Okay, well, I guess we are now online, given time and broadcasting. And good morning, everyone, for those that are in Europe, and about the others, depending on where you're standing now. So here we have today connected myself, Manuel Mameli from Lausanne, but especially Bart de Strooper, our keynote speaker of this morning. So Bart, thanks a lot for connecting at this early morning hour. And so we have Bart here. Why we have Bart sitting with us today? Because he's going to tell us about the secrets of Alzheimer's disease in a sort of way. Bart is an expert in the field, studying neurodegeneration in general, more specifically for the context of today, Alzheimer's disease in his intracellular form. How does that happen? The trigger motion. His career has been devoted indeed to analyze what are the pathways that may lead to the disease, and especially the disease in humans, by using and comparing, paralleling with animal models. So without further ado, I will let the stage to Bart for this talk. I invite all of you to ask a question through the QA answer, and we are going to deal with it, I would say, at the end of the talk, once Bart has told us everything. So the show is open and the stage is yours. Thanks, Bart.
Thank you very much, Manuel. So good morning to everybody. This first slide summarizes basically the raising insights into Alzheimer's disease. And so as you can see, I have this indicated this biochemical phase, which a lot of focus has been over the last years, over the last decades, in fact, because these are the typical biochemical lesions you see in the brain of patients when you look in the microscope. And these are abnormal accumulations of the A-beta peptide, these amyloid plaques, and then abnormal accumulations of the tau protein, which are these neuronal tangles. And so a lot of work has gone through this. The other side of the work has been on the clinical representation of Alzheimer's disease. And so, as you all know, Alzheimer's disease presents as dementia. If you look in the brain, you can see brain atrophy and a couple of lesions. And nowadays, we are also able to visualize these biochemical lesions in the brain. So basically, we can define Alzheimer's disease as a dementia with presence of amyloid beta and abnormal tau. The big oversimplification in this is that it takes actually more than 20 years to come from these biochemical lesions, the first appearance of these biochemical lesions in the brain, and the clinical phenotype dementia. And the second big problem here is that dementia is a symptom from many other diseases, other neurodegenerative diseases. So it's a symptom, and it's not really sufficient to talk about Alzheimer's disease. So over the last 4 or 5 years, we have started to look much more about what links these biochemical lesions to the brain atrophy in the late stage of the disorder. And the reason is obvious. If you can work on this intermediary phase, you will probably prevent dementia. And so the cellular phase is characterized by changes in all the cells of the brain. So today I will talk about the insights we have in this cellular phase. So a second thing I absolutely need to clarify is that even sporadic Alzheimer's disease, so the large bulk of Alzheimer's disease, has still a very strong genetic component. So ApoE4, apolipoprotein 4, is the strongest risk factor. But Swedish studies in twins have shown that about 2/3 of the risk of Alzheimer's disease is probably linked to your genetic makeup. And so this genetic makeup has been partially characterized in genome-wide association studies. And here you see the classical Manhattan plot. And you see here at least 40 genes now, which are canonical genes, or gene loci, to be more precise, which have been associated at genome-wide significance with Alzheimer's disease. But below this threshold, there are also many genes, as you can see here, which show a certain association, a certain statistical certainty to be associated with Alzheimer's disease. And these many other genes add to, when you calculate or when you add them to a polygenic risk score for Alzheimer's disease, they add to the risk for Alzheimer's disease. So showing that also these genes, which are the gene loci, which are below the threshold, are adding to your risk of Alzheimer's disease. And so this makes it very feasible to ask a couple of important questions. So the first is, of course, how do we use this genetic information to generate a functional hypothesis for Alzheimer's disease? The second point, which is very important, is to ask the question, how and under which conditions will genetic risk manifest? And it's important that you realize that these gene loci, which have been identified, are risk loci. So that means that they will only manifest when you have risk. You can compare it with driving a car with a belt or without a belt. When you drive normally and you have no belt, you will not really know, or you don't see the impact of not carrying this belt. But when you have an accident, then the belt becomes important. And it's the same with genetic risk. It's only when you test it in a situation where the risk is important, you will see what its role is in the disease process. And that makes it, of course, very difficult to understand these genetic risk factors in research. And then finally, so this is the essential question then, can we investigate these risk genes in a pathological relevant condition? And so the problem is that you need a good model to do that. And Alzheimer's disease has no good animal models. We have mouse models which develop these amyloid plaques or these neuronal tangles, but none of these models have a convincing link between the amyloid plaques and the neuronal tangles. And they also have no convincing link towards neurodegeneration and dementia. So some aspects of the disease are recapitulated in these two different models, but there is no complete model. On the other hand, we can use brain material from patients suffering from Alzheimer's disease, but that's really the late stage, that's post-mortem. There are changes there which are caused by the agonic state of the patient, and we miss completely the initial phase of the disease, which is, in my opinion, the most important one to study. So we were thinking, can we bring together the human genetic data, which are clearly linking molecules to disease, with experiments coming from the mouse models, mouse transcriptomic data. And so in the next few slides, I will show you that we can. So here you see the setup of the experiment. So we took amyloid mouse, APP we call it, amyloid plaque forming mouse, controls and transgenics, tau tangle forming mouse, controls and transgenics. We took the hippocampus at 2 stages, 4 months, which is initially of the disease, in early phase of the disease in these models, and 10 months, which have behavioral changes. So the 2 mouse have similar behavioral changes. And so we can look at 4 months and at 10 months what happens, and we can compare the wild type with the transgenic at age, genotype, and we can also study the age and genotype interaction. I have no time to go into the technical details, but we can look to the final results, which are very instructive. So you have here the comparison over age between the tau model and the APP model. And you see here the differential expressed genes listed over this graph. And what you immediately see is that most of the genes cluster around this transversal axis, showing that changes in the tau and in the APP model over age are rather similar. So these 2 mouse models age in a similar way. When you compare the APP, the amyloid mouse, with the tangle mouse, and you compare and you do the same comparison, then you see that there are dramatically differences in the way that the tau affects the expression of genes and the APP or the amyloid plaques affect the expression of genes. And you see here, for instance, these yellow changes along the tau axis. These are genes which are mainly involved in neuronal function. So it's very interesting. Our mouse mainly downregulate genes involved in neuronal function. And this fits with the idea that the tau pathology is very close to the neuron pathology. For the amyloid and the cognitive changes in Alzheimer's disease. For the amyloid beta pathology, you see complete different patterns. So most of the genes are upregulated. So these are the downregulated genes, these are the upregulated genes. Most of the genes are upregulated, and they are a complete different bunch of genes. There are genes involved in inflammation, neuroinflammation. So the conclusion is that there is very little overlap in these 2 mouse models. So the tau changes neuronal genes, the amyloid changes inflammatory genes. And so when you look for the age genotype interaction, you see that there are little changes in the tau axis. So the changes are occurring immediately and affect neuronal function very early on, as soon as the tau transgene is active. But for the APP, we see that over time, many more genes start to be changing. So there is a time effect in the amyloid mouse. So now the whole challenge was here not to identify what happens in the mouse, but to see what happens in human and whether we can make sense out of the changes in the mouse and to what extent the changes in the mouse reflect the genetic risk of Alzheimer's disease. And to do this, we go back to the GWAS studies, which I was explaining to you, and where you see that at the genome-wide, well, slightly below, so at the genome-wide, which is 5^-8, we have 40 genes, should be listed here. And then if we increase or lower, basically, the threshold for the p-value, the cutoff, you see that we get increasingly more gene loci included in the analysis. So for instance, at 0.1, which is a high p-value, admittedly, you see that about 2,700 gene loci contribute to the overall risk of Alzheimer's disease. And so what we did here is take this list of Alzheimer's disease genes or loci and our age genotype and age genotype interaction, and we did gene set enrichment analysis. So we looked for the presence of these genes in the changed genes in our different comparisons. It's a complicated explanation, but we have published this. You can find that back in the paper of Enrique Sirksma in Embo Molecular Medicine. But so to make a long story short, what we found is that there is only a strong enrichment of human genome-wide association gene candidates in the interaction of amyloid with age. So there you see that whatever threshold we take here, there is a strong enrichment of Alzheimer's disease risk genes. So this brings, so when we look then to these AD risk genes, they are all enriched for inflammatory functions. Not surprisingly, as I told you already, the genes which are changing in the amyloid mouse are mainly involved in inflammatory functions. So immune response, regulation of cytokine response, etc., etc. All these categories are enriched in the response in the mouse to the amyloid plaques. And so here you see an interesting intersection. So we take here all the genes which are identified in GWAS studies for Alzheimer's disease at the p-value of 0.001. We take here the interaction of all the genes which are differentially expressed in the APP transgenic amyloid plaque mouse. And then we take all the genes which respond to the amyloid APP blue module. And you see here that in the intersection of this, we find 18 genes, some of which have been identified as the canonic genes for Alzheimer's disease, so the high risk for Alzheimer's disease, including ApoE, clusterin, CD33, ELC gamma 2, which are very well-known genes associated with Alzheimer's in GWAS studies. But then we find also a couple of genes which at the same level of risk are identified in our approach and which were very low, with very low certainty associated with Alzheimer's disease in the GWAS studies. And these are this bunch of genes like a TREM2-like gene, GPC2, a SIG gene, XB, etc., etc. So this shows you that by this approach, we can enrich for interesting genes involved in Alzheimer's disease. And so to make this story, to wrap up this part of the story, so what we find is basically that the gene risk for Alzheimer's disease, that we find deregulated genes, which are in the loci associated with risk of Alzheimer's disease, they determine how microglia respond to the amyloid plaques. And here you see a couple of functions, again, repeated, as I said, and this is a representation of the microglia and all the different genes we identified in response to the amyloid plaques. So the conclusion of this story is basically that the genetic risk genes in sporadic Alzheimer's disease define an inflammatory response to amyloid plaques, much less to tangles, and point in the first place the microglia for the cellular response in Alzheimer's disease. Obviously, this is an oversimplification, I'm just saying that microglia are an important part of the cellular response. And as we will see later on, we have also data showing the involvement of the other cell types, as I indicated in my introduction. But let's now focus a little bit more on the microglia. And so we wanted to know what microglia are really doing in response to the amyloid plaques in our transgenic or in our knock-in mouse model for Alzheimer's disease. And again, to make a long story short, we took our mouse model, we look at 3 months, 6 months, 12 months, and 21 months, which gives you an increasing amyloid plaque deposition. And we isolated the microglia at the different stages to look what happens in the microglia response. And so here you see the classical cloud, the Disney plot of the microglia, the different. So every dot is a transcription profile of a microglia from those experiments, and they are all put together here. And you see that we get 6 large clusters of microglia, so different transcriptionally defined groups of microglia. So the most important one here is this amyloid response or activated response microglia. We have also these homeostatic clusters. But let's look to the data, to the essence of the data, which you can see summarized here. And so what you see here is these 6 different microglia clusters. And in brown are the 4 stages of the amyloid plaque forming mouse. And you see that this homeostatic cluster is relatively stable in the amyloid mouse, and you see the same. It's a little bit higher. There are more homeostatic microglia in the control mouse. But then you see immediately here a strong decrease in this homeostatic cluster 2 in the amyloid plaque forming mouse over time. And that's shifted, these microglia are shifted to another profile, which is this activated response microglia. And you see that this increases very rapidly in the amyloid plaque mouse. You see the same change, but much less outspoken and only at a very late stage. 21 months is very old for a mouse. You see that also in the aging normal mouse, we see this activated response coming up at 21 months old. So this is very interesting. The amyloid plaque forming mouse induce a response in the microglia, which you can see to a much less extent in aging microglia. And so we can then also do pseudo timing. So here you see all these microglia again, but now put in an order the most similar microglia closer to the beginning of this pseudo timing representation. And then you see that basically these microglia may evolve into very dark group and an activated response microglia group. And so to make, again, a long story short, you can then go for the gene expression in these different arms. And you see here a strong enrichment in the activated response microglia of ApoE. So ApoE becomes becomes more than 100-fold upregulated in microglia which are exposed to the amyloid plaques. And so, as I said already, ApoE is the major genetic risk factor for Alzheimer's disease. And this is really surprising to see that this major risk factor, which is usually expressed in astroglia, appears to become strongly upregulated in microglia when they see amyloid plaques. There are also other genes, but I have no time to go into the details. And so this is another representation to show the strong ApoE upregulation in the amyloid response genes. So where is this ApoE expressed? Here you see an in situ hybridization experiment with an amyloid plaque in the brain of our mouse. And you see the nuclei in blue. And then you see here the ApoE mRNA expressed in this microglia clustering around the amyloid plaques. And it's really only in the amyloid plaque exposed microglia that you see this upregulation of ApoE, as you can see here also. So this brings us to the question whether ApoE is really important for the microglia activation, or is just a marker of a cell state of the microglia. So we went for an ApoE knockout and repeated our single cell experiment as shown here. And again, to make a very long story short, here you see the different categories of the percent of microglia per genotype in these famous activated response cluster. And this activated response cluster is in fact very similar to the DAM cluster as defined previously by Ido Amit. So you see, if you take ApoE away, that so you see here this amyloid mouse has a lot of this ApoE positive or increased expression microglia, and you see that in the ApoE knockout, this cluster almost disappears. Another cluster, the immune response, the interferon response cluster, which is also increased in the amyloid mouse, is not affected by the ApoE deficiency. So it's very specific it's very specifically needed for the ARM or DAM response of the microglia in this mouse model for Alzheimer's disease. And so when we look again to the amyloid plaque here, now this time in white here also, and you see here, this time in red, the ApoE, but it's now the protein which is stained. So it's an immunohistochemical staining. The previous one was mRNA staining. And you see that this amyloid, this ApoE is secreted by the microglia and binds to the amyloid plaque. So it's really covering the amyloid plaques with ApoE. And here you see these activated microglia, which are swollen and which are associated with this amyloid plaque. And so in the ApoE knockout mouse, where we had no activated microglia anymore, first of all, you see this much less dense amyloid plaques, indicating that ApoE has an effect on this amyloid plaque formation. And then what you see also is that the microglia are present in this brain, but they are not forming these clusters of activated microglia around amyloid plaques. So this clearly shows that the major gene, the major risk factor for Alzheimer's disease, is involved in the response of the microglia to the amyloid plaques. And so the conclusion of this part of the story, and I could go in all the details, but is that ApoE, the GWAS risk factors of Alzheimer's disease, not all of them, but several of them, age, as I've shown you, and gender, for which I had no time, but the female microglia become much faster into this activated response mode than the male microglia. So ApoE, GWAS risk of Alzheimer's disease, age and gender affect microglia response to amyloid plaques. So this brings me to the next step in my presentation, and that is what basically happens now in this amyloid plaque niche. So I showed you, and this is a picture of Alois Alzheimer, and he was already obsessed by the cellular changes in this amyloid plaque niche, the cellular niche around the amyloid plaque, as you can see here. And here you see a picture I took from Michael Henneka, which shows this extreme inflammation of microglia around the amyloid plaque. But it's very clear that it's not only microglia which are responding to these amyloid plaques. And so what we wanted was an unbiased approach to what transcriptional changes occur close and far away from the amyloid plaques. And that's why we introduced a couple of years ago spatial transcriptomics in my lab. And just before I start with discussing those data, here you see again our mouse model represented at 3, 6, 12, and 18 months. And you see the accumulation of the amyloid plaques, which is really massive at 18 months. But when you look at microscopical level, you see here these amyloid plaques, and you see now not the microglia stained, but the astroglia. And so the astroglia also give a big response to these amyloid plaques, which is relatively mild at 3 months, but it becomes really massive at 18 months. And so we wanted to know what happens in the cellular niche of these amyloid plaques. And so here you see basically the setup of the experiment. We take a slice of 10 micrometer, which we stain. We have a slice here of 10 micrometer, which we use for the spatial transcriptomics. And we have another slice of 10 micrometer, which we again use for staining. And so these two slices give us morphological information of the transcriptional changes, which we will analyze in this slide. And so here you see a picture. This is a brain slice on top of such a spatial transcriptomic slides, which we did together with Joachim Lundeberg and his collaborators. And so what you do is you permeabilize this tissue, and then you have the mRNA will diffuse on these spots, and on these spots you have poly-T sequences and a barcode, which is of course extremely important for the final result. Then the mRNA is binding to these spots, and then you can afterwards release the cDNA. And thanks to the barcode, you know where the mRNA, which you have sequenced here, is coming from, and you can reconstitute this picture, but this time with the transcriptomic information all over the place. And so then you can do this type of Disney plots again. This time not every spot here is now a tissue domain or a cellular domain around an amyloid plaque, or a tissue domain from the same size. And you can do expression analysis. And again, just to show that this really works, you see that we can cluster, for instance, the hippocampal areas in different clusters of this, that is tissue domain can be clustered according to different hippocampal areas. And you can cluster them also according to disease, which gives you the other axis here, where you see, for instance, that the wild type of 3 months is here, that AD of 3 months is here, and that then and that the other groups also cluster below. So this shows you that our sequencing worked. And then we have a lot of information of every tissue domain. So we know where it is in the brain, we know whether there is a lot of astroglia, etc., etc. But we have also this plaque index. And so what we did is we developed a method to automatically assign a level of amyloid exposure to a tissue domain. So we use tissue domains with 3 different exposures of amyloid. And so we have a quantitative way to measure that. And here you see then again this Disney plot, and we overlay here the intensity of the amyloid plaque spots. And of course you see here in the young mice, in the cluster of the young mice, non-amyloid mice, that there is almost no plaque staining. And here you see the plaque staining in the tissue domains of the older mice. And so now we can do very interesting all kinds of analysis, again, just in a nutshell. So we have 10,000 transcriptomic profiles, 4 time points, wild type and transgenic. And we have a regional annotation, and we have a plaque index for every spot. So we can ask here, what are the molecular alterations in the plaque niches? We have a temporal resolution, we have a genotype difference, and we have a spatial resolution, close to plaques versus distance to plaques. So here you see a picture again. And so I will show you one graph with real results. And so the graph is as follows. So you have here the axis of plaque exposure. So we have data for every gene, to what extent it is exposed to plaques. We can then do a differential expression of transgenic versus wild type, so genotype in this axis. And so here you will see the genes which are strongly upregulated in the amyloid plaque niche. And here you will see genes, for instance, which are upregulated in non-amyloid plaque area. And this is according the genotype axis, and this is according the plaque axis. And so here you see a part of the data. They are really massively, and I invite everybody to mine the data, which are completely available on our website. So here you see a first group of genes in red, which are which turned out to be upregulated according to the amyloid plaque match niche very early at 3 months, when there are no other symptoms in the mouse. And so we think that this is a kind of protective response to the amyloid plaque, increasing amyloid plaque loads. And when we analyze those genes, they are all oligodendrocyte or myelin related genes. And very interesting, at 18 months, this cluster of genes is downregulated. And then on the other hand, we have the plaque-induced genes, PIC we call it. These are this purple group of genes, which are not affected in the beginning at 3 months, but which then becomes hugely upregulated, both according to the genotype and the amyloid plaque niche. And when we look to the identity of these genes, they are all related to microglia astrocytes. And you can see the annotation here. And when we did an overlap with the arm phenotype, which I just described to you, there is a part overlapping with this activated microglia. There is also a part of the genes overlapping with the previous identified activated astrocyte phenotype. And then there are several genes which are more or less novel, and which have not been associated to these genes in the past. And so here you see the list of the genes, no time to go in every gene, but overall, we see a strong enrichment in gene ontology for complement activation, endocytosis, lysosomal degradation, oxidative stress, immune response, and antigen processing and presentation, which is likely to overlap with the ARM response. So the most interesting and fascinating result is basically here summarized in this plot. So we have here all the ARM genes, all the big genes put around, and you see here how they interact with each other in a WGCNA analysis. So basically, we analyzed here how these genes change with each other together in different conditions. And then they automatically clustered in this green group of genes, which are namely astrocyte genes. And you see here how they are co-regulated in the wild type situation. And then here a bunch of microglia genes, which are also weakly co-regulated in the wild type situation. And then these genes, which are not much co-regulated with neither the astroglia genes or the microglia genes. And then you see here for increasing exposure of amyloid. And then all of a sudden, these genes start to interact with each other. So I mean, they start to be co-regulated with each other. And so this suggests very strongly that the astroglia genes here and the microglia genes here are co-regulated over these two cell types. And also these genes, these extra new genes, become incorporated in this WGCNA network when exposed to high levels of amyloid plaques. So this brings me to the next conclusion. The PICs are a gene regulatory network over microglia, astroglia, and oligodendrocytes, coordinating the cellular response towards amyloid plaques. So I'm going to skip this conclusion slide because I mentioned all these things already while speaking. And so the last 5 minutes, I just want to share an initial model we are developing now to study and to translate these findings into real human microglia and in real human brains. I'm not going to talk about our neuron chimera mouse where we transplant human neurons derived from iPSC cells in a mouse brain and have very interesting phenotypes, but you can find some details about that work in our paper in Neuron. But I can tell you that we developed this model now to a very, very nice full model, but I have no time to explain that today. But I'm just going to briefly talk about the work of Mancuso, Renzo Mancuso in my lab, who developed a nice mouse model for human microglia. And one of the reasons that we did is that we realized that although the mouse model, as I've shown to you, mimics aspects of the human disease, a mouse brain is not the same as a human brain. And a lot of genes which are expressed in human microglia are not very well expressed in mouse microglia, as I will show you. So we developed a mouse model where we could transplant human microglia derived from iPSC cells into a mouse brain. And here you see a section of a mouse brain, and every red dot is a human microglia, which has replaced the normal mouse microglia in this brain. So you see, it's a very effective method. And here you see characterization of these microglia. They are sitting in the same niches as the mouse microglia, as you can see here. So they really go and fit themselves in the mouse brain. And here you see then pictures of these microglia nested in the mouse brain. And so then, I mean, is this important? Well, I can show you here an experiment where we injected amyloid beta into the tail vein of a mouse transplanted with human microglia. And we tested the effect of this amyloid, which goes into the brain, on the expression profile of this human microglia and the mouse microglia. And here you see that this is really, here again, Disney plots, no time to explain, but you see that, that the beta has massive effects. And here you see a temporal curve, again, such a pseudo timing. And here you see profiles of individual genes, which are changed when the mouse and the human microglia are exposed to the amyloid. And you see really that in these clusters, which are chosen because they are overlapping or similar in the mouse and the human, but you see that in these clusters, these genes have dramatic different responses, especially here, as you can see here in the human mouse, in the human microglia, the extremely different responses compared to the mouse microglia. So it's important to take the human component in the modeling of Alzheimer's disease. And so if we briefly compare the transcription profile of the mouse microglia, and we look to genes which are relevant for Alzheimer's disease, like APCA7 or BIN1 or other genes which have been identified in GWAS studies, SORAL1 for instance, then they are present and they are interesting to study in the mouse microglia. But when we use human microglia, you see a staggering increase in genes which are not really very well expressed or are very different in composition in the mouse microglia. And you see here really the relevance to use human microglia to study the genetic risk of Alzheimer's disease, in particular when you want to do gene interactions and try to understand how a polygenic risk score affects the behavior of this microglia. And so this work is underway, and I hope to talk about that in a next seminar. So the overall conclusions of my talk are that in sporadic Alzheimer's disease, amyloid plaque is a trigger, not a driver of the disease. So you can have plaques, but you don't need to become diseased if the cellular response becomes under control. And so the cellular response, I want to expand it from microglia to astroglia and other cells, is determining whether you get Alzheimer's disease or whether you are protected against the trigger of amyloid plaques. The neurodegeneration is probably downstream, or it's very likely downstream of the neuroinflammation. And neuroinflammation, you need to see it broader than microglia. It's a response of the oligodendrocytes, astroglia, and microglia. And tau is downstream of the cellular interaction, as I tried to suggest in my first part of the talk. So the people involved in this work are Wei-Ting Chen, who has really been the driver of the spatial transcriptomic work in my lab, and then Mark Vierse and Ashley Liu, who have done most of the bioinformatics with a lot of clever tricks to analyze the spatial transcriptomic data. Carlo has pioneered the single cell analysis of the microglia work, which I've just shown to you, together with Nicolas Fattorelli, who was responsible for most of the bioinformatic analysis of those experiments. And then Nicolas works also together with Renzo to develop this microglia model I presented to you at the end of my talk. And then Annerieke Sirksma, and it should be an R here, Sirksma, has been central in the first part of the talk where we compared mouse and human for their responses to amyloid plaques. And also she worked together with Ashley and Mark for the bioinformatics. So thank you very much. This is my final slide with acknowledgements for my different funding resources, and a drawing here of the UK Dementia Research Institute, which is a fantastic initiative in the UK, where we bring together centers in different top universities all over the UK to work on dementia, and where we hope to find cures for this devastating disorder. Thank you very much for your attention, and I will stop sharing my screen and see whether there are any questions.
Thank you, Bart.
Thank you, man.
As usual, incredible amount of data, and I invite all of the attendees to share your question across the Q&A, and I will then lead it to Bart for the discussion. For the moment, people, I guess, are still writing, but then I have a couple of points maybe to launch already the discussion. Bart, according to you, okay, microglia seems to have, together with astroglia, a central role. But then, if I'm not mistaken, what you provide us here is a sort of a snapshot in different momentums that are going across the disease. And you call this activation of microglia. But here I wonder, how far or how close are we to understand the real temporal dynamic of these microglia? I mean, I guess these guys are going up and down, controlling very timely this. So what do you think we are still missing, and at which level
we
of
are
analysis?
missing a lot. They are missing a lot. I think the main thing here is that all of a sudden this simplistic view that amyloid is causing synaptic dysfunction and then you get a whole disease becomes a really complicated interaction of different cell types where you get protective and damaging responses. And I didn't have time to talk about that, but the main drive of my research is to understand why certain cellular responses will keep your brain going even if amyloid plaque is there, and what responses are going to lead to a decaying brain and a collapse of the brain. And so I didn't talk about the neurons and the neural circuitries, but in the end, it's all about how long you can keep your neural circuitries working normally. And so, the point I want to make here is that your microglia and astroglia are a very important aspect of the defense of your circuitry towards these accumulating proteins. And so that's basically it. And so the second point is here, the dynamics of the whole system. That's also why I think that we cannot go further without our mouse or animal models, because the only way to capture the process over time is using such models where you can start from the beginning, where patients, humans, have no signs, nothing. So you even don't know. So it's even making our studies, case control studies, complicated, because you probably also have in these control groups patients which have biological Alzheimer's disease without any clinical phenotype. So we cannot capture that very well in humans. So we need the models there. So that's an answer. A second answer. And then the third part of the answer is that indeed all the cells have very dynamic responses, and it's probably that they become that you have upregulation of this ARM response, but it could be then downregulated again at phases where the disease is less pronounced, or whether when there is something, it could be that an inflammation, peripheral inflammation, activates the whole thing and drive the disease a little bit more forward. So it's complicated interaction which goes over decades, then to finally end up with the collapsed brain. And then it's too late to treat also. I think that's a very important realization. We don't have to treat Alzheimer's disease. Well, we should try to do the best what we can for these people, but we will not have a good treatment at that stage. So that's also an important insight.
Okay, Bart, you have a question from Andreas Nold.
Yes.
I read a question for you maybe, so people get to know. Would it be correct to say that there is a transition between a first phase, which is potentially reversible, and in which amyloid puts stress of the tissue, or this triggers the disease, or a second phase that is rather tau-driven? How do you see it?
I agree with that type of thinking. And we know basically that this is the case from the familial Alzheimer's disease cases, which are all mutations which increase amyloid beta or change amyloid beta. And so they all end with dementia, and they all end with tangles and tau pathology. And the tangle and tau pathology is in these patients, again, more correlated with dementia than the amyloid plaque accumulation. So there is a dissociation between the clinical phenotype and the initial biochemistry of the disease. And so the model which I try to present fits much better with such a complicated, explains or potentially explains much better the disease even in the purely amyloid-driven pathology. And so the logic is indeed to think then, because it takes so much time, that you have a whole time where this amyloid is in your brain and irritates or stresses your brain, but your brain is just able to cope with it. The microglia, the astroglia, the neurons, we know very well that neurons and circuitry have homeostatic responses to amyloid, and Nina Slutsky published on that. So there is a whole homeostatic response to this stress, and that takes years before it's broken. And part of the breaking of the mechanism is the tau spreading throughout the brain. That's the second hit, which is probably already, and I don't know whether the tau is really driving the neurodegeneration at that moment. It could be that the tau is part of a bigger game there, where the brain, where the neurons are collapsing. We have ourselves shown that the neurons in Alzheimer patients, and also in our mouse model, in our humanized mouse model, they start to show necroptosis markers and the kinases are phosphorylated of the necroptosis markers. So in parallel with the tau appearance, you get also necroptosis markers which become stained, and that's also kinases. So I think that while the neurons are collapsing, a lot of kinases get activated. They phosphorylate the tau, which disconnect from the microtubuli and is then able to form tau inclusions and tangles. And they activate other kinases which induce necroptosis. And maybe there are other aspects there in the cell which we don't understand. So I think it opens a lot of interesting boxes. But I also think that that stage is already a late stage to do something to prevent the whole system. So at that moment, we need to stabilize the disease or do whatever we can to halt dementia, yeah, basically.
Maybe I have a provocative discussion to launch here. I was hit a little bit by the humanized mouse model that you proposed there, where you derive these human microglia and then reinsert them, let's say, in the mouse brain. I think back in time there has been also this speech gene that have also been looked at for that, the FOXP2, if I recall correctly. Now, we get these microglia that are human microglia. We put them in a context that is mouse neuron, astrocytes, circuit, and so on. And I understand that then we can get some relative information, but then what are the pitfalls there? What do we risk?
Zillions. I agree. Every model has its limitations, and the art is to try to understand, to take into account the limitations and then to make conclusions. So what I also think is that if you do mouse work or model work, you have also to go back to the human patient and try to see to what extent your predictions can be confirmed, but it's always limited. The functional predictions are very difficult.
But do you think that the gene...
I will, just to say, we have a control there. So we take the microglia, the human microglia, and we transplant them in again, an immune deficient mouse, but we transplant it in the brain and we look what happens. And there we see that these microglia have a very homeostatic phenotype. And we compared those microglia with microglia we derived from surgical resected specimens from humans. And we could show that there is a very strong similarity in the expression profiles we get from the human microglia transplanted in a wild-type mouse brain with the human microglia isolated from a patient. So I think it's a very good model for human microglia. It's probably one of the best we can have at the moment, because if you put human microglia in a dish, they become very activated and do not show that phenotype. So within the limitations and agreed, so there are probably human, for instance, one very nice example is we need to humanize in the mouse the CSF1, the CSF1 growth factor for microglia, which is essential to keep them going. And so the mouse CSF1 has a couple of amino acid differences with the human CSF1. And so if you transplant, we have in this mouse the CSF1 gene, we have transformed, other investigators at Regeneron have mutated those genes to humanize them, so that these mice can receive human microglia. And so that tells you already that how important it is to know the difference between the human and the mouse molecular biology. And so there are probably other aspects which we will not be able to model in this model. For instance, if there is a human-specific gene, and working on that too. So we have also these transplantations of human neurons in the mouse brain, and it works remarkably well. Pierre van der Hagen also, we use that model, and then we bring in, we didn't do that experiment yet, we can bring in the human microglia and see how the human neurons interact with human microglia. And the reason to do this is that you need a three-dimensional structure of the brain. You can try to model that in dishes, It's not easy. So the only alternative I see is organoids, human brain organoids, where you would have these interactions. But there we have the problem that it's so difficult to get amyloid plaques or neuron tangles in those organoids. There are close to what we see, to the extent we were able to model this already in mouse. And so what we do with our approach is just go a little bit faster than the organoids and trying to do human modeling in the mouse model while others are developing better and better organoids.
Thanks, Bart. So we have 2 minutes before we are automatically locked. So Tara is a freshman and probably a very young scientist, and I would say she deserves to have a little bit of this question. And basically she's looking at, she's asking whether the difference in genetic between families more sensitive or susceptible lies on microglia differences. Bart, your quick comment.
Well, she apologized for her question, but I think this is just a brilliant question. That's the whole essence of what we try to do here. Yes, I think that that some people are completely protected against Alzheimer's disease or against the amyloid plaques and the tangles, thanks to their genetic constitution. And we want to know these resilient genes to do something, to use that knowledge to protect those who don't have this resilient gene. So in essence, this is the main question of my group to find that. And it's not only in microglia. I'm sure that there are also resilient genes in other cell types. But the microglia are certainly, as the GWAS studies tell us, part of this important piece of the puzzle. Thank you very much.
Perfect. So in 30 seconds, I would like to thank, in first place, Bart for being available for this, all of the attendees for listening to these new studies and new avenues into Alzheimer's disease. And I invite all of you to keep following NeuroMatch. And Bart, has been great to have you here this morning. And thank you all for for being here. Thanks, Ali, for your technical support. And
okay, I think most attendees are here, so let's start. So welcome everybody to another session at the NeuroMatch 3 conference. I'm Christoph Metzner. I will be the host of this session. And today we have an interactive session, which means we're pretty much relying on the attendees to ask questions, and we want this to be very active or interactive. So if you have questions, put them in the Q&A window, and please also indicate if you'd like to come on stage to ask your questions. Also, a big thank you to Ali, who's running the whole room here on the back end. And yeah, so let's come to our first speaker. First speaker is Jean-Jacques Orban de Sivry. He's from the KU Leuven, and he's a professor in the motor control and neuroplasticity research group. And he will talk about are age-related changes in internal model function linked to poorer somatosensory function. So JJ, the floor is yours.
Yeah, thanks for the introduction. Thank you all for coming or attending the talk. So this question here was motivated by the following Twitter poll that I put a few weeks ago online that asked about the link between somatosensory acuity and internal model recalibration. And 80% of the people who actually responded to this poll were convinced that there was a link, while this actually contradicted what we have found in the lab in 2 different studies that I'm going to describe now. So in the first studies is about the task curve and cam feedback task, where people don't see the hand and they have to bring the hand from the starting position to the target here, the black circle. And they are told that the cursor that they see on the screen is completely uncoupled from their hand movement. And so the cursor will always go in this direction to the left, while the hand has to go to this target, even though they don't see the hand. And what happens is that with training, people start to drift away from the target. And this is represented here on this graph. So you have trial number, and this is the hand angle. And you see that a group of young people here, on average, they drift around 15 degrees after 400 trials. But what was interesting to us is to see that when Kuhn ran this experiment in older people, that this implicit adaptation, this drift of the hand, driven by the deviation of the cursor, was larger in older people than in young people. And so we were wondering whether this was actually linked to the fact that older people could not localize the direction of the hand movement properly. And so it was a sort of proprioceptive prediction error. And if you don't know where your hand goes, then of course you might be tempted to let your hand drift further away. So Kuhn, in his next experiment, tested this where people were asked to reach to a target, and then the robot could deviate the hand in one of those directions that are represented with color lines. And people were asked to report whether they were on the left or on the right of these targets. And we did that for many trials, perturbation on the left, on the right, and so on. And we were able to build psychometric curve. And the slope of this psychometric curve gives us an idea of how accurately people can localize their hand direction with respect to the target. And when we do look at the slope for young in blue and old people in orange, we see that there was no difference in how accurately people could localize their hand and the confidence in this hand direction detection task. And so that cannot, of course, then explain the large difference that we saw in implicit adaptation. We also tested other sort of somatosensory function tasks, for instance, a position matching task, where people, with the robot move one hand and you have to move your other hand, and there was no link. So now that was one task. We had another task that's called the sensory attenuation task, where you received a force on your left hand, and then you, by pushing with your right hand on your left finger, you have to reproduce this force. And when people do that, for instance, they have to reproduce a force of 2 Newton, and when they push themselves, they actually push 3.5 Newton. So they push more than they have to. And this is actually not because they can't feel the force, because when they reproduce the force indirectly, they actually do that properly. And this is those to arise from this model, where proprioception is in a Bayesian way merged with a prediction from the internal model, and that gives us this overcompensation in force. And of course, the idea is that if you would then have worse proprioception, you would rely more on the prediction, and you would have even more overcompensation. And so this is what my student Manasa tested recently, but with the arm rather than with the fingers. So here, the subject first received a force in his on his left arm. Then he has to reproduce this force by moving a slider with his right arm, for instance. And so moving the slider produces a force on the left hand. And so the yellow arrow has to match the red arrow. Or you can do that also here by exerting a force with your right arm. So the right arm pushed to the left, the force is transmitted to the left robot, the left arm, and so produces these forces. And again, you have the yellow arrow has to match the red arrow. There are three possible forces, 4, 6, and 8 Newton. And when we do that in the slider condition, so in this first condition, so this is the force error. So that's the difference between the red arrow and the yellow arrow. And you see that for a group of young people and a group of old people, they're doing quite well. They're not pushing enough, so there's a negative force error. But if you look in the mirror condition, where they exert the force on themselves, then you see that for the younger people, the force error is still slightly negative, but less than in the slider condition. For the older people, there is definitely more overcompensation. Yet we do not think that this is linked to somatosensory function, because the slider condition, which is a measure of somatosensory function, is not worse in older people. And we also tested the position matching tasks to people in these old people, and they were also doing fine. And so basically we have here two experiments where, and this reproduces similar results from Walpay. But we have, so here two experiments where basically we found larger sensory attenuation or larger implicit adaptation, which are two proxies for internal model function in older adults. But this seems to be completely uncoupled from any possible deficit in somatosensory activity. So we're left with this question. This is why I chose this interactive talk. Why is there a difference, or what could explain this larger internal model function in older adults if it's not somatosensory function? Thanks for your attention.
Thank you very much, Jay. Yeah, so if you have questions, put them in the Q&A window. And please also indicate if you want to come on stage to ask them. Maybe to get the discussion started, can you comment a bit on, well, what your ideas are? What could explain this difference?
Yeah, so I mean, that remains a big question, something that we want to investigate, because at this moment we really don't understand why it is. I mean, we've tested different things, but so far we've been unsuccessful. So, I mean, there are two possibilities. Either somatosensory function has nothing to do, and then we need to rethink of the way we think about motor control, or our measures of somatosensation are not good measures, because those are actually conscious measure of somatosensation. So people have to report things. And that might not be what the brain is actually doing. I mean, we know that sometimes things that you perceive consciously are different than what the motor system uses. An example of that is the size-weight illusion, where people are still prone to the size-weight illusion, while the motor system can actually lift object with the right amount of force.
Okay, so Katinka, you wanted to, you're muted.
Yes, thanks, Jean-Jacques. It's interesting work. I would like to ask a psychologist question. And is there any chance that this could be related to an experimenter effect, where the elderly place more confidence in the authority of the scientist and assume more that any feedback they get from this person is correct, in contrast to students?
Yeah, well, the thing is that people are completely unaware. I mean, the manipulation in the sensory attenuation or the adaptation, people are completely unaware of what they are doing. They are unaware that they're pushing more, or they are unaware that their hand is even drifting. They just think that they're bringing the hand in front of them. So I don't think that this is really, that could be explained by a conscious, by the fact that older people would listen differently or apply the instructions differently. Yeah. I don't see how that would work in this context.
Thanks.
So we have another question by, Gwendolyn. Yeah, just go ahead. You're still muted, Gwendolyn. You have to unmute your mic, please.
I didn't. Okay. So did you do any correlational analysis? And that's one question. The second is, when you did all kinds of somatosensory measures, and I think the last one, the force matching, you see some differences. But that one is the most complicated one. How are you going to interpret that difference? That's just my thought. The other thing is, I know you observe more implicit learning from the air clamp in the elderly. Is it possible that the strategic learning is less? Will you interpret in that way?
Yeah. So so the last question, I don't think that in the task-driven clamp feedback there is any strategy. I think people are asked to basically apply, the only strategy that they have to apply is to bring their invisible hand on the target. So I don't think there is a difference in strategy there, or nothing that we have observed. So your first question was what was the type of analysis you asked about?
Did you do any correlation analysis? Because you can.
Yeah. So we did plenty of correlation analysis, and I mean those are significant group. I mean, it's 35, 40 people per group, so it's reasonable groups. It's not enough to detect very small correlations, but we didn't really find any strong correlation. Nothing that would be big enough to actually explain the difference we see. And your last question, what was it? Was your second question? What was it about?
Is how you're going to interpret the force matching data, because you see the group differences.
Yeah. So the force matching data, it's well, I mean the way we interpret it, I don't really know. I mean, we thought that it might be. I mean, that's what also Volpe suggested, that this would link to a worse somatosensory function in older adults, but that's not really what we find. So in the slider condition, at least in the group that I showed you, the older people were even better than the younger people in the slider condition, which is the condition that where, that is the best proxy in that task of somatosensory function. So it's difficult to really put forward this argument that older people would be worse, but at the same time, they have this larger sensory attenuation. So we don't really see any strong age-related decline in somatosensory function in the older people that we are able to bring in the lab. That doesn't mean that there is none, but even though they don't have any somatosensory impairment, they have this sensory attenuation. So might be that there's no link there, but yeah, that remains a big question.
Yeah, maybe the last reason is because the sensory integration is different. They give it different weightings to perception and the vision.
Yes, right? but it is. Yeah, but in theory, if you give a different weight, it's because the reliability of the signals are different, and so that's what Volpe proposed, is that if you have a worse somatosensory function, you will give a lower weight to proprioception, and that's what will yield larger sensory attenuation. So if you want to give different weight, you need to have a less reliable source of information. At least that's how they interpreted it, and that's not what we found.
We also have another question from the audience with a quick answer. So Aaron Wong says, we know there are differences between static and dynamic proprioception. Have you looked at both?
Yes, so the unlocalization task is the position matching task, is more static one, but the other one with the reaching direction is more dynamic one. And even there, we didn't find any difference. I mean, that's the one where we thought we would find something, because it's very close to actually what people experienced during the task and feedback task. But even there, we didn't find any differences. So yeah, I mean, that's dynamic. Of course, I mean, you can think of many different types of somatosensory function. I think the one we picked, we thought was the one that had the highest chance of yielding a good result, and it's not the case. So yes, I mean, we can test more. We haven't tested all possible somatosensory tasks, but I think if it doesn't work with that one, I don't really see which task would work. So yes, we had one static and one more dynamic active movement, which we thought was very similar.
Thank you. So we've run out of time. There's another question in the Q&A, JJ, so maybe just type in your answer there.
Yes.
And we will move on to our second speaker. Our second speaker is Katinka van der Kooi. She's a postdoc in Jeroen Smeets' lab at the Vrije Universiteit in Amsterdam. And she will talk about a reach trajectory can be learned using binary reward feedback. So Katinka, take it away.
Yes. Spotlight and present. So I'd like to start by first thanking everyone for the opportunity to present here, for the room hosts for volunteering to do this, and especially the people in different time zones watching. I'll talk about reward-based learning of a little bit more complex reach trajectory. But what is reward-based motor learning? So imagine these girls practicing their dance move, and they're not sitting in front of a mirror, but they have to rely on their coach telling them whether they did it well or not. Well, that means that they have to engage in reward-based motor learning. They have to add variability, so they have to change their movements following failure. And when their coach tells them they are successful, they have to maintain that behavior. Well, this works well in simple tasks. But when many kinematic factors can contribute to performance, exploring all these different factors induces excess variability that hampers learning. Well, those familiar with reinforcement learning might think, yeah, but there's a way to solve that. When we have the binary reward feedback and many factors that can contribute to the reward, what you could do is you could solve a credit assignment problem. For instance, in the extreme case where only errors in slant would contribute to rewards, you could find out that this is the main factor to explore and only add variability in this factor. It is known that humans can do this for relatively simple tasks at least, but in our lab we also saw that 3D learning is much more difficult than 1D learning. So in this experiment, we wanted to ask, does solving the credit assignment problem for the participant by providing factorized feedback on one factor at a time improve learning? We used a trajectory matching task where participants matched a 3D trajectory they viewed in VR and where they received only binary, so red or green, performance feedback. We focused on two different factors, slant of the trajectory and also the length of the trajectory. And of course, it's the composite of these two factors. There were two factorized groups and one non-factorized group. So the factorized group, so there was always a baseline phase without feedback, and we also ended with a phase without feedback. And in between, there were 3 learning sessions. So the factorized slant first group received feedback on slant first, then on length, and then on the composite. For a length first group, this was reversed. The non-factorized group, in contrast, received feedback on the composite of the two factors throughout the experiment. We wanted to ask, do participants in the factorized groups learn more? So does the orange group learn less? We measured learning as the difference between the absolute error in the baseline phase without feedback and the third learning phase. We started out using frequentist statistics, non-parametric tests, because we're dealing with absolute errors. And based on reviewer comments, we also added Bayes factor for interpretation of the strength of the evidence. So before we say anything about factorization, of course, we want to know, can the factors be learned? I'll walk you through these graphs one by one. On the left, we have the composite, so the multi-dimensional task. What you see is in thick lines is the group medians going from baseline to the third learning phase. You see there's a lot of variability between participants. Therefore, the effects are difficult to see. A Wilcoxon-signed rank test showed, however, that the composite task could be learned. Bayes factor showed that the evidence was only anecdotal. For the other two factors, we see similar results. So here we have slant, again, a lot of variability. Do I say it wrong? Yeah. A lot of variability between participants. Significant learning, and the same for length. Now let's look what happens with factorization. On the left, you have the factorized group. on the right, there's the composite group. And on the y-axis is learning. And learning is the difference from baseline to the third learning phase. We had expected that the vectorized groups would learn more. Clearly, this is not the case. So we do find moderate evidence that vectorization does not benefit learning. We found weak evidence that the ridge trajectory can be learned using binary reward feedback. I had to nuance these conclusions a lot based on the Bayesian evidence. In retrospect, we underestimated power problems in studying reduction of natural biases that vary between participants and also studying them in the multidimensional environment associated with a lot of noise. Thank you.
Thank you very much, Katinka. Yes, again, if you have questions, put them in the Q&A window and also indicate if you want to come on stage. Yeah, so, okay, JJ, you have a question? Go ahead.
Yeah, when I was, thank you for the talk first. Very interesting. When I was listening to your talk, it reminded me of this model framework called Optimal that is used in skill learning, for instance, for learning sport and so on, where they basically found that if you instruct someone to, for instance, pay attention on part of its body to perform a task, that person will do less than if you encourage this person to look or to at the the global movement or at the goal of the task. So you have to turn your body rather than make your hand like this and so on. And basically when they studied that more in sport and so on, they found that giving global instruction is much more efficient than giving detailed instruction about the movement. And so yeah, I wonder whether you feel that this is the same idea, that the fact that you don't really benefit from giving really the details. What's more important is really to have the global instruction, the global piece of information as feedback.
Yes, so what might be related, something that I looked at is, does performance decrease in the performance irrelevant vector? Visually, I thought I saw that, but there was no statistical evidence for that. But I think that definitely there are also theoretical reasons why you would think that you get a negative effect because performance in the irrelevant vectors reduces.
Thanks.
Do we have more questions? So if you're in the audience, you can also raise your hand to come on stage and ask questions.
Otherwise, I have a question if there's none.
Yeah, sure. I mean, let's do it as interactive as possible.
Right. So in retrospect, we had this problem of dealing with participants starting at a different baseline level. Of course, we knew that before, but in retrospect, we also thought more about it. And I thought perhaps the right way to analyze this is with an analysis of covariance, where you use the baseline bias as a covariate. Would the audience agree with that?
Yeah.
Are you saying when you're doing the pre-test, you're using the covariance structure to quantify the baseline performance, right? But I'm just saying, I also have the same problem when I doing those model skill learning studies.
Yeah.
You found so huge inter-subject differences.
Yeah.
Sometimes we just want to calculate the mean or the variance, but it's just not enough. Sometimes it's really frustrated for that. So in fact, I just comments. This is a hard problem, and
yeah, hard. Yeah.
Yeah, unfortunately, the only way I did is the task you're using is actually complex because I'm also using virtual reality in a 3D space, and then we're having trouble. So sometimes I trying to do those 2D tasks.
Yeah.
And I have a question for you. Is the I didn't get it. How did you do the factorization? Because in fact, you quoted one of my early papers with Gregory Dan. We also doing this kind of a credit assignment, but I don't know how did you do it?
How we did it. So we have a straight trajectory slanted in depth, and we focused on the slant of the trajectory and the length of the trajectory.
You mean your score or your reward depends on the slant first, then
yes.
Oh, okay, I got it.
Yeah.
All right.
And the combination was just a factor difference.
Yeah, it's just in my 2013 paper is we found people can deal with those composite reward function.
Yeah.
They're just good at it. Yeah.
Yeah, so that I interpreted that correctly then, right? So that it looks like they can solve a credit assignment problem.
Yeah.
Or would you say something different?
I didn't see any difference. In your data, you also show the efficiency of doing that kind of credit assignment.
Yeah.
Okay, we have another comment from the audience. So Margaret Botsma says, we have turned to multi-level analysis to control for these baseline differences, studying the age effects on motor learning by adding participant as a random factor in the analysis. Yeah. Margaret, if you want, you can also come on stage and discuss further. Okay, and I think Jeroen wants to come on stage. Ali, can you pull him to the panel? You're still muted, Jeroen.
Yes, well, I can comment in the meantime.
Yeah, sure.
When you go to these more complex analyses and considering the variability you have in more complex movements in 3D, yeah, we would have needed a huge sample size. So it's good to be aware of these things. I always love going more complex, but it comes with a cost of measuring many participants.
Okay. And that's already quite a lot. How many did you have there?
I had 20 per group, so that's not a lot. I come from a psychophysics background and it felt like a lot, but it's not.
Right.
Okay, unfortunately, I think Jeroen has disappeared again. We're also running a bit out of time, so thank you again, Katinka, and thanks, everyone for this interactive discussion. And we move on to our last speaker for this session. Our last speaker is Quanlin Wei. He's a professor in the motor control laboratory at Peking University in China. And his talk is titled Sensory Motor Adaptation by Task Irrelevant Feedback. Stage is yours.
All right, let me find my PowerPoint. Share. Can you see it? Can you see my PowerPoint?
Yes, we can.
Great. So, it's great. JJ is in front of me, so I don't need to introduce a lot of background. So basically, we know visual motor rotation. What JJ just showed is air clamp, and we know this is one typical type of sensory motor adaptation. But people think it has two components. One is the implicit one, and one is the explicit strategy. Explicit strategy, basically, you can measure it by using those aiming report paradigm. And just before you're reaching for it, and you ask people where you're going, and that's the red cursor on the right graph, and your actual hand position is more deviated way if you are successfully adapt to visual motor rotation. Then we can decompose the total learning into two parts. One is the explicit strategy part. One is implicit part one, which is a kind of unconscious, which is more sensory prediction error driven. Then indeed we can find this is one of the data set. So if we ask people to do visual motor rotation twice, they will learn faster in the second time around. And if we look at it closely, as you can see, the black line is the total learning. That's your hand deviation. And you can decompose it into explicit part and implicit part. And during the second time adaptation, you will learn faster. Look at the black line, you learn faster. And also, people attribute to this kind of a saving effect as a recall. It's just a recall of your learn strategy, which is shown by the red line here. And my question in the current study is, I think there is a third component, that is, because when you are seeing this kind of a visual model transformation, which means cursor movement is deviated from the hand movement, and simultaneously, you kind of learn some conceptual knowledge of this kind of this visual model relation. Once you have this conceptual knowledge, it could lead to savings, leads to long-term effect of adaptation. So if that is the case, I can make 3 predictions. One is possible to learn or showing saving effect without adaptation itself, without forming strategy, without implicit learning. Once you know this concept, you should learn faster. At the same time, you see the same kind of cursor movement. The second hypothesis is, given its abstract nature, because it's kind of conceptual learning, this effect should be independent of error direction, error size, or even independent of task. I will show you later. My third prediction is not arbitrary visual model transformation can be learned. You have to learn similar concept to have this kind of effect. So what I do, I introduce so-called double cursor instead of a single cursor. Your hand is moving the green cursor forward, but there is task irrelevant cursor, which is the red cursor. The red cursor always goes, say, 30-degree clockwise or counterclockwise to your hand cursor. So instead of using arrow clamp, you can control the green cursor to hit the target all the time. So I call it double cursor. And the red cursor is the irrelevant feedback. I ask you to ignore it. And once you expose to this kind of double cursor, I can ask you to learn a regular visual model rotation. And how do people do? So this is a single cursor. Okay, here we go. So the first graph is the control group. You just see the first learning and the second learning. Indeed, people learn faster if they learn the visual model rotation twice. They learn 30-degree twice. And the second and the third graph are for the double cursor. For the double cursor, people can hit the target accurately because they have a green cursor to control. But once they learn the visual model rotation using a single cursor visual model rotation, you can see they learn much faster than the control group first time. Basically, we see savings. And even sometimes the double cursor moving in the opposite direction, you see the same effect. All right. Then the other thing I have to point out is during the double cursor movement, there is no implicit learning because I can use the catch trials, which is showing here. Where's my cursor? The catch trial basically shows people have no implicit learning, but they still exhibit saving effect. So my second prediction is the effect should be size invariant. So I give some jitter to the double cursor. And in fact, the double cursor moving in some random directions, and it never covers a 30-degree, which is the angle that's showing the saving effect. And we found the same saving effect, which means the size doesn't matter here. Then the third experiment, as I said, I use a novel task, which is called a mirror reversal. Mirror reversal means your hand moving in one direction, but the cursor moving in the mirror symmetrical direction. And here we have the same concept of visual model rotation, right? Then see how people react. Yes, people show saving again. And the other task is I change the visual model relation. I use so-called visual model gain. Visual model gain is another type of adaptation, but is different from rotation, different from directions. And I can show you the result. If you're using the mirror reversal, great, you can still show savings. But if you're using visual model gain, sorry, no savings, because the amplitude scaling is different from the direction rotation. So my take-home message is like this. So sensory motor adaptation is possible, the same effect is possible without adaptation itself. There is no implicit learning. There is no actually to acquire some explicit strategy. And the effect is area direction independent, is also independent from size, task, but it has to be involve a similar rotation concept. So in fact, in motor learning, feedback is always task relevant, but task irrelevant feedback can also be learned. I think our study is the first study to show so. And but in visual perceptual learning, in fact, people found simply expose people to task irrelevant feedback, they can learn it. And so that's my talk. People can learn sensory mode adaptation without actually learning it.
Okay, thank you very much, Kunlin. Again, if you... I'm muted. Oh, sorry. So again, if you have questions, put them in the Q&A or simply raise your hand and we'll put you on stage. Yeah, JJ, go. You had a question?
Yeah. Thanks, Kunlin. Very interesting talk. I was wondering, because so when you mentioned savings, is that for the explicit component or the implicit component as well? I mean, on the total adaptation, it was very clear, but I didn't know in your data which component showed saving.
Oh, sorry. I go through it too fast. It is an explicit component. You see the blue line?
Yeah.
They just learn. They got the strategy so quickly because they have the concept already.
Yeah, right. Yeah, but in your graphic, it was, there seems to be some difference in the implicit component, but I wasn't sure whether this was the case or not. Thanks.
Implicit component, I don't think so. Is more or less equivalent to the control group first learning.
Yeah. Okay, great. Thank you.
Sure.
Okay, thank you. Any more questions from the audience?
So maybe I can ask a second question while we're waiting. Then. So when you say forming a concept, or I mean it's vague. I mean, so getting a sort of strategy. How do you see that exactly implemented? What type of system is that linked to working memory? Is that linked to more the older literature on action observation? What do you think? What's the nature of that conceptual knowledge?
Yeah, that's a great question. Is for adaptation, people think it is more kind of low-level learning, right? And then we found that people can develop a strategy. But I think I go a step further, which conceptually is just because visual motor rotation is too simple. And once you know the cursor can deviate from your hand in different directions, you get this idea. Once you get this idea, it will impact your data. For example, I think this is a reminder to our field. For example, I just recently published a paper using the air clamp you just showed, and people learn air clamp, and then we test the saving effect. Yes, there is saving, but the air clamp is kind of implicit learning driven, right? But why there is saving? Then I realized because when we ask people to do air clamp, we ask them to ignore the cursor, ignore the task irrelevant feedback. Then they get an idea. They get the idea that the cursor could be moving in a different direction. They get the concept idea. That's why they show saving. My paper is published in Journal of Neurophysiology this year, but this just prompted me to think, oh, maybe there is some other learning component we just ignored before, right? Because for explicit strategy, we have to learn it trial by trial. We learn it gradually, but the concept, you can get it instantaneously. It just reminds us visual motor rotation is too simple for truly improving implicit adaptation.
Yeah. Okay. So.
I'm sorry.
Oh, sorry. There's one question from the audience. So Barbara.
Hey. I have two short questions. Like the learning via implicit feedback you show here, is it equivalent to if you would tell the subjects that what the visual motor rotation is? If you would tell them, oh, you have to aim 30-degree like counterclockwise, That's the first question. And the second question, like, I really like this idea of implicit and explicit feedback. But do you think this also applies to animals? Like if you look for visuomotor rotations in monkeys, for example, do you think, I mean, it's hard to test, right? But do you think they also have this explicit and implicit component?
Wow, that's a great question. So let me put it this way. My experimental paradigm is showing two cursors, right? But I think doing animal studies is too hard because our paradigm basically tell people to ignore one of the cursors, right? Even for arrow clamp, which is only showing a single cursor, we also need to tell people to ignore the cursor. But I believe in animal studies, you can test implicit adaptation very quickly because they don't understand the language, but once they control the hand cursor first, if they have a hand, then the hand cursor is moving in a irrelevant fashion, but that will introduce implicit learning because they still have cerebellum, right? So, but for my double cursor paradigm, it's very hard because we have to use some kind of cognitive control saying, okay, please ignore the second cursor. But the single cursor one should be applicable to animal studies.
Thanks.
Sure.
Katinka, you also had a question or comment?
Yeah, let's see what I can pose it very quickly. Thanks for fascinating work. I had to get my head around why the feedback was task irrelevant, but still relevant enough to learn from. And I wondered, is the key feature that it is movement that is correlated with your own movements? So would you still expect the same effects if the second cursor wasn't correlated with your own movements?
Oh, that's a great question. Yeah. It goes this way. In fact, that's something we can do next. If it's not temporarily coupled here, my guess is in certain window, you can still introduce implicit adaptation.
Okay.
But even though it's not time coupled, you can still get the concept because it's a spatial concept. So I will look at it in two different ways. Implicit learning really will be hampered by delay, saying the temporary delay, but the concept learning, no.
Yeah. Thanks.
Sure. Okay, thank you, everyone. So we're unfortunately running out of time. So thanks again, JJ, Katinka, and Guanlin for 3 very interesting talks and a very interactive and amazing discussion. Thanks also to the audience for the great questions, and thank you also to Ali, who's doing the back end here. And yes, enjoy the rest of the conference, and goodbye, everyone. Okay, we still have no attendees, and you're still downloading. So I think we can wait for another 1 or 2 minutes. Oh, there now people are coming. So we're still missing 1 speaker, so we'll wait for another 1 or 2 minutes before we start.
Okay.
Okay, wait, there's this way.
So
I was actually really looking forward to speaker number 1's talk.
Yeah, me too. It seemed pretty interesting.
Yeah.
Yeah, I don't know what happened, but so Smaranda, once you're ready, let me know, and then we
Let me see if this is okay. I can't download the photo of my lab, so I'm going to be that one person who does not thank her lab and does not appreciate her supervisor.
Well, you can say their name.
Yeah,
and thank them.
they're not
okay.
tuning in anyway.
Okay, so I think we should start now. So welcome everyone to another session here in Great NeuroMatch 3 conference. We'll have a traditional session here, so 12 minutes talks, 3 minutes questions. Nonetheless, if you have questions, put them in the Q&A. If we have more questions, then we have time. You can also take it offline. Unfortunately, we only have 2 speakers in this session, so it's Miranda and Anna. And we also have Alexandra, who's running the back end and solving all the technical problems. I'm Christoph Metzner. Will be the host of this session. And again, if you have questions, put them in the Q&A window. If you want to chat, use the chat function. Alexandra will also post some information how you can contact our speakers or recent papers, websites, etc. And yeah, let's start with our first talk. So our first speaker is Miranda Badea from the University of Hong Kong. She's a PhD student in Xiaodong Wang and Wujian Wu lab, and her talk is titled Modulating RPTP Sigma Promotes Functional Recovery in Experimental Models of Multiple Sclerosis. So Miranda, the floor is yours.
Okay. Thank you, Christoph. Let me see if I share here. All right. Oh, this isn't popping up. I think this is also too big. But in any case, thank you everyone who's joining us. As Christoph already mentioned, my title is something around modulating receptor PTP sigma, improves functional recovery in animal models of multiple sclerosis, which is apparently a secret according to my PPT. But one thing that I want to talk about is receptor PTP sigma to start with, which is a receptor for inhibitory molecules in the central nervous system. And this particular receptor binds to proteins called CSPGs. So in my experiments, I use a peptide that binds to this receptor and relieves CSPG-mediated inhibition. And it was designed in Professor Jerry Silver's lab at Case Northwestern. So the idea behind that is that in response to any form of neural injury, the central nervous system will secrete extracellular matrix proteins, particularly chondroitin sulfate proteoglycans. So these proteins have been shown to inhibit myelin repair, and they are also inhibitory for axonal growth. That's the main focus point in research so far. Now, what's recently been shown is that in active demyelinating lesions, such as those that occur in multiple sclerosis, these CSPGs can accumulate around the lesion sites, and they form a physical barrier for myelating oligodendrocyte precursor cells, or for other cells that may attempt to access the demyelinating lesions and perform their normal functions. So knowing that CSPGs inhibit both axonal regeneration via their receptor PTP sigma, and that this also occurs via PTP sigma in the case of oligodendrocyte outgrowth and remyelination, I considered whether alleviating the presence of CSPGs or their interaction and downstream signaling via PTP sigma may actually relieve this inhibitory environment around demyelinating lesions and permits access for the cells that are required for regeneration. In this case, initially they would be myelinating oligodendrocytes, but potentially also astrocytes and microglia with a role in phagocytosing and clearing out the debris. Okay. So this led me to my hypothesis that perhaps modulating receptor PTP sigma can promote regeneration, attenuate pathology, and lead to functional recovery in animal models of multiple sclerosis. Now, the characteristic features of these models is typically demyelination, because multiple sclerosis is the most common autoimmune demyelinating disorder in humans, and is characterized by demyelination, neuroinflammation, increased gliosis or activated reactive glia, and ultimately leading to axonal degeneration. Now, I looked at all 4 of these aspects in my project, but due to time constraints, I'm only going to address a few today. So the model I use is called chronic EAE, experimental autoimmune encephalomyelitis. I would say this is the gold standard for typical MS research. And it involves injecting, in my case, it's myelin oligodendrocyte glycoprotein incomplete Freud's adjuvant into mice. Which should induce an activated T cell response, ultimately generating MS-like inflammation in the central nervous system. And if anyone's interested, that's the dose that I use, because you can get quite a lot of variability based on the concentrations or amounts you use. So following induction, I would observe symptoms, and starting from peak of disease, I would inject these mice either with a vehicle solution or with the peptide I previously mentioned. Because these mice develop central nervous system inflammation and ultimately paralysis, I scored them for motor function, as you would see on the left side of the screen. So the mice I chose for my experiments, they all reached a score of 4, in which they had both hind limbs paralyzed. So I'll start, if this will change screens. Okay, oh, this isn't. So as I mentioned before, starting from disease peak, I would inject my mice with ISP, and I'll show you a comparison of what the mice look like at the end, right before sacrifice, the ones that have only received vehicle. As you can see, they cannot move their hind limbs, and they're quite sorry. This is the inflammation site you would observe in the back, where I injected complete Freud's adjuvant, and this is a mouse that received ISP treatment. You can still see that its tail has not recovered, but it can use both hind limbs. So this was a consistent trend amongst the animals that received ISP treatment, and this started around 1 to 2 weeks after the beginning of treatment, which was at the disease peak. I sacrificed these animals and then looked at the ultrastructural level, and here is just semi-thin sections, so 1 micron thick sections stained with toluidine blue, where you can clearly see that there's a change in the quality of the tissue. The most aggressively affected regions in the lumbar spinal cord are the ones highlighted on the right side. And so those would be both ventral and dorsal regions close to blood vessels, so primarily perivascular cuffs. And as you'll see, the axons that are normally there quite well structured are either lacking or severely damaged. And there are regions with a lot of dark spots that are either degenerating myelin or infiltrated cells. I looked at neurofilament loss because, as I mentioned, axonal degeneration is the ultimate endpoint for these models and could explain the severe hind limb paralysis. And what I saw is that animals that had received ISP treatment had significantly less neurofilament loss. So they had a lot more integrity along their axonal tracts in the white matter. And I measured that as a percentage of the surface area based on signal intensity of neurofilament staining. And compared to control, as you can still see, there's quite a difference. However, compared to the vehicle animals, ISP-treated mice had significantly less loss. Interestingly, these areas of neurofilament loss co-localized with areas of increased expression of chondroitin sulfate proteoglycans, which, as I mentioned before, are inhibitory to regeneration. So this would support my hypothesis that they accumulate around demyelinating region sites and may inhibit regeneration. Interestingly, these regions are also rich for APP, which can sometimes be used as a marker for degeneration. I then looked at immune cell infiltration because, as I mentioned, this is a autoimmune model. And I found that ISP-treated mice had significantly fewer CD3 or CD4 positive cells in the tissue that I imaged. Even though T cells do not express PTP sigma as a receptor, it's possible that due to reduced neuroinflammation, there was a decreased presence, or there was decreased attraction for T cells to infiltrate around the perivascular cuffs. I then looked at the resident immune cells of the central nervous system, microglia, and I found that there was significantly more microglia activation in the EAE mice that only received vehicle as compared to ISP-treated mice. So it's possible, and these cells do express PTP sigma, so it's possible that ISP helped their own, interfered in the interaction between microglia and CSPGs to modulate their inhibition in response to these molecules. And I found that these regions of microglia-rich lesion sites that also colocalize with CSPGs intensely are also found in areas where you cannot detect myelin to the same level as you would in controls or even ISP-treated mice. So again, this treatment, this simple peptide that targets the receptor PTP sigma, could alleviate this particular response. So looking at the cell level, because I was interested to see why or how microglia respond to CSPGs, I coated microglia that I obtained from newborn pups. I seeded them on cover slips coated with aggregate, and I observed that CSPGs can inhibit their attachment and their spreading. And this happens in a dose-dependent manner. So the more CSPGs would accumulate in response to injury over time, the more microglia activity might be inhibited. While they do not die, they cluster, and this could potentially lead to more activation. Then I switched to a second model that does not have an autoimmune component. So I use cuprizone-induced demyelination. I'm actually missing Christoph. I might miss my cues of going over time. I'm sorry. So I use cuprizone-induced demyelination, in which I fed my mice with 0.3% cuprizone every day in their chow. And I also injected them with rapamycin as a supplement to make sure that we can separate demyelination and remyelination stages. This model should cause systemic demyelination, particularly in the brain, with progressive recovery when cuprizone is removed from chow. So I had groups of animals that only received the demyelinating chow and rapamycin. I had age-matched naive controls that never received cuprizone and they were just on regular food. And then I had mice that underwent the demyelination model and then received either vehicle or ISP treatment. And what I found interesting is that despite these animals not having severe motor symptoms, as you can see here, all animals recovered and did not have issues on the rotarod, and they all traveled the same in an open field test. They did show signs of anxiety-like behavior, which coupled with a fear conditioning paradigm demonstrated that they might have cognitive issues in response to the demyelination model. And these cognitive issues were partially resolved by ISP treatment, as you can see with ISP in mice showing similar freezing times as the naive treated animals, untreated animals. However, there was a difference between cued and contextual fear conditioning, which might be related to the areas of the brain that are involved in these two types of memory formation acquisition. So I sacrificed these mice and I stained them for myelin to see what exactly the situation was like in the regions that are involved in these processes. And I found that cuprizone feeding induced severe demyelination in the corpus callosum, partially in the hippocampus, but extremely at the level of the amygdala, which would be responsible for the memory changes observed. And very interestingly, ISP treatment significantly increased the myelin levels in all of these three regions, with the hippocampus and amygdala showing hypermyelination in response to treatment. So overall, what my experiments would say is that these paralyzed mice, or mice with cognitive issues that showed reactive astrogliosis, activated microglia, and possibly T-cell activation in response to the CSPG presence, once they received ISP treatment, both astrocytes, microglia, well, this phase not as much astrocytes, but the cells that participate in all of these pathological processes were restored to their normal function, and mice could recover their normal cognitive and motor function. And that's it. I would like to thank my supervisor. I would hope he's here, but probably not in the chat. And everyone who helped me throughout my PhD, and all my friends who provided extreme moral support, particularly during the last 2 years when we were confronted with both extreme social unrest and tear gas at every street corner in Hong Kong, and then with the pandemic that kind of set us back in all possible ways. And thank you very much to everyone attending and all the other participants and panelists. I really look forward to hearing your talks. And please let me know if you have any questions. Thank you.
Okay, thank you very much, Smyrna. So because we only have 2 speakers here, we do have more time for questions, I guess. And Anna, I saw you already posted 2 in the chat. Maybe you just want to ask them yourself.
Yeah. So the first question was just, I was thinking about it as you were going on. Did you shave the mice for surgery, or was that hair loss a result of the EAE?
So I have to shave, yes. So I shave, and I use also hair removal cream, because in response to the CFA, they can form lesions, and actually a bit like craters in their skin because of how strong their immune response is to the MOG injection. That's why they also have these massive bumps. That's literally the injection.
Oh, wow. Interesting. And then I had a question on your method for quantitating the IBA1. Just curious, because I quantitate IBA1 too, and I'm always curious how other labs do it when you had the sections of tissue. How do you guys quantitate that?
So I would actually do the whole scan image. I do that over confocal with the TileScan Z-Stack. And then with the Zen software that we have from the confocal, measure the total surface area that the microglia occupy within the white matter. I didn't measure intensity as much because I didn't feel like it's a strong enough indicator of activation in that sense. I would have looked at other markers, I guess.
Yeah, exactly. We had similar issues when we were trying to figure out how to quantitate IBA1 and what to do. And so instead of doing anything at all standardized, we individually had to count each one that had morphology. And so your way sounds like it might be slightly less time intensive.
I couldn't. I honestly wanted to try doing that. I feel like that would have been much more accurate, but mine are more amoeboid-like. So the morphology is indistinguishable one from the other.
Yeah, I imagine it'd be really difficult. And then just one kind of general broad overview question, if I can. I don't want to dominate the question and answer. But wondering, I mean, like, you know, in neural injury models, right, it's always kind of a balance of like there's an initial immune response and you want that because there's beneficial aspects of it. But then if it goes on too long, that's really kind of when it becomes detrimental. And so I'm wondering what the beneficial effects of CSPG could be. Like, why does the body release them in the first place? Is there a beneficial?
That's a very contested topic. As someone at quite close to the bottom of the food chain, I don't want to step on any toes, but as far as I know, there's two sides to this. There's actually a good paper published, I think, 2015, showing that it's protective. The scar formation is protective in response to injury, but there's quite a few people that would contest it. So maybe I would think that it makes sense if they restrict the injury site temporarily so that there's not constant progression of whatever is happening, the injury, the demyelinating lesion, and so forth. But then the problem is with degrading all of the inhibitory molecules that are present, not just CSPGs, but even myelin debris is inhibitory. There's a lot of things that can cause issues there. I've actually upset people
with my answer to this question previously.
Oh,
So it's very loaded.
yeah,
Sorry.
yeah.
So-
I'm just wondering because we get similar questions from reviewers and stuff, and because we say, oh, we want to, get rid of the glial scarring, and then some people push back and say, well, the glial scarring's there for a reason. It's good. You can't get rid of it. So I know it's interesting, but I didn't know specifically for CSPG if it was the same. Yeah.
Definitely is, yes.
So we do have more questions from the audience. So, well, Sophia is, I'm just a first-year PhD student, so this might not be a good question, but can you explain a bit more about the role of CSPG in relation to your intervention? So I know that generally they're inhibitory, so are you trying to suppress their expression?
So for specifics, let me go back to the first slide. Actually, I think I put that in the wrong order. So I don't try to affect their expression. There's other compounds that inhibit CSPG expression, but as Anna mentioned, I think they actually serve a purpose at the beginning of pathology. So what I try to do is using this peptide is to inhibit downstream signaling after the initial onset of disease. So it's more of hijacking the signaling pathway through this one particular receptor, but there's other receptors that also bind to CSPG, so it's not a 100% canceling their signaling. It's partially blocking it, I'd say perhaps like a 50 to 60%.
Okay,
I'm not sure.
One last quick question, because then we can also have some more time for Anna later. I'm gonna divide that equally. So Sean asks what the reason was behind using female mice. So I guess the idea this.
Oh, okay.
Why not male mice? Yeah, and if you've checked for cycle and everything.
Yes. So for female mice, why I chose them is because they respond much stronger to the EAE model. That's a general, quite known aspect of the model itself. Apparently, women are more likely to develop autoimmune disease, potentially for a hormone-related reason, but for mice, we don't really know why. It's just that my success rate would have been much higher with females than with male mice. And for anxiety-like behavior, I did not check the estrus cycle for my mice, but all of my mice are all female and kept together. So I think over time they might have matched. I'm not sure if for mice it goes like it does for women, where we would tend to cycle together. I think you would know more about that. But yeah, it was quite a consistent reaction.
Okay, well, thanks again for a very nice talk and also a nice discussion. So our second speaker in this session now is Anna Jaratana from Rutgers State University. Well, actually, she's now moved to Zurich to do a postdoc, but she presents her PhD work at Rutgers. And the title of her talk is The Role of Genetic Polymorphisms in a Mouse Model of Traumatic Brain Injury and Personalized Treatment Approaches. Anna, the floor is yours.
Great. All right. So, all right. Thanks everyone for coming here, and thanks again for the organizers for putting this on. It's a big undertaking. So the title of my talk, again, was just said, so I don't need to repeat it. It's a mouthful. But I'm Anna Jaratana, and I did my PhD in the lab of Dr. Janet Alder at Rutgers in the Department of Neuroscience. And so I'll be talking a bit about that work today. So basically, traumatic brain injury is a disruption of normal brain function that can be caused by a bump, a blow, or a jolt to the head. And in the United States in 2013, there were 2.8 TBI-related emergency department visits. There were 50,000 TBI-related deaths. And there were 5.3 million people with a TBI-related permanent disability. And so when someone has a TBI, they come to the emergency room. It's classified as either mild, moderate, or severe. And so for this project, we really wanted to focus on mild TBI. And the reason for this is that mild TBI accounts for 75 to 90% of the TBIs that occur. It tends to be underreported and undertreated. And it's often ignored, which results in a premature return to work or play And when this happens, there are repeated mild TBIs, which can have significant long-term consequences. And so physicians have long noticed that certain people recover better after TBI than others. Two people can have very similar injuries and have very different outcomes. And our question was, why? Why is this? And so my hypothesis was that there are differences in genetic polymorphisms that play a role in the variable outcomes that occur. And I hoped that by understanding the mechanism through which the variable outcomes occur, that then I could develop targeted therapies that would rescue the vulnerable genotype. And so we decided to look at SNPs at two different important proteins in the brain. The first of these is BDNF. BDNF has a SNP site at the 66th position in the prodomain. And the results from this are in our paper that was published in Frontiers last year, if you're interested. And then we also looked at ApoE, which has SNP sites in two positions, the 112 and 158 position. And these results were just accepted in scientific reports, so that'll be out soon again, if you're interested. And so our overall experimental scheme was to look at the VAL-MET and the VAL-VAL SNPs for BDNF and the ApoE4 and ApoE3 SNPs for ApoE. And we submitted the mice to these repeated mild TBI conditions or a sham control condition. Then we looked at the differential outcomes, and then we tried to develop personalized therapies for the differential outcomes that we saw. And so we found that the VAL-MET and the ApoE4 injured mice have a larger injury volume at 21 days post-injury. So you can see here we did MRI scans of the brain, and these are the representative groups that I'll be comparing. So I'll draw your eye to the important comparisons. We have the brain injury volume site here for the VAL66-MET injured mice, and the important comparisons are to compared to its sham. So you can see it has a larger injury volume than its sham control. And then again to the other injured genotype, so to the VAL-VAL injured genotype. And you can see there's significantly bigger injury volume in the VAL-MET injured mouse. And then again for the ApoE4 injured mice, you want to compare to their control sham, and then again to the other injured genotype. And so we quantitated this. And so for these graphs, again, I'll draw your eyes to the important comparison. We found a significant difference between the injured mice for the BDNF and their sham controls, and again for the ApoE and their sham controls, and then importantly between the two injured genotypes. And so next we wanted to figure out what was happening kind of behind the scenes, and so we decided to look at immunohistochemistry markers. So for apoptosis, we looked at cleave caspase 3. For neurodegeneration, we looked at fluorojade-C. For activated microglia, we looked at IBA1. For phosphorylated tau, we looked at AT8. And for astrogliosis, we looked at GFAP. And these are all representative images from the BDNF mice, and we quantitated them. The graphs are in the paper. If you're interested, I won't show them here for time's sake. Basically, a summary chart of what we looked at. We looked at the 1 and 21 day time point and the cortex and the hippocampus. And basically anywhere there's a green check mark, that means we saw a significant difference where the VAL-MET injured mice had more than the VAL-VAL injured mice. And so kind of to summarize it, we basically saw in the schematic in the bottom that cell death, neurodegeneration, phosphorylated tau, and activated microglia were all significantly higher than the VAL-VAL injured mice at the 1 day time point. By the 21 day time point, we no longer see that difference in apoptotic cell death, but we do see a significant difference in astrogliosis, which makes sense if you think about the time course of injury. Apoptosis tends to be a more rapid response, whereas astrogliosis can take a longer time to build up the glial scarring formation that occurs. And so next for the ApoE mice, we looked at, again, apoptosis. We looked at neurodegeneration, we looked at activated microglia, and we looked at phosphorylated tau. And you can see represented images here for the ApoE3 and ApoE4 injured mice. And again, a summary table of what we found for these. Very similar thought here, whereas the green check mark represents a place where the ApoE4 injured mice had significantly more of the detrimental marker than the ApoE3 injured mice. And so what we found here was that for cell death, neurodegeneration, and activated microglia, the ApoE4 injured mice had higher levels at 1 day post-injury, and that by 21 day post-injury, there was a significant difference in phosphorylated tau that then persisted. So we wanted to figure out what some of the possible mechanisms could be for this. And so in order to do this, we decided to use Western blot analysis to look at levels of neurotrophic factors. And in particular, we looked at BDNF. And so some samples of our Western blot analysis here. If anyone's familiar with BDNF, I'd be happy to go more into detail, but I'll keep it brief here. Basically looked at mature BDNF over time. And so mature BDNF or total BDNF, and looked at the relative differences between the different forms of the neurotrophic factor. And again, I'll draw your eye to the important comparisons. We see that there's a significant increase in BDNF in the VAL injured mice, and the VAL-MET injured mice have significantly less total BDNF. And in the same case, the ApoE 3 injured mice tend to have significantly more mature BDNF than the ApoE 4 injured mice. So it's not exactly apples to oranges, but basically mature BDNF is beneficial, and so having more of it is a good thing. And so that's what we show that the ApoE 3 and the VAL injured mice have relative to the vulnerable genotypes. And so given this, we wanted to develop targeted treatment options for these two mice and these two genotypes. So for BDNF, we decided to focus on that 21-day time point, which if you remember from the chart was kind of that more affected time point. And so in order to do this, we used an AAV BDNF, which was to increase mature BDNF levels. And proof of concept showed that when we did this, we were able to increase mature BDNF levels, and we were able to activate the beneficial TrkB signaling pathway. And for ApoE 4, we decided to focus at that 1-day time point, because if you remember, that was the one that was kind of more affected in these mice. And so we used Briastatin 1, which is a drug that's currently in clinical trials for Alzheimer's disease. And it acts to mimic ApoE 3 actions, and it activates PKC epsilon. And so those were the two targeted treatments that we decided to focus on. And we saw that the personalized treatment in the VAL-MET injured mice was able to reduce astrogliosis and activated microglia at 21 days. So again, I'll draw your eye to the important comparisons. Here we found that when we compared the VAL-MET injured to the VAL injured, as we previously had done, there was a significant difference. But then by giving the Briastatin treatment, we were able to reduce this, these elevated levels of GFAP for astrogliosis. And again, the same thing for activated microglia. We see this difference between the genotypes, and that's ameliorated by the treatment that we give with the AAV BDNF. And then for the ApoE 4 mice, we give this treatment, and we looked at Florajade and activated microglia, which if you remember were the things that we found that were most affected previously. And so when we give this treatment, we're able to reduce levels in both neurodegeneration and activated microglia. And so next we wanted to figure out how this was affecting cognitive function and behavior in the mice. So we did the Morris-Water maze test. And so the Morris-Water maze test investigates spatial learning, and then the Morris-Water maze probe investigates spatial memory. So you can see here we place the mouse in this tub with cues that are in the room, and the mice have to find their way to a hidden platform, which is right here by the square. And so the first day they don't really know where it is, they're exploring, and by the second, third, fourth day they're getting better at finding where it is. So as by the 6th day, they basically are put right in the tub and they learn that it's there. And this is what happens when you can see that they're actively learning over time. And in the probe test, the day after, we remove the platform, and mice with an intact memory will swim in the area where the platform had been, whereas mice with impaired memory will swim kind of all over the tub and they won't have learned where it is. And so what we found was that the VAL-MET injured mice have impaired learning and memory, and that our personalized treatment was able to improve learning. So I'll draw your eye to the comparison here. The dark green bar is the VAL-MET injured mice. So they have a longer latency to platform, which means it takes them longer to find the platform over time. So they've reduced learning relative to the dark blue bar, which is the VAL injured control. And then for the time spent in the northeast quadrant, which analyzes memory, they spend less time there. So they have impaired memory for the dark green relative to the dark blue. And then when we give the treatment, which is shown by the bright green, we're able to improve learning, although we do not see a significant improvement in memory in this case. And then for the ApoE 4 mice, it follows a similar platform. So the ApoE 4 mice have impaired learning and memory, and our personalized treatment for them was able to improve both learning and memory. So again, the comparison of the dark red bar to the dark orange bar in both of these cases. And then when we give the drug, which is the pink bar, we're able to decrease that latency to platform, improve learning, and increase the time spent in the northeast quadrant or the memory. And so just to summarize what I've kind of represented and what we did in our project, some of this I've talked about, some of it I haven't, but everything on the screen is what we found to be a risk factor for the BDNF Val-Met injured mice after our treatment paradigm. And then when we give the AAV BDNF treatment, we're able to improve the activated microglia, the astrogliosis, the impaired learning and memory, and also lower BDNF levels and increase pro-BDNF signaling, which I didn't talk about here, but happy to answer questions about. And ApoE4, we found was a risk factor after TBI for everything on the screen here. And when we give our bryostatin treatment, we're able to improve the increase in neurodegeneration that we see, the activated microglia, the impaired learning and memory, the impaired fine motor balance, and lower BDNF levels. So some future directions for our research is just to kind of delve more deeply into the rescue mechanism, to look more into different signaling that might be happening with the neurotrophic factors, to look at TrkB signaling for the BDNF paper and for the ApoE to determine if BDNF and TrkB are important in the bryostatin treatment and if they're necessary for the beneficial effects that we see or not. And then also to look at longer term time points. Repeated mild TBI tends to have long-term consequences. So it'd be very interesting to look kind of further out to see what those more significant effects could be. And then also looking at combined genetic effects. It's very easy in mice to pinpoint one specific gene that could be a problem. However, in people, that's probably not the case. It's probably multiple genes that affect each other. So kind of looking at combined genetics from the candidate genes and also using kind of big data to do an unbiased approach to kind of scan the genome. So again, I'd like to thank my lab, my mentor, Dr. Janet Alder, Dr. Smita Thakuravaria, all the students who helped me with all of this, all my funding, and again, the organizers for having me here to talk about this today. Thank you.
Thank you very much, Anna. So if you have questions, put them in the Q&A. I have to say I don't know nearly enough about the topic to ask a smart question, but maybe you can, in a couple of sentences, put this into the context of treatment of TBI in humans. So yeah, what can we expect from that? Can you translate it easily, or yeah?
I mean, yeah, so that's the hope, right? So like now the problem is there's no treatment for TBI, there's just supportive care. And I think the problem is that, it's this balance of the immune system, right? Like, so they've tried immunosuppressants, but you need some immune system response to help. And so a lot of it is with timing and things like that. So I really think that it's by finding kind of genetic vulnerabilities that will help us figure out, okay, well, some people are fine. Why is that the case? And if we can boost that in people who are vulnerable, then maybe we can let kind of the immune system take its natural course and do what it needs to do while just kind of supporting it and helping it to do that. And I think that's the key. So like, for example, bryostatin is in clinical trials. I haven't followed it too recently, but it's specifically been shown to help for ApoE4 carriers. So it may not have any beneficial effects for ApoE3 carriers, but translationally, you could think about this as, you get your genotyping panel, you have a TBI and you come in and they say, oh, you're a risk factor for these like 3 genes, we can help support you in this way. Versus now it's like, okay, well, we can give you, rest and relaxation and dexamethasone or, whatever it is. And so it would be helpful in order to kind of just say, okay, we can quantitate you into risk strategies and then give you a specific drug that might help you. I mean, also another thing it could be used at is just prevention, right? So even if you can't come up with a therapy yet, you could, give people the option to know, and then maybe their kid doesn't play a contact sport, maybe they play tennis. It's just, it allows you to make individual risk choices as well.
Okay, yeah, thank you very much. Any more questions?
I actually have a question related to the behavior tests, to the the water maze one. I was wondering if you did any control tests for their motor behavior. Perhaps the time it took for them to reach the platform was because of any motor dysfunction they suffered. It seemed like the motor cortex might also be injured. Yeah.
Yes, great question. We did, because we're right over the somatosensory cortex. And so we use as a baseline, an exclusion criteria is mice that have significant impairment, so much that they take longer to get to. Their swim speed we look at. So if their swim speed is too slow, then they have a significant motor impairment. And then also the distance that they travel as well. Yeah, so we use that as a control, because otherwise, you're exactly right. It would be hard to tease apart that, because we also do the Rotor test, and we've done different fine motor tests and things like that. So we do see some fine motor differences, but we really don't see kind of like the gross, that they can't get to where they need to go, so that you couldn't test, you know, learning and memory because of the motor.
That's great.
Yeah.
Okay, oh, yeah, go ahead.
Oh, sorry, I had
one
more.
1 or 2 minutes, yeah.
Sorry, just because I'm not very familiar with BDNF. So is it normally overexpressed in response to your injury model? It seemed like the animals that had an overexpression of BDNF were a particular, were only the ones that had the proper TBI, not sham.
Yes, exactly. So it's a really complicated story, and it's really hard to get into, because on the other hand, our lab has done studies that are just on BDNF. So it's secreted as a pro-BDNF, the pro-neurotrophic factor, and then it's cleaved to become mature BDNF. And pro-BDNF and mature BDNF have two very different actions. So pro-BDNF kind of activates a detrimental cell death pathway, whereas mature BDNF activates a neurotrophic regeneration pathway. And of course, it's not as simple as that, because it never is. But basically what we've shown is that after TBI, BDNF is upregulated, but there's some slowdown, and nobody really knows exactly what it is yet, between the pro-BDNF being cleaved into mature BDNF. And so if you have a significant increase of pro-BDNF, that can be really detrimental after cell injury. So we've done other studies where we're looking at if you give, for example, they give TPA after strokes, and that cleaves pro-BDNF into mature BDNF. So we've done studies where we've given TPA after injury to help improve that along, so you have less pro-BDNF and more mature BDNF. So one of the things we looked at here that I didn't want to go too far into, but basically is that we want to see an increase in mature BDNF, not just an increase in pro-BDNF, because that would be detrimental. And so we also looked at the ratio of pro to mature BDNF, and specifically for the ApoE4 mice, we saw significantly more pro-BDNF than mature BDNF. So we looked at the ratio. So that's one of the things that's also happening with them. Yeah, but it's a great question, because it's kind of not as simple as more is better. It's kind of more of which and more of this, and how to calibrate it.
And when.
So, exactly, yeah.
Okay. Thank you.
Yep.
Okay, thanks again, Anna and Miranda, for very nice talks and a nice discussion. Yeah, so enjoy the rest of the conference. And
thank you very much.
See you, everyone. Goodbye. Yes, so if you, as always, if you have questions, put them in the Q&A. You can also use the chat to talk to each other. And yes, we have a traditional session, so 12 minutes talks, 3 minutes questions. Nevertheless, feel free to put as many questions in as you'd like. The speakers can also answer them offline then. We will also post some information on the speakers' websites, papers, Twitter handles in the chat, so feel free to look at them and contact them, follow them. Yes, so let's start with our first speaker. So our first speaker is Cameron Hassal from the University of Oxford. He's a postdoc in the Lawrence Hunt Lab, and his talk is titled Regression-Based EEG Analysis Reveals Temporally Scaled Signals During Interval Timing. So Cameron, the floor is yours.
Well, thank you very much for that introduction, and thanks everyone for coming to my talk. I'm very pleased to present today results showing temporally scaled signals in EEG. This work was done in collaboration with Jack Harley, a former master's student, and was supervised by Niels Koling and Lawrence Hunt. So action and perception in the real world require flexible timing. For example, the same piece of music can be played quickly, presto, or slowly, largo, and everything in between. We can also recognize the same piece of music played at different tempos. But how do we do this? Several models of timing have been proposed, and one in particular focuses on our ability to produce the same action over different timescales. That model is temporal scaling. which arises out of a recent animal work examining neural activity during interval timing. On the left here, you can see a monkey that is cued to produce either a short or long interval. On the right, you can see the response profiles of several neurons. Most of the recorded neurons in medial frontal cortex scaled their response to the duration of the timed interval. That is, the same response profile was stretched or compressed, depending on the interval to be timed. Contrast this with fixed time components, that is, activity that occurs over a fixed time scale, like these responses over here. The authors concluded that flexible motor timing relies on the speeding up or slowing down of a common network within MFC. Our goal was to determine whether or not these fixed time and scaled time responses could be dissociated at the scalp. This is challenging for two reasons. First, activity in the cortex is mixed at the scalp. A conventional EEG analysis, the method of event-related potentials, or ERPs, can only account for fixed time components. What I mean by that is that conventional averaging procedures assume that neural activity progresses over a fixed timescale. For example, you can see response-locked ERPs related to interval timing. Differences are apparent in the ramping activity, however, these signals share the same fixed timescale. Thus, it is unclear whether they contain any scaled components. In order to explore unmixing procedures, we simulated EEG in an interval timing task. In this task, participants see a cue, wait for a target duration, then respond at one of three target times, either short, medium, or long. We simulated two fixed time components, one for the cue, one for the response. We also simulated a scaled time component which spanned the delay period between cue and response. We summed these components together with some noise, and hopefully you can see in the resulting signal, components are now mixed. To illustrate what the results might look like in a conventional ERP analysis, we constructed cue-locked and response-locked averages, shown on the right. As you can see, adding in a scaled time component resulted in ERP differences that are consistent with previous literature, that is, different degrees of ramping activity. Our goal, however, was to unmix scaled time and fixed time components in order to recover the ground truth signals. So how did we do that? We used a regression-based approach in which the continuous EEG is modeled as a linear combination of underlying components. In our case, time-lagged regressors represented the contribution of our signals around each event of interest. These are variables or stick functions, that is, ones at the time when the component contributes and zeros otherwise. For much more information on this approach, I recommend checking out the Unfold toolbox. As you can see, we've included two fixed time regressors, one for the cue and one for the response. We then added a scaled time regressor whose contribution spanned the interval between cue and response. The idea here is to estimate a single component that accounts for the delay activity across a variety of timed intervals. And one thing to note is that we can solve this equation at multiple sensors. Thus, it might be possible to generate a scaled time topography. We then applied this new regression approach to our simulated data, and we were able to recover the original components, that is, fixed time components for cue and response, and a single scaled time component. You'll notice that the horizontal axis of the scaled component is not time per se, but rather the proportion of the interval. That's because this scaled component is spanning over multiple durations. So with this technique in mind, we conducted an interval timing experiment and collected some real EEG. In our temporal production task, participants learned to produce one of three target intervals, short, medium, or long. Blocks were preceded by a metronome to indicate the target interval. In the temporal judgment task, participants listened to a computer-produced interval, then judged the interval as correct or not. In the production task, we will be modeling fixed time components for the cue and the response, and a scaled component spanning the delay period. Likewise, in the judgment task, we'll be modeling the cue and probe as fixed components, and then a third component that spans this the gap between them. These plots show that participants were able to do the tasks quite well. They came close to hitting their targets in the production task, as indicated by the mean response times on the left. They were also able to correctly judge computer-produced interval, as indicated by the mean likelihood of making a correct judgment on the right. To align our results with previous EEG work, we constructed conventional ERPs, which interval-dependent ramping activity locked to the end of the interval, locked to the response. But these are fixed time components. Our regression-based approach revealed a scaled time component in both tasks that was distinct from the preceding and following fixed time components. Thus, we have successfully unmixed scaled time and fixed time components at the scalp. The signal appears to be consistent across tasks, both in the shape of the waveform and in the scalp topographies. And note that as before, the horizontal axis for our scaled component is not in absolute time, but rather relative time. That is the proportion of the interval that has been covered. We further tested our analysis procedure on open data from this paper by Bresca and Duell, in which they examined rhythm-based expectations. The goal of their participants was to respond to a visual target that appeared sometime after a visual cue. When we included both the warning light and the target light as fixed time components and a scaled component as before, we again revealed a temporally scaled signal. This was a successful test of our method because although the fixed time components differed from our task, that is visual versus auditory, the scaled time component was remarkably similar in shape and location. So one question we had was, how does this temporally scaled signal relate to behavior? To answer this question, we used principal components analysis, or PCA. You may have heard of PCA as a dimensionality reduction technique, but what you might not know is that principal components often represent the temporal derivatives of a signal and may be related to component latency. This appeared to be the case with our data. When we ran a PCA on epoch data, the first principal component, PC1, matched the shape of our scaled time signal, shown in green. The second principal component, PC2, resembled the temporal derivative of this signal, which is supposedly related to the latency of the overall signal. To confirm this, we basically added in more or less of PC2 to PC1, so we sort of reconstructed the data in order to see what adding more or less of PC2 does to the signal. And indeed, it appears to shift the latency of the signal earlier or later in time. Thus, we considered PC2 scores to be a trial-by-trial index of component latency. We then related PC2 scores to trial reaction time data by binning responses as early, on time, or late within each of our conditions. Collapsing across conditions, we see that earlier responses tended to have greater PC2 scores compared to later responses. Thus, the peak, or the latency, of our scale time component was related to response time. The earlier it peaks, the earlier you respond. In conclusion, we've shown that temporally scaled signals can be recovered via regression-based analysis. In particular, we were able to unmix fixed time from scaled time components in EEG. In the future, we think it would be exciting to look for scaled time components associated with other processes that are known to unfold over different or varying timescales, including sequential actions, such as in music or speech, as well as decision-making, for example, evidence accumulation. So I would like to end by thanking my collaborators on the project, as well as all the agencies who funded the work, And a big thanks to all the organizers of NeuroMatch. And thanks for your attention.
Thank you very much, Cameron. Yes, so if you have questions, put them in the Q&A window. So there's one question by Francesco Giosi, who asks, he's just asking for a paper to that references to this work. I think Alexandra has already put some information for that in the chat window.
Yeah, it's brand new work, so no paper or preprint yet, but stay tuned for that.
Okay. Do we have more questions? There's another one. It always takes a bit of time to type them. So Dongil Chung says, very interesting talk. Did you have a chance to check if the same framework being applied to decision-making problems where individuals may get to solve in different timing? Or where individuals may get to solve in different timing, I guess.
Would you mind repeating that question?
Yeah, so I think the question is if you've applied your framework to decision-making problems,
right?
Where they solve problems with different timing.
Yeah, no, that I mentioned that at the end that we're interested in this. Our lab also does work with evidence accumulation using a similar regression-based framework. So far, we haven't looked for scaled components within that framework. We're still working with fixed time components. But one of the sort of motivations for this work, when you look at these traditional ERPs, you can sort of see that one possibility is that these signals are actually being scaled. So you have a hunch that there might be some scaling going on there. So we definitely see a similar type of activity in evidence accumulation.
Okay, and one last short questions by Joe Arahua. Did you see any differences in your results for the regression analysis depending on the type of referencing used, especially in the topographic maps?
Good question. I mean, I would expect that as with conventional analysis, the use of choice of reference is going to impact the topographies. I haven't actually explicitly looked at that, so we use an average mastoid reference to be in line with where they're looking at these ramping signals. But certainly, is something that would be interesting to add, just to kind of confirm that we are dealing with something that is like a regular topography and sort of behaves like a regular topography. Question.
Okay, well, thanks again, Cameron, for this very nice talk. And we'll move on to our next speaker. Our next speaker is Priyanka Ghosh from the National Brain Research Center in India. And she's a PhD student in the lab of Arpan Banerjee. And her talk is titled Spectral Features of an Oddball Task Across Modalities. So take it away, Priyanka.
Hi, I hope my screen is visible to everyone.
Yes, we can see your screen.
Okay. Good evening, everyone. In India, it's an evening time. So yeah, good evening from my end. And thank you so much for the introduction. The title of my presentation is Spectral Features of an Oddball Task Across Modalities. So before I start on with the presentation, I would just like to brief about what do we actually mean by an oddball. So something like salient distractor, like suppose you guys were listening to me while you were attending the talk and the phone kind of rings out. So you guys listening to me, that's because part of your goal-directed attention, and it's the frontal regions of your brain kind of directing your sensory regions, your eyes or the ears to kind of listen to me. That's the goal while you're doing the task, as opposed to while the phone rings, that's a sensory stimulus which is salient enough that kind of captures your attention and reorients your attention from the goal-directed task to kind of attend to the sensory stimulus, which is your phone bell right now. So that is something which is attended by a process known as bottom-up attention. Why so? Because it's the sensory regions which kind of attend to that stimulus first, and which is later figured out by the topmost hierarchy of brain regions, say the frontal or the parietal regions, to kind of go and attend to the like where is the what's the source of signal, or what's whose phone exactly is it that rung, things like that. So yeah, so the ability to detect and orient attention towards salient significant changes around us is critical for survival. For example, a populous country like India, that's like something that we need to perform on an everyday basis. For example, while you're driving a car, that's like a goal-directed attention. You have to judge, say, the distance with other vehicles on the road and other traffic signals, as opposed to a sensory stimulus, which is salient as a pedestrian, pedestrian crossing your car. That's something that requires immediate attention and effect, and can actually have devastating effects if you do not act on time. So while we need focused attention on any goal-directed behavior, the temporal component of attention becomes very important in conditions that demand rapid shift of attention, like a train brakes during driving upon seeing a pedestrian suddenly crossing the road. But do the underlying neural signatures involved in this kind of an attentional shift remain same when we look at a static versus a dynamic stimulus? So what do I mean by static stimulus? Something that's stationary, and a dynamic stimulus is something that is moving. For example, you have to identify your friend on the platform while you're traveling in a moving train. So versus when the train is not moving. So of course, there is a difference in which the brain kind of processes a stimulus that is moving versus a stimulus that is not moving or is stationary. So when the train is moving, it's definitely more tough to identify your friend on the platform, as opposed to when the train is stationary. So what we wanted to know is that what are the neural correlates of such saliency processing when it comes to shift of attention from moving versus a static stimuli. So just to give a broad structure of what's already there in the literature till now is, so attention, as they say, can be classified into two broad categories. One is top-down attention, which is goal-directed, as I already mentioned. So this goal-directed attention is mainly controlled by what is known as the dorsal attention network, which consists of areas in the brain which are intraparietal sulcus, the frontal eye fields, etc. On the other hand, there is a completely different kind of attention, which is bottom-up attention, which is mostly responsible which is mostly stimulus driven and is controlled by the ventral attention network, which consists of a region in the brain known as the temporoparietal junction, and mostly the areas of the lateral prefrontal cortex involving inferior frontal gyrus and middle frontal gyrus. So so this is just another picture that kind of tells us what is an oddball exactly, or what's a salient stimulus, something that has a pop-out effect, or I can say a salient effect, which kind of grabs your attention even though you do not intend to attend to it, because that's not a part of your goal. So to study how the brain kind of attends to these kind of salient stimuli or oddballs across modalities, we started on with the visual modality first, and kind of took up two stimuli which vary in their spatial and spatiotemporal components. So one was a static stimulus, which was stationary in nature, and one was a dynamic stimulus, which was moving in nature. So this is the experimental paradigm that we use to do EEG. And why we chose EEG is because the nature of such attentional shifts upon the occurrence of salient stimuli is something which is very transient and rapid. And because of the sluggishness of the hemodynamic response, fMRI does not have the capability to capture this kind of transientness. So that's why we and literature doesn't have much evidences from EEG to kind of capture what are the neural signatures that underlie such saliency processing. Yeah, so coming back to the dynamic stimulus, this was a simple random moving dots experiment where the participants had to tell the net direction of the moving dots. So our task was divided into 3 categories. Number 1, where there was no saliency introduced. So this is something, so yeah, in the no saliency condition, the coherence, so the coherence was fixed for all conditions. There were 3 conditions: no saliency condition, with saliency condition, and neutral conditions. So the task was to identify the net direction of moving dots. So the dots could either move in top, down, left or right direction. The participants had to identify the net direction and respond using the respective arrow keys on the keyboard. So by coherence of 0.6, what we mean is that there are 100 dots on the screen. There'll be 60 dots which will be moving in a particular direction, and the rest of the 40 dots will be moving randomly. So that's this condition. The condition for the with saliency condition, the task remains the same for the participants, with the only difference of a salient dot or odd dot, which is introduced at a time interval of say 150 milliseconds after the stimulus begins, which moves randomly. Well, the participants were not aware that such kind of a dot is going to pop up, but this was mostly introduced to serve as a distractor and kind of deviate the participants from the task. And just to have a control, these are neutral trials which actually did not have any directions but net direction, but the participants were unaware of it. So this was the most difficult condition for the participant. Similarly, we had a static stimulus where the participants had to kind of look and arrange for the change in position of the cross in the second picture with respect to the first picture. Like for this, for example, in this picture, the cross moves from left hand side to right hand side. This was the condition without saliency. The condition with saliency had the similar task with the only difference of a salient stimuli that was introduced into the picture in the second picture. However, the task remained the same. In the neutral picture, however, this was again a control. The control was made in such a way that this cross was kind of not moving to the left or the right side, but was exactly on the midline. So the participants could not actually decide on to which side it actually moved on to, left side or right side. Yeah, so for this we recorded EEG data from 22 participants and with simultaneous behavioral recordings. So as expected, the reaction times for the trials without saliency were the least, followed by the trials with saliency because of the distractor that came into picture. Followed by the neutral trials, which showed the highest reaction times, which was expected because they were the most difficult trials for the participants. So yeah, interestingly, this pattern was shown for both the dynamic stimulus and the static stimulus as well. So WT is nothing but without saliency trials, ST is with saliency trials, and NT is neutral trials. So yeah. So with the EEG time series, we did a simple Fourier transformation and obtained the power spectra. And this is the power spectra for the dynamic stimulus. We bandpass filtered it between 0.1 to 80 Hertz. And what we saw is that if we compare it for the trials with saliency, without saliency, and neutral trials, okay. What was the difference was that there was only an enhancement in alpha power spectrum and no other frequency showed any enhancement. We see this for the static stimulus as well. So this was a common thing that alpha oscillations kind of subserve a role which is very specific to the salient distractor as such. We wanted to kind of see if the sources are also similar when it comes to dynamic and static stimulus for while processing saliency. And we found that if there are common sources that give rise to these alpha oscillations, which included the insula, temporoparietal junction, the LF. So basically, these are the regions that are shown in the first slide, which were actually responsible for processing saliency, but what was not known is that it is the alpha oscillations that underlie the processing of salient distractors. And it's the source, these sources communicate in the alpha oscillations. So just to see how these sources communicate, we did a Granger causality on the time series for the same spectral band, which is the alpha oscillations. And what we saw is that there is a common pattern of information flow when it comes to a goal-directed task, and the same task with goal-directed task when compared across dynamic and static tasks, which is like inhibitory control from LPFC to the VAA and the anterior TPJ. Whereas when the same task is presented with an oddball, irrespective of whether it's a static task or a dynamic task, there is this information pathway that emerges for both the task conditions, which is the VAA kind of initial causates or has a causal influence on the anterior TPJ, which has a causal influence on the insula, which kind of has a causal influence on the LPFC in turn. So this is the kind of a characteristic pattern of information flow where it comes to processing of oddballs across tasks while doing a goal-directed task. So this is for the visual modality. This is the next is a very preliminary study which we also replicated for the audio-visual modality and the visual modality. But this is a very preliminary stage. We haven't. We are yet to do the source localization in this. Yeah. So that's about it. So we want to establish these commonalities of alpha oscillations and the underlying sources with the other modalities as well, and see if there is a very common role that can be kind of classified for alpha oscillations and the role of alpha oscillations as such. Thank you so much.
Yeah, thank you very much, Priyanka, for this talk. We have time for maybe one short question. So if anyone has a question, put them in the Q&A window now. Maybe I'll quickly ask one. So in this Granger causality analysis, you found that there's this this oddball stimulus entry. Yeah, if you go one slide down, where you have this basically the overview of the network. So you basically found that there's this this oddball influence that changes the flow of information, right? So is that basically a switching from this goal-driven top-down attention system to the stimulus-driven system?
Exactly.
Yeah.
That's what is happening. So, and as we've seen that the goal-driven task is something that is kind of taken care by a top-down attention. So we can also say that LPFC, which is which kind of is more in the topmost hierarchical part of the brain, is kind of driving the regions which are lower in the hierarchy of the brain, so VAA or anterior TPJ, which very neatly kind of brings about what has already been there in the literature.
Okay, thank you very much. Let's move on to our third speaker. Our third speaker is Benedikt Ehinger. Benedikt is a professor at the Stuttgart Center for Simulation Science at the University of Stuttgart. And well, as Cameron has already mentioned in his talk, he's the developer of the Unfold toolbox, so make sure to check that out. And Benedikt will talk about analysis of EEG event durations. So take it away, Ben.
Can you hear me? Can you hear me? Can you see me?
Yes, we can hear, we can see everything.
Perfect. I was a bit worried because there were some hiccups. Okay, yeah, so this talk is going to be perfect in this session because Cameron did all the hard work explaining the methods. I will talk about very similar topic. And this works. Yes. Okay, cool. I will start with a very basic task. This is the oddball or P300 task. It works like this. You see letters A, B, C, D. And some of the letters are targets, like the C in this block. So you have to press the right button. And if you see a distractor, for instance, like the B here, then you would need to press the left button. And what we typically see in this kind of experiments is a result like this. So here's time. Here would be a microvolt of the average event-related response. And we see that we get this P300 effect, which is the target has an increased activity here. So in this talk, I want to raise 2 points, and maybe they are related, and maybe they are potential confounds. And the one is that the event duration, so the duration in some sense from the stimulus onset until we press a button, is different between two conditions. So first of all, it varies, as you can see here, reaction time varies, and also it's different between distractor and target. And this, this can directly, similar to what Cameron was talking before, can influence how the shape of our response looks like. And the second potential confound is because we are actually recording overlapping potential. So what we mean with this is that we could have isolated response to stimulus and isolated response to a button press, but we are actually recording both of them together. Now, because we have different reaction times between conditions, these different conditions might have different distances and so also have different overlap in our overlapping EEG. Okay, so what we did was, what I did was run some simulations, and later I will also show you applied to a data set. And in the simulations, I give you an example now with this response shape. So you have a positive peak, and then you have how the ERP looks like depends on the event duration. And I have it's a larger design, but I have conditions with and without overlap. And in the condition with overlap, if I have a response like this, the next response would also occur already during the first response. So we have an overlap here at the end of the responses. And for this one, we would have another overlap here. And then, you can, this is obviously what we're measuring, and we're trying to exactly using the Unfold toolbox and the approach that Cameron already introduced. We try to recover basically this shape, and we have various ways that we can do this, and we will see which ones might work better than others. Okay, so if in the left corner I have this no overlap symbol, then there's no overlap in the simulation. If in the right corner I have the no overlap, I also don't model any overlap. So this right now is basically a very standard analysis or that I could do. So this is again the simulation kernel. I try to recover this. I'm using the Unfold toolbox, which you should really check out. It's awesome. It makes such analysis very easy. And if you're familiar with the Wilkinson notation or this R formula syntax, then you can see that here I'm modeling now this EEG. I'm modeling by an intercept that would be the normal event-related potential, but also by a linear effect of duration. And if I understood correctly, this is also what Cameron did in his simulation. Right? If I do this, then I get something like something out like this. So what we can directly see here in the condition where there's no overlap and there's no noise, that we cannot really recover this kind of shape from our simulation. What we can also do is instead of taking a linear effect, we can take a nonlinear effect. So that means that we allow that we are not fitting a single line per time point, but we can also fit more complex shapes. And if we allow to do this, then with some artifacts here or here, we can recover the shape. So now let's have a look what happens if we add some overlap, but we're not actually modeling the overlap. Again, the linear effect doesn't look super good, maybe similar than before. Now we have a little bit more noise added. And if we do the spline effect, but we are not actually modeling the overlap, then, well, we get something that looks artsy, I would maybe say. I don't know, not very good. So it's clear that we also have to model the overlap. And if we do this, then the linear effect still does not recover our simulation here, but the spline effect can actually recover this. Now, this is just one simulation kernel, and I don't want to show you all the images, obviously. So we take some procedure or some accuracy score, and in this case, for each of these curves, we just calculate the mean squared error between this yellow curve here and the yellow curve here, or the yellow curve here, and we get a kind of score. These scores differ between the shapes, so I normalize this. Sorry, here's the mean squared error to the ground truth. 0 would be perfect reconstruction. 1 would be, what's 1? 1 is if I would just have one shape, so just a normal event-related potential without any event duration information. Okay, so this is the shape we saw before. I have other shapes, something like this, a boxcar, and maybe a half dropping off shape here. If we add the linear effect, then we can see we are much better, typically, except in a boxcar, we are better than just fitting a fixed constant. But if we add the spline, then we are, well, we can pretty much recover any shape to some extent. So there will always be some error, even though there's no noise in the simulation yet. I don't have time to really go into the noise, but I want to show another thing that you could do. And that is, you could, instead of fitting it as a duration or so, you could categorize the event durations into, let's say, 8 bins or something like this, and then try to recover the shape for each of the bins. And this is something like this, and I was a bit surprised that it looks that's so flat. But okay, the thing is that this is still better than assuming the linearity, but it's also a little bit over optimistic because we are actually checking where the bin center is exactly where we are also checking the mean squared error. Okay. I want to show you also how this could look on real data. So I'm making use of exactly this experiment in the ERP core dataset. And that dataset has 36 subjects that I could use, 200 trials only. So in total, 200 trials. So it's not that many. I generally don't use baseline correction. And this average reference. Okay, so I showed you this before, distractor target, that's a very typical effect. And yeah, please, these are preliminary results. As Christoph said, I'm setting up my own lab, and well, it's challenging to find time for research. Okay, I'm assuming that there's overlap of the potentials in the EG. For now, I'm not modeling the overlap, and let's see what comes out. This is my formula, so I model trial type and RT. I also model the button press, but it is not shown here. That may be a distraction only. I only show one condition because else it would be too messy. I show the condition effects later. Okay, this is normal ERP if you just run an ERP. This would be with a linear effective reaction time. So we definitely see that there is something to be explained by reaction time. And this would be the same effect just with splines. And as you can see, there's qualitatively, there's some similarities maybe here. Maybe it's a bit stronger or not. I don't want to go too much in the details. I also not sure how much I trust all of this to interpreting small details. But now I want to see what happens if I also model the overlap. So I get a shape that looks quite similar to what I had before. The reaction time is now effect is now, well, much reduced, I would say. But I get vastly different results if I allow the reaction time effect to not be linear, right? So then I uncover actually, I would say 2 reaction time effects. One maybe at 400 milliseconds and one even late, going probably into the period where the button press definitely are. Okay, and now I promised that I also look at the conditioned effects. So this is again what we saw before. This is distractor, this is target, this is the typical ERP that you would get. That's what everybody's been doing in some sense. If I now add the overlap correction, then I get an ERP that looks quite similar, I would say. Maybe the effect even starts a little bit earlier, but okay. This is not, I would argue this is not the things that we should look at, but we should probably look at what will be coming up here. And now the interesting bit is if I control for RT, so I model the reaction time, and now I can look at what would happen if distractors and targets would have had the same reaction time. right? What kind of potentials would I get? And then, as you can see here, for the stimulus-related response, there's not much effect left anymore. And yeah, there's I don't show it here, but there is still an effect of target and distractor on time locked to the button press, but it's not time locked in this case to the stimulus onset. Well, okay, preliminary results, we have to do many checks. Where are the statistics? This is not statistically tested yet. I want to test it on other data sets and see how it generalizes. And just, yeah. Well, I will first thank my new institution for giving me the possibility to set up a lab. I want to thank all the contributors of the Unfold Toolbox and for this project, especially Olaf Diemigen, Annalisa Gerd, Philip Allday for their constructive discussions along the way. And the take-home messages are, well, 1, you should probably model your event durations. 2, you should probably model them non-linearly and not linearly, but it depends on shape. And 3, you can model event durations and overlap both at the same time, which is great news, I think, for us. Yeah, thanks. I'm happy to take questions.
Thank you very much, Benedict. Maybe one question from myself. So you showed that, well, modeling, well, especially the nonlinear modeling improved your outcomes, but when you do the nonlinear modeling, you add another parameter or parameters. How do you find a good or the best parameter? And how do you know what's a good parameter if you don't know the ground truth?
Yeah, that's a very good question. A question that is there are multiple answers to. Right now, we are working on automatic methods to do so. The point is that if you get very flexible, if you allow very flexible regression splines, then you would also run into overfit. And you can do cross-validation in the Unfold Toolbox to try to find this number, but it's not. It's just a challenging problem with this kind of large regression problems to do it automatically in a regularized way or something like this. But that would be options that you have. Yeah.
Okay, we have more questions. So Etche Zainab asks, well, first of all, thank you. It was very interesting. And I wonder if you can differentiate longer overlapping activations such as gate and cognition.
Yeah, this is. It's a very interesting question. We have a data set, but we haven't looked at it yet for gate. Because gate has this time locked that maybe go over seconds or so activity, I think you can subtract it in some sense. There are also ICA-based methods that try to subtract the gate method. Maybe they are also suitable. I don't know.
Okay, and another question from David Ricardo Quiroga Martinez. Does that potentially mean that P3 effects are mostly related to behavioral responses and reaction time rather than purely cognitive processes?
Well, probably not, I would say. I don't know. There are papers on P3 that show that P3 is partially stimulus locked and partially reaction time locked. And so maybe the stimulus locked version of the P3 is just in some sense a consequence of the different reaction times and different overlapping potentials. Maybe it's hard to say at this stage, but yeah, it's a possibility. That's something that I'm raising here. Yeah, we'll see. Probably it's both, as always.
Okay, thank you very much, Benedict. And well, thanks again to all the speakers, Cameron, Priyanka, and Benedict. Thanks to the audience for the very nice questions. And also thank you to Alexandra, who was running the back end and make sure everything went smoothly. Yes, enjoy the rest of your conference, everyone, and goodbye.
Good to go. Great. We'll just give it another few minutes here. As people arrive. All right, so I'm gonna get started here as people are coming in, and just introduce myself in the session. So hey everyone, I'm Adam Steele. I'm a postdoc at Dartmouth College in Hanover, New Hampshire, USA. I've been told that we should give a few moments just to allow people to trickle in as the webinar is starting. So I'll use that time to say everyone in the United States should make a plan to vote next Tuesday. If you already have a plan, call one of your colleagues and make sure they have a plan. And if you can't vote in the US elections, call someone that you know in the United States or send them an email and just make sure they have a plan, because it's super important. So we have a great lineup of speakers in this session who are going to be presenting three really interesting talks about the integration of sensory information during perceptual decision making. This is an interactive session. So the speakers are going to give a 5-minute talk and leave 10 minutes for audience discussion and questions. If you'd like to come on stage to ask your question or chat with the panelists, there will be time for that. So just post in your question whether you would like to ask the question yourself or whether you'd like me to ask it for you. Otherwise, please post in the chat. Make sure, if you choose to post in the chat, just make sure that your messages are sent to panelists and attendees, so that way everyone can see what you're saying. Because this is an interactive session, if you'd like to introduce yourself in the chat, now is a great time to do that. Also, we have the Q&A tab for questions, and you can post there at any time. If you post, for those on Twitter, please tweet about this session using the hashtag NMC3. So our first talk, with that, I'll get started with the session. So our first talk is going to be given by Jacopo Haschen of the International School for Advanced Studies. He'll be presenting his work related to a recent bioRxiv paper, which we will link to in the chat. Jacopo's work is titled Dynamics of History-Dependent Perceptual Judgment.
Okay, hello. Hello, everyone, and thank you for being here. So it is known that perceptual decisions might vary from time to time, even if we present the subject with identical physical stimuli. This variability can be partly linked to the history of previous trials, which is itself composed of several factors such as stimuli, choices, and the outcome of those choices. So in a recent work from our lab, we have focused on the role of stimulus history in shaping perceptual decisions and its updating dynamics. Specifically, we have trained rats to collect reward by classifying tactile vibrations delivered on their whiskers as either strong or weak, depending on the mean speed of the vibrations. We used 9 different values of speed, and in particular, we had 4 high-speed stimuli, 4 low-speed stimuli, and 1 stimulus which was in between of the 2 categories and was therefore unfair and rewarded randomly. We also replicated the task in humans by providing vibrations on the fingertip and by rewarding the subjects accordingly, according to the percentage of correct trials. So the first important observation that we made was that recent stimuli influence perceptual decisions in a repulsive way, which means that the stronger the speed was in trial N-1, the previous trial, the less likely the rat would be to classify the current stimulus as strong, so that the psychometric function in trial N would move to the right after a strong speed and would move to the left after a weak speed in trial N-1. This effect did not only hold for the immediately previous trial, but also for further trials in the past. In fact, we see that even when we consider trial N-2, -3, and so on, up to about 6 trials in the past, we observe this repulsive effect the magnitude of which seems to decay exponentially. We therefore decided to try to model this effect. And in particular, we devised a recursive model of rats' decision criterion, which works as the following. So here on the x-axis, we have the time course of the session. On the y-axis, we have the speed of the stimuli that are happening here as dots. And the blue line represents the rat's decisional criterion, which moves around as stimuli happen during the session. At each new stimulus, the decisional criterion is updated by moving closer to the value of the stimulus itself, according to a certain time constant, which is the only parameter of this model. So here, for example, there is a weak stimulus happening in the session, and the decisional criterion moves down. And therefore, the next stimulus, which is an unfair stimulus because it lays on the boundary, is more likely to be classified as strong, since the criterion was below the central category boundary. Of course, perceptual decisions are stochastic, and therefore we can think of this decision criterion as the center of a logistic function that moves around as the criterion moves around. In this way, we can reproduce the psychometric functions that we fitted to the rat's data and that I showed you before in the previous slide. After this, the next topic in which we were interested was whether the intertrial dynamics would somehow influence the impact of the previous stimulus. For example, we could expect that after longer intertrial intervals between stimulus n-1 and n, the effect of stimulus n-1 would decrease on stimulus n. And here we consider the intertrial intervals in our data set. And specifically, if we isolate only the trials following a short intertrial interval and only the trials following a long intertrial intervals, and we look at the impact of previous stimulus on the current stimulus, we can replicate an analysis similar to the one I already showed you. But surprisingly, we see that the effect of stimulus n-1 seems to increase with intertrial intervals, so that after longer ITIs, there is more impact, more repulsive impact of the previous speed. By looking at a finer detail of this effect, we can see that the bias due to stimulus n-1 seems to grow continuously with ITI length, and this effect seems to saturate after a few seconds. We therefore decided to refine the discrete model that we previously proposed by making it a continuous model in which the decision criterion of the subject is attracted to the memory trace of the immediately preceding stimulus over time. Here, for example, again, the preceding stimulus was a weak stimulus, and the decision criterion moves towards the memory trace of that stimulus, influencing the next decision. This model reproduces the behavior of the discrete model that I already showed you, but it adds that it can explain also the effect of intertrial interval that we observed in the data. And in fact, when we compare the time constants of the two models fitted for different animals and different session subsets, we see that the parameters are very highly correlated, indicating that the continuous model can explain the discrete model. Surprisingly, we see two clusters of animals that have a longer time constant and animals that have a shorter time constant. And the animals that have a longer time constant seem to perform better than the animals that have a shorter time constant, likely because they can better estimate the sensory environment and therefore produce a more stable decision criterion to perform in our task. So thank you for your attention, and I'm now open to questions.
All right, thank you very much, Jacopo. That's super interesting. I really like this. One thing I had a clarification question is that the speed that you're talking about here is not the speed of the animal's choice. It's actually the speed of the stimulus that you're presenting, right?
Yes, exactly. So what I'm
sorry, I know this
about,
was a very early point, but I wanted to make sure that I was totally
if I did
clear.
not make if I didn't make it clear enough. So this is the stimulus that we give to the rat in terms of a noisy vibration applied on the whiskers. And what matters is the mean speed of the plate that simulates the animal's whisker during the vibration. So we have vibrations that have a higher speed on average and have to be classified as strong, and vibrations that have a lower speed on average and have to be classified as weak.
So does your model of the animal's choice integrate is it purely an accuracy measure, or is it an integration of both the speed that the animal responds, which could be like some kind of proxy for their confidence, or not? Because that could also be interesting to know how the animal's own confidence is adjusting the decision boundary. And then I'm going to ask Tom. I'll ask Tom Marshall whether he would like to come on stage to ask his question. So Tom, if you could post that in the chat, that would be great. Sorry, Jacopo.
Okay, yeah. So it's really relevant, the point that you raised. And can I reply?
Yep, absolutely.
Okay, sorry. So yes, in fact, when we look, for example, at the ITI distributions, it could be that since the confidence of the animal can be affected by the difficulty of the stimulus that is presented in the previous trial, this effect is not purely determined by the inter-trial interval between stimulus n-1 and n, but also by the difficulty of the stimulus in trial n-1. And therefore, we also replicated this analysis by subtracting the response time of the previous trial from the overall ITI value to make sure that we rule out this potential confound, and we obtain the same result. I'm not showing it here, but.
Great. Yeah, that seems like a really important thing to control for.
Yeah.
Stefan asked a clarification question for replicating the psychometric functions. Did you use the 20% fastest and 20% slowest ITIs?
But I think I got that now.
Oh, you got it. Okay.
It seems like those are just the error margins in the plot, right? Or so to say, the confidence bounds, or.
So yes, sorry. So here it's 20%, lower 20% and higher 20%. Actually, so these are just quartiles. And in this specific plot, we compare the lowest quartile with the highest quartile. And we consider only trials that had this, in which the ITI was comprised in these intervals.
Okay, thank you.
And yeah, sorry.
So I'm going to ask Tom's question. If he'd like to come on stage for clarification or to clarify the point, just let me. Oh, here he is. He's going to ask his question.
Yeah. Okay. Can you hear me?
Yes.
Okay, great. Yeah. So I just wanted to know what was the training regime you used for the animals, because depending on how well they were pre-trained on the set of stimulus speeds, I guess you could consider shifting a criterion. You could sort of reconceptualize that as just learning and learning better about the set of possible stimulus intensities. So is there a sort of conceptual difference there, and what was the training regime you used prior to the experiment?
Yes. So the way we trained the animals was initially they start just by learning to poke their nose into the sensor and waiting for a vibration. In the very first stage of the training, they're just cued to respond to the correct side. And then progressively, we introduce stimuli in which the rat is not cued. So it has to learn that there is an association between a strong vibration on one side and a weak vibration and the other side. And we progressively introduce more difficult stimuli until we get to the entire stimulus set of 9 stimuli. And we only start collecting the data set once the animals perform well enough in the final task with all the 9 stimuli, usually around 75% performance. Right. And what's interesting is that data is not shown here, but it is in the online manuscript, in which we see how the performance changes in the early part of the session with respect to later part of the session, so that rats, even if they have been well trained in this task and they know the rule very well, they know that for strong vibrations they have to go right, and for weak vibrations they have to go left, and vice versa for different animals. But they need to tune their decisional criterion in the early trials of the session. So the psychometric function in, say, the first 5 trials is much less accurate than in later trials.
Right. So there's it's almost like there's kind of learning on different scales. There's the sort of a cross session learning, which they have to get to a certain level of before they even begin. But then there's also like a within session learning that's happening as well. Is that right?
Exactly. And our model is precisely trying to capture this component, which is related to the within session learning.
Yeah.
So they have to consolidate this decisional criterion. So they need a few trials to get it running, and then they can perform, I would say, usually between 70% and 80% correct trials, depending on the rat. But as you saw, for rats with a longer time constant, they can better estimate the sensory environment because they can remember more stimuli from the recent past, and therefore they have a more stable criterion that is closer to the center because they're averaging more past stimuli.
Yeah, I see. That's really interesting. Thank you very much.
Thank you.
Great. So that was a really excellent talk. We're going to have to move on to our second talk of the session, but thank you again.
Thank you.
So our second talk of the session is Dominik Szymanski of Cardiff University, and his talk is titled Integration of Information in the Perceptual Decision-Making Task. So take it away.
Great. So thank you very much, everyone, for tuning into my talk. As this is supposed to be an interactive session, feel free to ask me questions at any time during my talk. To give you a little bit of a background, perceptual decision-making is a process by which humans decide between available options based on the information from the external world. This is opposite to the value-based decision-making, where people tend to make decisions based on the internal representation of available options. And so far, the evidence from animal studies, but also like human cognitive experiments, shows that brain tends to like add the information from like multiple cues. However, the details on how this like addition like works is not quite clear, especially that most of the studies use cues or attributes that have like a different accumulation rates. And we believe that to investigate that phenomenon in details, one needs to use the experimental setup when this accumulation rates would be normalized. So we propose an experiment that is a modified random dot motion discrimination task. You can read the details of our experimental setup in this pre-registered report at OSF. But to summarize briefly, our hypothesis is that we believe that from our like previous experimental work, that the integration of like information from multiple information sources happens non-linearly, and that there is also a difference in performance when people integrate information from information sources that are more correlated. And as I mentioned, to investigate that process, we extend this classic random dot motion discrimination task that you probably know, such that we add yet another axis. So here in the part 1 of our experiment, actually one of the axes is shadowed to black, and we ask our participants to focus only on a single axis. We also manipulate the angle between the axes to increase the strength of the correlation between the information input. And in this first part of our experiment, we use a staircase procedure to determine 2 coherence levels. One which is more difficult, and the second that is like easy. We do it with this procedure that when participants respond correctly to 2 consecutive trials, then experiment becomes more difficult. When they make incorrect choice, then it becomes like easier. Similarly, with this like more easy condition, we use this research that you have at the bottom of the slide to get an optimized setup for this forced choice staircase procedure. And once we determined the coherence, stable coherence level for difficult and easy condition, we are moving on to the second part of our experiment, where 2 of the axes are actually highlighted, and people need to pay attention to both of them. And here we give to our participants 2 like major conditions. So in the first one, they need to take a look at the both axes. So if we, for example, have coherence 2 that has like 10% and coherence 1 that is 5%, people need to combine that information together. Whereas in the second subcondition for congruent, on the one axis there is like 15% of coherent dots. In the second axis, there is like 0 coherence level. So even though the total information on the screen is the same, it is provided in a different way, and we wonder how people combine that information. Similar with incongruent condition, when the second axis has opposite direction to the dominant dots movement. We also introduce this sort of like a dummy control condition just to as attention check. To reduce the fatigue of our participants, we split the whole experiment into 6 blocks. All the conditions are shuffled, randomized, and they consist of like 2 repetitions of the whole sequence. So here you can see the gist of our experiments after the part 2 of the experiment. As you can see, after like a little bit of training, people usually become quite skilled in that task. So, and because of the whole pandemic situation, we couldn't really do this experiment in the lab setting, so we had to move it online. We use a really nice JSPsych JavaScript library to implement our experiment. We stored it on the Pavlovia server, and we used Prolific service to recruit our participants. If you're interested in learning more about how to run that kind of psychophysics experiments online, you can read our blog post when we shared our experiences from that. And since that experiment was done by participants at home, we had to introduce quite strict acceptance criteria. So the first one was that indeed the convergent coherence values agrees with the theoretical values, such that the coherence 1, which is like more difficult, is lower than the easier coherence 2. We wanted to make sure that actually the difference between the coherences is possible to perceive by participants in the part 2 of our experiment. And also we introduce some extra performance checks for the part 2 to eliminate participants who are just pressing buttons randomly. In total, we collected 45 recordings for one angle, and with a little bit of overshoot for the second angle to give us 0.9 power to detect within group difference of a medium effect size. And here you can see that indeed in the staircase procedure, participants tend to converge to some stable value. We were taking 8 last reversals to determine our like average coherence level for 2 staircase procedures. For the coherence 1, people tend to converge around like 45 steps. For the coherence 2, it was like around 65. But more importantly, on this figure, you can see the values to which people converged. For the coherence 1, it was like around 8% of dots coherence. For the coherence 2, it was around like 15%. There was no significant difference between the angles, but this is kind of like expected, as in this part 1 of the experiment, what people were doing was actually this like classic random dot motion discrimination. So actually the rotation angle didn't really matter. Okay, when it comes to the accuracies in the second part of the experiment, here we can see the plots broken down between like various experimental sessions. So two different angles, and single axis denotes the condition when information was available only on the one axis. Addition condition means that people had to combine the information from two axes of the experiment. And as you may see, there is like a slight positive trend here, which means that people tend to be better when the information was available only on the single axis. However, there was no really significant difference when it comes to the strength of the information. So the angle between our axes, so that a little bit contradicts our like initial hypothesis here. When it comes to the reaction times, on the other hand, as you may see in the congruent condition, people tend to be faster in the single axis condition, and it took them like longer to respond for the condition when they had to combine the information. On the other hand, in incongruent condition, people tend to respond faster when they were like subtracting information, and slower when the information was available only on the single axis, which sort of agrees with this like common speech accuracy trade-off phenomenon. Again, in terms of reaction times, we didn't see any significant difference between the angles of the input sources. So that pretty much summarizes all the behavioral results we've got so far. And as this study is still ongoing, we are working on the model that would be able to actually explain that data. And we want to extend this classic Wong and Wang model that was used to explain on like more biologically plausible level the classic random dot motion experiment. And here we hypothesize that actually we might deal with like two pairs of like neuronal pools that somehow communicate each other to, and now we want to tune the parameters of that model to actually reproduce the behavioral results we observed so far. So to conclude, I presented you the online psychophysics experiment, which is extension of random dot motion discrimination task. We noticed the higher performance for conditions with like single information source. On the other hand, people tend to react faster when they were subtracting two coherence movements, and they were actually slower when they were like adding that information. We also noticed no significant difference between the more correlated information sources. So that's pretty much everything I got. Thank you very much to all my like funding buddies, and here at the bottom you have my online presence, so feel free to connect if you want to learn more about this. Thank you.
Great, thank you, Dominic. That was again great talk.
Sure.
So we've got time for questions. We'll take the first question from Stefan, who asks, did you experiment with also staircasing the angle between the two RDKs? Or did you start only to consider the two angles that you used in the experiment, which were 20 and 45 degrees?
Yeah, we did not. However, that would be actually like interesting thing, and we are like fully prepared actually to extend this like experiment with the way it is like implemented. So far, we just wanted to focus on those like two hypotheses that we have, which was like the strength of the like information source, whether like people actually do pay attention to it, but it seemed that they do not. However, if we actually introduce some like staircase like on that, indeed for like very, very strong like information sources, so when basically the angle is zero, that might reveal some like interesting effects.
Yeah, I think that would be really really interesting to
get
see.
that edge case.
Yeah, it seems quite interesting, because the results in the second set of data that you were showing, it seemed, if I was reading your plots correctly, that the angle did matter in the 45. There was a difference between the 20-degree case and the 45-degree case. Was I reading that correctly?
No. So actually, we actually didn't see like any difference, neither in terms of like reaction times nor in terms of accuracy. However, like it was like in even with like 20 degrees, as you may see, like the two axes are still like very easily like separated. But the interesting bit is like what happens when it's basically like difficult to, you know, like discriminate between them.
Yeah, and it seems like I guess the presentation is quite rapid, so you don't have to worry about the peripheral. Ideally, you wouldn't have to worry about the peripheral information so much, where they could be getting like just isolated from the axis that's would be most relevant, I suppose. But how can you be sure that that was the case in an online setting? Are you able to like insert some kind of catch trial to make sure that that doesn't happen?
Yeah, so well, I mean, first of all, you know, this is just a little like you know, excerpt. So we basically try to put that stimuli exactly like in the middle of the screen, and we were also like monitoring the resolution of the participant screen. So another acceptance criteria was that they have like large enough screen such that we know that, you know, like the stimuli is like centered on the screen. So this like, you know, like the peripheral like vision is not a problem here.
Yeah. So if there's any other questions, you can post them in the chat, and Dominic will be here to answer them. In the meantime, I think we're gonna have to move on to our final talk of the session. So thank you again for that talk, Dominic. And our final talk is by Stefan Appelhoff from the Max Planck Institute for Human Development. And his talk is titled Control Over Sampling Boosts Numerical Evidence Processing in Decisions from Experience.
Yes, hi everybody. Can you see my slides?
Yes, we can see the slides.
Okay, cool. Today I want to present some results from my current study that shows that control over sampling boosts numerical evidence processing and decisions from experience. And the big research question for this study is how does control over sampling impact choice? And it's motivated by the fact that for many decisions, humans can base their choice on sampling experience. However, this sampling can differ quite strongly in terms of the actual control over the process. For example, when buying a car, you can decide both which cars to try and for how long to try them before buying them. However, when you do a standardized job interviews, you may only get 2 applications and you have to select a single one of them. Previous research has shown that this control over sampling does impact the final choice. And in our study, we are interested in how the neurocognitive mechanisms are affected by that, or how they affect that. Specifically, we are using a numeric sampling paradigm where participants first sample numeric outcomes between 2 options, and then after some time, they make a one-shot final decision for one of those options and get a reward for that. And the reward comes up in the form of a random draw from that finally chosen option. And crucially now, we vary the amount of control that participants have over the sampling process. In full control, they decide both where to sample and for how much to sample. Whereas in partial control, they only decide where to sample. And after a fixed number of 12 samples, they have to make that choice. So here the number of samples is fixed. And finally, both of these these conditions have control conditions or baseline conditions that are associated with them, where participants only see a replay of the sampling and they have to base their final choice on that replay. Now let's turn to the results. To check whether the control over sampling truly influenced the final choices at all, we had first a look at the behavioral accuracy. And here we saw that participants were significantly more accurate in identifying the option that was more profitable when they had full control over the sampling as opposed to only partial control or no control. We then also wanted to see whether the integration of sampling information over time was different. So we calculated decision weights for early, mid, and late samples in the sampling sequence. And we found a clear recency pattern overall in that late samples had a higher weight than earlier samples in the sequence. However, this did not show to be any different between the different sampling conditions. The recency was approximately the same. So much for the behavioral data. Next, we looked at our EEG data to get an insight into whether early perceptual processes, early visual perceptual processes differed between the conditions. For that, we looked at two early visually evoked potentials, the P1 and the N1, and compared the amplitudes. However, it turned out that the amplitudes were absolutely comparable, and so there was no difference between full, partial control, or no control in terms of these early visual processes. Okay, now we know that early perceptual processes are not strongly affected by the level of control over sampling. But what about later cognitive processes? And to shed light on that, we made use of earlier findings by Spitzer and colleagues 2017, who showed that in a different numeric paradigm, participants encoded the number information along a neural number line so that the multivariate representation of a 1, for example, is closer to the multivariate representation of a 2 than it is to a 5, for example. And in our study, we harnessed this finding to see whether the number line encoding is different between levels of sampling control. And our results show exactly that, as you can see in the plot. Firstly, there's a number line encoding in all of the sampling conditions. But secondly, this effect is significantly stronger under full control, but not partial or no control over sampling. And in split analysis, we furthermore showed that this effect is present throughout the whole sampling sequence. That is, it arises early on. The final result I want to show to you today is based on the question whether the accumulation of sample evidence differed between conditions. And for that, we looked at the amplitudes of the P3 ERP component in early, mid, and late samples over the sampling sequence. And interestingly, the accumulation was steepest under full control again, as can be seen in the plot on the lower left. And now showing this as a summary plot together with the no control baseline conditions, we see that the accumulation happened in every condition, but significantly steeper in full control. And this is again mirroring the same findings that we found for behavioral accuracy and for the number line encoding. And that already brings me to my summary, that control over sampling boosts numerical evidence processing. And we found that under full control over sampling, there was a higher accuracy. There was no difference in early visual perceptual processing. However, we did find a stronger encoding of numerical information that was evident early on in the sequence. And finally, steeper accumulation of evidence in neural signals. However, no difference in evidence integration in the behavioral data. I want to thank my collaborators and supervisor Bernhard Spitzer and Ralf Hertwig, my lab and data collection team, and of course you for listening and joining.
Great, thank you, Stefan. That was it's super interesting. I like the idea of the number line, like the numbers encoding strength being particularly important for the decision making here. I'm wondering I like that idea. I'd like you to explain it a bit, just a bit more for me, why the strength of the similarity to the neural number line should be the determinant. Do you think that this is driving people more toward making the decision drive closer to this decision boundary because their kind of internal representation of the number is stronger, or is it some other type of process?
So I think it could go into several ways here. So if participants have a stronger encoding of the numbers in the space, in this paradigm, the numbers also directly relate to a reward. So a 9 also means a higher reward than an 8, and an 8 higher reward than a 7, and so on. And if the multivariate neural representations of these people are ordered in a way that is consistent with the way these stimuli, after all, are rewarding, then that probably shows that they have a better grasp of the whole situation, and that can improve their behavioral performance.
So when you when the participants are making the decision, so this would be like in the behavioral results that you have, do the essentially my question is like, if they see a 9 at that, like, do they stop sampling very early? Because it's very unlikely that they would receive a number on the end of that number line. Like that would be very unlikely if it was a low choice pool. Does that make sense? Or perhaps I misunderstood exactly how you were doing this 2 choice, like how the distribution of samples from the 2 choices was going to work out.
Yeah, so they could sample from either option and draw numbers out of the options, and they would get an impression of how often one option or the other option would provide higher numbers or lower numbers. So each of these options had an expected reward, and this expected reward was different. And the task was mainly to find out what's the better option in the long run. And what you're saying is what we see partially in the data, that when participants experience these extreme numbers, for example, a 9 or a 1, which were the bounding numbers, so there were rewards from 1 to 9. If they saw a 9 or a 1, they reacted slightly differently. We see that in ERP amplitudes that, for example, the P3 amplitudes were U-shaped actually, so that the extremes had a much higher amplitude compared to the other numbers. So there's definitely something that we are also still looking into with these extreme values.
Yeah, because that makes a lot of sense, because that's where like the majority of the information is in these cases, and it also it also is the maximal differentiation in the RSA. So is there any way that you can is that something that you feel that you need to control for in the representational similarity analyses, or is that a facet of the data that's actually helpful for you? Is the question. Like, how do you think about that?
That's a great question, and we have considered that, and we also have a second RSA model. So I have shown you the number line RSA model. We have a similar model that we call the extremity model. And in that model, the extremes are more heavily weighted. So that's a model that would weight a 5 as more similar to a 6, and the extremes to be more similar to each other, so a 1 and a 9. And we did analysis with that model as well on the data. And we did analyses where we orthogonalized the extremity model and the number line model to see whether the number line model still has the effect that we find after we partial out this extremity model. And that is what we find. So the number line effect that I showed in this presentation is preserved. And however, the results with just the extremity model are still a bit inconclusive. So I didn't share them today, but we're still working on that and trying a couple of things, thinking about on how to share these results.
Yeah, that's really encouraging, because that does suggest that it's about the number encoding rather than being about this kind of information content. So in the replay trials, are they replayed previously what they'd already seen, or are they replayed so that way if they did end up seeing one of these extremes, they would notice to make that selection? Or is it like a randomly generated new stream of numbers and it's just matched for the number of samples that they chose?
So these are actual, these are actual replays, and but these are replays where the participants are unaware that they are the replays of their own stuff. So a participant would, for example, come in, do the first part of the task where they actively sample, and then there's a short break, and then they would see the replay of what they just did. And we always perform post-hoc interviews with the participants to get an idea whether they realized that. And actually none of them did, which is maybe understandable given that they are sampling like 1,200 times.
Yeah.
Now, difficult thing was to counterbalance these things. So when a participant comes in and he's supposed to see the replay first and then do the active sampling, That's obviously impossible logically. So for these people who started with seeing the replay, they actually saw the replay of another person. And that is just how you have to counterbalance it. It's a small compromise that you have to do for this kind of experimental design.
Yeah, I think that makes a lot of sense. And always, I suppose you, depending on just how greatly people's the influence of how great of individual differences they are, actually crossing people's participants' experiences also seems like it could be a reasonable thing to do. But the counterbalancing is tricky there if you want to actually represent participants their own experiences. Cool. So I think thank you for that. I think we need to wrap up here. I want to thank our speakers again for this session, and the audience for listening. We hope you guys tune in to future NeuroMatch sessions as they continue on in the week. And yeah, have a nice day, everyone. Ciao. We'll be able to come on stage too, so that's nice. Great. So we'll just give another minute for people to come in, and then I'll introduce the session. Arrange my windows. Okay. Wow, that's I understand Mohammed's feeling of empowerment. It does feel like you're in a bit of a fishbowl as somebody in a Zoom webinar, but here we are. We can always bring you on stage if you'd like to be present, though. We would like to do that. So I'm going to get started. Hey everyone, I'm Adam Steele. I'm a postdoc at Dartmouth College in Hanover, New Hampshire, USA. I'm just going to give another moment for people to trickle in, and I'll use that time to say to everyone in the US to make a plan to go and vote. If you already have a plan to vote, please call your colleagues and make sure they have a plan. And if you can't vote in the US elections, you might consider to call your US colleagues and ensure that they've got a plan, because it's super important. So we've got a great lineup of speakers for this session who are going to be giving 3 interesting talks for discussion on visual processing. This is an interactive session, so the speakers are going to give a 5-minute-ish talk and leave 10 minutes for audience questions and discussion. If you'd like to come on stage to ask a question, there will be plenty of time for that. So just post your question in the Q&A or in the chat, and include whether or not you'd like to ask it on stage or whether you'd like to have me ask it for you. Otherwise, you can post in the chat at any time. Just make sure that you have your messages set to panelists and attendees, so that way everyone can see what you're saying. Because this is an interactive session, if you'd like to introduce yourself in the chat so that way everyone can see who's here, feel free to do that right now. Just post your name and where you're calling in from. And the Q&A tab is open for questions. If you are on Twitter, please tweet about this session using the hashtag NMC3. So with that, we'll get started. Our first talk is being given by Chen-Shi He of the International School for Advanced Studies. I think that may be incorrect though. I'm sorry if it is. Her talk is titled, "What is the nature of representation in tool selective areas in the parietal cortex? Effects of category, shape, and spatial frequency." And again, I apologize if that's not the correct affiliation.
Can you guys see my screen now?
Yep, you're good.
How about the audio?
Audio is great.
Okay, thanks. So hello everyone. Okay. Oh. So every day we encounter numerous objects and we retrieve different aspects of knowledge about these objects, including its category, its shape, color, the function, and also the context that they usually appear in. And among these objects, we are particularly interested in one special category, tools, because tool cognition usually recruits a dedicated brain network, including two important parietal areas. So these color patches are the ROI overlays from all of our participants. The blue patch is roughly the inferior parietal lobe, and the red patch is roughly the superior parietal lobe. And we want to know how these two areas support different aspects of tool knowledge. And more specifically, we want to know whether they represent more category information of tools or more shape information, namely the elongation property of tools. We're particularly interested in elongation property because this property affords many action and manipulation on objects. So in our fMRI study, we used animals and tools with comparable visual features measured by GIST statistics to examine the independent effects of category, shape, and spatial frequency. So this experimental design and stimuli has been used in one of our previous studies, focusing on the ventral visual category selective areas. So we include both low and high spatial frequency images, and with two types of shapes, one round and the other elongated, for both animals and tools. So first, we want to build the potentially different roles of SPL and IPL based on their differential functional connectivity with the other two selective areas beyond the parietal cortex. And we defined those regions of interest using a separate localizer, which used tool and animal images with naturally varied visual properties. So in this way, we defined 5 ROIs, including SPL, IPL, precentral gyrus, and ventral visual areas, which is posterior middle temporal gyrus and medial fusiform gyrus. So the first basic thing that we found was significant correlations among all those areas, except between MFG and precentral, which is not surprising. And the interesting part is that we found this clear contrast between SPL and IPL. So specifically, the medial fusiform gyrus has stronger functional connectivity with SPL compared with IPL. And in contrast, precentral gyrus has stronger connectivity with IPL compared with SPL. So based on these patterns, we anticipate that the more connected with an area, then the representational nature would be also more similar to that area. So that means because we found the SPL is more connected with the medial fusiform gyrus, then we would predict that they would represent more semantic category information about tools, because we already found in our previous study that the medial FG represented mostly category information. And in contrast, the IPL would represent more action-related properties in this case, it's the elongated property which affords actions. So we investigated this prediction by using the representational similarity analysis. So we did this by correlating the multivariate neural patterns within SPL and IPL with our theoretical models. So for instance, here, the 2 model means that the neural pattern similarity would be higher among items within the 2 category, but it would be lower among items within animal category or cross categories. And we are also interested in high spatial frequency, particularly because previous studies have suggested that recognition of man-made objects or tools compared with animated things rely more on the high spatial frequency information than low spatial frequency information. And we also included a few interaction models by multiplying the basic models with each other. So here we can see that the best models are the interaction models for both RIs. But if we only look at the basic models, we also observe this consistent contrast, which shows that for SPL, 2 model outperformed the elongated model, but for IPL, elongated model was better than the 2 model. So in summary, our functional connectivity analysis and the representational similarity analysis actually gave quite convergent evidence, showing that SPL has stronger connectivity with the medial fusiform than IPL, and mostly represents semantic category of tools. And IPL has stronger connectivity with precentral than SPL, and primarily represents information of elongation. And it's possibly because elongation provides action affordance. And these results revealed how different aspects of information are represented in parietal 2 selective areas, which show different connectivity strengths to other 2 processing areas to support recognition and potential action planning. Thank you very much.
All right, lovely talk. So we have plenty of time for questions. We have one coming in right now from, well, I'll just read the question. So based on motor imagery studies, I find it possible that the tool images activate the sensory motor cortex and sensory motor network. What do you think would happen if you present objects instead of animals to compare tool-related activity?
Okay. Yeah, I think that's a good question, but maybe I need some clarifications. So does she or he mean that what if I compare tools with other non-tool objects?
Mm-hmm. I think it's, yes, yeah, okay. The clarification is confirmed.
Yeah, I don't know actually, because I indeed noticed that for other people, a lot of people have investigated by using stimuli with only tools and non-tools. And they have some interesting, but very mixed, very not inconsistent findings about this. So I think this needs to be like investigated more systematically.
Mm-hmm.
Yeah. Right now, I cannot have a very clear conclusion.
I think one thing that I was thinking about, which is somewhat related, is whether or not you could differentiate the tools more finely based on how they are being interacted with. So like a saw movement is much more similar than like what you had with a microphone, which were just 2 examples that stuck out to me. So doing something like including an RSA model that had like movement type or similarity of use case could also be an interesting way of getting at that question.
Absolutely, I absolutely agree with you. So when we designed this study, the 2 stimuli was not actually very optimized for like including a wide variety of the way of manipulation. So that's why we didn't include something like a manipulation model or something like that. But if we may have a wide variety of two stimuli which have allow us to order the tools by the way we manipulate them, then it would be a very good point to do that kind of analysis.
Yeah, I think that would be really potentially enlightening, especially as the one of your regions, I guess it was the inferior parietal lobule, looks like it's much closer to like somatosensory cortex and sensory motor areas. So it wouldn't be inconsistent with your results. So I'm going to ask another question that's come in. How were the results from the high frequency RSA? My expectation is that since IPL and SPL are located along the dorsal pathway, which are comprised of mainly large filters, they won't capture much info.
So with high spatial frequency, actually, they are represented significantly in both ROIs. Can you see my slides on the RSA results still? Yep. Yeah. Yep. So if you can see the bars for high spatial frequency model, now it's abbreviation for HSF. So
can you use your mouse to point to it? I think it's harder. It's hard to grab. Yep, right there.
It's here. So it's still significant. And for SPL, it's, for IPL, it's here. So both of them are significant. So if you know, there is a paper by Bradford Mahon, 2013, on Journal of Cognitive Neuroscience. They actually tried to use low and high spatial frequency images and observed this double dissociation in parietal areas, but they did not exactly separate between IPL and SPL. Maybe they're mixed a little bit together, but they do found this segregation. So for one subregion, it's more sensitive to low spatial frequency, but for the other subregion, it's more sensitive to the high spatial frequency. And they said that the high spatial frequency sensitive one actually receives information from the ventral visual area. But in our case, like it seems there's no sensitivity for low spatial frequency, but only for high spatial frequency. So it's kind of conflicting or something that we need to reconcile later with other studies.
Okay, great. And then really quick answer to our last question. Were there animals which were graspable?
Okay, let me go back to my, is there any way that I can show the stimuli set? Probably. I don't quite remember graspable. Maybe I remember there's an animal we used is needlefish. Is that graspable?
Well, if not graspable, perhaps like touchable. But I think if you want to discuss that further in the chat, that would be probably the way to go because we need to move on. But thank you. Okay. That was really a really great talk, very clear. And I'm going to go on to our second talk in the session, which will be given by Raul Hernandez, titled "The Visual World in the Dog Brain."
Okay, so I'm going to share my screen. There you go. Is it? Good. Okay, thank you for the introduction, Adam. So this is the name of my talk, The Visual World in the Dog Brain. So first, I would like to introduce you to my dog, Odin. Odin, it's a very interesting dog. He does very particular things. When he sees my cat, Kami, he has this very particular behavior. He stays really still. He tries not to look at the cat directly and not to make any sudden movements. In this way, he can stay for more time looking at the cat. so the cat doesn't get scared or he doesn't attack him. But he does something very different when he sees his friend Paula. When he sees Paula, he knows that it is time to play. So he tries to find a stick. He grabs the stick and takes the stick to Paula so she can play with him talk. But it is different when he sees Rohan. Rohan is his dog friend. When he sees Rohan, he knows that it is time to play. So he goes to her, he tries to chase her, and they interact. Now, the thing is that in order for Odin to know what he has to do whenever he sees any of them, he needs to recognize them. So what we know is that the studies in the primate brain tell us that the visual information enters through the primary visual cortex, but then in order for the primate brain to recognize an object or to recognize something, it needs to process information in the ventral temporal cortex. Now, when we look at the studies done in the carnivore brain, in particular, this is the example I'm showing you from the cat, we know that the primary visual cortex and the secondary visual cortex are similar. So these two are the same tissue. These two evolve in both species. But after the primates and the carnivores separated, the evolution took the visual pathway through different stages. And we don't know which region if there is a region in the carnivore brain that is similar to the temporal ventral cortex, or if there is a region that is encoding the information about the categories. So my question is, does the dog cortex possess an analogous region to the primate ventral temporal cortex? And in order for us to answer this, we train 12 dogs to remain still inside the scanner. We also scan 13 humans. So we were playing them videos of different things that I'm going to show you here. The videos are videos that, as you will see, they are moving, they are changing. You can see very different things going on in the environment and the different things that they are showing. But in general, they show you videos of cats, dogs, humans, and as a control, cars. Okay. So what we did then is that using representational similarity analysis, we took a portion of the human brain, a region that we know what it is doing. So we know that the early visual cortex is, I'm marking here the primary visual cortex. We know that it processes low-level visual information. So we took a sphere in the human brain, and we searched in the dog brain for regions that are performing a similar encoding than the human early visual cortex. But then, in order for us to find the ventral temporal cortex, we did the same. So we took spheres in the ventral temporal cortex, and we look at the patterns of activity of the human brain when they were looking at our stimuli, and search for these similar patterns, but in the dog brain. And this is what we found. So now I'm showing you here the overlap in orange. And in general, what we found is that the early visual cortex of both species seems to be the same as the previous studies are telling us. But in the case of the dog, the region that is encoding something similar as the ventral temporal cortex in human is more spread around the cortex. It's not focused in a particular region as the human brain, but it is also covering part of the early visual cortex. So our conclusion here is that the primate brain seems to have evolved to have very separate regions that are performing different processes in general. But in the case of the dog, the cortex is not so specialized. Seems like these processes expand from the primary visual cortex, but they also cover other parts of the temporal ventral cortex. So that's it.
Great, thank you for that. Oh, I'm getting a little bit of feedback. Nope, now I'm not anymore. Okay. So that was a great talk. Very cute dog. We have plenty of time for questions. So if people want to ask a question or post in the chat, if you have any questions or comments. We have one question that's just come in. How did you search for a region? It seems to be too many degrees of freedom associated with such a process.
Yes. So what we do here is that we create a dissimilarity matrix. So we take the processes, we compare how the brain is processing the different videos. We create this dissimilarity matrix. The dissimilarity matrix is telling you how different a particular brain region is processing a pair of stimuli. So we take all of our videos. We build this dissimilarity matrix using the patterns of activity of the regions of interest. In this case, of the primary visual cortex and also the ventral temporal cortex. We then search in the dog brain. Now the problem is that you might get many false positives. So in order for us to control this, we use permutation testing. So we repeat the same process, but relabeling all our stimuli prior to the analysis. And we build a distribution on how we expect, how many correlations we expect to find that are not really of interest, that are just noise. Yeah, and using this distribution, we can then find out if the correlations that we're finding are actually accurate. They are actually representing the stimuli. Yeah, have I answered your question?
Great. If you'd like more clarification, just post in the chat. So we have another question. We have several questions. So one question is the visual cortex ROI seems, and the human seems like it corresponds to peripheral rather than foveal vision. So what do we know anything about dog eccentricity? So like how dog's periphery might be represented differently.
No, actually, we don't have that many information in particular about dogs. There are studies that have looked at the visual pathway of cats, but they are invasive studies. In the case of dogs, this is a growing area. We expect to, in the future, be performing several other experiments in which we can look at particular details of the visual pathway.
Yeah, it would be really interesting to do like a retinotopic mapping study or something in the dogs, if possible. That would be I think very enlightening in how the visual system is organized.
Yeah.
Of course. So here's another question. Were you able to eye track the dogs to ensure that they were watching the videos?
Actually, the groups that have been studying dogs in the scanner, many of us have tried to use eye tracking. It's really hard to use it. The dogs have this white area in the eyes, so it's really hard to track what they are looking at. What we do here is that we put a camera outside the scanner, and the camera is looking at the face of the dog. So we just know whenever they are falling asleep or not. And after we take the scan, if the dog move a lot, we discard the scan. But in general, we know that they are looking because they have their eyes open.
Yeah, I'm very impressed that you're able to get your dog dogs to stay still. My dog would certainly not be as compliant. Here's a question. What was the rationale for the choice of cars as your control stimulus?
We wanted something to which the dogs have some experience, an object that they have seen in their life, and also an object that moves. So we know that they have certain familiarity with cars, and they know that the cars move. So that was it.
Cool. So here's perhaps a slightly longer question. Since their category representation is spread out in more than in humans, do you think that dogs are slower at categorizing? So is the cortical spread going to indicate some kind of speed of processing? Is there any behavioral or neuroscientific study that might indicate how fast or even well dogs process different categories of stimuli?
That's a really interesting question. I don't know of any study that have made this comparison, it has many problems. One of them is that in order for you to ask a dog, you need to have certain paradigm in which the dog can answer. But then you will need to control how fast the dog can answer in comparison to a human. Another approach is to do it using EEG, and in the case of the group in which we are working here in Budapest, they are trying to do EEG with dogs processing different words. They can probably give an answer about this, but I will expect a very similar processing speed. I mean, it shouldn't really change that much. We're talking about 200, 300 milliseconds.
Cool. I'm going to ask a question, which is that that's kind of been hanging in my mind, which is that like you've only taken a very small region of human ventral temporal cortex or occipital temporal cortex, perhaps. But we know that for humans, there's quite a large spread, and the representational pattern that you're going to get really depends on where in ventrotemporal cortex you sample, or whether you sample take, for example, the entire ventrotemporal cortex as a single region. So my question is, how did you decide to put your ROI in a specific spot? And have you considered sampling more widely across human ventrotemporal cortex to establish a representational pattern?
Yeah, that's a great question. I've been thinking of this like for over a year. So what I would like to do is like take every portion of the human brain and test it against every portion of the dog brain. And I actually tried several times of this. But if you think about it, the analysis is huge. I mean, if you take every single voxel, you will have millions of maps. I tried different approaches. I also use hierarchical. There is an analysis using functional connectivity that reduces the areas that you can use. So it creates like a mosaic on the brain. It splits the brain in different regions that are coordinated between them. So I try to use this. So I break the human brain in different parts, like in 200, 500 parts, and I try to search for regions in the dog brain. I couldn't find something really good, like something really consistent. So I stopped doing this. What I show you here is just like the tip of the iceberg of all the analysis that we did. But in just general, we choose these regions because they had the highest peaks when we use multivariate pattern classification. And we assume that, okay, so these are the regions that are performing better in the classification. So probably are the regions that have more information about the categories. But we also test some other things, like whether some dog regions have increased tuning against different categories of the stimuli, and other models, like a model from the primary visual cortex.
One potential way to do your results is actually to kind of flip it on its head, and rather than look for regions in the dog brain that show similar representation to the human brain, would be to look in the rather than look in the human brain and then try to find those representations in the dog brain, would be to like take parcels in the dog brain and try to find matching representations in the human brain, and then do a little bit of reverse inference there. I don't know if you've thought about that at all, but that could be interesting too. Yeah, sorry, I didn't mean to cut you off, but it's very interesting talk. We're gonna have to move. We're gonna have to move on to our final talk of the session. But feel free to keep the conversation going with the attendees in the chat. And there is one additional question for you in the Q&A. So thank you again, Raul. Our final talk is going to be given by Ajat Azimi, and it's titled Representation of Spatial Frequencies in Neural Populations of Human Temporal Cortex.
Okay. Hello, everyone. Thanks for accepting our invitation to this keynote. I'm Ajat Azimi, a master's degree student in artificial intelligence at the University of Tehran. The title of my talk is Representation of Spatial Frequencies in Neural Populations of Human Temporal Cortex. Actually, this is the result of our latest work, which I'm going to talk about. Let me start my presentation with a little game. Here, all of you must probably see a little red dot in the center of your screen. I want you all to just look at it. Please just focus and look at it. Don't blink or anything. Okay, 1, 2. Please don't look anywhere else, just look at the red dot. 1, 2, 3. Okay, as you must have seen, for a short period of time, you must have seen a picture. But the question is, what did you see? Could you describe it? How was it? Was it animate or inanimate? Like the picture on the right. Was it more similar to the chair or to the dog or to the apple? The next question is, did it contain any human faces like the picture on the right? And finally, was it a blur photo or a sharp one? Maybe you can answer these questions or no. But we have these questions in our mind, whether our brains are our brains capable of answering these questions or not. To answer these questions, we designed an experiment. Our experiment contained 155 grayscale images, including 73 animate images and 82 inanimate images. We showed these images, actually you can see our stimulus set on the right side of the screen. We showed these images to one subject and recorded 6 signals. Our subject was actually a pharmacologically intractable epileptic patient who had 50 electrodes in his temporal cortex. We had selected 5 areas in the temporal cortex, including right hippocampus, right and left supplementary motor area, right pre-supplementary motor area, and the right anterior cingulate gyrus. We had 10 electrodes in each of those 10 areas. After that, we set our paradigm for our experiment, which was really close to the little game that we did together in the beginning of the presentation. Each trial started with 100 milliseconds of fixation. After that, we showed the stimulus for about 200 milliseconds to the subject. And after that, the subject had to choose whether he has seen a face or a non-face picture, following a 500 milliseconds of rest. Okay, after actually recording these signals, we wanted to see if our signals have any information about the questions we had in mind. We started by visualizing the signals in the form of ERPs. Here you can see the ERPs of the 3 categories of signals based on their spatial frequencies on the right side. Here we have plotted only the signals from right hippocampus. But let's have a closer view. Let's compare each categories. Actually, let's compare them in groups of 2. Here we can see that by comparing the categories 2 by 2, there are some points in each trial that there are significant differences between each of the 2 categories. This means somehow that maybe actually a different representation of this type of images in the brain. To discuss this more, we actually trained some classifiers. Here you can see the performance, the accuracy of our SVM classifier through time. We did the classification 2 times. Once for a face versus non-face classification, that you see here. Here we have the whole population of neurons on the left side of the screen, and on the right side of the screen, we have trained our SVM by the use of only the right hippocampus. We can see that by as the time goes through, our classification accuracy goes high, which means the perception of the image category actually. After that, we again repeated this experiment, but this time to classify between high spatial frequency versus low spatial frequency. Here again, we can see the in the form of bar charts with standard error drawn in red lines, that our accuracy goes high, and again, there's a significant information behind the signals that we have recorded. After that, we wanted to see how distinguishable our signals are from each other through time based on their categories. To do that, we did a separability index analysis. Let me have a short review of separability index. So in order to calculate separability index, we first calculate separability between each class, between actually the subjects, the samples of each class, which we call here actually as indexed I. And the total within class scatter is called as index W. We then find the scatter of between the classes, which is called as index B. Finally, our separability index is calculated by this formulation. Okay, now let's see how our SI changes through time. Here we can see how our temporal dynamics of SI is. Here the 0 indicates when we are showing the stimulus to the subject. As you see, the signals start to separate from each other through time right after we have shown the stimulus to the subject, and it has a nice peak here. So we can say that there is there is almost information in the form of our signals. Actually, that's it because of the short time that we had, but we have done much more analysis that I like to show to you, but I don't have time. So if you want to know more, I'd be grateful to show them to you. Feel free to contact me. Thank you.
So thank you. That was a really great presentation, really clear. We actually, so if anyone has questions, they should post them in the Q&A. You do have time to share the results there if you would like to, just to generate more questions. Otherwise, we can open the floor for discussion, whichever you'd prefer.
I believe it is better if we let them ask their questions.
Great. So if anyone, when anyone has questions or comments, please post them in the chat or the Q&A. I wanted to just get your thoughts on the spatial frequency representation in the hippocampus. That seems like quite an interesting finding. Do you mind going back to that result there?
Sure. Which slide do you mean?
It was keep going back. I believe this is it. This is spatial frequency in hippocampus, right? Can you go right? Can you go one more?
Yeah, I think this is a better slide, right?
Yeah, this one here. So in this case, the plot that we should be looking at if we're comparing the spatial frequency dynamics is the leftmost plot, correct?
Yep.
So it looks like you've got kind of an antiphase oscillation, whereby there's like periods of time where, especially early, you can see like right at about 200 milliseconds, and then again at 1,000 milliseconds and 1,200 milliseconds, it seems like there's this like pretty strong oscillation. Did you have any thoughts about that?
Yep. Actually, after reviewing the literature in this field, we saw that the same results have been seen, and there's actually other experiments being done by invasive methods. And I believe this is actually the time that it takes for information to transfer from the eyes of the subject to the back of his head. Actually, I mean, to go through the temporal cortex. That is the reason behind this dynamic, I believe.
Right, but I guess that it's interesting that the phase of the oscillation they seem to be an anti-phase. I just didn't know if you had any thoughts on whether information might be coming from a different stream, like perhaps subcortical stream for low spatial frequency information, for example, because I think early signals about faces are propagated, at least to the amygdala, not necessarily the hippocampus, rapidly from a collicular input. I think there was a paper about that at some point. But I really think that the fact that you see a difference between high and low spatial frequency is very interesting. Yep. So we've got time for one more quick question if anyone has one. So I'll give it another couple seconds for that. In the meantime, we've got messages coming in that you had a very clear presentation, so that's great. And with that, I think it's actually about time that we can adjourn the session. So I want to thank all of our speakers again. These were all really great and interesting talks. I think it was really cool work. I'm glad that you had the opportunity to share it with us. Thanks to the audience for listening, and we hope you guys come to future NeuroMatch sessions this afternoon. All right.
Thank you.
Have a nice day, everybody.
We'll get started. Yeah, let's just give it a few more seconds and then I'll give my intro spiel. Okay, so I think that we have critical mass. Looks like we've already got over 50 people in the room and more people joining every second. So we'll go ahead and get started. We have a tight timetable to stick to. So my name is Sean Escola. I'm an assistant professor at Columbia University. And I'm really actually very interested in behavior. I study the motor system. And I'm interested in the idea of like motifs of behavior. So what are the sort of behavioral primitives? And so I've been really interested in the literature about how we take really rich, complex measurements of behavior using video and other techniques, and then decompose those measurements. And we're really lucky today to have two of the absolute bleeding edge people in the lab in the world doing this research in their labs. So we'll have two presentations by these groups, and then have some time for discussion. So first up is Mackenzie Mathis. Mackenzie is an assistant professor at EPFL in Switzerland. And before that, she was in Nao Chita's group at Harvard, and had actually a little bit of an interlude after that, where she was working independently at Harvard as well. And then we'll switch over, and Liam Paninsky, who I actually know reasonably well, he was my PhD advisor many years ago, is a professor of statistics and neuroscience at Columbia, and has been exploring many different approaches to applying statistical approaches to analyzing different kinds of data, and has recently gotten interested in the problem of analyzing behavioral data. And so he and members of his group will also give us an update on ideas that they've been having. So with that, I'll kick it over to Mackenzie. So go ahead and get us started. Thanks a lot. Glad everyone's able to join.
All right. Thank you so much, Sean, for the introduction and for inviting me here today. I'm excited to speak about our Toolbox Deep Lab Cut. And I also am very eager and excited about the notion of being able to deeply analyze behavior and using these sort of modern techniques to sort of decompose behavior into a dimensionality that's maybe more tractable to actually study. All right. So just as a bit of a segue to introduce myself a bit to the community, in general, our laboratory is situated in sort of this triangle of neuroscience, engineering, and computer vision. We use mice as a model system, and we're very interested in understanding how the motor system can be dynamically adapted on both short and long time scales. And so part of this quest to really understand how the nervous system drives these types of behaviors, we've sort of embarked in the last few years on developing new tools and techniques utilizing computer vision in order to study this in greater detail. And of course, I won't have time to talk about this today, but we also work on using deep learning tools to model the system for both the sensory, i.e. proprioception, and motor side. Okay, but of course, we all sort of have this problem of trying to understand, if we want to understand the behavior, like the motor system and those neural circuits that drive it, there's this big gap in between understanding, the limb of this animal and what type of cortical information processing is going on. And so part of this is just being able to measure this behavior with really high accuracy is really crucial for understanding these circuits. So I'm not going to talk about this work in great detail today, but feel free to check out our research on our website if you have more questions. So the notion of measuring movement, of course, is not new. People have thought about this for a very long time, including, I'm sure, this famous example that you know from Edward Muybridge, where he had a scientific question asking if all four limbs of the horse were off the ground at the same time. And moving slightly more modern, of course, David Marr and others have shown us that, getting the gestalt of an animal down to sort of the skeletonized version actually gives you a lot of information about not only what animal it is, but the motion of this animal. And so in modern times, we can use marker and markerless tracking. And so things from marker-based, of course, humans are very compliant. We'll stand in front of green screens, and you can easily track these dots and digitize them nicely. But this is pretty painstaking to do. So here's a really elegant example from John Donahue and Michael Plaque many years ago, where they used marker-based tracking to understand the motor cortex of monkeys making complex 3D movement reaching. But the field of deep learning sort of just blew up onto the scene around late 2014, 2015. And in particular, this subset of computer vision called pose estimation has really taken off. And so we know now that we can essentially track the key points or sort of skeleton representations from this pixel space and do this with really high accuracy. So for example, here's open pose with humans arguably in the wild on a pier in Sydney, dancing, the camera view is changing, the image statistics are changing dramatically, and yet we can still track these individuals. So for those of you that might be newer to deep learning, essentially they work like this, but actually in my talk, we're going to start to actually break this down a bit more. But in general, the idea is you have an image in pixel space, you have a predictor, and you have a pose that you get out at the end. The catch being that you need, of course, to train this predictor, and typically you need a lot of labeled data to get such performance as you see above. So for example, this is trained on a data set of about 25,000 different images, around 40,000 different individuals, and then it can generalize quite well. But that's a rather daunting thing to ask of a laboratory to be able to label so much data. And so we wanted to take a different approach to try to see if we could kind of use this in a lab in a meaningful way. And so we thought about this in 2017 when Alex and I started, and we sort of came up with a list of challenges that were quite unique to animal pose estimation. So first of all, animals have highly different bodies. So we didn't want to make a rigid solution like building a skeleton or having constraints a priori if they weren't needed. We didn't want to have to label a lot of data. We wanted really accurate 3D postures. We eventually wanted this to be really fast because I wanted to be able to do closed loop optogenetics, as I'm sure many of you do as well. Of course, animals don't all exist in isolation, so we wanted multi-animal tracking approaches. And in particular, we're quite interested in this notion of generalization and robustness in both machines and the brain. So this opens a lot of new research avenues for us as well. Okay, so deeply inspired by a really lovely algorithm called DeeperCut that was published in 2016. We started to look at these different high-performing human pose estimation networks to try to see if we could sort of use this in the laboratory and what it would take to get down to very low data loads. And so here is the toolbox that resulted from that, which is called Deep Lab Cut. The general idea is that you take images, you label these images with your hand annotations, or well, computer-based annotations, and then you train a network end to end, and you have a decoder layer that predicts not only the location, but can refine this location in space and give you a confidence readout of how good the network is actually doing. And so the video I'm showing you on the right is a mouse that was not in the training data set. And so these are images that were labeled, trained on this network for a few hours, and then this can generalize to other mice in very similar looking settings, i.e. here in my laboratory. Okay, and so just a couple key takeaways from this original paper. One was that importantly, you didn't need thousands of images to do this with really high accuracy. You could get away with a small number of frames. So on the left, you're seeing a plot that is showing the distance and errors from the human ground truth annotation to the network prediction. So this Euclidean distance between them in pixel space, and then the number of training images that were needed to achieve that accuracy. To give you a sense of scale in this data, the mouse's nose is only 10 pixels wide. So anything essentially below 10 pixels was really good performance for the question that we had. So that was resulted in about 50 frames getting really nice performance. On the right, you're also seeing that even if you have a network that has only seen one animal in this context, you can put more animals in this similar environment and will actually generalize to new animals and even find multiple animals. So the power of these networks is quite impressive, and we'll
higher order cognitive processing, it says there. What I'm going to rely on, and actually I think the community relies on, is this image on the right-hand side, which comes from a paper from Massimo Scanziani's lab back in 2013, where they described, if you like, the canonical interneuron circuit. And one of the reasons why it's nice to rely on this is because all of these subtypes that you can see here, we've got a somatostatin positive interneuron, a VIP positive interneuron, and a parvalbumin positive cell. We can target these reliably using Cree driver lines. And so we can explore their function, explore who they talk to across in vitro to in vivo models. So this is what we're sort of going to take forward. What I do want to do now is just sort of recap briefly, so we're all at the sort of same level, interneuron development. So a whole host of labs have contributed to this body of knowledge, and I've just listed a number down below in alphabetical order. But the simplistic view is that there are a number of neurogenic niches in the ventral forebrain, and I've put two here, the medial ganglionic eminence and the caudal ganglionic eminence, that will give rise to cortical interneurons. And these will tangentially migrate up into the developing pallium, into the cortical plate, where they will then integrate with the pyramidal projection neurons, which are glutamatergic. The medial ganglionic eminence is largely defined by a transcription factor called NKX2-1, and this is an image from my lab where we have used the NKX2-1 Cree to drive GFP in those interneurons. And then we've also done immunohistochemistry for parvalbumin and the neuropeptide somatostatin, which gives us, two of those populations defined in the Scanziani canonical interneuron network. Now, in terms of when the cells are born, work that we did in the Fischel lab established that the parvalbumin and the somatostatin from the MG, color-coded blue here, are some of the earliest to be generated. And then at a delay, this sort of diverse array of cells from the caudal ganglionic eminence, which all express the ionotropic serotonin receptor, the 5-HT3 receptor. They're born slightly at a delay and then will sort of migrate upward. I've just removed all the detail from this picture because actually one thing that will then strike you, if the remaining dashed line that I have running here segregates the boundary of layer 4 and 5A of somatosensory cortex. And what you will see is that there's a much more dense sort of GFP profiles in the deeper layers, and they're much more sparse in superficial layers. And this is, we now know this is sort of reflected primarily in the differential distribution of somatostatin interneurons, which are primarily located in infragranular layers, as opposed to the 5-HT3 receptors. And I've picked out VIP positive interneurons in particular, which are in supragranular layers. The parvalbumin cells, we find them pretty well all over. What you will see in this immunohistochemistry is that we've got the odd red, the odd blue cell, which is not GFP positive, because they're coming from other areas of the basal forebrain, and Oscar Marin in particular has been exploring those neurogenic niches. Then the next thing to point out is actually that in the adult, we have the canonical cortical circuit, if you like. So sensory information will come into layer 4. It will then go forward into layer 2, 3, where it will be processed sideways.
Early next year as well. Okay, so coming back to this notion of data augmentation, you actually have a lot of options. So it's not that just those, say, 50 labels that you've actually labeled are going into the network and being trained. We do a lot of back-end augmentation. Namely, you can do things like rotate and scale and crop, or invert the image, or if you know you're going to have a lot of motion blur, you can actually blur these images already. And that goes into the training to sort of augment, if you will, the capacity of this network to learn in all these different instances. Even things like fog and snow, if that's something that you imagine encountering in your research, is possible to add in on the back end. And so actually, I just wanted to highlight too, this primer comes with all the code to reproduce these figures that I'm going to be talking about today. So you can play around with this and also use these techniques in your own networks. Okay, so really the kind of take home from this part is how do you get the most out of your data? And so what I'm going to show you is a video using 3 different data augmentation methods, and the initial training set is the same. And notably, the mice were labeled such that they're kind of always just facing the left of your screen, if you will. And so we have on the far right scale and crop, which would just change the size and draw cropping boundaries, versus an ImageAug package that was developed, and TensorPack that allowed for more rotations. And so even just this simple selection of saying picking ImageAug or TensorPack on the exact same data can dramatically increase performance. So you're seeing this kind of diagram where here, for example, there's some switching between the nose and the tail because the network just never got any training examples of such a switch, but we can essentially save that with data augmentation, which is a nice thing to be able to do. Another really important feature that I highlighted, but I want to come back to, is this notion that you can evaluate performance at this stage, both in terms of metrics, like in this pixel space errors, but also just looking qualitatively at the data, like these videos I just showed you. You'd be kind of maybe surprised to know that the scale crop actually had lower pixel error, but then if you look at the performance using ImageAug, for example, you would notice that it's qualitatively higher. So here on the bottom left side, what you're seeing are plots of the percentage of key points that are considered accurate. So if you take one key point and draw a bounding around it, you can say within this small bound, you can consider that an accurate key point. And so this is a case where you have different mice are in the data set, but all with the same camera on the top row, and then different mice, but with different cameras, but it hadn't seen this. And so you see the network performance is quite poor. This is a very different camera situation. But then if you can just add a few frames from this new camera and quickly retrain this network, you actually can rescue this performance. So you yourself can also use this notion of transfer learning to continually to improve your own networks. So even if in your own laboratory, say you switch mouse strains and it looks very different or it's a different color, you can just add some new frames and retrain that initial network to get this performance back up to where you really need it to be able to do your downstream behavioral analysis. Okay, also within our package, we have some post-processing tools for you. I mean, of course, we don't provide the full pipeline of saying doing all the clustering and behavioral analysis, because there's a plethora of amazing tools to do this with. But we do give you ways to sort of clean up your data and make sure that it's as good as you want it to be. Oops. And so on the top left, I'm just showing you in one line of code, you can apply different models like filtering, Kalman filters, or an ARIMA model that's used for time series analysis, like predicting the stock market. And on the right side, you get some qualitative metrics of how the tracking is looking in a particular video, and then a readout of the confidence of the network, which can kind of be indicative of the quality if you're going to scale this up across a larger data set. It's helpful to look at these types of metrics and do a little bit of pre-processing on your data, or post-processing, if you will, from Deep Lab Kit. Okay, so we're really embedded in this larger ecosystem where we work in computer vision space, but it's fully built on the Python stack. We provide user interfaces to both do large-scale pipeline computing. You can deploy to Amazon, to Docker, to DataJoint, also, of course, to Google Colaboratory. There's neuroscience-specific tools that have Deep Lab Cut integrated, and there's a lot of nice tools now that are using the outputs of Deep Lab Cut to do types of behavioral analysis and downstream quantification and modeling. So coming back to this really quickly, I'm just going to give you one brief slide on the next three things, since some of it is published or being talked about in other places in NeuroMatch. So 3D pose estimation, we discussed how you can use multiple cameras to get quite nice reconstruction by training only one neural network from all camera views. And that was highlighted in our Nature Protocols paper last year. We recently released code and tools to do real-time feedback based on posture. So tomorrow, Gary King, who is a postdoc in my laboratory at Harvard, will be discussing this in great detail. So please go see his talk. He's a fantastic speaker as well. But nonetheless, just to give you a bit of a teaser, you can do this very quickly with using anything from powerful res nets to more shallower but faster mobile nets that are even designed to be run on cell phones. And then, of course, for offline processing, you can get much faster speeds. This is really for real-time processing only. Jesse this morning talked about our newest edition with multi-animal tracking, which is a really complicated problem, as hopefully you can attest from these videos. But he's built some really nice solutions along with Alexander to really try to tackle these problems as best as possible. And so in the very last few minutes, I think I have 10 minutes exactly, I'm going to just talk about this notion of what we envision in order to make more generalizable and robust networks for the community. So for example, what I've told you so far is that we've given you the tools and the resources to sort of swiftly tailor your own customized neural network for your task at hand. So whether that's locomotion, a mouse on a wheel, or a horse, or rat reaching assays, so on and so forth. There are a lot of common things, though, that we could be harnessing collectively as a community to build these resources in a more sustainable and reproducible fashion, versus each of us making up maybe potentially our own niche solution. So for example, there's a lot of behaviors that people do where animals are head fixed, they're doing go, no-go tasks or discrimination, or potentially pressing a lever, or of course, open field assays and behaviors across different rodents. Or reaching something that's near and dear to my heart, but I'm not the only lab doing this, right? So you can think on a larger scale about how can we actually combine our efforts to make tools that will work for everyone, or at least be a really good starting point for you to do transfer learning on top of this. So maybe, for example, if you have a rat doing a reaching assay like this, if you had a network that was already trained and very good at particularly, identifying this rat and these types of reaches, but you have a little bit of a different setting, then, for example, you might be able to take someone else's network, retrain it slightly, and then use it in your own situation even better. So we really wanted to build this in a scalable way. And so, you can think about this problem from many different levels, but one of them, for example, is this, namely, one laboratory, one experiment. I think this is, nearly solved. Experiments within a lab, right? Like if I have a mouse in one behavioral setup and I have a mouse in a different behavioral setup, of course I can train a neural network that will work across all these different setups and different camera views and different lighting consistence, right? The networks really have a lot of capacity to learn all of this. The next question is how well can it work maybe across labs? If I give you my network, how well would it work in your laboratory with maybe different mice in completely different setting and different individuals and very different diverse backgrounds? So I was alluding to some of the research that we're doing there. Another kind of larger question is, what about an animal group? Could we do this across all mice and rats? Could we make a network that could effectively identify all of these different animals? Or maybe across all mammals, right? Like, let's just dream big. And so we're trying to tackle this from a sort of computer vision's perspective and thinking about generalization and robustness. But at the same time, we know that data is really effective at doing a lot of this for us, right? I told you about data augmentation, but of course the input data is really, really important to use this with. And so we recently released this website called modelzoo at deeplabcut.org. And the idea is that we're actually providing you pre-trained networks on particular classes of animals or problems. And these are now trained on many different individuals, for example. And so just to give you a sense of what this looks like, if you go to this website, you'll see, of course, what different models are currently available. If you have a cat or a dog at home, you can see one example user from Twitter that just tried it on his dog or her dog that looks quite different than the example I have on my website. And this is all done in Google Collaboratory, so you don't have to install anything. You just click on this button and you can just run. So you just connect to Colab, you'll get a GPU. You can upload your favorite video, and you actually even get the whole project folder and the weights already downloaded. So if you want to take this now and use it in your own research, you absolutely can do that. And if it doesn't work for you, hopefully the idea is that you can get in touch with us, we'll make it better, and then share it back to the community. But to do that in a systematic way, we just released, I guess, a few weeks ago from two talented students, Stefan and Maxime. They worked on this beautiful web app. So if you go to contrib@deeplabcut.org, you'll see lots of different animals that you can just click and label. And eventually this type of interface will also be available for you as an individual user to use potentially a fully online version of DLC without having the pain of installing any GPUs. But again, we're trying to mitigate that a bit by giving you interfaces to Google Colab so you don't have to install anything except for just clicking through the buttons. But in general, the idea of the data that we're getting in are things like this dog, where you have multiple users have clicked on the same point. And so this also opens up some nice ways to think about, what is the ground truth key point in this instance? And then we throw all this data back in and continue to improve these models in an iterative fashion. Okay. So yeah, I hope today actually just to try to build some intuition about how these tools work. And hopefully if you're interested in using DeepLabCut, you can find all the resources for this. But in general, most of the packages available now are having this similar workflow. And for me, I'm sort of envisioning a future where you can sort of immediately say, is there something pre-trained? Can I use it? Can I build on it? Can I make it better? Can I add more key points or ignore some? And then you kind of just run through this pipeline in whatever fashion you envision. Okay, so hopefully in this short talk today, I've convinced you that DeepLabCut is an effective tool to build customized deep neural networks for your tracking problem at hand. Animals have highly different bodies, and so this really allows you to build your own tailored solutions as best as possible. You don't need a lot of training data to do this. A few hundred frames or even less is often very much sufficient due to transfer learning, as I described, and pointed you to some more resources. And then using efficient data augmentation, of course, helps in this many ways. It can be quite fast, so you can do 100 frames per second with ultra low latency, as you can find out more tomorrow, or please check out the paper. You can also go to dlclive.deeplabcut.org to find more resources about using the online version in your laboratory. We provide graphical user interfaces, so it's really just click and play. We have support for both 3D and multi-animal tracking, and we're really working on this vision of providing users this robust and generalizable tool for pose estimation to be used in neuroscience and beyond. So with that, I'd really like to thank the people in my laboratory that do a lot of the work with me. Of course, Alexander Mathis and I started this off together, and it's been a great collaboration. Jesse Lauer, who's the lead developer now, and Gary Kane, who developed the DLC Live package, and the other students that I called out, of course, and the funders that keep our doors open. And I'm looking forward to your questions and discussion later. Thanks so much.
Well, thanks, Mackenzie. I appreciate the presentation and the update. So we have a couple questions. Maybe I'll kick it off if you don't mind. I was really interested by this question. Is it's not necessarily the case that a object classification network would need to have information that's then useful for POEs. So the fact that the transfer learning works is it wouldn't necessarily have had to. And I was wondering if what your thoughts are on that. If there's anything you guys have done to sort of encourage these sort of classic ImageNet, ResNet kind of networks to have the geometrical information about body pose as well as object information, and how that sort of interplayed in your ability to be successful with tracking.
Yeah, no, it's a great question. Yeah, I think that's a good point, right? It wasn't immediately obvious that this would be the right solution. So I think in some sense that was sort of our contribution to the field in many ways. But yeah, so I think what we're, I guess in layman's terms a bit, the idea is that you're imbuing these networks with good natural scene statistics ahead of time versus just random weights, right? And so they've seen, a lot of different images. Like ImageNet is, a million images of random things, but they're real world things, right? They're like cars, bananas, there's mice, there's animals in the data set already. And so, for example, you can treat that as a bit of a segmentation problem as well. And in fact, Deep Lab Cut is using it as kind of treating key points as a segmentation problem. And so we're sort of predicting exactly which pixel it should be. And then we have this location refinement layer that goes in to clean this up so you can get even sub-pixel accuracy. And in the newest version, we actually added like limb vector fields such that you can get even higher precision and performance if you want by you build kind of edges along these different nodes in the bone. That works in multi-animal, so it's really helpful to do an animal assembly such that like animal A is and not animal B. But even for single animals in challenging cases, this is a really helpful addition in many aspects. Yeah, so I'm not sure if I fully answered your question because I still think there's a lot of questions about transfer learning that we're working on. And so I didn't mention it, but one thing we definitely have set up in the last year is a benchmark for this. And so we're interested in using like kind of real world size data sets and really pushing the bounds and looking at the robustness across this diversity of networks. And so that data set is released, and so people can also play with this in their own hands as well. So.
Cool. Well, we've got a few audience questions. So Aaron Wong asks, how does the tracking accuracy compare to traditional marker-based systems, particularly for 3D and real-time tracking?
Yeah, that's a great question.
And Gunnar had a follow. Sorry, just for a second. Gunnar just now had a follow-up. Gunnar, one of the Neuromatch organizers.
Sure.
I'd be particularly interested in derivatives, for example. Many people want to know how to analyze.
Very little in layer 4 at these early ages, at the P4 to P6 window. But in the knockout, we only see local GABAergic input. This looks very weak, and the reason I would propose for that is, work from Gord Fischel's lab and others has shown that somatostatin cells are really important for the maturation of PV interneurons. And I think what we're seeing here is that we're really not getting, the PV cells aren't maturing, and they're certainly not compensating. So I would maybe argue at this stage, there's no homeostasis. In terms of thalamic input, we got quite a pronounced result. At the early ages, we saw inputs in the wild type, again shown in blue here. But in the conditional knockout of SNAP25 in these somatostatin cells, we never saw a response. Now, it does recover. It recovers quite strongly by P7 to P9. So, just a few days, well, immediate few days after this silent period, we see thalamic input. So this inspired us to ask, actually, what is the impact in terms of perception? in these animals. And so we've now moved in vivo. And what we do is we do a multi-whisker stimulation using a piezo. We record using a silicon probe in S1BF, having confirmed the location using intrinsic optical imaging and post-hoc, because we label the probes with dye. And we collect a whole range of activity. We've got spontaneous spindle burst activity that we see postnatal day 5 to postnatal day 8, which is this earliest window. We look at sensory adaptation, so we do multiple stimulations of the whiskers. And we've also looked at speed encoding. And because this is a brief talk, I'm really only going to touch on the spontaneous spindle burst activity that we see and the sensory adaptation. So this is the work of Liad Baruchin, who's a postdoc in the lab. And I should say that Liad is on the job market, so please do snap him up, because I think you'll agree this is really fantastic work, and they're not trivial experiments to do. One of the key tweaks that Liad did, and you know, I think this is a little stroke of genius, was we not only did our somatostatin cells, which is the ones I'm obsessed with, but we also did VIP interneurons, so we can target those with the VIP CRE. And again, we cross these with this SNAP25 to conditionally silence that population through early development. Now, why do the VIP cells? Well, the thought is they're late born. Okay, they're coming from this caudal ganglionic eminence. Maybe they're late to integrate, although I have to say you should check out the work of Cristiano Bagnoni from the lab, which is now on bioRxiv, which actually negates that. They contribute to the early circuit. But the other thought was, if we silence these, maybe we will sort of boost, we will remove this inhibition of the somatostatin cells, and we might see like the opposite effect to when we selectively silence the somatostatin cells, because we'll get disinhibition of the pyramidal network through this canonical interneuron circuit. Okay, so first of all, let's have a look at the spindle burst activity. Now, spindle bursts in adults are associated with sleep, but what do neonates do? Well, they sleep. That is their behavioral repertoire. And there's a lot of work out there. I mean, immediately I'm thinking Rustem Kazapov's excellent work, Heiko Luhmann, and many others that have explored spindle bursts and their contribution to network maturation. And so this is done postnatal day 5 to postnatal day 8. We have a wild type at the top, somatostatin VIP. And really the take home of this slide is that the spindle burst duration is normal, no matter what interneuron population we take out. The average frequency within the spindle burst is roughly 15 Hertz, is what we would expect. But when we conditionally silence the somatostatin interneurons, we see reduced number of spindle bursts, or at least we observe a reduced number of spindle bursts in neocortex. If we then look at spontaneous multi-unit activity in these animals, what we see is actually a shift through development. So the first thing that is really obvious, and it's significantly different, is that at postnatal day 5 to postnatal day 8, we see this massive reduction in multi-unit activity in the somatostatin silenced animal. They seem to recover, and everybody is the same as we go from the critical period through to just before active whisking, at which point we see that both in the VIP and the somatostatin silenced animals, and we see excessive spike activity, as you can see in these traces up in the top right. Now, the way we're thinking about this at the moment, and I would love some feedback if you have thoughts, is that we know from Rustem Kazapov's work that the early spike activity is almost entirely associated with spindle bursts. So we're observing fewer spindle bursts in neocortex. So it's not surprising, therefore, that we get this significant, or we see this significant effect early on. We also know from Alison Barth's lab and many others actually that somatostatin interneurons are controlling network activity in the adult. So this would explain basically removing some inhibition from the circuit. So that's why we're getting much more spike activity, spontaneous spike activity. And finally, in terms of the VIP cells, this sort of fits with some excellent work from Renata Batiste-Brito and when she was in Jess Cardin's lab, which shows that VIP cells actually are inhibiting pyramidal cells at rest. And this is something we've seen in my lab when we've interrupted or have interrupted VIP signaling in the developing brain. Okay, so let's move on. And this last few slides that I'm going to go through, we're going to look at the sensory response adaptation in neocortex. So to test this, say Leah did 2 stimuli at a range of interspike intervals. I'm showing you one here, 250 milliseconds or 0.25 seconds. And we then analyze the local field potential, which I'm going to argue sort of reflects the lambic synaptic input into layer 4, or certainly synaptic activity within layer 4. And we're also going to look at the multi-unit activity, which is spiking the neuronal activity within layer 4 itself. We have focused primarily, and certainly the data I'm going to show you now is focused primarily on layer 4. The reason is it's easy to compare across ages. I'm showing you the P5 to P8 here, and you can see this really strong layer 4 response and the multi-unit activity. So it looks like there's slightly more multi-unit activity with this second stimulation. And at later ages, yes, we see more activity outside layer 4, but you can just about make out, although maybe it's dirt on my computer screen, that there is some sporadic multi-unit, low levels of multi-unit activity in other layers, but just restricting our analysis to layer 4 means that we can more readily compare across the whole of early development. Okay, so one of the sort of first interesting observations that they had in relation to this. Draw your attention to this gray, light gray line here, which is the paired pulse ratio at various interstimuli intervals at postnatal day 5 to postnatal day 8, which is the time period when we would expect the layer 5B for loop to be present. And what you can see is that essentially we're getting almost failure of the second response at these early ages. Then it sort of peaks and then it drops away again. And there's some thought that maybe this is sort of engaging later circuits, interneurons, some sort of reverb here. There's certainly the lack of early responses, probably because the thalamus is just not transmitting the information to the cortex. Then again, up to the lead up to active whisking, we still have a similar relationship, although it's flattening off here. And then we hit the sort of what we would like to call active whisking stage, and you can see that it's significantly different from this. Now, when we silence somatostatin cells and VIP interneurons, we don't really see much effect. The only possible effect is that we could point to is that with the VIP cells, it looks like we're not getting this second dip. And so maybe they're involved in this sort of late integration when you've got stimuli 1.5 seconds apart. But really, the impact is relatively minimal on the LFP. And this supports the notion that we're just silencing the cortical interneurons, we're not really affecting thalamic information coming in. However, if we now look at the multi-unit activity, the first thing to note is that P5 to P8, we actually see facilitation in the second response, and you saw it in that 0.251 example that I showed earlier. But across many animals, we see it almost selectively at 0.5 ISI. This sort of fits with work again from Rustem Kazipov's lab, where they did intrinsic optical imaging and see this large hemodynamic response in the early S1BF at the same frequency, and also work from Heiko Luhmann's lab, which has very convincingly showed LTP in vivo at the same frequency. So this sort of fits with their data, which was very reassuring to see, because in the somatostatin animal, we see no facilitation at all. It basically plateaus off. It's a completely flat line here. So this would suggest to us that somatostatin cells are contributing to that facilitation in the second pulse. And it's not just a general interneuron thing, because when we silence the VIP cells, we're seeing a similar response. In actual fact, maybe slightly enhanced, which would back up our argument that the VIP cells are potentially engaging with the somatostatin cells early on. So I'm going to bring this to a close now. What hopefully I've convinced you in this short talk is that layer 5 somatostatin cells are important. They, as I say, we've mapped the circuit all the way through development, and we see them sequentially engaging with the layers that the thalamus is engaging, that sensory information is moving into. And why is this? Why in S1BF have we seen these somatostatin cells? Well, I think it means they're ideally placed to meet those sensory processing requirements. They're in deep layers, they're early born, and they have these ascending axons. But, and I really must stress, and this is the last slide, all interneurons contribute. And the work of Rosa Cassat, Natalia DeMarco here, and indeed ourselves looking at the VIP cells, Christiana's work, suggests that every subtype of interneuron is involved during early development. But the system has evolved to make the best use of what is available to it at developmental time, okay? And this is an interesting thought. By the way, I should say this diagram here is one that we were going to put together for our SFN mini review. And it's just making this point that there are many routes of information into the early cortex subplate, the layer 5B somatostatin cells, and the 5-HT3ARs in layer 1. What I like about this is it's almost like a sandwich. You've got the bottom and the top of the sandwich, and eventually we're going to have the filling in the middle, actually the processing that's going to come forward from layer 4 to layer 2, 3. I'm just going to put this up and take a sip of water. Other cortical regions, of course, have different requirements. And this is a thought I want to leave you with, because S1BF, we've got to get it online early. We've got to get it up and running, because touch is going to be important to this animal. V1, we know from the work of Takao Hensch and many others, has much later critical period. It is sort of into the 4th postnatal week. So we might expect that it's going to use different interneurons. It's going to have a different repertoire of interneurons that it can rely on in order to sort of enact that critical period. We might want to think about M1. And how M1 relates to S1BF and so on. And, I think this is a really intriguing question that remains to be explored. We've touched on it slightly in Christiana's work, where we looked at the emergence of the long-range connection onto VIP cells. And that's also sort of kicking off fairly early. And then I have two fantastic colleagues in immediate space in Oxford, Armin Lack, who does prefrontal cortex, and Adam Packer, who's exploring the claustrum. And they've really taught me to think about other cortical areas. I've got to get out of S1BF. And these are particularly interesting because obviously they're getting, substantial cortical connections. I should also mention, obviously, we've got Ileana Hanganu-Opat and my former student, Paul Anastasiades, who's now set up his own group in Bristol, who again are looking at these questions of the early circuits in prefrontal cortex. But my guess is they're going to have different interneuron circuits, so they're going to rely on different scaffolds. And we already have some evidence to that, and this is the last thought I'm going to leave you with. We have from the Tolius lab, we have evidence in the adult that layer 4 interneuron circuits are different in sensory areas. And those of you that heard the Worldwide Neuro talk from Gord Fischel, but if you didn't, you can go check out this bioarchive paper that they've released, where they've done rabies tracing to look at long-range connections onto neurons and find differences. Okay, so each sensory area is going to rely on the interneurons that it has to compute that information and get it, the information through into cortex. Okay, that's it from me. I just need to thank, obviously, all my funders. This is the lab at the moment. This was a photo taken this summer. The hardcore patching squad, all wearing appropriate PPE. And particular mentions to Leah Baruchin and Filippo Getzi, whose work I've shown today, the in vivo and the subplay. Also Andre Marco Smith's CoMind, and Paul Anastasiades, who's now a PI at Bristol University. And of course, everybody else in the lab who contributes and explores the wonders of early interneurons, and my collaborators in Oxford. Okay. All right. Hopefully I'm now back on screen. Okay. So I guess we still don't have a host, so I'm going to just go through the questions. Yeah. Okay. So first up, we have one from Jimmy Dooley. Hold on, just making sure we've got everybody here. Yeah, okay, I'm supposed to give some moments, but let's get on with Jimmy's question. Could you elaborate a little bit more why you think an ISI of 0.5 Hertz is facilitated, and then why that facilitation goes away when you remove the somatostatin interneurons? Is that a behaviorally relevant ISI? Great question, Jimmy. Okay, why do I think 0.5 Hertz is the frequency that's facilitated? I think that's because that's what the network can deal with. Now, what we know is through development, you can see facilitation at, increase. Actually, sorry, I should say it's 2 Hertz. It was 0.5 ISI. Some people have shown that in adults, 8 Hertz is a good frequency for facilitation. I think that's what the somatostatin cells can deal with. Now, why do I think the somatostatin cells are important? Because actually they're the GABAergic input that's binding the activity of the cells, of the spiny stellate neurons in layer 4. They're effectively synchronizing them so that they can then fire in an effective way. Maybe, as Heiko Luhmann showed, we might see long-term potentiation. So my guess is that, as then parvalbumin cells mature, again, because the somatostatin cells are driving that maturation, the activity is, thalamic input is driving that maturation, we can then start to sustain increased frequencies, getting up to sort of, like the 8 Hertz. I hope that answers your question. What happens when we remove the somatostatin cells, of course, is because then, it's not so, we're not readily coordinating the cells, we're not synchronizing them. And as such, we don't see that facilitation, but there's plenty more to go at. Okay, I'm going to go to the top. Matt Buchan. Hi, Matt. Good to have you online. Hi, Simon. What are your thoughts about the subtype specific role of somatostatin neurons during this developmental process? Is it the same neurons changing their inputs, outputs morphology, or is there some die-off changing of regard, etc.? That's another really good question, Matt. You know, we, with Andre's study, we actually targeted a very particular subtype using the LPAR1 EGFP line. And that's the population that we get with these amazing morphologies. There are obviously other somatostatin cells within layer 5A, 5B that have other morphologies with some with sort of amazing sort of lateral axons and dendrites. Given that morphology of the LPAR I showed you, where it has the axon going down into the subplate and the axon going up all the way through to the marginal zone to layer 1, I do think the LPAR population are coordinating across the layers. Do other somatostatin cells also contribute? Yes. We've tried to get a handle on this with some of the wonderful sort of transcriptomic markers that are coming out of the Allen Brain Institute. And there was a wonderful paper from Alex Nacker, gosh, I guess a couple of years ago now, where he also explored the diversity of layer 5 somatostatin cells. I'm doing what the community does, which is sort of essentially brushing over that diversity because I use the somatostatin creed line. But I think that diversity will become important, and I really hope that we will get better resources to target those subtypes and get at them. My bet is that it's the LPAR that's doing it all, but I'm biased in that way. I'm going to say that because that's what Andre recorded. Do I think some of them are dying off? Yes. You know, immediately I'm thinking Oscar Marin has shown very elegantly that there's a lot of interneuron death. But you know, that remains to be seen. Okay. Najat, let's have a look at your question. I hope I'm saying your name right. Somatostatin neurons receive high thalamic input early in postnatal development that is transient. Do you know the role of this early synaptic connectivity and why is it lost, since thalamic feed-forward inhibition in mature circuits is mainly mediated by parvalbumin interneurons? Do you observe this early thalamic transient connectivity on somatostatin interneurons in other cortical areas other than the somatosensory cortex? Okay, so great questions. Do I know what the role is for the early thalamic engagement? Well, I would propose that actually the somatostatin cells are almost providing feed-forward inhibition, and you know, so they've kind of taken the place of the PV interneurons, because that is what somatosensory cortex has to go with at this time, because the PV cells aren't mature enough to enable, you know, the processing. Why is it transient? I don't know that it is, and I think this is something we will definitely explore. Whether or not it's as strong, you know, I guess one of the things is, you know, PV inhibition is so strong. When layer 4 PV interneurons mature, they're slamming GABAergic inhibition onto those spiny stellate cells, and so maybe we lose the insight onto the somatostatin cells at that time, because their inputs are then going out onto, you know, the dendrites. So I don't know that they are transient. I guess, you know, we have the olig-3 Cree line, so we could check that, and that's a great point. I do think some other labs, that maybe Gord's lab, have shown that it is transient. I think we're of the opinion that it maybe sticks around. Your second question is the million-dollar question, which is what I was implying at the end. And yes, we have looked in other cortical areas, and we see different transient connectivity. Okay, we see different scaffolds, and maybe you know, hopefully we can all get together for Society for Neuroscience next year in Chicago. I will be giving a talk then, and I'd be more than happy to talk about that. But yes, we have mapped different cortical areas, and we do see different early interneuron circuits, kind of reflecting what Gord Fischel has talked about, and also what the Tolius lab have shown for layer 4. So I hope that answers your question. Okay, Priscilla Ambrosi, what do you think is the mechanism behind the changes in connectivity of somatostatin cells through development? Is the incoming thalamic input necessary for those changes? Yeah, great. What do I think the mechanism is? Yeah, I would love to know. What we have done is we've done a couple of experiments that address this point. The first is we used what is called the cortex isolé model. And this is André Gauffinier and Tazir. It's a science paper from a number of years ago. And this is an animal which has no corticothalamic or thalamocortical connections. And we see this layer 5B4 loop disappear at the same time. Now, Liad has done some in vivo recordings, and actually they're not completely cortex isolé. There is a very small delayed sensory input that's maybe coming through other cortical areas. And so there is activity in those animals. They have spindle bursts. And so we think maybe just the generic activity is causing that change. Then there's another experiment we've done, which is to return to the infraorbital nerve transection that we reported in Andre's original paper. And I want to thank my reviewer too, because reviewer 2 said to us, well, actually, you know, how do you know that infraorbital nerve transection is attenuating sensory input? We make that argument in the paper because actually if we do the infraorbital nerve transection, we see a prolongation of the layer 5B4 loop. It is maintained. Again, we've revisited that. And actually what it looks like is that there's excessive activity. And so we're maintaining the 5B inhibitory input onto 4 because essentially the cortex is panicking. It's getting too much activity. However it senses that, it's then, we're seeing maturation of the local PV cells, but we maintain that 5B connection as well, just to put an extra brake on the system, okay? So what do I think the mechanism? It is activity, and it is thalamic. And just to go right back to the start of the talk, you know, I guess what I should be pointing out is that the interneurons are like the primary responders, okay? They're, you know, the reason for the policeman at the front end was they're sort of controlling the activity within cortex, and they're making sure everything is appropriate, okay? And when they get inappropriate activity from the thalamus or from somewhere else, they have to deal with that, and they deal with it in some really intriguing ways. And that is definitely something that we're continuing to explore. Okay, the last question I can see at the moment is Pierre Orhan. What are the trade-off the system needs to make an S1BF compared to V1M1? Intuitively, why would it not use the same circuitry? Great question, and something we think about an awful lot. I think, you know, S1 is one of the first sensors to come online, and so it, you know, it needs to process that information in an effective way. And at that time, the somatostatin cells, the somatostatin cells are the only ones there. Why don't the other ones use it? I honestly don't know. One could imagine with V1, maybe it's because we don't need the tight input-output relationship between layer 4 and layer 5, because, you know, S1 has a direct sort of motor component as well. Okay, I'm being told privately that we're going to be kicked out in 2 minutes. Is it 2 minutes now? But hopefully, Pierre, that has answered your question, okay? But it's a great point. Okay. All right. Thank you very much. Look after yourselves, everybody. Interesting times, as they say. And hope to catch up with people at a live conference in the not too distant future. All right. Bye then.
New work for us. So we're very excited to share this with everyone and hopefully get some feedback at the end. So in this framework, we're going to start by taking a sequence of video frames and push them through a convolutional encoder neural network. This encoder outputs two sets of latents. So the first is a set of supervised latents, and these are required to reconstruct the ground truth markers. And these are output by DGP, for example. The second is a set of unsupervised latents, which should capture variability in the behavioral video that is not accounted for by the supervised latents. So if there's variability that's not being accounted for by the markers that you're tracking, then these unsupervised latents should be able to handle those sources of variability. And then finally, we reconstruct the frames from the combined set of latents, both supervised and unsupervised. Now, the loss function we use to train this model contains four principal terms. So there's one for the marker reconstruction and one for frame reconstruction. And there's another term that orthogonalizes the entire latent space, which helps with both interpretability and ensuring that the marker information in the supervised subspace does not creep into the unsupervised latent representation. And finally, there is an additional term that encourages the unsupervised latent representation to be a factorized distribution, which further helps with interpretability. So now I want to describe the latent representations learned by this model when using the IBL data, starting with the supervised latents. I also want to point out that for this portion of the talk, I'm going to consider two labeled key points in the video, the left and the right pause, as you can see labeled in blue and yellow in the upper right image. So here you can see the DGP markers for a segment of test data plotted in black, with the reconstructions from the semi-supervised model in blue. And as you can see, this model produces near perfect marker reconstructions. As a comparison, we next fit a vanilla variational autoencoder, which is a fully unsupervised method, which compresses the video data but does not use the markers to define the latent space. Once we've trained the variational autoencoder, we then perform a post hoc linear regression between these fully unsupervised latents and the markers, plotted here in orange. And you can see that we're not able to capture all of the marker variability. So this demonstrates that incorporating a supervised latent space not only aids in interpretability, but it also captures important variability in the video that may be overlooked by fully unsupervised methods. And this is especially important if you're tracking key points in the video that maybe don't account for a lot of pixel variance. So if you're tracking some very small body part that you care about, say like the nose or the pupil area or something like that, that's going to be very hard for these fully unsupervised methods to pick up. And so we can actually account for those by incorporating them into the supervised subspace. So now that we've seen the model can capture the marker information in the supervised subspace, we can ask how well that supervised subspace is mapped to the reconstructed frames through that frame decoder. So to make this more clear, imagine that we have a latent representation for a particular video frame. So we have values for both the supervised latents and the unsupervised latents. So the question is, what happens if we take the dimension associated with, say, the horizontal position of the left paw, schematized here with the blue dots, and move the blue dot up and down? We change that value, keeping all other values in the latent representation fixed. So if our model has correctly learned to map that dimension to the reconstructed frames, we would expect to see the left paw move back and forth horizontally in those reconstructed images. So this is in fact what we see. So our model has learned a single latent dimension that is mapped to the horizontal position of the left paw. And here the red dot indicates the target pixel value that we specify in the supervised subspace. So this sort of demonstrates that supervised marker is actually being mapped to what we expect in these reconstructed frames. And importantly, there is no vertical movement of the left paw, as that's accounted for in another dimension, or any movement of the right paw. So next we can manipulate the dimension corresponding to the horizontal position of the right paw, and again see that the model has effectively learned this mapping. Similarly, the model has learned to properly encode the vertical position of the left paw. Next we can see that it has also encoded the vertical position of the right paw, but in this video, the right paw doesn't actually move up and down that much in the training data itself. And so there's not a whole lot of dynamic range here. Okay, so similarly, we can ask how well the unsupervised subspace is mapped to the reconstructed frames. And this is interesting because it also gives us some idea of what information is contained in that unsupervised subspace. So now we'll choose a single unsupervised latent dimension to manipulate, fixing the remaining unsupervised and all of the supervised latent values. So for the first of the two latents, you can clearly see the jaw opening and the tongue extending, while other aspects of the image, notably the paw positions, stay constant. So we can apply the post hoc label of jaw dimension here. The second latent corresponds to local movements of the paw along the base of the holder, most likely capturing small movements not accounted for in the tracking. So here we can apply the post hoc label of local paw dimension. So the question is, what can we do with these latents? And the answer is that we can use them for a variety of downstream applications, but with increased specificity. So for example, we can segment just a subset of the video, for example, the paw or the face. We can decode individual dimensions of behavior beyond what's being tracked by the markers. And we can have more interpretable predictors for neural encoding models. So we plan on exploring all of these directions and more in the future with this type of model. So now I'm going to pass it back to Liam to talk about other future directions that we're pursuing.
Yeah, great. So I'll just kind of speed through this because I think we're kind of low on time. But clearly, you know, there's always much more exciting work to be done. Two particular directions that we're really interested in are, you know, as Mackenzie mentioned, multi-animal data sets bring their own challenges. I think we really need to kind of reformulate some of the constraints that we used in the single animal case to the multi-animal case. That's kind of work in progress, and hopefully next year we'll have something to say about that. One thing we have made a little more progress on is thinking about how to incorporate multi-camera information. So of course, you know, all the results we presented so far have been based on a single camera. But once you have multiple cameras, that gives you another huge source of kind of semi-supervised or unsupervised soft constraints you can build into your model to train better neural networks to perform tracking. So here's a beautiful case from Rick Warren and Nate Sautel's lab, tracking basically, you know, two views of the same mouse here. And what we're drawing down here on the bottom, these lines correspond to the epipolar lines, basically using information from the top view to provide constraints on the bottom view. So hopefully kind of, and then again, we can train the neural network using these multi-camera constraints to get better performance. So that's still work in progress. Next slide. The other big thing I think in progress, and this is kind of a general topic for computational neuroscience more generally, is really kind of constructing infrastructure that's reproducible and scalable and easy to use. So again, Mackenzie kind of nodded to this as well. These new tools are great, but in many cases you need to buy a GPU. You might need to maintain some complicated hardware. You might need to do a lot of annoying software installs, and I think that kind of scares away some users who might really want to use these tools. So obviously the kind of Google Colab approach is a great way to kind of skip some of those steps, but there are limitations on you know how long you can run and limitations on the hardware and the reproducibility of Colab. So with John Cunningham's group, we've been working on cloud approaches for solving some of these issues on AWS. So that work is described in this bioRxiv preprint by Taiga. Deep Lab Cut is actually hosted on this system now. This is actually the first system we got up and running on this platform, and then we're working on incorporating deep graph pose in this system as well. So anyone who's interested in that, please check it out, and we'd love to hear some feedback. All right, next slide. Yeah, so I guess we just wanted to stop here, add some pointers to the papers and the code, and we'd love to have questions. So thanks again to the organizers and to the audience for your time.
Okay, well, thanks Liam and Anka and Kelly and Matt. That was a lot of really cool stuff. I was thinking when I was watching, the final part with Matt's presentation, it struck me, why did the unsupervised latents have any locality in the, I mean, is that something that just emerges naturally when you're analyzing behavioral data, or is there something else that I'm sort of missing in the process that would give some possibility for interpretability of the unsupervised latents?
Yeah, that's a really good question. So we were actually motivated to move in this direction because initially in this BehaveNet paper that we have on the screen right now, we were using just standard autoencoders to do the video compression, just fully unsupervised video compression. And the problem that we were running into was that a single movement of a paw, say, would be represented across all of the latents. There was no notion of locality, as you mentioned. And for the purposes of that paper, that wasn't such a big idea, but some of the downstream tasks that we want to perform with these low dimensional representations, it would be really nice to say, this dimension is, a jaw dimension, this is a paw dimension, this is a tongue dimension, or something like that. And so, yeah, so there's a bunch of lost terms in this model, but the two that are important for your question, Sean, are the orthogonality of the latent space. So that's not something that a standard autoencoder has built into it. And we found that orthogonality is really important. It's kind of like a PCA type constraint. And then on top of that, the unsupervised latent space by itself, we constrain that to be, or we don't constrain that, we encourage that to be a factorized distribution. And so that really gets rid of correlations between the dimensions. And we found that that's important for getting this sort of localized representation that we have. So, the idea is that movement along one dimension should be independent of movement along another dimension. And so as long as, so the example that I showed with the jaw opening and closing and the kind of paw moving back and forth, as long as those happen at different times throughout the training data set, then ideally we'll be able to separate those into two different latent representations. Now, if those two things happen together all of the time, if the mouse moved its paw along the holder every time it opened its mouth, then we wouldn't be able to disentangle those. But as long as they, sometimes one happens, sometimes the other happens, sometimes they both happen, then that factorization in the latent space allows us to separate those two things.
I guess conversely, that would provide a path then to sort of identifying what are the highly correlated behavioral sort of, motifs across an entire body or field of view in a camera, and what are more independent or can be independent.
Yeah, exactly. Because if there are two say what we would consider separate movements that always co-occur, then in those reconstructed frames, we would kind of see them both associated with the same dimension, because there's no way for the model to learn that those are two separate movements. Yeah. Cool.
We have one question in the chat, in the Q&A. Folks, feel free to put any more in there. So Christian asks, this is super cool. How do you know the dimensionality of the latent space of key point movements? Do you have a loss on the number of latents?
Yeah, so the dimensionality of the latent space of key point movements, that's a number that is determined by the number of markers. So for example, in the IBL data set, there were 2 markers that we used, and each of those have an X and a Y coordinate. So the supervised latent space is just 4-dimensional. Now we could try to play some fancier games with that, but for now it's it's just a one-to-one mapping. Where you might do something slightly different with that is, say you have the video of a mouse face, for example, and maybe you want to use deep lab cut or deep graph pose to track the pupil. Then you might have 4 key points to track the outline of the pupil, and in that case you have 8 total marker dimensions. But you could reduce that into three, right? You could have like the center of mass, the X and Y coordinate of the center of mass, and then you could have like a pupil area or pupil diameter dimension. And so you could use 3 dimensions. But yeah, for it, it's just a one-to-one mapping basically. In terms of the loss on the number of latents, I'm guessing this refers to the number of unsupervised latents. Yeah, so this is a tricky question. Any sort of model that has some sort of unsupervised component to it, there's always the question of how many latent dimensions do you have? And in a lot of the data sets that we've worked with, we've found that this can actually be a pretty small number. It can be 2, 4, 10, something like that. But I can't give you an exact number. The way we do it is just fit a range of them and kind of look at the results. But unfortunately, there's no recipe for saying in this data set you should have 6 latents, and that's the final word on that.
Maybe I'll ask just one more question of this team, and then we can sort of invite everyone to have a larger conversation. Folks, please put some questions in if you want. So Anki, it seemed like the point that I took from your presentation was that the key to the difference between deep graph pose and deep lab cut is related to this enforcing the confidence map to be unimodal. I mean, there's the additional temporal and spatial constraints, but that's sort of like maybe more like extra sauce almost. The key idea was this unimodal confidence map, and it just made me wonder: is that the right interpretation? 1, and 2, are there other ways that might be imposed? Right? Is there something specific about the way that you chose to do it that was sort of the most natural and made the most sense? I'm just curious if you could give us some insight on that.
Yeah, thanks, Sean, for the question. Yeah, that is the one key sauce we add to this post-estimation model by imposing a kind of unimodal Gaussian constraint to the output of the confidence map. And at the beginning, we don't really have a strong intention that we want it to be unimodal, but it's more like we construct the unsupervised loss. We construct the loss for the unlabeled frame as the label frame does, and then it becomes very unimodal, and then we feel like this Gaussian bump is a very good constraint for that purpose. But this is actually only valid for single animal tracking, because we only have one body part we try to predict, and when you get to multiple animal tracking, you can't really just naively impose a one mode Gaussian to it. You have to come up with some other variants of structural constraint to the output, to the confidence map of the unlabeled frames. So I wouldn't say this is the only constraint we should add, but this is the most natural one for single animal tracking for unlabeled frames. Does it make sense?
I can add a little more to that. So we didn't start by saying these confidence maps should be Gaussian. Really, that fell out of the variational inference approach that we used to basically pull information from the unsupervised, from the unlabeled frames, and hand that off to the model. So we happened to choose a Gaussian posterior on these unlabeled markers on the unlabeled frames. If we had used a different approximation instead to that posterior distribution, then we would have gotten kind of different constraints there.
I see. Okay, so why don't we open it up? So obviously, panelists can feel free to engage with each other, and we can take questions from the audience. So the idea of developing these tools, of course, at least from my point of view as a neuroscientist, is then to be able to use them in the downstream applications of interrogating behavior, or maybe correlating it to neural activity, and looking at perturbations of behavior. And I'm just curious if you guys want to talk a little bit about your thoughts on how these, how has these efforts really pushed neuroscience forward? I mean, we can start there and sort of see where the conversation evolves. So, Mackenzie and Alex, you guys want to jump in and give us a go, or?
Yeah, thanks. I mean, as I kind of alluded to in my talk, I feel like there's two large camps within neuroscience, right? Those that want to be able to do, say, behavior classification, and those that are doing kind of motor control things. And I think in both domains, pose estimation has been fruitful. I mean, I guess, as others have pointed out, right, this is a relatively new tool. I think Deep Lab Cut came online in the spring of 2018. So it's also kind of nicely coincided with this whole neuropixels movement, if you will. And so there's been a lot of really nice work that others have done using these tools already in published papers, right? So it's been exciting to see like what type of neuroscience has been done by considering movement more. So as a movement scientist, I was excited to see more focus on behavior and movement. So.
Yeah, I would agree with that. I mean, there's really two types. So I think also there are two types of users. Ones that had always basically done motion capture in some sense, or shied away from it because of the tremendous manual cost. And then they are obviously immediately embracing it and immediately have questions. And I mean, the labs in mind, or the research questions in mind, is really a lot of motor control, a lot of hand tracking, a lot of arm tracking. And there have been amazing papers, including, yeah, well, I don't need to mention names. There are really lots of fun studies. And I think the other side is really there's a large fraction of people that kind of maybe they never saw the need to do motion capture, but now, as Mackenzie was also alluding to, there's also this kind of growing appreciation that behavior as an inference copy or for many other reasons is actually everywhere in the brain. So it makes sense to measure it, even if you don't necessarily do motor control or motor neuroscience.
All right. So maybe any response for that from anyone else, or we can keep moving?
I would. I think we agree with everything that was just said. It's really been, like I said, inspiring to watch this, movement, pun intended, kind of take off over the last couple of years, and really kind of transform a lot of the way we look at the brain and kind of think about how to approach interesting behavioral questions. I think the other, implicit thing here is kind of the community and the open sourceness of these tools, which has really kind of accelerated science, and really it's kind of a beautiful kind of positive feedback loop that's really kind of pushed the field forward. So that's been great to see as well.
Yeah, I agree with Liam here a lot. Like, I mean, Liam has been, for example, has been known for many years providing amazing tools. And I think historically, a lot of the tools, I mean, Liam can maybe correct me if I'm wrong, a lot of tools emerged as like small scripts in MATLAB that were built on a lot of code, of course, and then were maybe difficult to use. And I think what has been happening in the last few years is like with the growth of Python, with the growth of Stack Overflow and all these things, I think, there's been really an amazing emerging community of users, of students using tools, adapting tools, changing tools. And I mean, Deep Graph Pose, I guess that's the name, is an example, because I mean, it is literally built on Deep Lab Cut and has Deep Lab Cut source code in it, which is, I don't know, didn't count, but I guess it's like 75%, or I don't know. So I think that is really the amazing thing that is happening. And that's why open source, I think, is so powerful, because with permissive licenses, we can really build on each other's work. And if someone feels that some digit jitters in their 55 labeled frames, then they can put in a prior to have a unimodal bump, and then kind of improve the algorithm for this. specific need. I think that's why it's really so exciting to be in this field right now.
Yeah, definitely. No, I'm a huge proponent of open source, and as I'm sure as other developers in the panel and the audience know, it's also a labor of love, right? It's a lot of work to maintain this code. It's very easy to like publish a paper and then say it's open source and like dump the code online, but to really like maintain it and grow it and build that community is like truly now my hobby, right? Like it's a whole life force to maintain Deep Lab Cut, but it's really rewarding, and exactly as Alexander said, then you can see what people do with it and build on it in really cool ways. So
yeah, and in that sense, I think we should also give a nod, and I mean Mackenzie did it in her talk, to I think computer vision has, for example, a really amazing open source community, and
absolutely
we have both grown very much enjoyed that community and growing into it. And
yeah, I mean, I think it's apparent in I don't have behavioral data myself that I analyze, but I've been sort of casually observing the field, and just the interest and enthusiasm that has grown, I think probably largely around the ease of utility is so important. And on the Liam, you mentioned very briefly, but I know you and John have put a lot of effort into this project to get as many neuroscientifically useful tools online as possible in the lowest impact to entry way that is possible. And I think that's great. I really appreciate the effort for the community.
Yeah, I mean, I think Python has kind of come a long way in the last couple of years, but there's still a barrier to entry on the usability side, on the kind of maintainability side, scalability. People having to install and kind of maintain their hardware and software stack. So I've really, John has convinced me that moving towards kind of cloud solutions is really the way to go to make everything more easy to use and more reproducible and more scalable. So yeah, I'm really a strong proponent of moving in that direction.
Maybe we can change gears. I really want to talk about what do these approaches open up? Everyone's touched on that a little bit, but let's be as speculative as we can now. Go all the way to the end, to the limits. So what can we do with really highly detailed estimated body positions in humans or animals that we haven't been able to do before?
Well, so I'll start off with this one. So kind of referring back to the intro that Anchi gave, I think that right now experimentalists are able to record potentially very high dimensional neural activity, and also video data, which is very high dimensional. And if we really want to understand behavior in and of itself, or understand the link between behavior and neural activity, we can build a lot of fancy models, but these really require the data being quite low dimensional, right? We can have these complicated dynamics models that tell us how, what the action is that the animal is engaged in at any point in time, or, maybe we can try to infer what the torques are on various joints or something like that. But a lot of these models require low dimensional representations of the behavior. And so I think all of these approaches are really making those low dimensional representations richer and more detailed. And from there, we can really start to do the downstream work of connecting that with neural activity, or pulling out information about the kinematics or the actions or something like that. And so I think this first step is really important to enable those further downstream modeling efforts.
So it's I think you used an interesting phrase, make the low dimensional representation more detailed. It's kind of an oxymoron, but I guess what you mean is you want the low dimensions to be the useful dimensions. Is that the right way to say it? Yeah. Okay.
Absolutely.
Yeah, that makes a lot of sense.
Yeah. And I mean that for example, the classical work of Ronald Johansson, I guess, has highlighted that that's one of the interesting things about describing behavior as key points, for example. I mean, there are things that it cannot capture, but there are these famous videos where you just see a bunch of moving dots coherently. And if we watch them and they are the relevant key points of humans, for example, we can easily see the social interactions and what is happening. I think the other example, I mean, I kind of want to start off, I want to say something like in some sense, it just has made digitization easier, but I think as like one new thing that can be done that couldn't be done historically, that's really kind of closed loop feedback, I would say. And there I want to highlight maybe 3 studies so one recently from Daniel Huber, who had this paper in Nature Methods, his lab, and they showed, for example, so they have this really cool camera that follows an animal around in a 3-dimensional arena and thereby kind of tracks it, thereby centers it, and thereby has only very few pixels. So that is already a hardware solution for kind of getting the relevant low dimensional bits. And then they use deep lab cut to extract the pose. And then they showed several things. One of the things is they conditioned the animal, they basically tried to see if they can condition an animal to do a particular pose. And that, of course, no, you can only do if your algorithm doesn't run for many hours and only after the fact you know that the animal is doing this particular action and you can basically reinforce it. We have a preprint where we did something with a dog of a postdoc with deep lab cut live. The reason was during the shutdown we couldn't do experiments, so the first author actually worked with his dog. And Bob Dada at Cosine, I think maybe 1 year ago, they had a cool example where they basically used an online version of MoSeq, you could say, very briefly. And what they showed is that I think they stimulated VTA. And so of course, now if you take ongoing behavior of an animal, you can decompose it into an ethogram. So you have all these states and how you transition through it. And then one question that you can ask yourself, and that they asked themselves, and it's a great question, is if you kind of reinforce a particular action a lot, how would that affect the whole ethogram? Will it mean that neighboring behaviors go up in that graph, or is it generally everything like, so and that is really possible only with this online VTA stimulation. And that those, I think, would be three cool studies that I liked a lot, but there are many more.
Cool. We've got a couple of questions in the chat. So maybe actually I'll start with the one that just popped up from Chris, who is one of the NeuroMatch organizers. He talks about if you build tools for closed loop stimulation during EFIS experiments, that the latency is very important in these cases. So do you think the cloud solutions are going to be able to support these kinds of experimental setups?
Yeah, that's a good question. This is something we're just starting to work on now, so I don't have a definitive answer. Obviously, it depends on your upload bandwidth, particularly. I think there are certainly applications if you need true kind of millisecond latency where it's going to probably make more sense to do things locally. There are a lot of other applications where you might need tens of milliseconds latency, and cloud solutions will be kind of just a ticket. So I think it is going to depend on the application. There's certainly going to be some applications where you want to do things locally, but again, it's kind of hard to set up kind of real-time feedback systems. There's kind of a long list of things you have to kind of set up and get working properly. And so if we can kind of put solutions in the cloud, I think it's going to be much more impactful and much easier for people to use in a routine and reproducible way. I guess one other thought, kind of responding to the last question, I think kind of the markerless tracking stuff. One of the reasons it's so powerful is because you know exactly what answer you want to get out, right? You want to track fingers or paws or noses or fins. It's very easy to see if you're doing a good job or not. With a lot of the kind of downstream things like these ethograms that were mentioned, it's a lot more qualitative, right? There might be disagreement between multiple people about what is actually a different behavioral state. And so I think, you know, we've worked on these tools and contributed a little bit to these kind of temporal segmentation tools along the lines of kind of generalizations of MoSeq. But I do think there's still a lot more important work that needs to be done there to make those tools more robust and real time and interpretable. So I think over the next couple of years, I think there's going to be a lot of work in that area that needs to be done.
So Liam, I really appreciate you brought that up. As you know, I'm really interested in this idea of you know, being able to identify behavioral motifs, and you could do that. You could try to do that just from raw video data, right? You could say, okay, well, let's do some clustering.
Exactly. I mean, that's the approach that the MoSeq paper takes. That's the approach that the BehaveNet paper that we had at NeurIPS last year takes. And yeah, I think you're certainly going to do better if you have access to good markerless tracking. You know, why wouldn't you incorporate that into your representation and try to do a better job with that?
I mean, I guess the converse is that you're really implying sort of heavy priors, right? The things that you then are tracking. And so, you know, I guess that if your priors are right in order to identify what the appropriate motifs are, then that's good. But I you know, what about merging these data streams, right? Using, I guess in a way, Matt's a model that he presented is sort of about that, identifying different latents in the activity. But if you, it's not exactly the same as merging marker-based dimensionality reduction with others as a way to get to behavioral ethograms.
Yeah, I mean, I think Matt's work is exactly along those lines. So I'll let him answer.
Yeah, I mean, so you know, with that model, at the end of the day, what we get is a supervised and an unsupervised subspace, and we can concatenate those together and have this full representation, or it's, you know, not completely full, but a richer representation that contains both marker information and non-marker information. And then from that latent representation, we can then fit these downstream models that do behavioral segmentation, for example. And so that allows us to sort of incorporate that knowledge of what we do want to track from markers, but also allow us to capture other pieces of variability in the video that might be important for the downstream goal of segmentation in this case.
Have you done that yet to look and see what are the what's the ethogram you get from the video alone, from the markers alone, or from some sort of hybrid approach?
No, we haven't done that yet, but that's definitely on our to-do list. Another thing that I'll say that is maybe not so interesting neuroscientifically, but is kind of a technical issue that I've run into with some videos that I'm working with, is that there's actually mechanical pieces in the video that are moving around. And if you just do fully unsupervised dimensionality reduction in those videos, then you get latents that correspond to these mechanical pieces, or really the movement of the mechanical pieces is in all of the latents. And so one thing that we're doing with those is actually tracking the mechanical pieces, and you can put those, separate them into their own subspace, and then say everything else is actually the animal behavior, and that we can segment. But yeah, so that's definitely you know, to do on our part is looking at how segmentation or other things like that change as you incorporate more information.
Cool. So let's jump back to the Q&A. So we have a question from students, and I like this. It's pretty open-ended. These kinds of analysis, what can we learn anything about cognitive functions? Can we, you know, how does this open the great questions in neuroscience of what's going on in terms of cognition, and have you guys had any ideas about
well, I mean, I can give a cheap answer. Sure. You can use it to do pupil tracking, and the pupil is super relevant for cognitive behaviors. I mean, yeah, but that's of course very cheap.
Yeah, I mean, I would add that, of course, movement is the way that we interact with the world. So you can get a lot about intent of an individual or an animal by its prior history and its future movements, right? So that gets maybe to a deeper question about what is cognition and how can you measure it anyways. But I would still argue that having a clear and crisp and robust way to measure behavior is going to be really important on the quest for understanding consciousness.
I think you're exactly right. The idea is that when you look at a lot of studies in cognitive neuroscience, the triggers that are used to base inference are these very large scale phenomena. I pressed a lever or whatever it is, right? Yeah. Whereas if you can have this sort of richer behavioral representation, then there may be an opportunity to get at intention and attention and all of these other phenomena.
And I think this also goes into the clinical space too. Oh, sorry, maybe we're cross talking Zoom. I was just going to add that I think this also works in the clinical space really well, right? Getting away from scales that are 0 to 10, but making really meaningful quantitative metrics. I mean, most of us probably have more technology in our living room than we have in a hospital bed, and that's kind of shocking and a shame in many ways, right? So I think hopefully these types of technologies will also, and they already are, I mean, DLC has been used in 2 clinical trials, and there's many people developing like new stroke scales and Parkinson scales off of markerless pose estimation. So I think that's a really exciting space for, as a developer, like exciting to watch anyways.
And I mean, one hallmark of cognition, at least for humans, is certainly language, for example. And I mean, in language, we also know that there are a number of latents that play a role. Like you can tell, for example, now if I'm nervous or not. And all these things just from my voice, and like having kind of more quantitative tools that don't just get the symbolic knowledge in behaviors, but or the symbolic output that kind of subjects give, but really how their bodies kind of accompany that, I think is really important for figuring out how the brain works actually, or cognition works. But
yeah, I wanna just add a little bit things that I feel is very exciting, that based on their rich like behavior extraction from the video, something we can borrow from human research or human pose estimation is, or from robotic research, right? When people really get the rich representation of behavior, you can try to learn like a cognitive model, reinforcement model, to understand the reward representation, to understand how the animal represent the reward or value function during decision making, during like spontaneous behavior, and try to infer those kind of cognitive states from the behavior data only. I think that is a very interesting thing. Some people have done a lot of research in human and robotic science, but I think when we get to the stage where we can obtain very rich representation of behaviors, people can also do that kind of thing with animals. Yeah, I think this, yeah.
Yeah, I think just kind of one more note building on that. I mean, there's another question about, what can we do simultaneously with behavioral video and kind of large scale neural recordings, which is, of course, a topic that's near and dear to all of our hearts. I think, if you're interested in cognitive variables inside the brain, I think the last couple of years have really shown that you need to condition out the sensory variables, but even more importantly, you need to condition out the motor variables. So you need to model what's going on behaviorally, so that you can expose interesting kind of internal cognitive states. So I think all of these kind of things kind of play nicely with each other, and it's going to be fun to watch where the field goes over the next couple of years.
And I mean, another example along those lines is also, for example, know that how saccades of monkeys, if they're more confident, which is a cognitive variable, you could argue, influences how fast they make saccades and things like this. So motor really has lots of correlates of highly cognitive variables like confidence.
All right, well, I think we should probably wrap it up with that. I mean, let's go ahead and solve consciousness. We'll be back this time next year.
Perfect. I'm there for it.
So I really want to thank the 2 groups and our panelists. So that's Mackenzie and Alexander Mathis at EPFL, and Liam Paninsky, Anki Wu, and Matt Whiteway and Kelly Buchanan at Columbia. Thanks a lot. And we'll see you hopefully at some more of the events today and tomorrow, the last 2 days of NeuroMath.
Great. So we'll just continue to let people come in for another minute, and then I'll introduce the session.
The live stream is on.
Yep, I see.
Cool.
Thank you.
Sure.
All right, so I'm going to introduce the session now. Hey, everyone, I'm Adam Steele. I'm a postdoc at Dartmouth College in Hanover, New Hampshire, United States of America. I've been told that we have a few moments as people trickle in, so I'll use that time to tell everyone in the United States to please vote on Tuesday. Have a plan to vote. If you've got a plan or you've already voted, call up a colleague and tell them to vote. And if you aren't in the United States or you can't vote in the US election, please contact one of your US colleagues and make sure that they have a plan to vote, because it's extremely important. So with that, we've got some outstanding talks right now on coding in entorhinal cortex. As a reminder, we've got the chat where you can post messages and clarification questions. Just make sure that you have the messages set to panelists and attendees, so that way everyone can see what you're saying. If you'd like to introduce yourself now, so that way everyone knows who's in the room, feel free to do that. Just say your name and where you're from. That would be great. We also have the Q&A tab for questions. You can post questions at any time, and please do upvote your favorites to make sure they get asked. If the questions don't get asked on live, the questions will remain up and our panelists can comment on them and answer offline. For those of you who are on Twitter, please tweet about this session using the hashtag NMC3. So with that, I'll get started. Our first talk is from Holger Dannenberg from Boston University, and his talk is titled Neural Dynamics Underlying Coding of Location and Running Speed in the Medial Entorhinal Cortex. So the floor is yours.
Yeah, thank you very much, Adam, for the introduction, and thank you very much for having me here. It's great. Yeah, as you said, the title of my talk is Neural Dynamics Underlying the Coding of Location and Running Speed in the Medial Entorhinal Cortex. And before I start, I first want to thank all current and past members of the Hasselmo Lab at the Center for Systems Neuroscience at Boston University, where I'm a postdoc, and of course also the funding institutions that support our research, in particular the National Institutes of Neurological Disorders and Stroke. So let me start my talk with a simple thought experiment. Imagine an animal more or less randomly foraging in an environment on the search for food. Once the animal has found a piece of food, it often wants to return to its shelter to consume the food in a safe location. And the animal can do so, and it can do so by taking the fastest route possible, even if it has never taken that route before. And a key question is now how internal self-motion cues are integrated with external sensory cues to form a cognitive map of the environment for the purpose of memory-guided spatial navigation. And decades of research has shown that the hippocampal formation is central to the formation of a cognitive map. The activity of neurons in the hippocampal formation is temporally organized, for example, by theta rhythmic oscillations. Theta refers to a frequency range between 6 and 10 hertz, and is often referred to as the online state of the brain. And importantly, theta rhythmic activity depends on the medial septum and the diagonal band of Broca, which is a brain region in the basal forebrain. So in the following 10 minutes, I will first present data on the coding of location and running speed in the medial entorhinal cortex. And then in the second part, I will then examine the role of medial septum inputs for the coding of running speed. And in the last part, I will then present data on the effects of visual cues on neural dynamics for the coding of location and running speed as a function of time. So all the experiments I'm presenting today were performed in freely foraging mice that carried a light fiber for optogenetic manipulation of neurons in the medial septum, and tetrodes for multiple single unit recordings and also recording of local field potential activity in the medial entorhinal cortex. So let me start with a short introduction to grid cells. Grid cells in the medial entorhinal cortex have been hypothesized to provide a metric for space. And grid cells fire at multiple locations that fall on the vertices of equilateral triangles, thus kind of forming an hexagonal grid spanning the environment. In most computational models of grid cell firing is updated by integrating a velocity signal. And in this talk, I will focus on the nature of the speed signal in the medial entorhinal cortex. So current grid cell models rely on a linear running speed signal. And the existence of linearly tuned speed cells in the entorhinal cortex seems to fill that gap. However, many speed tuning curves are better fit by a saturating exponential function, as shown on the right side, as opposed to a linear function. And moreover, speed tuning curves do not allow conclusions about the time scale of firing rate modulation. So what do I mean by the time scale of firing rate modulation? Well, a firing rate is not a physical property of the spike train, but it is derived by integrating spikes over time. This raises a very important question, namely, which time window should be used for the computation of a firing rate signal. And I want to illustrate that here with an example. In the upper panel, a short time window is taken to compute a firing rate. As a result of using such a short integration time window, the firing rate shows fast changes over time and appears very variable. In the lower panel, a longer time window is taken, and that results in slower changes in the firing rate signal. And importantly, the underlying spike train is exactly the same in both cases. This raises the question, of course, which firing rate signal is most useful for the brain, and we cannot know the answer a priori. So in order to address that question, we computed the firing rates of entorhinal speed cells using different integration time windows ranging from as short as 125 milliseconds to as long as over a minute. And what we found is that the integration time window that maximizes the speed score, that is the correlation between firing rate and running speed, was in the range of seconds, namely about 3 seconds in rats and 12 seconds in mice. So this finding has implications for current models of grid cell firing. Because if the speed code by firing rate requires integration over seconds, the question is what happens to the regular hexagonal firing patterns of grid cells? And we tested it by using a model of grid cells and feeding in speed information to this model. As you can see in the panels on the right side of the figure, the spatial periodicity of grid cell firing was largely disrupted when running speed information was integrated over time windows larger than 200 milliseconds. And this data indicate that grid cells may use actually another speed signal. And one alternate speed signal in the hippocampus and entorhinal cortex is an oscillatory signal by theta oscillations. In particular, the local field potential theta frequency increases with running speed, as shown in the right panel. So previous experiments have shown that both theta oscillations and grid cell firing depend on medial septal inputs. Since grid cells may integrate running speed information, we tested the hypothesis that the medial septum is important for conveying speed signal to the entorhinal cortex. In order to test this, we optogenetically inactivated neurons in the medial septum by expressing the inhibitory opsin arginrhodopsin T, which is a light-activated inhibitory proton pump, and simultaneously recorded LFP signals and speed cells in the medial entorhinal cortex. So these experiments were performed in freely foraging mice during alternating epochs of a baseline and optogenetic inhibition of medial septal neurons. And by this, we demonstrated that optogenetic inhibition of medial septal neurons largely reduced theta oscillations in the entorhinal cortex. So the figure in the middle shows a time frequency plot where the frequency of local field potential oscillations is plotted on the y-axis and the time is plotted on the x-axis. The power of theta oscillations is color-coded with red colors indicating high power and blue colors indicating low power. And you can see that the theta oscillatory power almost instantaneously decreases at the start of the laser stimulation for optogenetic inhibition. And then theta power returns to baseline levels after the end of the laser stimulation. On the very right side, you can see that the slope of the local field potential theta frequency versus running speed relationship is also reduced during inhibition of medial septal activity. And taken together, this data demonstrate that the medial septum is important for potential oscillatory speed signal. But is the medial septum also required for a speed signal by firing rate? So to answer this question, we compared the distribution of speed scores between inhibition and baseline conditions. The speed score measures the correlation between firing rate and running speed. And we found that the modulation of speed cells firing rates remained unchanged on the population as well as on the single cell level. And these data show that the firing rate modulation of entorhinal speed cells does not depend on medial septum inputs and indicates that the brainstem pathway via the medial septum may not be the primary influence on the speed signal by firing rate. So one alternate way to estimate running speed is the use of vision, in particular the use of static visual cues or optic flow. Previous studies have shown that visual inputs are important for good cell firing, and a key question is how visual inputs are integrated in the coding of cell location. We therefore next examined the role of visual inputs for the coding of running speed and grid cell firing in the medial entorhinal cortex. So how do changes in visual inputs affect speed signals and grid cell firing? To answer this question, we recorded single unit activity in mice that randomly foraged in an open field environment during alternating light and dark conditions. And we observed large changes in the firing rates of many neurons, including speed cells. And as you can see on the left panels, those changes in firing rates during darkness resulted in changes in the slopes of the speed tuning curves. But then the right panels show the same data after normalization of speed tuning curves within conditions. And as you can see, the normalized speed tuning curves now look very similar across light and dark conditions. And normalizing firing rates within conditions therefore demonstrates that those changes in the slope of speed tuning curves, as seen on the left side, can be actually fully explained by the changes in mean firing rates of neurons. Conversely, the running speed dependent gain in firing rates of speed cells is not changed during darkness. So we next examined if the proposed oscillatory speed signal by local field potential theta is changed during darkness. And we found that the slope in the theta frequency versus running speed relationship was reduced in darkness. Previous data have shown that visual inputs sharpen the grid cell metric. And we found also that the spatial stability of grid cell firing was indeed reduced in darkness, but the spacing of grid fields was largely preserved. So grid cells are important for spatial memory, and this raises the question, what is the time course of those changes in grid cell stability after changes in visual inputs? And so we analyzed grid cell stability as a function of time. And by doing that, we could demonstrate that changes in grid cell stability show a fast and a slow component of change after transitioning between light and dark conditions. And this suggests that grid cells integrate velocity signals over time. And intriguingly, as you can see on the right side, the time course of changes in grid cell stability almost perfectly aligned with the time course of changes in the local field potential theta frequency versus running speed relationship. And consequently, the spatial stability of good cells was highly correlated with the slope of the local field potential versus running speed relationship, which we could interpret as an oscillatory speed signal. So taken together, these data indicate a link between the speed modulation of local field potential theta frequency and the stability of good cell firing. So to summarize all of that, in the first part, I hope I could convince you that the speed signal by firing rate becomes optimal at seconds-long timescales. And then I've demonstrated that optogenetic inactivation of medial septum inputs reduce theta oscillations in the medial entorhinal cortex, as well as the slope in the theta frequency versus running speed relationship. But at the same time, medial septum inactivation did not alter the speed signal by firing rate. And then in my last part, I showed that good cell firing is maintained in darkness, but with reduced stability resulting in larger grid fields. And this change in stability showed a slow component of change and was strongly correlated in time to changes in the slope of the local field potential theta frequency versus running speed relationship. So with this, I want to end my talk, and I'm looking forward to questions if there are any. Thank you very much.
All right, great. That was a really fantastic talk. Very impressive work. Emily Airy-Jones has posted 2 links to the papers that were discussed in the chat. So thank you for doing that. Balash has asked a question. Did you investigate the time scale of theta frequency coding of running speed?
Not yet. So it's currently on my list of doing that. So that's also very important. But what I can say is that, from looking at the data, that it's actually it seems to be that it's more similar actually to the timescales of the firing rate coding. So, you might think that the coding by theta frequency might actually be faster because, you have that potential. But then you also still have a lot of variability. So a lot of studies, like average theta frequency over time or over trials and so on. So a lot of that, short time scale variability is often smoothed out. And so we have to see if it's really, in the time scale of like 250 milliseconds or so, we're in our grid cell model, the grid cells were breaking down. So the analysis is still outstanding, but if I had to guess, I would actually say that it's not that fast.
Interesting. That's really interesting. If anyone has any other questions for Holger, you can post them in the chat, and he'll be monitoring that. Unfortunately, that's the only time that we have for questions from this talk, but thank you. It was really outstanding. Our next talk is going to be from Joseph Monaco, and his talk is titled Spatial Theta Phase Coding in the Lateral Septum, a Theory of Allocentric Feedback During Navigation.
Almost there. Okay, are we good to start?
Yep, you're good.
Oh, wait. Hold on. Crap. Sorry. 1 second. I had too many talks up. Okay. Well, I had some notes, but I can't show them at the same time, so that's kind of a... I will just forget about the notes then. Does anyone know if there's a way to do that better?
If you go into your screen sharing options in advanced, there's a way to just show a portion of your screen. That could work.
And you should play it in a window, I guess.
You can also share just the PowerPoint presentation. That worked for me.
Oh, okay, yeah, I got you. All right, so right there, and then stop the share. Yeah, I'm sorry, I'm wasting time here. Okay.
All right, excellent.
Excellent. Okay, sorry about that. Thanks for the introduction. I'm sorry for wasting a minute or two there. Also, thanks to the Neuromatch organizers for putting on this greatly needed meeting. So yeah, I'm a computational neuroscientist. Much of my work is focused on theories of the neural computations underlying spatial cognition, including interactions between place cells and grid cells. But in this talk, I'd like to describe some data and some modeling that potentially supports a theoretical role for septal neurons in maintaining the allocentric reference frame of the spatial cognitive system, at least in rats. Particularly, I'm going to focus on the question of path integration and how you might be able to maintain an allocentric reference frame during path integration, which basically comes down to the problem of how do brains reset the accumulation of encoding errors. So path integration, as has been described, is the critical component of navigation that contributes spatial information by integrating self-motion signals. Resetting is necessary because integrating self-motion accumulates errors in position estimates over time. And that's kind of illustrated by this green trajectory here shown for this journey. And so there's a lot of different ways of doing that. There's been two main kind of camps, theoretically speaking, for how to model path integration and how it might interact with landmarks and cues in the environment. One of my much older papers kind of explored the oscillatory interference mechanism, which is where if you have theta rhythmic oscillators that are also modulated by direction and speed, then you can combine them in different ways to form grid cells, for instance. Those were models from John O'Keefe and others, and Neil Burgess. But I showed that you could generalize this or genericize this into a place field, more of a generic spatial mapping model, where if you randomly, if you have random preferred directions with these VCOs, these velocity controlled oscillators, then you can build place fields that are stable and based purely on path integration. And this was work with Jim Knierim's lab of modeling his data on circular tracks. And so this is just kind of showing how the amplitude of synchrony can map into space through path integration with oscillators. But one of the things that I was really concerned about in this paper was how does a sensory cue actually feed back into the phase code in order to stabilize a phase code from drifting away. And so in the bottom panels here, you can see that if we posit some amount of phase error and we want that to be corrected at some point in time, that represents the interaction with an external cue, then I simply posited a very kind of abstract feedback process represented in this diagram on the bottom right, which is what I needed to be able to study this in terms of remapping and partial remapping. But this was definitely a black box. So this kind of really raised the big question, like, is there an actual neurobiological basis for having this kind of spatial feedback, particularly in the phase domain? So the question is there a phase code and oscillatory code outside of potentially outside of hippocampus that could serve a resetting function for path integration? And so this data is a result of a collaboration with Tad Blair's lab at UCLA, where he performed these very long duration recordings in an 80 centimeter cylindrical arena, basically standard random foraging tasks, while recording a reference hippocampal LFP signal. He recorded essentially from all of the subcortical brain areas highlighted here that are in one way or another interconnected with the hippocampal and entorhinal formation. And so he recorded from all of these areas, and I basically took this into a data analysis paradigm to look at the amount of spatial information carried in phase, in the theta phase, as well as correlating that to velocity and position and other characteristics. And I just to kind of give the hint of where things are going, I found this kind of spatial phase code, which I think might serve this theoretical role that I described only in one place, and that was in lateral septum. and lateral septum happens to be the primary subcortical output target of the hippocampus. So it's possibly the entrance point to an interesting feedback loop if we consider all the interconnectivity within these networks. And so I'm going to take you through what some of this lateral septal data looks like. And just as a brief overview, if you record what a theta signal is, probably this crowd doesn't need that. You can record the LFP, you can do a bandpass filter, find where the peaks are, and then we can take the spike timing from relative to peak to peak within each theta cycle, and we map that to the phase domain that goes from 0 to 2 pi. And just to be, so that explains the y-axis of a lot of the plots that I will show. And so here's just a standard spike trajectory plot of one of these cells, and you can clearly see there's this is a very long, I think a 2-hour recording. So you've got the gray trajectory there with the random foraging and the red spikes, and you can see clear spatial modulation. So if we look at the top map here, you can see that the firing rate, clearly illustrated. There's a broad place-like field and on the west side of the arena, which that's great. We've got spatial modulation in lateral septum. That's been shown sparsely in the literature, but it has been shown before. What hasn't really been shown is that relationship to the ongoing theta oscillation in the hippocampal system. So if you look at the bottom map here, this is a phase map of the average phase at every location. And you can see that there is a correspondence between the pattern of modulation in the rate map on the top and the phase map on the bottom. And so this is interesting. This is over a very long period of time, a very long recording, and there appears to be a very strong relationship between rate and phase. So that's kind of the basis of the idea going forward. And I quantified that by looking at the phase rate correlation, the circular linear correlation for basically the pixels in these maps. And you can see what that looks like. So you get kind of the expected negative phase rate relationship. And I termed these cells, I called them phaser cells. So these are lateral septal phaser cells, for want of a better word. We'll see if it catches on. But basically, we're going to analyze that correlation, but the correlation kind of immediately brings to mind that you can explain this with a fairly simple phase coding mechanism. So if you posit that there's some cell that it receives an inhibitory theta rhythmic input, such as the magenta sinusoid that you see here, and then you also posit that, well, maybe it also receives a slowly changing or slowly ramping depolarizing input, like the green triangle wave here. And that's all you need to get kind of the phase coding relationship that we just saw, where as the input increases, you start to get activity in the cell, and then you get more and more activity within each theta cycle as the input goes up, but then the activity within each theta cycle also occurs at an earlier time, or it initiates at an earlier time. So you have this joint modulation of phase and rate. And critically, once an input slowly ramps up, it can slowly ramp back down, and symmetrically you see the same exact thing. The phase will deflect all the way back up to the baseline phase once that input has gone away. So there's no kind of hysteresis or learning going on here. So this is a symmetric bidirectional phase coding mechanism, and something like this has been posited for phase for place cells in hippocampus before learning or any kind of network effects have come into play. But the idea is that this gives you a scalar code. So this is basically the co-modulation of phase and rate means that that phase is basically just a conversion of rate. It's analogous to taking the spatial information in a rate code and then putting it into the phase domain. And so we can think about functionally, why would you want to do that? And it is in particular a high contrast with what you see in typical place cells in the hippocampus. So on the right, I've taken a figure from Souza and Tort, the 2017 paper where they analyzed a large place cell data set and showed this kind of canonical unidirectional asymmetric relationship between phase and rate. So as the animal goes through a field, the phase will continually go down and it does not return. However, if you had a phaser cell field, as the rate goes up, the phase would advance, and then as the rate goes down, as the animal leaves the field, the phase would delay back up to the previous level. And so looking for this type of phase code, I set up a number of different criteria, well, 4 criteria, 3 important ones. So looking at the spatial phase information, looking at the total phase shift, like how much does it change within that correlation, and then the strength of that phase rate correlation, essentially. And then using those criteria, I was able to filter TAD's entire data set, the subcortical data set, all those single unit recordings into cells that meet these phaser cell criteria and those that don't. And particularly, it's important to look at whether this is actually a stable code or not. So just looking within session, I compared up to the first hour of a session, the early part, up to the last hour, the late part. And so if we look at on the left, both a spatial correlation, and on the right, a change in that total phase shift, this kind of illustrates that you do maintain spatial correlations across these long duration recordings, and the phase shift that comprises or that constitutes that phaser cell code does not significantly change. Most cells remain within about pi/4 or about 45 degrees across these multi-hour recordings. And then you also want that to exist across days, and that is basically what we found with the curves looking very similar. And between days with identified units, we can kind of track what each individual cell is looking like across days. And the vast majority of them do not have significant changes or flips in the direction in their phase shifts for this phase code. A couple of them do, but most of them are pretty stable, which is pretty good. That's what you want to see. But then the last thing besides stability is that you want to make sure these spatial responses really are spatial and not just a confound of spatial correlations of other aspects of the trajectory. So to kind of deconfound that, I trained a GLM, a generalized linear model, with both spatial predictors and trajectory-based predictors. And so these variables called LMQ, or just linear and quadratic. So this is up to second order spatial variation. And then the trajectory predictors are wall distance, speed, and direction, basically. So this top grid, this top grid is showing you that the responses are utterly dominated by the spatial factors and almost not at all the trajectory-based factors and other confounds. And even if we look at the maximum possible contribution that each of these predictors made, which is the bottom plot here, you still see that dominance of the spatial relationships, though that does kind of also reveal that there is this hint of a trade-off between how spatial the cells are and then how tuned to speed they are. If you look at the sorting along the fourth column with S here. I'm not sure if people can see my mouse, actually. I'm waving my mouse around a bit. So once that's kind of established that these are spatial cells and they are pretty stable, we have those criteria. We can kind of see where these cells fall. So on this plot, I'm showing all cells that had significant spatial phase information. And so that spatial phase information is on the x-axis, and then the total phase shift, that phase modulation is on the y-axis. And you can see we have these cells with a negative phase rate correlation here in this bottom part of the plot. And these circles are, the size of the circle correlates to how strong the correlation is. So these like nice strong phase rate correlations showing these strong negative phase modulations, giving us lots of information about space in phase, which is great. But then the surprising thing is that if we look at the top, we also saw cells. So the cells up here with positive phase shifts. This was surprising because of that simple mechanism that I implied earlier, that I described earlier. You wouldn't expect higher firing rate to correspond to later firing in that intuitive model. So there's probably something else going on here, and this potentially has interesting implications. So just to show some examples, these are 5 different examples of those negative phaser cells from different animals. The top row shows you the rate maps, the middle row shows those phase maps, and then the bottom shows the phase rate correlations, just like the example cells I showed earlier. And you can see there's like wall responses, there's place-like responses, place boundary, conjunctive responses, and then kind of like broader, but still spatial responses. So there's a strong diversity here.
Joseph, you have 1 minute left.
Darn, okay. So here's an example of the positive phaser cells, which are not as strongly spatial. So if you look at the phase rate, the trajectories of these cells, you can see across phase, they interleave very nicely. You can see in this plot here. And then if you look at the typical firing phase of these populations, you can see just how interleaved they are. So at any moment in time, you've got information coming in the phase domain about space from one population or the other. And then so very quickly, the modeling of this is that I had a dynamical circuit model with a very simple structure here using feed-forward suppression of the positive cells by the negative cells, and you can get this complementary relationship pretty much exactly, which is very nice. And then I use that GLM as a generative model to generate spatial tuning curves for both 1,000 negative phasors and 1,000 positive phasors. And I use those as an input layer into a set of downstream theta bursting target neurons. And so these are just random target bursting, these are random theta bursting neurons that are not path integrating or spatial. The question is, can we make them reset to a path integration signal? And so with different types of supervised phase codes, which I won't get into detail because I'm running out of time, we can actually see how well these codes were learned by these downstream target cells based on a very simple competitive learning mechanism. And so once you have that, we have a very small number of these cells. You can actually do population decoding of just the phase, and you can see that, well, this top structure didn't work very well, but the bottom one did. And that actually is going to lead to a very rapid phase resetting mechanism, basically a sub-second reset mechanism for path integration. So that's basically the idea, that we've got the fairly simple network structures and circuits, and using this kind of like location-based synchrony idea, there might be a subcortical pathway for phase code feedback in the spatial system that might support path integration and other elements of navigation. And I will end there.
Perfect. Okay. So we have to move swiftly on to make sure that Balázs has enough time for his talk. You do have a few questions in the Q&A from Eleanor, so I would encourage you to go check a look at that. And thank you again for this great talk. I'm going to move swiftly on to Balázs Uefilushki, whose work is titled Sampling-like Representation of Prospective Locations During Hippocampal Theta Sequences.
All right, thanks Adam for the introduction. And I also want to thank the organizers for the opportunity to present my work here. So I am Balázs Uefilushki from Budapest, and in this talk I will contrast different ways of probabilistic representations in the hippocampus. And this is joint work with Gergő Orbán. So assume that you are participating on a conference in the post-COVID times, and you want to find your way back to a nice restaurant in a busy city. You might use a mental map of the city to evaluate possible routes and to predict their probability of success. But the world is full of surprises and maps are inaccurate, so your predictions are inherently uncertain. In fact, the further you move into the future, the less reliable your predictions become. We are interested in how the uncertainty of these predictions is represented in the brain. So the hippocampus is essential for model-based predictions and planning. Just as we have seen in rodents engaged in spatial navigation, the activity of place cells repeatedly encode the trajectory of the animal in subsequent theta cycles. So starting from the past at early phases of the theta towards the possible future locations and the late theta phases. So this provides a unique test bed for testing representations of uncertainty. We know the variables encoded in the population activity, the location of the animal. The uncertainty increases systematically from the beginning of the theta cycle when representing past towards the end of the theta cycle when representing future. And data sets with activity of hundreds of place cells are recorded routinely for thousands of theta cycles, and these data sets are publicly available. So whether and how hippocampus represents uncertainty associated with these predictions during theta sequences is not known. There are several ways of representing a probability distribution in the neuronal population activity. First, in product of experts type representations, which is closely related to linear probabilistic population codes, or PPC, one can represent an exponential family distribution using their canonical parameters. Next, mixture of experts type representations, closely related to distributed distributional codes, or DDC, neuronal activity represents expectations of nonlinear functions under the encoded distribution. In sampling-based representation, the population activity can represent samples drawn from the probability distribution. And finally, in maximum a posteriori or map encoding, any variability in the neuronal responses is unrelated to uncertainty in the population, and the population only represents the mean activity. So first, I will focus on the product of experts type representation. So here, the distribution is represented as a weighted product of nonlinear functions, where the lambda weights represent spike counts associated with the canonical or exponential parameters of the distribution. In general, the entropy of the distribution decreases with lambda, the spike count. So in a population of neurons with tuning functions uniformly tiling the support of the distribution, the mean is encoded by the location of the activity hill, and the variance is inversely proportional to the amplitude of the hill. So in these product type representations, the firing rate is the lowest at the theta phase where the most uncertain future locations are encoded. And the question is whether we can observe this in the hippocampal population activity. So we tested this prediction in two different data sets, one on a linear track and one on an open field. So what is the theta phase that encodes the future locations? So when we plot the phase of a place cell spikes as a function of the position, we can see that the cell starts firing around 270 degrees. That is when its place field is still in front of the animal. Alternatively, we can decode the position of the animal from the activity of all neurons within a small time window spanning 1/3 of the theta cycle. We can observe that the average decoded location relative to the animal varies systematically within the theta cycle and shows a significant forward bias around 240 degrees. So this is the theta phase that encodes the future locations. The firing rate does not vary too much, and in contrast with the product of experts type prediction, it has a peak, not a trough, near the theta phase encoding the most uncertain future locations. We obtained very similar results in the open field experiments. Thus, product of experts type representation is not supported by this data. Next, we studied the DDC encoding. In DDC, the firing rates of the neurons correspond to expectations of the encoding functions or basis functions under the represented distribution. So intuitively, the variance of the distribution is encoded by the diversity of the coactive neurons. We developed the maximum likelihood decoder that estimates the encoded variance from the spikes in early, mid, and late phase of the theta cycle. We found that the estimated standard deviation was almost always near zero, indicating the encoding of a single value instead of a whole distribution. So the mean encoded as D was not significantly larger at the late phase of the theta, when the most uncertain future locations are represented. In contrast, we could recover the increase in the maximum likelihood decoded SD in matched control simulations. And again, we obtained similar results in the open field experiments. Thus, mixture type representations including DDC are also not supported by the data. Finally, we studied sampling-based representations. Here at each time step, a different sample is drawn randomly from the distribution to be encoded, and the population activity only encodes this sampled value. We assume that a single sample in this case corresponds to a full trajectory that is played out during a theta cycle. We reasoned that if an independent trajectory is sampled in each theta cycle, then the variability between the represented trajectory should be large, whereas if it is only the most likely trajectory that is represented, then the cycle-to-cycle variability should be small. Specifically, we define cycle-to-cycle variability as the difference between the two-dimensional trajectories encoded in two subsequent theta cycles. So the expected value of this cycle-to-cycle variability equals the sum of the decoding error plus the change in the mean encoded trajectory in the case of the map encoding, whereas it is larger by the internal uncertainty of the positional representation in the case of the sampling. So one could naively argue that defining feature of a sampling is an increase in the variability for more uncertain future locations. The cycle-to-cycle variability is higher at the end of the theta cycle. However, using synthetic data, we could show that this would be also true for all the other models. The expected mean change in the trajectory is larger for more uncertain future parts of the trajectory, as shown here in this little graph. So therefore, to discriminate between map encoding and sampling, there is one more step. When measuring CCV in real data, we need to decide whether it is small or large. To do so, we can compare it to the internal uncertainty of the positional representation that we can estimate from the data in the following way. So the posterior variance is related to the trajectory encoding error, which we define as the difference between the encoded trajectory as the true trajectory of the animal. Assuming that the animal's internal model of the environment is consistent, the difference between the true and the posterior mean trajectory equals the decoding error plus the sigma square, which is the internal uncertainty of the position representation, posterior variance. And similarly for the sampling, this is the decoding error plus 2 times the sigma square. After subtracting the decoding error that we can estimate from the data, the trajectory encoding error thus reveals the magnitude of this posterior variance. Now, if we compare this trajectory encoding error with the cycle-to-cycle variability, CCV, the difference is what we call the sampling index, which is going to be positive for sampling-based representations, and it's going to be negative for map encoding. Okay, so we validated our approach using synthetic data matched to the real data in terms of movement statistics and firing rate, spatial information, and variability in the theta sequences, and found that indeed this sampling index can reliably discriminate between sampling and map encoding. Finally, the sampling index was consistently positive for all 8 experimental sessions that we analyzed and were recorded from 4 different rats. Thus, our data supports a sampling-based representation of uncertainty during hippocampal theta sequences. In summary, we found a large variability of the encoded trajectories during hippocampal theta sequences, and this large variability is consistent with random sampling of prospective locations and suggests that a dynamic generative model is implemented in the hippocampus. Finally, we found no evidence for spatial ways of representing uncertainty, either in a product or in a mixture type representation. I would like to thank Brad, David, Andres, and Yuri for kindly sharing their data, and also to my colleagues in the lab for discussions. And thank you very much for your attention. I hope I'm well with time.
Yeah, so we're going to allow ourselves to run over about 2 minutes, 2 more minutes if people want to ask any quick questions. I have a somewhat naive question, which is about the translation of your model between the linear and the circular track. Do you want to just talk about whether there were any quantitative differences?
Yeah,
between...
the main point that for the last analysis that we made, when we have to compare decoded trajectories with real trajectories, that is only doable in the 2-dimensional arena. Because in a 1D arena, you cannot really compare trajectories because they are all just the same.
Right, they're always the same.
But all the other analysis can be done in the 1D. And that's why we just choose that, okay, we should check those predictions also in the 1-dimensional linear track data.
Yeah, we have a question that's just popped in the Q&A. Balazs, in your analysis of cycle-to-cycle variability, did you see any evidence of alternation between two trajectories on alternate theta cycles?
Yeah, I think this is a beautiful paper from Laurent Frank's lab that showed that this is this sort of alternation. I didn't specifically check for this sort of alternation, but as you could see, I could see that the cycle-to-cycle variability was larger than what you would expect in matched control simulations. And this large variability could be an indication of such an alternation between different future locations. So the variability is larger than random sampling. It is consistent with like sampling opposite values.
Mm-hmm. Yeah, that makes sense. Okay,
yeah,
so we're gonna have
Great
to.
question.
Yeah, thank you for that. And thank you all three of you for giving excellent talks. This was a really interesting session. And thank you all for listening. And we're looking forward to seeing you at future NeuroMatch sessions. So, ciao, everybody. Which is actually in York, not in Birmingham. But I had in my head that it was York, UK. So you're actually much closer.
I thought that as well.
Oh, did you? Okay, cool. I'm glad I'm not the only one.
Makes sense. I think we do get that a lot.
Yeah. Cool. Then we're not. Then I'm not far off your time zone actually, because I was thinking, trying to figure out what session it was that you were talking about at 5 a.m.
Yeah.
Cool.
I think we're on the same time zone because it's around 1 p.m. for us right now.
Yep. Yep. I guess we must be then. That's cool. All right, I'm gonna get my screens arranged here and make sure everything's good. You guys can leave your video on while each other are talking, since it's the panel discussions, or you can turn them off if you don't want to be on video. That's also fine.
So our YouTube video is on, and our attendees is here as well. Yep.
Great. I'm just going to start. I'll introduce the session at 1:01 just to make sure we're like giving people ample time. The thing that I find funny about online presentations, like the moment before it begins, is like instead of the like low din of like people chatting in the room, you have this like really incredible pure silence. It's like it to me is like really what drives home it is the moment that is the most different.
Yeah, feel that.
It's all good. All right, so we'll get the session underway. I'll introduce us here. So hey everyone, I'm Adam Steele. I'm a postdoc at Dartmouth College in Hanover, New Hampshire, USA. I've been told that we have a few moments just to allow everyone to trickle in. So I'll use that time to say, everybody in the US, please make a plan to vote. If you have already voted or you've made a plan to vote already, please call one of your friends and make sure that they have a plan to vote. And if you're not in the US or you can't vote in the US elections, please call one of your US colleagues and tell them to vote because it's incredibly important. With that, we'll get started today. We have a great line of speakers in this session talking about visual motor adaptation. I'm really excited to hear everything that they want to say and also to have a discussion because this is an interactive session. So each of the speakers is going to give a 5-minute talk, and that'll leave 10 minutes for audience questions or discussion. There's ample time for that. If you want to come on stage to ask your question, we can do that. Or just we can unmute your microphone or give you the option to do that. Just post with your question whether you would like to have yourself unmuted or whether you would prefer that we ask it. Otherwise, post in the chat. Because this is an interactive session, like I said, it'd be great if the people who are here can introduce themselves in the chat. So why don't you, if you would like to, just post in there what your name is and where you're calling in from. Just so we can see everyone who's in the room. So you can post questions at any time. Please upvote your favorites. And for those of you who are on Twitter, please tweet about this session using the hashtag NMC3. So all of that said, we'll get underway. Our first talk is going to be given by Shana Mochalingam, and his talk is titled A Motor Adaptation Paradigm to Strengthen Implicit Learning.
Thank you, Adam. So I'll just go ahead and share my screen there. Hopefully that's working well. So like Adam mentioned, my name is Shana, and I'm from York University and in Canada. We had a little bit of confusion before the talk began about that. And I'll be talking about a motor adaptation paradigm to strengthen implicit learning. And again, in this talk, it's going to be in the context of visual motor adaptation. So when we learn a movement, and that is what I mean by that is when we learn to reduce errors, we have generally two different processes that are happening at the same time. So one is an explicit process and another is an implicit process. Explicit processes, you can consider them as conscious strategies to your movements. So these are nice in that they can develop very quickly, as in we can see a very obvious error and we can think in about a second what I'm going to do next to counter that error, but they're cognitively expensive. And one of the biggest problems with that is that they take a lot longer of a preparation time before you can initiate the movement. Implicit processes are, on the other hand, are more slower to develop. So they take a few different exposures to an error before they become fully developed, but they're more efficient. However, they can be limited and invariant, and I'll talk about what I mean by limited and invariant in a little bit. One really cool thing about implicit processes is that they do kind of free up cognition for other things. So for instance, if you've implicitly learned to adapt to a small error, say because of muscle fatigue or something, I can be having this conversation while reaching for something and countering that error. So again, in this talk, I'm going to focus on implicit processes and again, in the context of visual motor adaptation. So what do I mean by limited and invariant? A few studies in the past have looked at the amount of implicit adaptation that can occur when adapting to, in this case, again, visual motor rotations of various sizes. So you can see here on the x-axis that we have a bunch of different perturbation sizes in various studies, ranging from 30 to 60 degrees in all studies. But you can see that when implicit adaptation is measured in the y-axis, they're all very similar. So whether people adapt to a 30 or a 60 degrees rotation, implicit adaptation seems to stay around this 10 to 20 degree mark. There is some differences between studies, but within one setup, they seem to be fairly, again, invariant. Regardless of the perturbation size, you have a similar amount of implicit adaptation. Now, like I mentioned, there are various benefits to implicit adaptation, and it would be nice if we can bring this implicit adaptation to a higher level. So has this been done? So we do have one previous study from our lab in the past. Again, it's an older study, so we didn't really measure implicit adaptation as well as we do now. It wasn't isolated as well, but we do see that in two different groups. So in this group, in these two groups, people adapted to a 30 and a 70-degree rotation, right? The 30-degree rotation people or groups are in the white here, and the 70-degree rotation group is in the black here. So we can see that over 3 blocks, the 70-degree rotation group did have, again, this is implicit adaptation on the y-axis, it did show higher levels of implicit adaptation, going all the way up to like 40 degrees. Now, 2 things. One, like I mentioned before, implicit adaptation wasn't well isolated. So we do want to see if this difference between the groups is because of actual implicit adaptation differences or because of our measurement error. Another thing is that when people adapted to the 70-degree rotation, people adapted in a stepwise manner. So in that, what we mean by that is for the first rotation block, people only adapted to a 30-degree rotation, and then a 50-degree rotation, and then a 70-degree rotation. So we wanted to see if it was this methodological difference that resulted in higher levels of, hopefully, implicit adaptation. So how do we measure this? So we had 2 different phases. The first one is just an aligned phase where people reach to a target with that cursor aligned with their real hand position. And that's what we call this aligned cursor training. And interleaved, we had no cursor reaches, which means people again reach these targets, but they don't get visual representation of their hands. So they're basically blind reaches. And then people adapted to a rotation. So in this case, we went with a 60-degree rotation, which is a fairly large rotation. But again, within these training trials, we interleaved no cursor trials. But importantly, we were sure to ask people to either reach with a cognitive strategy. So again, when they're adapting, if they had a cognitive strategy that they were using, we told them to either use that strategy or to not use that strategy during the reach. And the idea is that if they're not using a cognitive strategy, if they're trying to reach directly to the target, but they still deviate by some amount, we consider that implicit learning. So here's how our experiment setup looks like. So people reached on a digitizing tablet here, and their hand was occluded by a mirror, and visual display was also shown on that mirror. So we wanted to test this abrupt versus stepwise adaptation paradigm. So in the abrupt rotation, so this was one group, in the abrupt rotation, people experienced the entire 60-degree rotation on the very first trial. So they went from a 0-degree rotation to a 60-degree rotation. This was very obvious. And this is what's usually done in visual motor adaptation paradigms. In the stepwise rotation, people in the first block of rotated training, they only experienced a 15-degree rotation. After they fully adapted to the 15-degree rotation, they experienced another 15-degree rotation, so 30 degrees in total, and so on until after 4 separate blocks, people experienced a 60-degree rotation, just like in the first group. So ideally, we wanted to see more implicit learning in this group, in this stepwise rotation group, compared to the abrupt rotation group. So here's another explanation of our paradigm. So this is a perturbation schedule. So on the bottom here, we have trials, and on the y-axis, we have what perturbation people experience. So again, they have an aligned phase, and in this abrupt group, people experienced a large 60-degree rotation on the very first trial, and the stepwise group looks something like this, where they do 15 degrees at a time until they reach a full 60-degree rotated group. So in previous literature, there's been some conflicting evidence as to whether something called a gradual rotation leads to more implicit adaptation. So a gradual rotation in this case is something like this, where over, in this case, it's 60 trials, people eventually again reach reach using a 60-degree rotation, but the rotation is introduced 1 degree at a time. Okay. So again, we wanted to see if that also led to higher implicit learning. And again, these little orange markers are these places where we had these no cursor reaches. Okay. And I just want to point out that I marked these as block 1, 2, 3, 4. That becomes important later. So this is just how performance looked like. So what I just want to highlight here is that all 3 groups, by the end of training, when they're during block 4, when they're adapting to a full 60-degree rotation, they perform similarly. So all groups are able to adapt to a 60-degree rotation. So now let's look at the implicit learning. So again, these are these no cursor reaches when we tell people not to use any cognitive strategies. Surprisingly, we see that with both gradual and abrupt rotation paradigms, people don't go above this 15-degree-ish limit that I mentioned before. So again, in this case, in blocks 1, 2, 3, and 4, by the time we measure these implicit after effects, people have adapted to a full 60-degree rotation. But again, implicit learning is still at this 15-degree mark. However, when we look at this stepwise rotation, we clearly see that by the time people adapt to a full 60-degree rotation, they do show higher levels of implicit learning. So I think we've robustly shown that this stepwise rotation is different. Again, we're doing experiments to see why this might be different. The whole pandemic has kind of slowed that down, but we can at least show that phenomenologically it is different than the other 2 experiments, right? So learning in this manner, for some reason, increases implicit learning. So what's up with this gradual learning? So again, I mentioned there is some difference in or conflicting evidences in previous studies regarding gradual learning. And what we see is that when we tell people to use a strategy, they're not able to use a strategy, especially in this very first block, when we first tell people or measure these after effects. When we tell people to use a strategy, they don't really have an explicit strategy developed at the beginning. It takes some time for people who have experienced a gradual rotation to come up with a cognitive strategy. So it's a little strange. It seems that implicit learning is not different between gradual and abrupt introduction of a perturbation. However, it's this cognitive strategy used that's actually a little bit different. So in conclusion, again, implicit learning is invariant within the gradual and abrupt groups. And gradually, sorry, I think I'm missing a word there. And gradually introducing a perturbation actually leads to a lowering in strategy use, right? Or it takes longer for people to come up with a cognitive strategy. However, introducing a perturbation in a stepwise manner, like we predicted, can lead to actual increases in implicit learning. So we do think this is important because, like I mentioned before, when it comes to like a rehabilitation sense, it's important that people are able to use or adapt to things while not having to cognitively think about the things that they're adapting to. So with that said, I'd just like to thank our lab. So here's Marius and Raphael, who'll be giving a talk very shortly. And from there, I'll take any questions.
All right, that was a great presentation, and that effect looks absolutely massive. So that's really interesting. I'm looking forward to hearing about how you'll follow that up in the future. You've had a bunch of questions pop up, and we have had a person with their hand raised. So I'm going to first allow Caro to unmute herself, and she can ask her question.
Great, thank you so much. That was a great talk. So I've actually used the stepwise paradigm as well, and what you're showing resonates with what I see in my data. I'm curious if you've maybe looked at whether baseline variability predicts or correlates with implicit adaptation, just because there's a paper showing that basically becoming aware of the rotation is dependent on your baseline variability, which then governs your detection threshold? And also, sorry, just secondly, do you think therefore this effect would depend on the tool you're using? Because maybe in joysticks there's more variability than in like a reach adaptation paradigm.
Right. So there's a few questions in there, and thanks for the question. We should definitely contact each other later after the talk, maybe, since you are working on similar things. So firstly, about the baseline variability, I am aware of that paper, and the reason we chose 15 degrees is that in general, that's around the detection threshold. So we wanted it to be not too similar to the gradual rotation paradigm, right? Like if you think about it, a gradual rotation is just a lot of 1 degree steps.
Yeah.
Right. So we wanted to make sure people were sort of, at least at the beginning, noticing that there was this rotation. So, but we didn't look at any relationships between individual baseline variability versus how they learn. Now, we do have fairly large numbers in these groups. They had 37 people in each group. So we'd assume that the baseline, I mean, again, I'd have to do a correlation to see if there's a correlation there, but I'd assume that within these groups, like we have the whole gambit, right?
Right.
So
yeah, because
regardless of sorry, I just wanted to mention, if baseline variability does influence this, it's not driving it. So we'd assume that within these large groups, like one wouldn't have a higher baseline variability. Because like I said, everyone did the exact same baseline task.
Yeah, it might be just interesting to look further into that, one group, just because my data doesn't allow me, I don't have enough subjects to do that analysis, but I also chose a 10-degree stepwise increasing rotation because of that reason, because of it's lower than the detection threshold.
Cool.
All right, so unfortunately, I'm gonna have to cut the questions short here because of time. But Shauna, you've got a bunch of questions in the Q&A. I will leave them open for you, so that way, at least until Raphael's done talking, so that way you can address them. If anyone wants to keep the conversation going with him, you can post in the chat too. But we are going to move on now to Raphael Gastruck, who is going to be giving his talk titled, I lost my notes, so I'm going to look up, there it is, the different mechanisms that distinguish motor skill acquisition from adaptation. So Raphael, the floor is yours.
Thanks, Adam. Hopefully everyone can see my screen and my pointer here. So yeah, hi everyone, I'm Raphael, and today I'll be talking about the behavioral mechanisms that distinguish acquiring motor skills from adaptation. And there are at least two types of motor learning often identified, and the first has been investigated extensively, and Shauna has talked about it in his talk earlier, which is motor adaptation, which suggests that a representational remapping has occurred somewhere in the brain. So a typical task would be to have participants reach towards a target. So in our experiment, it's in a radial array. They would use a digitizing tablet to reach, to get a cursor towards a target. But then we also introduce a rotation, and in our experiments, it's usually in the magnitude of 30 degrees. So you can see that the magnitude of the rotation is fixed no matter where the target is. But another type of motor learning is de novo learning, where a new mapping is expected, such as when you're trying to learn a new skill. So previous research have identified that the mirror reversal task is able to investigate this type of learning. So here we match the target locations for those in the rotation task, but given the mirror axis in the middle here, you can see that depending on where the target location is, they would have to deviate their reaches in different magnitudes. So here it's 15, 30, and 45 degrees. So our experiment was a within subjects design. So we counterbalanced for the order that each participant experienced the perturbation, as well as where the axis was located of where they experienced each perturbation. So some would experience the rotation in a vertical position, others in a horizontal, and same for the mirror reversal. And this, we also counterbalance for the target locations in relation to that axis. So essentially, no effects were observed. So learning one perturbation does not affect the other. And then we tried to quantify the learning for each perturbation across participants. So to make them comparable, given the different angles that they would have to deviate their reaches, we measured amount of compensation. So this is the percentage of how much they were deviating their hand relative to perfect compensation. So you can see that by comparing the two tasks here, both tasks, or participants were able to learn both tasks relatively quickly within 90 trials, but it was definitely more variable for the mirror reversal compared to the visual motor rotation. And as a measure of reach after effects, we simply plot the washout trials. So this is when the perturbation has been removed and they switch back to an aligned visual feedback. And we see evidence of reach after effects for the rotation, but not the mirror reversal. And as some other behavioral measures, we do look into reaction and movement times as well. So here I plot the aligned version as a kind of baseline or reference. And when we plot the reaction and movement times, we see that both times were slower for the mirror reversal compared to the rotation task. Now, in a second experiment, we wanted to explore the mirror reversal further using an online platform. So we had to simplify the task by using only two targets here in the 30 and 60 degree mark in polar coordinates. And basically, we had it only in the upper right quadrant of the workspace. And you could see like how much the participants had to deviate their hands in relation to the target position. So here I only plot the angular reach deviation because we're only comparing the two target locations. So perfect compensation for the 30 degree target would be at 120 degrees. And for the 60 degree target, it's at 60 degrees here. And the aligned session basically proceeds as expected. But once we introduce the mirror reversal, you can see that the 30 degree target, participants don't reach perfect compensation, unlike in the 60 degree target where they do reach near perfect compensation here. And also, we do not observe any evidence of reach after effects during the washout trials. But if you would notice, immediately during trial 1, reach deviations here are quite high already. So we wanted to look into this further to kind of try to explain what's happening here. So we did a plot of the participant distributions of the reach deviations for each trial. So here I showed trial 1, and it's a circle because it goes from 0 to 360 degrees in terms of reach deviations. The target is aligned to 0 degrees, and perfect compensation would fall on 120 and 60 degrees respectively for each target. But basically, you find that in trial 1, there's a bimodal distribution of responses here, which would explain the high deviations in trial 1. But as trials progress, and I'll show trial 2 here, as well as trial 90, you can see that 90 would be the last trial, by the way. Most participants eventually respond towards the correct location, and that's actually clearer for the 30 degree target more so than the 60 degree target because you still get responses near the 0 degree mark here. But this still doesn't answer why participants already know in which direction they should be reaching to begin with, even if trial 1 hasn't started. So we think that this may have something to do with our experiment. So since participants had to move the cursor to the start position at the beginning of the task, they could have explored the workspace at this time, even if trial 1 hasn't started. So as a separate analysis, I don't show it here, but what we did was we tried to compare the angular reach deviations with the movement time, or the time it took them to get to the start position, but found no relationship there for either target. So this is still an open question. It's something that we need to investigate further as to why they're already reaching in the correct direction here. But at least for this experiment, we were able to show that they are able to learn how to compensate for the mirror reversal in an online task. And just to summarize, the mechanisms underlying motor adaptation and de novo learning are distinct from each other, and future work will need to investigate the mirror reversal task further using an online platform. And with that, I'll open it up to the discussion and questions.
Great, thank you, Raphael. So there's, again, plenty of time for discussion, so you can go ahead and post any new questions for Raphael in the Q&A tab. it's really interesting that they are getting it on the first, getting the task on the first reach. Is your analysis checking at, you might have said this and I missed it, but is it the initial direction that they're going, or is it the endpoint that you're looking at here?
So we take a kind of like a proxy for the initial direction error. And to do that, we take basically 20% of the home to target distance. So if for example, this is the home position, if you can see my hand and the target here. So
Yep.
like 20% of that distance when they're moving out, that's the point that we get essentially.
And how much could they possibly, is it possible to know how much curve or like how much they may be correcting in that, or is that not something that you've been able to look at? And
we actually,
because 20% is actually that's actually a reasonable amount of distance to begin the correction, especially if they've noticed that they're going wildly off course.
Yes, definitely. And we actually did try to plot the reach trajectories as well, just to kind of see like where they're going throughout the whole trial, right? And we do see that a lot of participants are not making relatively straight reaches. Most of them are making like a zigzag pattern, probably because they're trying to get used to the mirror reversal, right? So depending on which sample I get from that reach, these results would differ in terms of the magnitude that they would show in the angular reach deviations. But relatively speaking, like the plot that I show here, like it doesn't really change in terms of like the curve that it shows.
Okay, interesting. So yeah, really, really interesting. So you've got a few questions popping up in the Q&A. Nick, I saw before that you're comfortable being unmuted, so I'm gonna go ahead and give you permission to talk, and you can unmute yourself.
Can we hear me? Is that good?
Yep, can hear you.
Cool. Okay, thanks for the talk, Raphael. That was really interesting. I just wondered if you wanted to comment on maybe whether there's faster adaptation in the mirror reversal task, perhaps because we're more familiar with that kind of visual transformation in day-to-day life than perhaps a rotation around a start position. And I just wondered whether perhaps that was a reason you'd see a faster initial adaptation, or whether you had any thoughts on that.
Yeah, I wouldn't know whether it's correct to say that we're more used to dealing with a mirror reversal type of thing in everyday tasks. But I think what's happening here is that once you do understand how a mirror reversal is working, then that's it. It's kind of like it happens in like a snap. You know how to deal with it immediately. But in the rotation, you kind of take more trials to become more exact with where exactly you're moving. So here, I'm guessing that in trial 1, they immediately find out like, oh, this is how the workspace is working. If I move here, the cursor goes there. And they could easily just apply this to the other target, since it's in the same quadrant, and they wouldn't have to deal with the target being at some other place relative to the mirror axis here.
But in theory, that could work for the rotation as well, right? You could just pretty quickly work out that that's the rotation, and then you just apply that to the other targets as well, unless I'm like missing something.
Yes, definitely.
Or do you just think that, and that's why I kind of thought that maybe, I don't know, more intuitively we understand a mirror reversal perhaps than like a rotation, but I don't know, maybe it's, you know, one of those things.
But also, like, just to add to that, probably it has also something to do with the explicit and implicit learning that you have for the rotation and the mirror reversal, where, like what you said, in the rotation, technically you can just figure out the strategy and immediately like show an advantage in dealing with it. And probably with the mirror reversal, it's more of an explicit strategy, or your explicit component or contribution to the learning that you're showing here.
Okay, thank you.
Thank you.
Okay, great. So we also have a question from Alexander Cates. I don't know if Alexander wants to come on, but we're short on time, so I'm going to ask it for him. In graph and experiment 2, which you actually have displayed right now, would perfect performance for 60 and 100 degree would perfect performance be 60 and 120 degrees to compensate? Why do you think, and if that's the case, why do you think that the participants are actually performing worse as they proceed in the task?
That's a good question, and I don't have the exact answer for it, but if I were to guess what's happening here, it may be that the mirror reversal task becomes harder as you move further away from the mirror axis. So the 30-degree target here would be kind of close to the horizontal line, basically. So it could be that participants just try to reach in a general correct direction and then just adjust for it afterwards, which we don't really capture in the measures that I'm showing here. But it could be like, as long as they're in the correct quadrant, then they could just adjust their movement to do the task or get the cursor to the target afterwards.
Great. Yeah, that's interesting. It's also, they're reaching, am I reading the graph correctly, that the reaches are becoming more extreme? So maybe they're overcompensating in this mirror, where their explicit strategy is super like exaggerated compared to the compensation they actually need to make. So that's kind of an interesting effect too, which seems counter to what you usually see in adaptation, which is like not quite achieving the correct angle.
Yes.
Cool. So we're gonna have to wrap up there. To give our final speaker ample time, and that is Marius Hart, and he is going to be giving a talk titled "The Different Mechanisms That Distinguish Motor Skill Acquisition from Adaptation."
All right, thanks, Adam. I'm actually going to talk about aiming evokes explicit motor adaptation. Let me minimize this.
Sorry about that, Marius.
There we go. Okay, so why am I talking about aiming here? You've seen a couple of talks where we talk about implicit and explicit learning, and it has been assumed for a while back that most motor learning is implicit, but there's actually very good evidence that there's a lot of explicit learning going on, especially in like these mirror reversal paradigms, for example. And often this is being measured with aiming. That is, you ask participants on every trial to think about where are they going to reach towards the target that you're presenting them. And to me, this has always seemed like you're giving people a clue, like the direction of the movement is very important. Maybe this is how you should solve this problem, which makes them more explicitly aware than they would usually be. So when we started looking at like we're very interested in these implicit mechanisms, but we want to make sure that what we're measuring is not due to explicit learning. So instead of using aiming, we decided to use the process dissociation procedure. Now, this is kind of what Shana talked about by using no cursor reaches that either include or exclude the strategy. But then when we are submitting our papers or presenting our posters, then our colleagues are asking questions like, why don't you use aiming, or why don't you find explicit learning with a 30-degree rotation? Because that's what's being shown with aiming. And to me, that is because like we don't want to elicit explicit learning. We want to look at explicit learning. We want to look at the implicit processes. And but of course, we have no evidence that aiming really elicits explicit processes. So that's why we decided last year to start doing an experiment with first an aiming group that has 24 participants, an instructed group that we tell exactly this is the rotation and this is how you are going to be able to counter this rotation. And then we also have a control group that neither does aiming nor gets an instruction. So this is what if this is what the workspace looks like, then people would start their reach here at the home position and they would go to one of 8 targets, let's say this one. And then in the aiming group, before they actually aim, they get this arrow and they can orient that however they like, to say like, this is where I'm going to actually move my hand in order to get the cursor onto the target later on. And then all groups also do this process dissociation procedure, where in no cursor reaches, we ask them to either exclude the strategy where we supposedly only get implicit learning, or to include the strategy where we get both implicit and explicit learning. So let me show you the paradigm and data, the reaching data. So first we start with an aligned cursor, and this is cursor training in these 3 blocks. And we have baseline no cursor blocks as well. And then in the rotated parts, they train a little bit longer. And we have, let's say, exclude strategy blocks and include strategy blocks that we can then compare. Here's the data for the control group. It's a typical learning curve with some decay in the no cursor blocks, nothing special. Then we have the instructed group that starts off at about 20 degrees. So they understood our instructions and were able to apply them at least to some degree. And here's the aiming group that does very similar to the control group. So this is not surprising so far. Of course, we also got these aiming responses from the aiming group, and they look like this. This is not as high of an aiming response as you would see in some other papers. It's about 4 degrees. So now let me talk about how the reach after effects we measured relate to this aiming. So here on the y-axis, I'll show what I call explicit learning, and that is the difference between include and exclude strategy, no cursor trials. There, exactly. So here's the control group, which is very close to 0, although not exactly 0. And the instructed group, which is, which like they know a strategy because we told them and they're able to do that. And then we have an aiming group, which is, I don't know, somewhere in between. But you may also notice that the distributions of individual participants, that's these dots, are very different for the 3 groups. So for the aiming group, it looks like there might be a bimodal distribution. So first of all, here are the distributions for the control and instructed groups, and you have clear peaks around 0 and around like, what is that, 18 degrees. And for the aiming group, there's like 2 kinds of participants. There's 2/3 of participants are very similar to the control group. It looks like they don't do any explicit learning, whereas 1/3 approximately has about the same explicit learning as the instructed group. So you might already say like, okay, doing this aiming makes some participants explicitly aware just as if they had an instruction. And if that is the case, then the measure of aiming, the aiming responses itself, like the deviations of those arrows from the target, should be similar to this explicit learning mechanism. So that's what I'm showing here, only for the aiming group, because we don't have aiming for the other ones. Here's the individual participants, unity line, and a regression. So, which is not different from unity and highly significant. Okay, so what does this mean? I think, first of all, I don't want to stop people from using aiming. It's super useful, especially if you're interested in explicit learning and if you want to continuously measure it, keep doing that. But we are not, like in most of our tasks, we are interested in implicit processes, and then this might be a good reason to use another method and to decrease the explicit learning and really be able to focus on that implicit process. Exactly, and so yeah, so then I have to say, like, all the hard work and the heavy lifting has been done by Arush. Thank you so much. And if people want to assign blame or criticize the study, then you should talk to me instead. All right, thank you so much.
All right, thank you, Marius. That was a really, really great talk. I think the demonstration of the difficulties with using aiming yeah, seemed like a great point. And hopefully we can have a good discussion about it here. So you've got a question in the Q&A. Doesn't say whether they would like to be brought on, so I'll just ask it for them. Can you please elaborate on the small magnitude of implicit learning compared to other papers?
Right, well, I didn't actually show the implicit learning. The implicit learning here is about 14 degrees. If you just look at the, so the graph, let me share the screen again. There we go. Oh, come on. So this is not implicit learning. That is the difference between explicit and implicit reach after effects. So basically, this is the explicit component. But if you really look at the implicit component itself, it's about half of the learning that people did. 14 degrees, or no, 12 to 14 degrees, something like that.
So the implicit and explicit is usually super additive, right?
Yeah.
Or is it usually sums to more than, or to less than it would take to fully compensate? Or what they fully end up compensating?
Yeah, that's what people say. So I actually, it's good that you bring this topic up. I have this extra slide here with an alternative view on the data. So if like this is your home position and this is your target, then aiming is supposed to be, and then you have what people actually do during training, these reaches, that's their actual reach. And then usually what people do is split up this bit in like, this part is explicit and that part is implicit. So in other words, total adaptation has to be the sum of implicit and explicit. And if you look at what the experiment does, then, which we program in an algebraic way, then that is correct. But I'm not sure if the brain would solve this particular problem in an additive way. If you do assume it, then like adaptation minus explicit should give you the implicit measure. Or the way we're doing it with PDP here is the include strategy minus exclude should give you explicit. I don't know if either one is true, actually. So let me show you our data here. So this is the total adaptation at the end of training for all three groups, which is kind of the same. This is the exclude strategy reach after effects for all three groups, which is also the same. So that means if you take this assumption that it's purely additive as true, they should all have the same amount of explicit learning. But even in like the aiming group, the include strategy doesn't get you all the way up to the total adaptation. So that it's not the case for the aiming group that this is additive. Now for the instructed group, it is kind of true, but that might be an artifact. And here for the control group, it's even less true than in the aiming group. So I'm not so sure if we should be thinking of the brain as doing simple addition. It might be a different type of process.
Yeah, sorry, go ahead.
Yeah. So there's also a, I don't know if it's already published, but it's a brilliant preprint by Jana Maresh, Susan Werner, and Ofer Danchin that says there are different types of explicit learning that you can measure differently with like these different measures. And maybe there's even like some, I don't know, dark matter of adaptation that we just can't measure with these methods. Right?
Yeah, it seems in particular with aiming that there may be some, perhaps some like calibration error or calibration adaptation that's going on there too. Like you may not have complete access to your aiming in some way. Anyway, I'm gonna let JJ come on, JJ Arban, who is, he's gonna be allowed to talk now. So JJ, you can unmute yourself.
Hi, can you hear me?
Perfect.
Yeah, hey Maurice, very clear presentation. So I was wondering how this, the exclusion part of the PDP process differs from what has been used by others to measure after effects, simply telling people, you know, here there is a perturbation, here there is no perturbation. And that's what people usually take as implicit, as measure of implicit adaptation. And so I'm not sure exactly how this compares to the PDP, the exclude part of the PDP that you use.
Yeah, the exclude part is more the classic reach after effects. What we add, like Shana did the same thing. What we do is we tell people after they did the aligned part, like remember what you did now, just before, because we're going to ask you to do it again. So reach without your strategy, and then when they do when they're in the rotated part, we tell them like, okay, if you have any strategy, forget that now and do what you did before, which hopefully you remember.
Yeah. I mean, that I guess because what we have been doing is simply showing the target and asking people, bring your invisible hands on the target. It's kind of a different type of instruction. I think it's tries to do the same thing, but it's a different type of instruction. And I wonder whether the instruction can play a big role here. I mean, those are critical, of course, to
we actually mentioned
the process
in the hand too. So it's not totally different, because what in a different sentence we tell people, like, you have to move your hand to the target, not the cursor. So
right.
What you did to get the cursor to the target, but focus on your hand.
Yeah.
But yeah.
Okay. Yeah, that's very similar in that.
Great.
Thanks.
Thanks, JJ. I'm going to disable your talking. The session is just about to wrap up, but I will ask Barbara's question. Just for understanding, does this mean that making the subjects report their aim interferes with how they solve this task? Super interesting. So I guess, yeah, if that was going to be like putting a pin in the point here, would you say that that's the summary or the take home?
Yes, for some participants.
Cool. So great question, Barbara, and I think that's a great place for us to end. So thank you, all three of you guys, for some great talk, and the audience for a great discussion session. This was a lot of fun, and we hope that to see you at future NeuroMatch sessions. So thanks, everyone.
Thanks, Adam.
Thanks, everyone. Thanks, Adam.
NMC.
Thanks, everyone.
Bye. Plan, call one of your colleagues to make a plan. And if you are not in the United States, or you are not able to vote in US elections, please do call one of your US colleagues and make sure that they have a plan to vote, because it's really, really important. So we have got a great lineup of speakers in this session who are going to be giving some cool talks on gamma oscillations. So I'm really excited for this. It's an interactive session. So the speakers are going to give 5-minute presentations, 5-ish minutes, and then that'll leave about 10 minutes for audience discussion and questions. If you would like to come on stage to ask your question, we're going to have time for that. Just let me know in your comment or question whether or not you'd like to be unmuted and ask your question yourself. Otherwise, you can post in the chat at any time or the Q&A. Because again, this is an interactive session, so I'd suggest that if people want to introduce themselves in the chat, so that way everybody knows who's here, you can just post your name and where you're calling in from. That would be, I think, really a nice thing. You can ask questions at any time, upvote your favorites, so that way we can make sure they get asked. And for those on Twitter, please tweet about this session using the hashtag NMC3. So our first talk is going to be given by Nosratullah Mohammadi. His talk is going to be titled Instantaneous Modulation of Gamma Rhythm by the Difference of Time Constants of Inhibitory and Excitatory Synapses.
Thanks, Adam. And thanks to NeuroMatch organizers for giving me this opportunity to be here. Hello, everyone. My name is Nosratullah Mohammadi. Let me just share my screen. Okay, can you see my screen now?
Yep, we can see.
Great. Under this, I'm a master candidate at Institute for Advanced Studies in Basic Sciences. And under the supervision of Dr. Ali Reza Valizadeh, I studied the effects of changing synaptic time constants on brain rhythms, especially on gamma oscillation. So why are we interested in gamma oscillations, and why we should even care how these rhythms are changing over time? Well, the answer would be that many studies on mammalian brains have observed the prominent existence of gamma oscillations during activities such as exploratory behavior in hippocampus and motor responses in premotor areas, and this list goes on and on. So those studies encourage us to find out, to dig in and find out more about the underlying mechanisms of modulation of gamma oscillation. So in this study, we chose integrate and fire excitatory inhibitory neurons as a building block of the network. We only focused on synaptic decay times of excitatory inhibitory neurons and fixed all other parameters in each simulation. And due to simplicity, we chose exponential curve or function for postsynaptic current curve to just have more control over the parameters, and of course to get more to increase our confidence about the results. So we created our network with 20% inhibitory neurons and 80% excitatory neurons with only 10% random connections. We simulated the network in a wide range of inhibitory synaptic decay time from 1.5 millisecond up to 3.5 millisecond, while we fixed the excitatory synaptic decay time to only 2.5 milliseconds. Previous studies showed us that increasing the inhibitory synaptic decay time would result in decreasing the gamma frequency. But other aspects of this change are left out. Here in this figure, you can see the power spectral density plot of one of the simulation with 1.8 millisecond of inhibitory synaptic decay time. So in order to study this effect, we have to first apply spectral regions to extract different spectral. In order to study, we have to first apply spectral separation to extract different spectral regions such as theta, alpha, beta, and of course gamma oscillation out of the total population activity. This can help us in future to study cross-frequency coupling such as phase amplitude coupling, which is basically that the phase of slower rhythms are going to modulate the amplitude of the faster rhythms. But what do we mean exactly by instantaneous modulation anyway? Well, we want to see if there's any meaningful correlation between the amplitude of the signal and the period of the signal in each cycle, meaning that if there's any, by knowing the amplitude of the signal, can we predict the period of the next cycle? And if so, how good we can predict it? And there are other terms such as the correlation between the period of each signal, each cycle, and the amplitude of the next cycles, and the autocorrelations of amplitudes as well as the period autocorrelations. So once we get the results, we can see that there are two different behavior in the simulation. In a smaller inhibitory synaptic decay time compared to excitatory synaptic decay time, those correlations that I mentioned are getting weaker by increasing the inhibitory synaptic decay time. But in larger inhibitory synaptic decay time compared to excitatories, those correlations are getting stronger by increasing the inhibitory synaptic decay time. And if we take Pearson correlation coefficient, you can see that the results are nicely classified into two subgroups. And in terms of phase amplitude coupling between beta oscillation and gamma oscillations, we can see that in smaller inhibitory synaptic decay time, there is a considerable coupling, magnitude of coupling, and with a tendency to phase zero, right? And by increasing the inhibitory synaptic decay time to larger numbers, both the tendency and the magnitude of correlation, or the strength of correlation, are going to vanish. On the other hand, when we look at alpha oscillation and gamma oscillations phase amplitude coupling, the phase amplitude couplings is just going under phase shift from phase 0 to phase I by increasing the inhibitory synaptic decay time. And it is the same scenario for theta oscillation and gamma oscillations, phase amplitude coupling. So the take-home message would be that there are different behaviors of amplitude period correlations, period amplitude correlations, as well as amplitude and period autocorrelations in two situations. And there is a phase shift in theta gamma phase amplitude coupling and alpha gamma phase amplitude coupling by increasing the inhibitor synaptic decay time. And last but not least, there is a decreasing of strength of phase amplitude coupling of beta oscillations and gamma oscillations by increasing the inhibitory synaptic decay time. And here I would like to thank my supervisor, Dr. Ali Rizavalizadeh, who helped me and guided me throughout this project, and my wonderful teammates who didn't bother to help me whenever I needed. And now I'm open to your questions.
All right, thank you, Nosratullah. That was a really great talk. Some very interesting simulations there. So we have some time for questions. So if anyone's got questions, you can go ahead and post them in the Q&A or post them in the chat. I actually had a question. If you can go back to the phase amplitude, or the phase coupling plot. Yeah, these three plots here. So I was wondering if I was somewhat surprised by the shift in phase amplitude coupling with the increase in inhibitory synaptic strength. Did that surprise you? And what, can you give me an intuition as to why that switch happens when you add more inhibitory, when you increase the time constant of the inhibitory synapse?
Well, I don't know the underlying reason for such phenomenon, because this is like one of the latest results of my study, and my next task is like going to answer these questions. This is a wonderful question, but yeah, I didn't see. I mean, as far as I know, I don't know any reason for such thing, for such happening, and yeah, I'm looking forward to answer those questions in future.
So what is the next step in order to answer that question? Have you kind of delineated that out? How you're going to interrogate that effect?
Yeah, like studying individual neurons would be the step one. So that yeah, observing like a bunch of random neurons, and I mean observing those individually in either the voltage, the membrane voltage, and like the frequency of activity, and there are many I mean aspects that we can look and see.
Yeah, that's really interesting. Are they different populations of neurons that are actually driving the different components of the frequency? So do the neurons in your because these are simulated neurons, do you find very discrete populations as far as what's actually generating each of these components of the spectrum?
Well, due to, I mean, because of the parameters I set, and this is only one neural network, right? I mean, neural network. It is one balanced network, and those, I mean, all of these frequencies in different bands are happening in the same local network. And yeah, I didn't look at that area like to focus on, I mean, those local regions, but that would be a great leap in the study, like see if there's anything going on there.
Yeah, it would be interesting to see whether you are, especially as you change the inhibitory current, whether you're creating distinct like how much you're differentiating different populations of neurons and kind of what the dynamics of that whole system are. There's a question that's just popped up in the Q&A from Caro. What was the composition of these EI neuron populations matched to any specific region of the brain? So is your study here meant to kind of be biologically plausible, if you will?
Yeah, that is a great question because, yeah, I mean, the parameters I chose is, I mean, from, I guess, CA3 region in hippocampus. And yeah, those are like typical parameters and the simulation that everyone do.
Sorry, I think we lost you for just a second. So you might want to repeat.
Okay. Sorry. So I told that, yeah, I chose the parameters from one of the simulations that happened on, I mean, simulate CA3 region in hippocampus.
Great, cool. So I think that's all the time for questions that we have, but that was a great, awesome talk. Thank you so much. And now we're going to move on to Zainab Esmailpour for her talk titled Mechanism of Temporal Interference Stimulation Using In Vitro and In Silico Models of Hippocampal Gamma Oscillations. So this should be a really interesting talk.
Thank you, Adam, for the introduction. And hello, everyone. I'm Zainab, and I'll be in the next 5 to 10 minutes. I'll be talking about mechanism of temporal interference simulation using in vitro and in silico models of hippocampal gamma oscillations. Let's just start with brain stimulation in general. There are like different categories of a simulation. Each has their own promises and also limitations. So starting with deep brain stimulation, it's first of all invasive, it's expensive, but at the same time you get a focal stimulation. Then moving to transcranial methods, either transmagnetic or electrical, we would have less focal, and also we cannot target deep brain regions, but at the same time these are widely available and easily to use. So the ultimate goal for us would be like having a method that is first of all transcranial, second of all it can reach deep brain regions. So recently there is this a study published by Grossman and his colleagues.
Zainab,
Yes.
I just want to interrupt you quickly to tell you that your slides, we are zoomed in on just like the very center of your slide. So some of the figures and things are being cut off. So you might just want to reset your screen share window.
Oh.
Sorry, I didn't want to, I was making
I mean, let's.
was making sure that that it was a problem before I interrupted.
Yeah, I mean, let me see. Let me stop sharing. Share. Then I saw this one.
Still, it's very
Still
zoomed in.
is very zoomed in.
Yeah.
Okay.
Or perhaps can you try share the application window only, or?
Meeting setting. Yeah, I don't know how to change.
Or we can go with the editing view, if that's.
So this is not zoomed in, right?
No, we can see everything. There is under advanced, and if you click on the screen share and then you click advanced, it should give you the ability to like crop just a small window of the screen if you don't want us to see your presenter notes.
Yeah.
But it might be what you know,
yeah, it's going to cause environmental issues.
Okay, it's fine. Just sorry about that.
yeah, I mean, okay. So this works then.
Yeah, we can see the whole slide. We're seeing your whole application window, but that's okay.
Yeah, but that is the issue. Okay, still it doesn't work.
Yeah, still zoomed.
Yeah, it's the first time that is happening. That is interesting. Okay, anyways, I guess for the interest of time, probably we can just move on.
So we were on, I think, your third slide there.
Okay, so yes, there is this method that is called temporal interference simulation, and it's the whole idea of that is having two pairs of electrodes that are producing sinusoidal, but in the kilohertz range frequency. And it gives, and these kilohertz frequency range are slightly different from one another in a way that it produces a really nice patterns of waveform throughout the whole brain. In the cortical areas, you would see purely sinusoidal waveforms, which I can show here, these areas. But when these two areas intersect with one another, because in frequency they are slightly different from each other, there would be an amplitude modulated waveform. And in the study that they have done in the, I believe it was mice, they showed that neurons are sensitive to amplitude modulated waveform, but not purely sinusoidal kilohertz range. So we wanted to test it in human. We wanted to see how it will be distributed in human. So we started using the finite element models of a human head. And as expected, we would see, with this method, we would see kind of targeting deep brain regions in a somewhat focal fashion, but also cortical areas would be exposed to high frequency stimulation. And the whole idea boils down to this fact that neuronal membranes have this low pass filtering property. So it would be dampening or attenuating all the stimulation. So it won't be sensitive to high frequencies, purely sinusoidal high frequency stimulation. But the immediate question that comes to mind is the amplitude modulated waveform doesn't have low frequency component. So how is it that the central nervous system can be sensitive to unmodulated sinus, cannot be sensitive to unmodulated waveform, but can be sensitive to amplitude modulated? So what is the reason? And also, if we want to go to human application, how much current do we need, or like how much sensitivity do we need for that? So in order to achieve this goal, we started using pharmacologically induced gamma oscillation, which has been studied extensively in neuroscience. So here you can see the experimental setup that we have two wires on the somatodendraic axis of pyramidal neurons in CA3. I'm recording from CA3A region, and we have this bath electrode just to reduce any noise because of the stimulation. And on the bottom, you can see that the method can produce stable gamma oscillations for about 2 or 3 hours. And also the frequency range of that would be around 20 to 40 hertz. Then we went one step further to see if we can explain the result that we saw with network model. So the network model that we picked was adaptive exponential neuron model. And in like a excitatory inhibitory network, and we used 2 subcategories of inhibitory neurons, and also a stimulation was modeled as a current that's injected to the excitatory neurons, assuming that inhibitory neurons, because of their geometry that is symmetric, won't be sensitive or depolarized because of the electricity. So going to our results, we started with purely sinusoidal 5 hertz, because that has been like a gold standard and has been replicated throughout the literature so far. And we can see stimulation can modulate the gamma activity pretty well. Then we started using amplitude modulated 1 kilohertz and also 10 kilohertz. One thing that comes to mind is noticing the intensity of the electricity that we need to produce the same modulation. And also another important thing that I want to draw your attention to is in the peak of the amplitude modulation, you can see there is a modulation of gamma oscillation, but when it goes to zero, this gamma activity or gamma power does not go to baseline, it goes to lower than baseline. And also even at the end of the stimulation, we see this rebound effect, which kind of calls for some kind of adaptation mechanisms as well. When we go to purely sinusoidal waveforms, there is a whole different response. At the beginning, there is this peak of response, and then when time passes, this adaptation mechanism kicks in, and the response at the end is kind of stable, but in the lower end, and then the same when the stimulation terminated, you have this rebound effect as well. So far we know that there is evidence because of the carrier frequency, high frequency carrier that you're using, you need more electricity or more electric field. That is an evidence for the low pass membrane properties of the membrane, low pass frequency properties of the membrane, and also this adaptation mechanism that we see here. So in order to quantify, because we have used different intensities for each of the waveforms to see if there is any nonlinearity or monotonicity or not, we quantified the power of gamma throughout this band of 20 to 40. Here we go from, on the top you can see 5 Hertz sinusoid, 100 Hertz sinusoid, and 2 kilohertz sinusoid. You can see from when we go from 5 Hertz to 100 Hertz, their behavior totally changes. So because of the fastness, the system doesn't respond to the trough, but does respond to the peak. And also for going from 100 Hertz to 2 kilohertz, we need to increase the intensity in order to produce the same response. Again, back to the membrane and its RC. Then going to amplitude modulated waveform, here on the bottom, you see all these 3 different ones that their envelope is fixed. The only thing is the carrier frequency. We go from 100 to 1 kilohertz and 2 kilohertz, and it shows that technically we see the same results, the same behavior, but increasing the carrier frequency would again make the system less sensitive. So we need more current in order to produce the same result. And also, again, these rebound effects are so obvious, and also the power that doesn't go back to baseline goes lower than the baseline. This is technically a summary of what we saw. The dynamic modulation, it was the quantification of peak to the trough of the modulation that we saw. And also a static is just the baseline changes, like static changes over the time. And it just shows that increasing the frequency from 100 Hertz to 1 or 2 kilohertz, we need to increase the electric field needed to modulate the brain activity. Again, showing the low pass filtering properties of the membrane. Okay, so I'm not going to talk about the model results. Technically, the model result reproduced all the experiment, but we learned 2 important lessons from that. One was there is this adaptation mechanism that in the model was modeled by the GABA-B dynamics that had this higher threshold and slower dynamics to make the system selective to amplitude modulated, but not purely sinusoidal kilohertz range. And also we learned that in our system, the responsive, in order to match the results to the model, we need to use the lower time constant for the membrane because of the fact that there are some highly sensitive compartment in the neuron that we are not using. So probably there are some highly sensitive compartment involved in the experiments as well. So I guess in general, selectivity is governed by adaptation mechanism, and sensitivity was governed by highly sensitive compartments of the neuronal models. And by that, I can thank everyone, and I think my time is up. I can take questions.
Thank you. That was a great talk. Again, some really interesting simulations here. So we have time for a quick question. I will pose one question as well. Can you explain the decrease below baseline in that slow modulation condition? So I thought that was quite interesting. And I was wondering
these
whether,
ones you mean?
Yeah, so you saw this like, yeah, I guess
that is, I think the whole system.
B1 and C2 both show it, although B1
and C2.
Yeah, so right now you're.
Okay, so B1 is technically sinusoidal, right?
Right.
So it's a low frequency sign. When you have this negative field, it makes any neuron that is active to be silent. Mm-hmm. So this negative is because of that.
That's to be expected then.
Yes, that is to be expected. But
in C2,
these ones, for the C2, these negative ones are because of the adaptation mechanism. Mm-hmm. Because technically in amplitude modulation, we don't have a negative one.
Right.
So what it is because in this phase that neurons are so active, there is an adaptation mechanism that kicks in and start, because that is inhibitory, silence everyone. And because that is slow, it takes some time to go back to not being active. And that caused this negative, let's say, reduced in power of gamma.
Yeah.
Which again, this is the same technically as the end of termination of the stimulation.
I see. It'd be very interesting in like a mouse model to do a pharmacological intervention and see whether that prediction would bear itself out. That would be really nice.
Yeah, I guess we were suggested to do some, use some GABA B antagonist to see if that is going to change the behavior or not. But yeah, that would be a really interesting experiment to do.
Yeah, cool. So we're going to move on to the last talk of the session. Thank you again, Zainab. That was a wonderful talk. And our final talk is Katarina Duker, and her talk is titled "No Evidence for Entrainment: Endogenous Gamma Oscillations and Rhythmic Flicker Responses Coexist in Visual Cortex."
Thanks, Adam. I just got kicked out of the session. I hope my internet connection will be stable throughout this talk. Let me just share my screen. I hope you can see the presentation now.
We can see it, yep.
Yeah, great. Okay, so yes, as Adam said, I looked into entrainment of endogenous gamma oscillations in human participants, and the paradigm we created was meant to both induce intrinsic gamma oscillations, strong intrinsic gamma oscillations, and a flicker response. So yeah, this is a bit of a trailer to a preprint that I uploaded beginning of September. So if you're interested, have a look under the QR code. We were able to look into two phenomena, oscillatory phenomena, being entrainment and resonance, but today I'm going to focus on the entrainment bit. Right. Yes. So like I said, it's we tested human participants, so we recorded oscillatory activity using MEG, and we had two conditions. One I'm going to refer to as the flicker condition, in which we simply flickered a patch on the screen with average luminance of the matched to the background. So for high frequencies in particular, this should be basically invisible. It looked like there was nothing on the screen, and we used sinusoidal flickers from 52 to 90 hertz in steps of 2. So we had 20 frequencies. The other condition, which is going to be the focus of this talk, I'm going to call flicker in gratings condition. So again, we had a 1 second baseline. Then we presented a moving grating stimulus, which has been shown to induce very strong gamma oscillations in visual cortex. And then we superimposed the flicker onto these gratings. So we were flickering the light rings, and then concluded the trial again with just the grating stimulus on its own. And this portion of the trial here, these 2 seconds of just the grating stimuli, we used to identify for each participant the individual gamma frequency. Now, like I said, I'm going to talk about the entrainment part. So the question we wanted to answer is, does rapid flicker entrain endogenous gamma oscillations? So we hypothesized that when we present a flicker, that the frequency of the intrinsic oscillator would synchronize to that flicker. So the power at the individual gamma frequency should decrease. And same for the phase. We hypothesized that the phase of our gamma oscillator would also synchronize to the photic drive. And as you may know, when we talk about entrainment, either in dynamic systems theory or neuronal entrainment, that works best if we get close to the endogenous frequency of the oscillator. So here this is shown for individual alpha frequencies in a study by Annika Notbohm. And we see that entrainment is strongest the closer we get to the frequency of the oscillator. Now, the messages I want you to take from that is that in our case, we don't find that the photic drive is able to capture the gamma dynamics. So we neither find effects on frequency nor phase of the gamma oscillator. But what we do see is something that looks like a coexistence of both responses in visual cortex. So here I'm showing the time frequency representations for the sensors of interest, which were the sensors that picked up the gamma oscillation the strongest. And if we look at one individual participant, here in these trials, we flickered at 52 hertz, as you can see here, and the individual gamma frequency was 58 hertz, as indicated by the dashed line. So I'm showing relative power change compared to the 1 second baseline at the beginning of each trial. And we see very nicely, we have a strong gamma oscillation at individual gamma frequency with an increase of 300% throughout the presentation of the stimulus. Now, when we turn the flicker on, we would have expected this frequency to be drawn towards the flicker, but instead we see that gamma doesn't really seem to be bothered by this 50 hertz stimulation. It just happily continues and stays on until the rest of the trial. Now, when I look at the grand average, before calculating the grand average, I aligned the frequencies for all participants to the individual gamma frequency. But we see the same picture basically. So obviously this is spectrally smooth, but again, gamma is happily oscillating at its individual gamma frequency and doesn't really seem to be drawn to the flicker frequency. So that was surprising to us. Now, this is obviously sensor data. Now, when we went into source space, here I localized the location of the gamma oscillation. So the strongest voxel showing the individual gamma frequency without the flicker, that is, and the flicker response, which I located using a just the flicker condition trial. And what we can see already here in the slices is that it looks a bit different, but not the strongest location looks a bit different, but not terribly different. But indeed, when plotting the M&I coordinates for each participant, so each scatter is one participant and one condition. The big scatters indicate the grand average coordinates. The individual gamma location seems to be inferior to the flicker location. And indeed, this difference along the inferior superior axis in visual cortex is significant and was at an average of about 9 millimeters. Right. So yes, like I said, I'm focusing on the entrainment question. I've brought the resonance. I brought a lot of data in the appendix, so if you want to talk about the resonance stuff, then I'm happy to answer your questions. And I would like to thank my co-authors and advisors who've guided me through this project, mainly, of course, my PhD supervisor, Ola Jensen. And obviously, I'm thanking you for your attention, and I'm happy to take your questions.
Great. Thank you, Katerina. That was a great talk. It's super interesting data, and I had a few questions. So if anyone else has questions, you can put them in the chat. So I had 2 points where I was interested in like particular pieces of your data. One was in the grand average spectrograms.
Mm-hmm.
It looked like there was more representation of the gamma frequency from the flicker in the case when it was below the IGF rather than above the IGF. It might just be the like way that you've marked with the lines where the stimulation frequency was, though. But it looks as though the band in that the band of flicker frequency in the upper set of plots is darker than the band in the lower set of plots. Is that just my eyes playing tricks on me, or is that a real effect?
That is definitely true. So that is another thing. Let me see. So we would have expected, based on resonance phenomena, that when we
that plot that you just showed from the previous data looked like it would be symmetric.
Yeah, so I think this might depict it a bit better. So what you can see here is when I plot relative power change at the flicker frequency as a function of frequency, I see that, oh, this should be stimulation frequency just. I find that the relative power change decreases as a function of frequency. So what you saw in the time frequency representation is definitely true. The amplitude of the flicker response is larger when I flicker at a lower frequency as opposed to a stronger frequency. I did not find any correlation with the individual gamma frequency, though. It's not that the gamma frequency is sort of the limit, and that's as high as I can go.
Is this frequency, so this is flickering really, really fast. Is this actually consciously perceptible at the, okay, so it's not something they can perceive.
I mean, we are not there. We have papers where we call it invisible in our lab when we use this method, because I went down to 52 hertz. It was visible. Some of them were, but for flicker frequencies above 60, you cannot really see it. So that is quite nice that we still get a nice response. So we can do a lot of things with that, but unfortunately we don't seem to be able to do entrainment with it, which would be nice. But
yeah, the other thing that I had a question about was with the source localization. What do you make of it being different? If you can go back to that piece of data, it looks like as you might I would have expected the flicker frequency to be more on the foveal pole, like the occipital pole, like more purely foveal. It looks like it's actually a little bit more in the upper bank. So perhaps more you're seeing more of the flicker represented in the lower visual field. Is that just the way that you're plotting the data? And then the IGF is actually really looks like it's right on the occipital pole, like right in foveal V1.
I think it's the other way around, actually.
Is it the other way around? Okay.
Yeah, it's the flicker is here, or at on the z-axis it's at 0.
Okay, sorry. I wasn't actually looking in I was looking at the brain maps, not actually the MATLAB or the whatever the 3D scatter plot there.
So the gamma oscillation seems to be a bit more spread out, and the flicker response is a bit more focal.
I see.
That is one thing. What I didn't tell you is, so when I look up the coordinates using Brotman areas, I do find that the flicker seems to be in V1 and the gamma oscillation seems to be in V2. I did not get into that because when I use a different, and obviously this is not peer reviewed yet fully, when I use a different atlas, I get the Culturine sulcus for both of them, even though it's different areas. But if we would go with the Brotman areas, then we could interpret a bit more as, I don't want to say top down, but it's in a higher order area in the visual hierarchy, whereas the flicker seems to just be driving visual cortex and not really be passed on.
Have there been other studies that have tried to localize the endogenous gamma frequency oscillations of visual cortex? Have they found similar like diffuse, not specifically foveal V1 type of effect?
Yes, so one of the first ones, which is by Nienke Hockenboom, that uses this paradigm, they found V1 and V2 because it spreads out so much. So that's why I don't want to emphasize too much on the whole V2, V1 story yet. And another interesting thing is that the flicker response and the IGF they could be in different cortical layers. So the gamma oscillations are more in layer 2, 3, whereas the flicker is more in more found in layer 4, which also makes sense with the whole. It's just a retinal. It's just you are just hammering the system with a visual with a photic stimulus.
Yeah, that's really interesting. I mean, it definitely looks like a more diffuse signal for the endogenous oscillations. So yeah, maybe it's not worthwhile to like precisely localize the peak of it, but just say it's more general. I think you're hedging your bet. It seems to me that you're hedging your bet in the right direction. But this is really cool. It's really great work. So I think that's all the time that we have for this session. So I will adjourn here. But thank you again to the 3 speakers. They were all 3 great, very clear talks. Thank you to the audience for listening, and we hope to see you at future NeuroMatch sessions.
Yeah, thank you so much, Adam. And thank you, everyone. Nice to meet you.
Thank you.
Thanks, Adam.
Bye, everyone.
Of developmental dysfunction of part dopamine interneurons or neural circuit function. So welcome, Renata, and the stage is yours.
Thank you so much. Thank the organizers for putting this beautiful session together with great other speakers on the topic. And thank you, Jota, for hosting me. So it is my pleasure to share this recent new work from my lab. So my lab is very junior, and this is actually the first time that I'm talking publicly about the work that we have recently developed. And as Jota said, it's about the impact of the developmental dysfunction of pre-development interneurons in circuit function. Sorry, I'm having some technical issues. Maybe. Okay, so as all of you know, the brain is constituted by a huge variety of cells. Those cells form here networks, and these networks eventually form circuits in different regions that connect with each other and convey information and allows the brain to do its job. Now, the reason why I like this cartoon is because the cells that constitute the brain, and in particular the cortex, which is what I'm going to be talking about, are very diverse. And they come in very many flavors of different kinds of patterns of activity, different kinds of connectivity, different kinds of markers they express, transcriptional profiles, and etc. So that has been a major sort of focus on my career, is being to understand how different inhibitory types contribute towards cortical function throughout development in adult cortex. And so I just told you that the cortex is composed by a wide variety of neuronal cell types, but they can be sort of compacted in a few main classes, or a few, as Gord Fischel would say, cardinal classes. And those cardinal classes include, the vast majority include provalbumin cells, which are inhibitory neurons, so express GABA, as well as do somatostatin and VIP. But they have a very specific feature. So they are fast spiking, meaning that they fire very fast. And they are perfect for vetoing runaway excitation because they target the soma of pyramidal neurons, also target other interneurons. But this somatic targeting make them very precise in terms of timing of their inhibition. Provalbumin interneurons receive input from the thalamus, as here it is, as well as local input, and send local output, mainly to pyramidal neurons, but also to other interneurons. And they have been widely involved and widely studied in mature circuits, but they're studied in developing cortex as being somehow difficult due to the lack of a way to actually label and target and follow the activity of these cells early on. Okay, so the question that I'm going to talk throughout this talk is, how does PV interneuron dysfunction contribute for disruptions that might be involved in neurodevelopmental disorders? Now, neurodevelopmental disorders is a very broad term, so you might be thinking, what are you meaning by neurodevelopmental disorders? But so there's one of the things that have become really obvious across the last recent genetic studies in human patients is that there's a great deal of overlap between genes that confer risk for a certain neurodevelopmental disease, let's say autism or schizophrenia, to also other diseases, such, let's say, ADHD or ADD or etc. So suggesting that there are some genes that somehow are sort of hubs for conferring neurodevelopmental diseases across many of them. Now, other interesting point that came from studies of other labs is that those genes are preferentially expressed from mid-fetal brain to sort of like the first soon after birth in humans, which in mice correspond to the first weeks after birth, and the fact that they are expressed at this time and they confer this sort of general propensity for neurodevelopmental disorders led us to hypothesize that there must be some sort of patterns or motifs of disruption in the cortex that it goes across different developmental disorders that are linked to neurodevelopmental disorders, and that would be the underlying cause. So the idea for us was not to focus in one gene, in one disease in particular, but actually find a gene that sort of encapsulates a variety of neurodevelopmental disorders. And so to do that and to address the question, how does the impact of provalbumin inhibitory neurons affects the maturation of the cortex and then process of sensory information in particular is affected in neurodevelopmental disorders. And to ask this question, we decided to perform a loss of function of a gene called MEF2C. Now, MEF2C is a gene that it's expressed in excitatory neurons, but is also expressed in provalbumin interneurons, and within those within the inhibitory neurons expressed almost exclusively in provalbumin interneurons. And we thought that it was particularly interesting because, as I mentioned, MEF2C is linked to a variety of neurodevelopmental disorders, including autism, schizophrenia, ADHD, ADD, and Rett-like syndrome, amongst others. It's expressed, so its expression is the best genetic predictor of a cell, an inhibitory neuron, become a provalbumin interneuron, even before they express the markers that constitute their identity. So MEF2C, as being studied, is also expressed in excitatory neurons. I'll just show you in a second, and it's being analyzed in the context of brain development, and is being shown to be involved in neurogenesis, migration, cell survival, and synaptic transmission. But all these studies were done in excitatory neurons, and there's really not much known about what this gene does in provalbumin interneurons. There's a haploinsufficient syndrome of MEF2C that leads to a variety of phenotypes associated with different neurodevelopmental disorders. And not only MEF2C confers high risk for autism and other neurodevelopmental disorders, but also it's a transcription factor. It is upstream of many other genes that confer risk as well. So that's why we decided to focus on this gene in particular to study how dysfunction of PV interneurons might lead to phenotypes, cortical phenotypes that might underlie behavior phenotypes in disease. So as I told you, MEF2C is widely expressed in excitatory neurons. Here you have MEF2C in red, and you can see it expressed in many cell types, and these are excitatory neurons, but it's also expressed in inhibitory neurons, and in particular, provalbumin inhibitory neurons. And you can see here that most of the provalbumin neurons express MEF2C, while the other two non-overlapping interneuron markers, such as somatostatin and 5-HT3A, which the three together are encompass the whole totality pretty much of inhibitory neurons, express very low levels. So within the GABAergic population is pretty specific. And I must say that MEF2C in humans is also expressed in provalbumin interneurons, was just recently shown. Now, knowing that, however, we set ourselves to study the hypothesis that MEF2C signaling shapes provalbumin interneuron development, and it is based on the motivation is based on it does in pyramidal cells, and then it's going to impact cortical activity and sensory processing. So before I jump into the data, I just want to give you a overview about what we're going to be talking when we talk about development, because I told you that MEF2C is involved in excitatory neurons, MEF2s are involved in many of these processes. So just a quick overview. GABAergic inhibitory neurons are born in the ganglionic eminences, the MG and the CGE, here illustrating from the MG, migrate tangential to get into the cortex, where they distribute across the lamina positions. They go through a period of programmed cell death that, depending on the cortical areas, can be between P7 and P15. And during this period, they are also spontaneously active. There's beautiful work from Rosa Cossart and Natalia Garcia de Marco that just recently published papers on that. And then go through, the network goes through circuit refinement. And for instance, in the visual cortex, which is the system that I studied that I'm going to tell you about, there's a lot of refinement that occurs at the time of eye opening or sensory input coming in. And so with this in mind, we set ourselves to study what happens when you remove MEF2C from LHX6 inhibitory neurons. So LHX6 is a marker that is expressed in AMG-derived interneurons, and this encapsulates both provalbumin expressing interneurons and somatostatin interneurons. However, because MEF2C is almost not expressed in somatostatin interneurons, we use this as a way to remove MEF2C from provalbumin interneurons almost specifically. So these are the mutants that we are going to be looking at, and we are going to cross it with a flux allele. And the first thing that became really obvious when we produce these animals is that the animals have behavior phenotypes or endophenotypes that are reminiscent of neurodevelopmental disorders such as ASD. For instance, they have this clasping of the paws in the back that has been linked to Rett syndrome. They have really reduced social preference both with the new sort of mouse that is placed on the cage or even with their siblings. But they also have deficits in communication and stereotypic behavior and sensory motor gating. And for the sake of time, I won't go through all of these, but they certainly appear to be a good model for NTD. Now, when we zoom into the cortex, into the visual cortex of these animals, what we see is that there's a reduced in the number of PV expressing neurons. Here you can see that in the mutant, there's like the cells are very faint, and these are almost overexposed cells. Even though the total number of cells is not massively reduced, there's a slight reduction, but they are not expressing the markers, suggesting that maybe they are not mature enough. And in the same line, they are also not expressing, they are not developing perineuronal nets, which develop in postnatal development and are thought to be important for synaptic transmission. So we saw those phenotypes and we asked, so what is happening to the provalbumin cells themselves in the cortex of these animals, and what is happening across ages. So most of the studies that we do are performed all the way from P10, to adult ages. I'm not going to show you everything for the sake of time. I will just show some snippets of some ages, and I will comment on what we see in the other ages. The one thing that we did was to patch by using slice patching from parvalbumin interneurons or parvalbumin interneuron precursors, cells that are still not expressing parvalbumin, but it will. And we also patch from excitatory neurons to ask what is the non-cell autonomous effect of removal of MEF2C in pyramidal cells. But I'm not going to show you much about that. But one of the problems of this project, and one of the problems that the field is having in wanting to look at the specifically parvalbumin cells early on, is that the only markers, the Cre drivers that we have are the PV Cre markers that the individual cortex start to only be expressed around P15, and there's no reliable recombination till somewhere between P25 and P30. So in order to be able to visualize these cells and also to manipulate them early on, we established a collaboration with the labs of Dimishstein and the Fischer lab, and have done. This is a big shout out for their labs, for the Dimishstein lab, that has created this beautiful, beautiful tool called the E2 virus, which uses an enhancer of that is specific for PV interneurons, and therefore allows you to, by using a virus, to selectively target PV interneurons in adult, but also during development ages, and with a high degree of specificity, as you can see here. This also allows you to look at beautiful morphologies of these cells and see how they mature over time, and to identify these cells, once, to manipulate these cells with optogenetics. Here we have optogenetic activation of fast spiking cells, and you can see that it's responding to light. And here we have in the same animal the recording. So this is an in vivo recording. These are raster plots from a RS cell, which is a putative pyramidal interneuron, saying that is being suppressed when you activate PV interneurons. So this tool is beautiful and works and allows us to actually target PV interneurons early on and see how they mature. And so what happens, so what I'm showing you here is a spontaneous EPSCs, so which is in order for us to evaluate the glutamatergic input that control and mutant cells are receiving, and these are parvalbumin cells, or that will become parvalbumin cells. And what we can see is that the frequency of the spontaneous EPSCs is massively reduced. And this was true starting as early as, P7, and kept being true throughout the life of the animal, suggesting that the synaptic transmission, at least glutamatergic transmission to mutant cells, is massively reduced. Now, we also wanted to go all the way from looking at a cell type in the context of what happens in a slice to go and to what happens in the in vivo network. And to do that, we perform everything I'm going to show you is going to be using extracellular recordings in mice that are head fixed, that can move on a treadmill or a wheel at their own will while their activity is being recorded. And we are monitoring their behavior state or state of the animal by looking at the locomotion, the facial movement, and the pupil diameter. And then we are going to ask, how does this change in a parvalbumin interneuron connectivity and maturation changes the local network? And all the recordings are going to be done in the visual cortex. So again, here's an overview. We do our recordings using laminar probes, so we can probe the network activity across all layers. And then we can measure local field potentials, which is the sum synaptic activity of the network, a variety of individual cells that are split in regular spiking and fast spiking, putative pyramidal cells and putative parvalbumin interneurons. And depending on the state of the animal measured by pupil or by facial movement, we divide them in quiescent or alert. And so the first thing that became really, really clear when we recorded from those animals is that they have these events that are highly synchronous, very short in duration, and that oftentimes start around the deep layers or layer 4 and propagate to the other layers. And these mice are not seizing as far as we can tell. There's never ictal activity or even pre-ictal activity. There's not like muscular activity suggesting that these mice are seizing, but these have these patterns of activity that are very resemblant of basically immature. So these recordings are in the adult cortex, immature spontaneous sort of synchronous activity that occurs in mice as they are developing, and as you've seen by other talks by colonies and so because we are recording between pups and adults, and we are recording animals before even eye movement, we actually devised a new metric for the state of the animal. And here we are comparing pupil, facial movement, and locomotion, and these are the different traces. And basically this plot is just to tell you that if you correlate the increase in firing rate that you typically see with locomotion or increased pupil to also facial movement correlates extremely well with the increase of firing activity here in red, and actually antecedes even pupil dilation or locomotion. So it's like sort of the most fine metric that we have, and that's what we are going to use for measuring alertness in our pups. So here we recorded from, these are the power spectrums for local fault potentials in control and mutants. And what became really striking is that when you look at their power spectrum, it is really completely different according to state. So typically in the controls, you see that high frequencies are dominated during periods of alertness. But in the mutants, that pattern was totally inverted. And here you can look at the quantification of gamma versus delta. That is like the total amount of gamma versus delta is similar between controls and mutants. But in controls, you have an increase of the gamma rhythms during alertness, and in the mutants that was totally inverted. And that was true for every animal that we recorded. Then when we look at the firing rate of individual cells, we were expecting actually that, given that we think that PV interneurons are not receiving synaptic input, there was going to be like this, we can be very silent cells, and then we would have massive basically firing rate increase in the pyramidal neurons. And here I'm showing you the firing rate of individual pyramidal neurons in controls and mutants. And you can see that there's not an increase in the firing rate in mutants. If anything, there's a mild decrease that we still have to record from our animals. And the same was true, like the opposite result also showed to be true for when we recorded for probably interneurons. We're expecting that their firing rate would be reduced, and it's not significantly different from the controls, but if anything, there's a trend towards increasing. And when we compare between quiescent and the alertness, like the trends are similar, independent of the state of the animal. Now, what was really fascinating is that when we start to perform cross-correlograms between regular spiking cells and resolute spiking cells, which is pyramidal cell, pyramidal cell in controls and mutants, or fast spiking cells in controls and mutants, or regular spiking cells, fast spiking cells, and again, fast spiking cells are pyramidal neurons, we saw that in any regime, there's a much, much stronger correlation between independent of state in the mutants. So here it is like an example cell, but that difference was really more striking between pairs of pervalbumin interneurons. Not only it was striking, but it was really increased during the state of quiescence, suggesting that during the state of quiescence, there's this input that arises to the fast spiking interneurons, and they are very, very connected and firing together. So that suggests that even though the overall firing rate of these populations is not changed, their synchronous activity it's very, very altered, and their fine temporal patterns of activity are destroyed. Now, Manal Salmi in the lab performed the same experiments in pups, and these are quite challenging, so a shout out for her experiments. So before eye opening and after eye opening, and we are doing these longitudinal analysis across ages. I'm going to just give you a snapshot. I think this is from sets of animals that are P15, or somewhere between P15 and 17. And what you can see is that contrary to the adult ages, in the controls, in the pups, we don't see this. So we see that we don't see this switch on the local field potential according to state. What you can see is that if you compare the quiet state LFP gamma over delta between control and mutant, it's reduced in the mutant, suggesting that overall there's less gamma. But the state modulation is still present. So the take-home message is that mutants have less gamma relatively when they are pups relatively to delta, but their state transitions are still sort of like in line with the general tendency of the controls. Now, when we recorded from spontaneous activity in terms of firing rates again, we didn't see any major difference. It's significant, but it's very, very mild. But when we look at the synchrony between by performing cross-correlograms between two parvalbumin interneurons or between parvalbumin interneurons and pyramidal cells, we saw that they are firing much more synchronously. So suggesting that they are like their connectivity is altered and their fine temporal patterns of activity is altered even at this time point. And we are really interested to compare this, what happens at these ages to what happens before eye opening, which we are currently collecting the data for. Now, I told you that we are doing these experiments in the visual cortex, and one of the reasons is because not only can look at spontaneous activity, but we can also look at visually evoked activity and ask how does sensory input that arises in a context in which PV interneurons maturation is truncated, how does it evolve? And so we have the mice still, in awake recordings, but now they are looking at the drifting ratings. And here, actually, I'm showing first the data of the pups, just because I think this is quite remarkable to show that by P15, both controls and mutants have cells that are visual responsive, and they are, pretty well tuned. So here I'm comparing regular spiking cells in control and in mutant and fast spiking cells in control and in mutant. And then we ask, what is the responsiveness of the cells to visual stimuli? And what you can see when you look at visual responses, which is basically what is a response to the stimulus relatively to baseline, there's not a big difference in the responsiveness of cells that are excitatory neurons. But parvalbumin interneurons are much less responsive, even though, and this is like really interesting, they are more responsive to visual stimulus at these ages than the pyramidal cells, but not in the mutants, again, suggesting that there's some impairment in the maturation of the parvalbumin interneurons or some deficits in the connectivity from visually that goes all the way from the retina to the cortex. And when we look at the orientation selectivity index, so we are looking at a variety of receptor field properties, such as orientation selectivity index, spatial frequency distributions, contrast response, but I'm just going to give you a snapshot. So what we saw is that cells overall at this age are very well tuned already. And here I'm showing you an example of a mutant cell that is a regular spiking cell, so a putative pyramidal cell, but in the mutant, the orientation selectivity was massively compromised or at least delayed.
Renata, 5 minutes.
Now we zoom in and we look at the visual responses in the adult animals. And here I'm showing just an example of a response that is a regular spiking cell to visual stimulus in the mutant. And the reason why I'm showing this cell is because one thing that we are analyzing is the patterns of responses that we have in the mutants. And one thing that we see very consistently that we don't see in controls is this like offset followed by an onset of response. And we are also looking at the local field potentials that look very different between controls and mutants. But we are doing a variety of fine-tuned analysis, both to describe the quality, so the the type of responses that we have, and then quantify those type of responses based on firing rate and local field potentials. But just as an highlight of a take-home message, what I want to tell you is that the visual responsiveness of controls and mutants is not massively changed, even though goes in the opposite direction than what you see in the PUPs, suggesting that there's a progression of the network towards, being more responsive to visual visual input. But one thing that was obvious is that the noise correlations, both in the quiet and in the alert state, in the mutants, was massively increased, suggesting that the sensory processing is going to be much more compromised. And right now we are following up with many other experiments along those lines, but we are also preparing these mice to do a visual task and ask how these changes in noise correlations and responsiveness of the cells impact the detection and the discrimination of a visual stimuli. And this is a story to tell another time. And so, because I'm about on the timeline, so I would like to summarize to tell you that we performed, so we were interested in having a way to disrupt maturation, particularly the postnatal maturation of parvalbumin interneurons, and ask how does that integration in the local circuit impacts sensory responses and also state-dependent responses. And what we show is that by removing this gene MAF2C that has been linked to a variety of NDDs, we changed the maturation of the PV interneurons. I did not show you all the data on these, but we believe that their connectivity is highly impacted, particularly thalamic connectivity, but we are working on that data set, and that's part of their developmental problem. That change in the maturation of the network leads to changes in the patterns of network activity that basically do not reply anymore, or even invert in terms of the state of alertness of the animal. It's like if the circuit does no longer know what is the overall state of the animal. And that emerges soon after eye opening, even though we are still curating the data for before eye opening, and evolves over time and leads to changes in the visual responses, mainly by this like massive increase in synchronous activity that exists in controls and in mutants very early on, but it appears that in a mutant, in controls, the natural process is that these synchronous events to cease, and then synaptic input comes in, and you don't have like these basically bursts that you typically see during development. And in mutants, seems that somehow they are stuck in that developmental stage, and perhaps is the underlying cause of all the other phenotypes that we are seeing. But this is so far just a hypothesis that we have. And so with that, I would like to just thank you for listening to me. And I would like to, give a big shout out for the people that actually did this work. So this work was done in a really close collaboration between 3 persons in my lab. Claire Ward, which is a PhD student and did most of the recordings and the data collection and many of the data analysis for the adult data that I show you. Ehsan closely worked with Claire to do a lot of the curation of the analysis of the data, and Manal, which did all the recordings of the PUBs and also contributed for data analysis. Now, this work could not be done without our collaborators. My always most close collaborator is Luke Scholzen and the Scholzen Lab. It is Luke, and we have we share many of the facilities of our lab, and we share intellectual property, and we share resources and etc. So it's been a really privilege to work along with him and his lab members. And then the Mishtain, Fisher Lab, and Castillo Lab are all collaborators that. The Mishtain and Fisher Lab contributed for the virus that allows us to visualize and manipulate PV interneurons early on, and the Castillo Lab is working with us in the slice electrophysiology, and this work was funded by the Simons Foundation.
Excellent. Thank you very much, Renata, for a wonderful presentation, which was also right on time. I don't see any questions from the audience. I hope you know how to do it. You just have to use the Q&A option and ask a question. Or you can, I don't know, raise your hand, and then I see a raised hand and invite someone in. So don't be shy. While you think, I start asking some of the ones that I have. So Renata, you said that you delete the EMF2C gene in juveniles, and then you see an effect in both social behavior and synaptic transmission, right? What happens if you delete it later on in the adult animal?
Yeah, so not much actually. So we are still doing the fine analysis. But so we did a variety of experiments, but one of them is that we remove it MEF2C PV3 driver, and that removal occurs somewhere around P30, even though PV is expressed early, but it takes some time to get rid of all MEF2C. And we don't see much phenotypes. We don't see. So it seems that this is a very whatever MEF2C is doing there afterwards is not doing something striking that we could catch with any of these phenotypes. So the local field potential looks normal, the frame rate looks normal, the visual responses look normal. So as far as we can tell, this is really a developmental effect rather than a mature effect.
I see. And what is the window within which you still see an effect?
So we start to see, so the earliest that we have is right now data analyzed is like around P15, and we see the effects that I show you, and with some degrees of variety. And the older animals that we have that we recorded are adults, and they have phenotype till they die. In terms of like what is the earliest that we can remove and not have a phenotype, I don't have the answer for that. So I know if you remove it early on during development, we have these striking phenotypes. If you remove it in the adult, we don't have any phenotypes. And right now we are doing the experiment in which we remove it in between, but I still don't have the answers.
Okay, we have one question from the audience from John Carlo Compista. John, would you like to ask live? If so, please raise your hand. Otherwise, I'll ask for you the question. Raised hands? No raised hands. So I will ask the question then. I would like to know about the future direction of studying MEF2C in autism. Is it going to be promising?
I think so. I mean, the MEF2C, deletion of MEF2C is syndromic. It's like a syndromic, leads to high syndromic autistic behaviors in humans. But SNPs in MEF2C also are strongly correlated with the phenotype of autism. So, so I think that's a direction to follow. I think in genes that you're looking at that are transcription factors that not just affect autism, but also affect all the neurodevelopmental disorders is really hard to pinpoint whether it's, autism or other diseases, mainly in mouse, in which, obviously we don't have the fine diagnosis, or not at all, that we have in humans. So I think the way that I see it is that it will impact the study of autism as so far that you're looking into basic underlying causes of autism. But those underlying causes are probably shared with other diseases.
Okay. Thank you very much, says John. Okay. Oh, hold on. That was answered. You cannot see. Oh, sorry about that. I don't think I can do something about it.
It's okay.
You cannot see the audience, yeah. So hold on, let me see if there was another question from the audience. No, I don't see another question, but I have written a few here because this is a subject that we're working in the lab, and I'm interested in it actually, the effect of PV interneurons on circuit dynamics. And okay, so another question that I had, when you see that the balance, the excitability doesn't change of these interneurons, why do you think is that the case? Is it because of some homeostatic mechanism that it's taking place?
Yeah.
Is the intrinsic excitability different?
Yeah, we really don't know. I mean, this was really puzzling data. So when we saw that with the slice electrophysiology, that the synaptic inputs across ages, and that was true later on too, are really reduced into PV interneurons, right? So you expect that PV interneurons are not firing, and then they're not going to be doing their job, and excitatory neurons are going to be firing a lot, or maybe there's compensation by other interneuron types, as you say, and regulates that. Our data does not suggest that, because in our data, when we record extracellular in vivo form provalbumin interneurons, they are still firing at similar ages. So what I think is going on is that they are communicating through other mechanisms, not just synaptic transmission. And there's some lines of our data that suggest they are communicating through gap junctions. So this is very preliminary, but that's like taking all together. So it looks like these cells in the mutant, they are very synchronous together, and not just that, but when we feel one mutant cell, a basket cell that is that is going to be expressing PV, you see many other cells that pick up the dye in ways that suggest that they are really actively engaging in gap junctions at later ages, which usually does not occur in the controls.
And you think that's why it is state dependent? I mean,
the state dependency, so we are looking, so to ask that question, the best guess that I have is that there's going to be, well, there's two possibilities. I mean, there's many, but there's two hypotheses that I think are the most likely. One of them is that, so during spontaneous activity, the networks are much more synchronous and are going fluctuating between these up and down states. And maybe, just like when there's like these up states, it's like these cells communicate amongst them and lead to these like sort of like there's a delay in the timing between excitatory and inhibitory inhibition that leads to these like sort of like synchronous events that then don't stay for very long because then the other interneurons kick in. The other possibility is that the connectivity into provalbumin interneurons is really changed. So, and the reason why we hypothesize that is that those events, those synchronous events, are also driven by sensory input, not just by the state of quietfulness, but also when you give a stimuli very early on. And so what I think is going on is that the synaptic input into provalbumin interneurons in the mutants is changed in a way that they are not receiving as much thalamic input and maybe not as much cholinergic input or other inputs that are from neuromodulatory areas. And so we are right now using rabies tracing in collaboration with the Official Lab to address that question.
I see. Okay, that's really exciting. I'm very interested to find out the answer to all these questions. Perfect. Okay, I hope there is no problem with the audience. Because I don't see much activity either in there, as you state. So, is there any other question from the audience? So, if there is a problem with asking, just let me know. Okay, I don't see anything coming up. So I hope it was not a problem in terms of communication. But anyway, it was a wonderful talk, Renata. Thank you very much for joining us.
Thank you. It was a pleasure.
And we wish you a wonderful evening.
You too. Thank you.
Okay. Bye.
Are going to talk about motor control of complex objects, and I'm really excited for these talks because it's really rare to see people do research on complex object motor control. And all three speakers are actually from Action Lab at Northeastern University. Our first speaker today is Dr. Marta Russo. She's going to talk about manipulating a whip, motor control beyond reach. She's a postdoc at Northeastern University in the Action Lab working with Dagmar Sternad. So without further ado, I'll let you take over, Marta.
Okay, let me share my screen, and thank you for the introduction. I hope you all see my screen correctly. And today I'm going to talk to you about a task, and I hope that I will interest you with a very exotic task, which is manipulating a whip. And a lot of credit of this project needs to be given to Alexei Krotov, who actually graduated with this project for his master in the Action Lab. So the rationale for this study is twofold. And we wanted to investigate, the first reason is that we wanted to investigate complex dexterity in humans. Because in motor control, a lot of study focused on reaching or catching and throwing, which are all tasks that, although require a lot of coordination, at the same time are not reflecting this broad spectrum of human dexterity. And in particular, what we wanted to investigate is human dexterity with flexible objects. Because in our daily life, we deal with the flexible and under-actuated objects. And that's what we wanted to investigate more in this study. On the other hand, in our daily life, we intertwine different actions that can belong to the discrete or the rhythmic framework. A discrete action could be a throw or a catch or even a reaching movement. And during our daily life, we're actually performing a lot of rhythmic actions, so like walking or running. And we believe that we need to investigate tasks that can put together these two frameworks and have a comprehensive theory that can investigate both kinds of actions in our motor control. And in order to do that, we decided to ask participants to manipulate a bullwhip, which is indeed an under-actuated flexible object of a high complexity, as it has hundreds of degrees of freedom. However, humans can actually use this object, and as you can see in the video here, we can use it with a very high precision, as April is doing, like playing Jenga. And taking inspiration from this task, we ask participants to hit a target with a whip. And to ask our second question, we ask participants to perform the task in a discrete and a rhythmic fashion, where discrete is a task, an attempt to hit the target with pauses in between different attempts, while rhythmic is a continuous set of actions, one after the other. So in particular, during the task, we collected the kinematics of the arm and the whip, and 16 participants were recruited and performed 5 blocks in each fashion, discrete and rhythmic. The task was set to be, the setup was set to be in order to have a 5 centimeters overlap between the tip of the whip and the target, which means that in order to hit the target, participants had to have the arm and the whip both extended in order to have a successful trial. We measure participants' performance by measuring the number of times they actually hit the target during the task, and we call that the success rate. And we also evaluate their performance by measuring the error, which is actually the minimal distance between the tip of the whip and the target. And as you can see in this figure, there is a larger inter-individual differences in the performance. But however, the discrete rhythmic performance also show a difference. And indeed, if we put together this result by pulling together the results of discrete and rhythmic and all subjects together, you may notice that participants achieved a better performance in discrete than in rhythmic. And this was confirmed by the success rate that was higher in discrete and the error that was lower, together with its own variability. And these results were confirmed in all blocks. So all the 5 blocks and all the 3 metrics. Then we asked, what was different between these 2 styles? So what were the participants doing that led to such a different performance? And first of all, we wanted to investigate the hand kinematics. So we noticed that due to the complexity of the object, that also the arm and the hand were moving in a simple fashion, but they had different phases during the motion. In particular, participants set the throw before actually attempting to hit the target, and then they actually threw the whip until they actually unfolded the arm and the whip in order to reach for the target. We wanted to look for these phases into the hand speed profile. And we define the first phase of the motion between the preparation onset and the throwing onset, defining the preparation phase, which is the first peak that you can see in this velocity profile. And after that, we defined the throwing phase as the second peak of the bell speed profile that was ending at the time at which the whip reached the minimal distance from the target. And I'm referring to these two peaks in the speed profile as this was a characteristic that many subjects had during the task. And this is what I'm going to show you here, where you can see the velocity profiles for all subjects from 1 to 16 in the circle around, and the two colors indicate the two different styles, discrete and rhythmic. You may notice that all participants had these two peaks in their speed profile, and that's also confirmed in the average that you can see here in the center panel. What we also notice, however, putting on this trial together, is that while the throwing phase was very similar between styles and between subjects, which is the second phase here, actually the first phase, which is the preparatory phase, was longer in discrete. And this was confirmed by the statistical analysis, and you may see that indeed discrete had a longer preparation phase as here in this inset. We believe that this affected participants' performance, and we believe that having more time to set the whip before the throw could actually help participants to reach a better performance, to achieve a better performance. But then we also asked how the whip needed to behave in order to reach the target, and what were the key features of the whip during the task. In particular, we realized that the whip during its motion was moving and sometimes was entangled, but then it could unfold close to the target and actually hit the target. And that was that required a good timing, because then the whip can keep moving as in a follow through, and then even more be more entangled. And this was an example of a successful trial. But in a non-successful trial, the whip could unfold and still be quite entangled while it's close to the target, and then unfold when it's too late to actually reach the target, which means that this will lead to a less successful performance. We wanted to then identify these two time instances in the whip motion, and particularly defining these two states of the whip, which is the time instance of the maximum whip extension and the time instance of the minimal distance from the target. So we wanted to define these two time, and we wanted to compute the difference between these two time in order to understand if participants were timing the unfolding of the whip correctly in order to reach the target. And as you may expect, when we compute this time difference, we realized that in discrete, this difference was smaller than rhythmic, suggesting that participants were timing the whip extension closer to the time of the minimal distance from the target, which led to a better performance. In conclusion, I showed you that in manipulating a bullwhip, there was a large inter-individual variability in the performance, but however, all the participants achieved a better performance in the discrete style. We ascribe this result to the fact that participants used a longer preparation in discrete, and they were able to time better the whip extension in the discrete style. I haven't told you that there were some other interesting features in rhythmic, which also are very interesting, and in particular the fact that participants improved their performance in rhythmic that we didn't see in discrete, and that actually the unfolding of the whip was smoother in the rhythmic style, and this was presented by Alex yesterday here at Neuromatch. So what is the take-home message from this preliminary analysis? Well, is that we believe that timing is a key factor in throwing a whip, shown both by the preparation phase duration and the time of the whip unfolding. But we still want to investigate further what are the kinematic features of the hand and the initial conditions of the whip that are necessary to have a successful hit. And we want to further investigate actually what are the control strategies that are engaged in the two styles and that they lead to such different control strategies. With this, I thank you all. I thank all my lab, my PI, Dagmar Sternad, and in particular, Alexei Krodov, who actually collected and analyzed this data. So with this, I thank you, the Neuromatch organizer, and I'm here for the questions.
So, I'm going to applaud on behalf of all the viewers. We have a few questions for you, so let's get into that. The first question is from Andrea, and she asks, how did you track the whip kinematics?
So, well, that's a good point, because it's a complex object that requires complex tracking. So we tested different approaches, but at the end, in the end, we developed customized markers that were soft markers, and we sewed basically the marker onto the whip, and they were small sponges covered with reflected tape. And that was the best in terms of accuracy that we could achieve with a motion capture system.
Cool. The next question is from JJ Orban, and his question is, were there different phases in the learning? Example, exploration, learning, and then consolidation?
So we ask the participants, first, when the participants come in, we show them, so the experimenter played with the whip and let the participant play with the whip for a few minutes. And then there were the 5 blocks for each style. So basically each participant performed 10 blocks. So I would say that was maybe a little exploration, but everything was happening during the session. We definitely wanted to investigate also what happens if the participant is given more time, or maybe is coming back another day and keep playing with the whip. One thing that I might say here is that all the participants were basically naive. No one is an expert, but few of them had some little experience with the whip just by random happening. And we saw that they achieved a better performance, but we couldn't find, so far at least, any difference between participants.
Cool. The next question is from Julia Aldemir. And the question is, really cool talk. I would assume that having shorter prep in rhythmic would also be facilitating. You say it helped them prepare, but rhythm is such a repetitive fashion that they wouldn't need to have a second thought about it, even though they spend time to calculate the distance extension. I would think it would be for once. Okay, the question's long. You said they showed improvement in rhythmic, so the repetitiveness is the case that actually facilitates, I guess. So actually, didn't you say that they, okay, yeah. So I think the question is, did the repetitiveness of the rhythmic task help in the learning?
It could be. We were kind of surprised that we saw improvement in rhythmic, but they didn't manage to achieve the same performance as in discrete. So I think that the fact they were repeating the motion in the same block, so one trial after the other, helped in improving, but at the same time constrained participants in the in that loop of repetition. So maybe it didn't have to explore enough the solution manifold, so they couldn't actually improve enough. But again, we should have more sessions to actually say that.
Okay, let's take one more question before we move on to the next talk. Maybe we can have like a short Q&A at the end of the session as well.
I can also reply in the chat if it's necessary.
Yeah. Do I understand correctly that in the rhythmic task, the trials were connected so that the trials are not independent of each other? So it's
Yes.
like all one movement.
So the recording was in one block during the rhythmic session. All the trial were happening without a break, so one after the other. And so we actually decided to parse the trial in the rhythmic in order to compare it with the discrete. But we are also going to investigate the whole session all at once, because indeed there was a whole motion, a continuous motion. That's also why we wanted to investigate this rhythmic fashion. So yeah, I think the answer is yes.
Great. So I'm going to leave the remaining 3 questions for later. And in the interest of time, let's move on to the next speaker. Our next speaker is Reza Razavian. He is an NSERC postdoc fellow at the Action Lab at Northeastern University, working with Dagmar Sternad. And he's going to talk about human control of complex objects, stochastic optimal control feedback, and beyond.
Thank you. I was looking for that. Okay, thank you for the introduction, Edie. My name is Reza Razavian. I'm in the same lab as Marta, and she presented very exotic results on manipulation of WIP. I'm going to talk about manipulation of another complex object, and I'm going to give it a bit of a modeling twist. So before I talk about that, I want to mention that we have learned a lot in the previous maybe 2 or 3 decades about properties and features of human motor control system by analyzing and designing experiments in a 2D motion and analyzing point-to-point reachings. For example, we have learned about the temporal aspects of motor adaptation and its relation to error, perceived error, or we learned about the importance of having internal models and optimality principle in guiding these movements. However, this perhaps is a very large simplification of the movements that we interact and produce in our daily activities. For instance, this is perhaps an exaggeration, but in this case, it is no longer visible what is the error signal that drives human motor adaptation, for example. Or when we are dealing with these extremely complex situations, how much information is available and what is the internal model representing here? Or what is the objective that the humans are trying to optimize in a context of an optimal feedback control? So motivated by all this, our research group has, in the past few years, have moved into analyzing complex objects and how humans deal with these situations. And our previous work has shown, for example, that when there are complex under-actuated dynamics, people, for example, choose predictability over their effort, or they seek stability of the object, and they actively try to reduce the transient behaviors in these interactions. And Marta just presented that learning, for instance, in manipulating a whip, the subject could improve performance in one style, but they couldn't in another style. So this has been the motivation for our research group to come up with new experimental paradigms. And one of them is inspired by carrying a cup of coffee where the liquid inside this cup can create first an underactuated behavior, and the interaction of this liquid with the cup creates a lot of not unintuitive, but complex and potentially chaotic interactions that the subjects had to think about and potentially correct for or compensate for. To make things scientifically tractable, we have simplified this by still keeping the important nonlinearity and underactuated behavior of the system by replacing the liquid with a ball rolling inside a circular cup. The system is still underactuated that can create potentially chaotic behaviors that subjects need to react to. In the specific version of this cup task, we were asking first of all, the system were simulated in a virtual environment, and the participants were interacting with the simulated cup and ball by moving a haptic robot that would produce forces back on the participant's hand, and they could feel the effects of this ball rolling inside the cup. And the goal of this task, if I can play this, was to carry the cup from the start box to the target box, and they were explicitly instructed, try not to lose the ball, and the ball would roll forward, and if it had reached the rim of the cup, they would lose the ball and the trial would be a failure. We simulated 3 conditions, and subjects performed 3 blocks. In one block, the ball was essentially glued to the cup, and the cup and ball system formed a rigid object. In the second block, the ball was free to roll inside the cup, and this would produce these underactuated interesting dynamics. And the third block, we had another perturbation to excite the system even more, and the subjects had to kind of find a new strategy and preempt for this perturbation in order to keep the ball and finish the task. So I'm just going to first show you how the subjects performed in doing that. As expected, when the ball was fixed and the object was behaving as a rigid object, they were showing this bell-shaped velocity profile that was smooth. And this is what we would expect from manipulating a rigid object. Things get interesting when there is this underactuated effects of the ball, and clearly the subjects no longer follow the smooth bell-shaped velocity profile, and their interaction forces also change accordingly. And the last interesting behavior that we see in the subject's data is the way they react to this perturbation that is visible in both their velocity profile and the interaction forces. Then the important question is, what processes are in place or can give rise to the behaviors that we observe? And to answer this, we took a modeling approach. There are a lot of questions to answer here. For instance, what is the objective that the subjects care about? Does the stability of this underactuated system matters? What is the effect of the body and the object interaction dynamics? And perhaps what is the role of sensor information coming into the system? To answer a few of those questions, we referred back to the existing hypotheses in the motor control literature. One of the earliest models in this regard is a minimum jerk model that puts a lot of emphasis on the execution, planning, and perhaps execution in kinematic space. And it postulates that the movements are planned as a maximally smooth trajectory. Another class of models is dynamic primitives. And this model puts a lot of emphasis on the dynamic side, and postulates that the movements are generated as the evolution of a dynamic system that drives the body and the interaction with the environment toward the target. And one important component of this dynamic primitive model is mechanical impedance, essentially a spring and damper that is being driven as if by moving its free end and moving it in through a space. And that zero force or reference trajectory of this spring damper system is postulated to created by an attractor dynamics that produces a stable motion through time. But often this has been replaced in the existing models with the same minimum jerk profile for a discrete motion. Lastly, an important model in the motor control literature is postulating that there is no pre-planned trajectory in place, and the movements are essentially being produced through a fine-tuned feedback gates that act based on sensory information that drive the system to the target. So what I'm going to show you next is tailoring these models as a generative model that can predict behavior in manipulating the cup and ball. As expected, all of these models have been shown to be successful in explaining or recreating behavior when the object is simple, a rigid object. All of them are virtually indistinguishable in their prediction of a smooth bell-shaped velocity profile and the interaction forces. Things get interesting when there is this underactuated effect of the ball rolling inside the cup. The pure minimum jerk model is obviously unaffected by the ball and continues to predict a bell-shaped profile, whereas also including an impedance operator does not change this prediction much. But the optimal controller that knows the dynamics of this ball predicts something qualitatively different, and I want to point your attention to this region of the velocity profile. And this effort minimizing controller predicts that the ball, as the ball rolls forward, it produces a resistive force on the cup that decelerates it, and this controller does not oppose that in order to minimize effort. So some of the important messages here is that the object dynamics is critical and its knowledge is important. And another thing is that effort minimizing controller does not replicate the behavior well in this scenario. Another interesting behavior happens when there is perturbation. And I want to contrast the prediction of these two models, the optimal feedback control and the one with an impedance operator. And as you can see, the optimal controller shows very stiff behavior, characterized by a small change in velocity and large transfer of the perturbation force to the hand. Whereas the impedance, the model with an impedance shows much more smooth and compliant behavior as observed in human data. And again, the messages here is that mechanical impedance seems to be an indispensable component in interaction with dynamic objects. And the feedback loops through sensory information is not enough to capture that. So the way to try to create a model that can combine all these messages, on one hand, we have the information that the mechanical impedance is critical in interaction with the environment and dynamical objects. And on the other hand, we need to have a mechanism that produces the reference point or zero force trajectory of these spring damper systems knowing the dynamics of the arm and the object. And the solution we came up with was to take these dynamic primitive framework and embed, instead of having a pre-planned trajectory for the zero force trajectory, we use an optimal controller to generate these attractor trajectories on the fly without any pre-planned motion, using a fine-tuned feedback gain that acts based on sensor information. And now here we are faced with an option. So either to minimize the jerk or maximize the smoothness of this trajectory, or minimize the effort in driving this trajectory. And finally, to the punchline, as you can see here, the effort minimizing model, although it shows much better behavior post perturbation, it still predicts the deceleration in the cup that we do not see in participant data. Whereas the jerk minimizing controller penalizes changes in cup velocity and predicts behavior that is well matched by the data. And with that, I want to just summarize what we have.
So why don't you pull up your summary slides and you can start, just because we might run out of time, and we can answer the questions. So first of all, that was a great talk. The question is from JJ Urban, and he asks, would your results for the optimal control model not be different with real arm dynamics rather than a point mass?
That is a good question. I haven't tested that, but given, I'm just giving you my guess, given the similarity of the results that is often observed in other motor control studies that compared a point mass to the human data and also arm dynamics with the data, a point mass has been good enough in explaining or replicating the behavior. So I would guess it wouldn't change things a lot, but that's something I haven't tested.
Great. So if attendees have more questions, they can continue putting them in. We'll try to do another short Q&A at the end with all the speakers. But now let's move on to our next speaker. Our next speaker is Mohsen Sadeghi, and he did his PhD with Daniel Walpert at Cambridge, and he's now a postdoc in the Action Lab with Dagmar Sternard. And today he's going to talk about merging control strategies for the manipulation of complex objects. So over to you, Mohsen.
Thank you very much. Can you all see my screen? And is my voice clear? Okay, I assume so. Thank you. I'm very pleased to be here, and the same session as my lab members, basically. And I think I'm going to have a very easier job explaining after Martin and Reza. So I'm Mohsen Sadeghi, as Nidhi mentioned. I'm a postdoc at Northeastern University, and I'm mainly interested in the control mechanisms underlying that manipulation of complex objects. And in particular, I'm very interested in rhythmic and discrete movements and their interactions. So when we perform movements or manipulate objects, depending on the goal of the task, we might use different types of movements. So in swirling a glass of wine, the main goal of the task is to bring the wine into a circular rotation. Whereas when we bring the glass to the mouth, the goal of the task is to translate the glass itself without spilling the coffee. And there's been a lot of questions and a lot of studies as to whether these two types of movements arise from fundamentally different control mechanisms. And fMRI studies in humans have shown that different but overlapping regions of the brain are activated when performing rhythmic versus discrete movement. And when learning, people have shown that the transfer of learning from rhythmic to discrete and vice versa is not a symmetric transfer. And lately, people have found that even distinct functional modules, the motor cortex of mice, is responsible for rhythmic and discrete movement. So it seems that there are separate mechanisms underlying these two types of movements, but these mechanisms perhaps are interacting. And that's because in our real life activities, we often perform complex movements, which are not purely rhythmic or discrete, but a combination of both or a sequence of one after the other. And the question is, how do we smoothly transition between rhythmic and discrete movements when we perform complex movements? And we're mainly interested in that question in the context of manipulating complex objects and in the real experimental setting as opposed to virtual. So Reza talked about the virtual setting where he studied the cup and ball manipulation. The importance of studying real interactions with objects is that there are real sensoromotor consequences, and these consequences could actually affect our decisions. So if you make a sudden movement when carrying a cup of coffee, you may have to deal with real trouble. So I think it is very important to basically move towards studying more realistic experiments. Now in the lab, we don't really work with a glass of wine, merely because subjects just get bored and start to drink it. So instead we replace it with a cup, simple cup on horizontal plane with a ball rolling inside. And here is the experimental setup, a table with a smooth surface and the custom designed cup on top. Subjects can basically slide the cup onto the surface easily. And on the top of the table, we have a camera which records the movement of the object, and we use shape detection and color detection algorithms to basically measure the position of the object and to detect the shapes on the table which define the task. So the task that we design is fairly simple. So subjects start with rotating the ball in the cup while in a home position, similar to swirling a glass of wine. And then auditory cues introduced, after which subjects are instructed to translate the cup from the home position to the target position. And in this translation, they have to try to keep the ball rotation as steadily as possible and try to avoid the ball from falling out the cup, which is a possibility. And after they arrive at the target, they have to restore the ball rotation as steadily as possible again. So here it's me sort of demonstrating the task. We start with the rotation of the ball, the home position, and then the translation to the target position. And basically we go back and forth between the two. So we collected data from 5 subjects. This was basically right before the pandemic. And each subject performed 8 sessions on 8 different days. And we had 150 trials per session. So in the end, we ended up with 1,200 trials for each subject. And I'm going to basically show the data from all subjects in this study. Here we have an overview of the trajectory of the cup and the ball with respect to the inertial reference frame. I'm showing here about 100 example trials, and the black line here is just a single example trial. And we're going to mainly focus on the movement of the ball. So we can take the ball trajectory and represent it in the cup frame of reference and basically break it into 4 different sessions or 4 different phases of the trial. So the rotation phase, you have a very fairly stable rotation of the cup of the ball, and then there's a go cue signals introduced, and then the translation is initiated here and then ended here, and then the recovery basically when they restore the ball rotation. And we can look at the ball position at the critical stages during the trial, which are mainly at the go signal, when they start the transition, the translation of the cup, when they end the translation of the cup, and basically the movement time from the start to the end of the translation. We can also introduce a performance index to see how well the subjects were performing. And that's essentially, we can take the ball trajectory with respect to the cup, and at each point we can take the distance of the ball from the center and plot that distance as a radius of the ball position against the time. And as you can see, that the distance is fairly stable at the rotation phase, and we can basically take the average of that and take that as a reference radius for the rest of the trial. So any deviation from this reference circular movement is essentially the error of performance. So we can basically represent as a root mean squared error of performance. So if we look at this sort of error for each subject here on the vertical axis, I'm plotting this error across the trials. Each dot is one single trial, and the solid line is a running average with a 20 trial window. And 3 of the subject, 1, 3, and 4, basically show very stable performance in terms of changes in their performance index. So there's no change in the performance index. We see learning in subject 2 in a peculiar way where he basically figures out how to do it, and then the performance improved instantly, and we have a gradual improvement in the performance subject 5. And overall, if you look at the moving standard deviations, as a measure of variability, the variability goes down for most part, which is an indication of learning. But we're mainly interested in the transitions between the rhythmic and discrete movement. So how do we sort of look at those? Well, essentially, what we can do is we can look at the position of the ball with respect to the cup and represent it as angle or the phase of the ball with respect to this horizontal line. And we basically measure that angle for different critical stages of the trial. We can basically plot the histogram of those angles in a polar histogram such as this. So we can start basically with the phase of the ball at the go signal, and we plot the histogram of that. We basically find a very fairly uniform distribution of all the angles across all the trials. And this is not really surprising because at the go signal, subjects are not really expecting the go signal, so they don't have any control over the phase of the ball. And if you fit a simple von Mises distribution to this data, you can basically estimate the concentration value here. It's just a little sidetrack. So if you have a von Mises distribution, the concentration value is essentially analogous to the inverse of variance. So concentration of 0 means a uniform distribution, and as you increase it, you get more concentrated distribution around a certain angle. So the concentration value is very small and close to 0, meaning there's a uniform distribution when the ball for the ball at the go signal. But what about when they start the transition from rhythmic to the discrete movement? If we look at the distribution of that, we can see that there's a clear modulation of the ball phase when they start to transition to the discrete movement of the cup. So this sort of discrepancy between the go signal and the start of the discrete movement shows that subjects are actually actively modulating the phase of the ball in order to basically have smooth transition towards the discrete movement. So how about when they basically end and arrive at the target? So we can look at the distribution of the ball phase at the target, and we can also see a very concentrated distribution around a certain angle. Now, this might be different across subjects, but also within subjects, they're all very consistent in the angle of arrival. Now, we know that they modulate the ball phase when they transition from rhythmic to discrete movement, but can we look at this plot, which is the target, and say they also actively modulate the ball phase when they arrive at the target? Because another null hypothesis would be that subjects just choose one initial angle at the initiation of the translation, and they basically play out the discrete movement, and it just happens that a ball arrives at a consistent angle every time. And so this is not really what is intended by subject. It's just a product of choosing the right initial angle. And to answer that question, basically examine that question, we can just look at the translation time, where subjects basically translate the cup from home position to target position. And if we plot the translation time against the ball phase at the initiation of translation, we get a polar plot such as this, where the angular dimension is essentially the ball phase at the start of the discrete movement, and the distance from the center is the movement time, the translation time. And we plot those against each other, we see this interesting spiral pattern which indicates a linear relationship between the time and the initial and the initiation phase. And that is basically the case for at least 3 subjects, but not so much for 2 other subjects. So what this means essentially is that if subject starts with an initiation angle, let's say here, the movement time has a bimodal distribution. So they either arrive at a target at this time, or they delay the arrival for this amount to arrive at a later time. And this difference between the two basically happens to be close to the close to the period of one cycle of the ball rotation. So what this indicates is that subjects are sort of integrating the discrete movement into the rhythmic movement of the ball rotation when they basically transition back from the discrete to the rhythmic movement. So this indicates that not only they modulate the ball phase when they transition from rhythmic rotation to the discrete, but also when they transition from discrete back to the rhythmic. So the question now is, what is the advantage of a spiral sort of pattern like this? So to answer that question, we did a very simple simulation where we simulated a cup and ball rotation with a sinusoidal movement of the cup. And then at a given ball phase and a given time of the given movement time, we translated the cup with a minimum jet trajectory to the target position. And then we basically measured the position of the ball and its deviation from perfectly circular motion. So here I'm basically showing an example simulation where you start with the rotation of the ball, then the minimum jet translation, and at the end we basically measure the deviation of the ball from a perfectly circular movement. And with that, we have basically independent variables where we change the translation time within this range, and then the ball phase within this range. And at a given phase and time, we calculate the error of performance. And we basically plot the error of performance as a function of these two variables. We get a solution manifold, which looks like this, where, sorry.
Sorry, if you can just wrap up quickly, we have a couple of questions for you because the session ends in 3 minutes.
Oh, okay. Yeah, it's the last slide, or second to last. So we have basically the initiation phase, the movement time, and the color is basically the color gradient is the error, the normalized error of performance. And the red dots basically indicate the cases where the ball escapes from the cup, so the ball falls out of the cup, so they're not really accessible. And if you look at these low error areas, these dark areas, you can see that there's a similar spiral pattern emerging from these simulations, which resembles the performance of the subjects of some of the subjects in the experiment. And we can basically overlay the data on top of the solution manifold, and you can see that they basically follow these dark areas of low error, and it happens for some of the subjects. Now note that this is not a model of the behavior. This is just a simple simulation that suggests that if subjects start with a given initiation phase and a given movement time, it facilitates the restoring of the ball rotation at the end. So with that, I'm just going to wrap it up with summary. So we showed that subjects basically modulate the ball phase.
You're going to get kicked out of the room in 2 minutes. Why don't you why don't you go ahead and answer one of the questions at least? So the first question is, could you address the difference between your experiment design and how the wrists and fingers in reality are used to manipulate a wine glass?
Sorry, how the fingers are used to. Can you repeat the second part of the question?
Yeah, so it basically asks, could you talk about the differences between your experiment design and how the wrists and fingers are used in reality?
Oh, well, I mean, it would be a different experiment if you have a different grip of the object, definitely. So the experiment design wasn't necessarily to replicate a swirling of the wine, but we just wanted to basically have a hand. I mean, we had limitations in capturing the cup with the algorithm, the shape detection algorithm. So we had to design the cup in a certain way. But I would say that if you were to grip the cup in a different way, then the results probably would be different, just because it's a sort of different configuration.
Okay, I think we'll have to close now because the next session is supposed to start, but I've screenshotted the questions and I'll send them to you. Maybe you can reach out to the attendees later. Thanks, everyone.
To have more audience. Oh, okay, numbers are popping up now. Great, now we can start. Hello, everyone, welcome to the session in the afternoon, at least in the East time. I'm not sure for other people. So I'm excited and honored to be the MC for this session. My name is Anchi Wu. I'm a postdoc research fellow at Columbia University, working with William Panensky and John Cunningham. And we have a really exciting line of speakers for this session to talk about attention learning happening in primary visual cortex. I'm not a big experimental neuroscientist, but I've worked with data from visual cortex as well. So I'm very excited to learn about the new findings and research happening in this area. And let me just briefly introduce the speakers we have. We have Doris Boina, who's a grad student working with Eric Schönbrunn and Stefan Mihalas. She's a grad student at the University of Washington. We also have Shraddha Shah, who's a graduate student working with Farron Briggs at University of Rochester Medical Center. And finally, we have Adil Khan, who is a PI at King's College London. So it's great to have these three amazing speakers, and I can't wait to start. So let's kick it off, starting with Doris' talk. She's gonna talk about single circuit in V1 capable of switching context during movement using VIP population as a switch. So Doris, maybe you can take the stage.
Thank you, Anchi. So as Anchi mentioned, I'm going to talk about a circuit in the visual cortex that can switch context. So I'm going to start with the observation that animals adapt to different environments. And a great example is the cheetah, whose whole body is adapted to achieve the high speed necessary to catch its prey. What I'm going to focus on today is less the adaptations of the body and more adaptations of the brain, and specifically the visual cortex to the statistical regularities of the environment. Indeed, we see that animals can navigate different environments and switch between them very easily. So it's just natural to ask the question of how brains adapt to the different environments and how they switch contexts. So by context, I'll define context here as being a state defined by certain statistics of the environment. And I'll focus in this short talk on the moving and static context, while mentioning that I can easily generalize this to other contexts as well. I'm going to focus on a circuit in the primary visual cortex that is considered to be a switching circuit. Why is it a switching circuit? It's a switching circuit because the activity in the circuit is highly modulated to the context. So during the moving state, we see that a certain population of neurons has activities that are highly modulated and highly, that are much stronger. We can also notice the fact that this circuit has been very well described in the literature. So we know that there are four main populations of neurons involved. There's the pyramidal excitatory population, and there's three inhibitory interneuron populations, the SST, the PV, and the VIP. The latter is important for our work here, our discussion here. We see that this VIP neuron activity is highly correlated with the speed of the animal, and we can think of it as a switch that turns on and off to movement, something that is not true for other types of neurons in the circuit. We can thus make a parallel to a general switching architecture that we can think of, where we can draw a correspondence between these VIP switching units and general switching units that turn on and off to different contexts. So an overview of this short talk is that, in general, I'm going to ask how brains adapt and switch context effectively. And first, I'm going to look at a model of circuit adaptations to the statistics of the environment, which is the context. And then in the second part of the talk, I'm going to look at circuits that switch context, and specifically in the framework of this visual circuit that I just described to you. Okay, so we start by looking at a model of visual processing for static images, and then for moving context in the case of videos. And these models have neural connections shaped by the statistical regularities of the context. Before I go into the nitty-gritty of the model, let me just define a few terms that will keep appearing, in the context of this talk. So classical receptive field is just the patch of space that the neuron responds most strongly to. And extraclassical receptive field is really this larger region of space that the neuron responds indirectly to through interactions with other neurons that have classical receptive field in this extraclassical field. So this model of circuit adaptation has been first proposed by Iyer and Halash in 2017. And it starts with the assumptions that neurons basically code for the probability of features being present. This looks like a complicated math formula. I'm not going to go much into it, except to say that the probabilities of certain features here are equated to the probabilities of features given information in the classical receptive field. This makes up the classical receptive field contribution times some extra classical receptive field contribution from the surround, where these probabilistic quantities get information from basically patches from the surround. And so let's ignore this math formula now, and this is at the basis of the theory. And the idea here is we're going to map these probabilistic quantities to neurobiological quantities like firing rates and weights of the circuit. So we have that the firing rate of a neuron in the circuit is equal to the classical receptive field firing rate, so small f, which is basically the firing rate as if only the classical receptive field was present in the visual space and nothing else, times the extra classical receptive field contribution, which is a weight sum of the firing rates of neurons that respond to surrounding patches n. So I'm going to build a little bit of intuition on this model. So we've mapped the weights W to be a function of probability of co-occurrence of certain features. So what does this mean? What does this mean? Let's suppose I have two features, FM and FN, which are Gabor filters of different orientations. And if these two filters co-occur together often, this means that the weights between neurons that respond strongly to these features are likely very strong and positive. If these features co-occur in the visual space by chance, the weights are likely weak. And if these features co-occur are inversely related to each other, then these weights are strong but negative. Also observe the fact that I've averaged over all images in my data, which means that these W's are representative of different statistics of the data set or of the world that I'm taking into account. Okay, so we can also generalize this framework for movement and as well by integrating not only spatial surround, but spatial temporal surround. So now the firing rate will be equal to the classical receptive field firing rate at time t times the extra classical receptive field contribution that now comes from t minus delta t in the past. So basically the surround comes now from delta t in the past, where delta t you can think of as modeling basically synaptic delay. So the time it takes for the information conveyed by surrounding neurons to reach the other neuron. This means that the weight is a function of a probability of co-occurrence of features that are now separated by delta t time window. So this formula can be generalized basically for both static and moving contexts. The difference is when an animal is static, the features at time t and t minus delta t is identical because nothing moves, which determines a certain statistics, which determines W static. In the moving context, these features are no longer identical, and so this determines a different statistics, hence W moving. So the flow I go through is I have two data sets, say I have images and videos. I compute the weights using the formula I just described to you, and then I plug in the weights to find the firing rates. So okay, these are images, filters, and for in the interest of time, I won't go into describing these weights or firing rates. What I want to convey, though, is that we start with the data set, so different environments, static or moving in the case of images and videos, and through this model, we describe through weights and firing rates, we describe these circuits that are adapted to these different statistics. Great, so we basically covered point one because this is a model for the different contexts. And now we go to the second point, which is the switching circuit. So we start from this idea that so far we have two optimal connectivities, W and firing rates for static and moving contexts respectively, right? We have these two circuits with FR static and W static, FR moving and W moving. But let's remember our V1 circuit, which seems to have a VIP binary switch and seems to do both of these operations. So in this case, we want to ask, how do we find a mapping between the two functional circuits given by star and double star and the physical circuit in V1 that I've sketched here? So I won't go into much detail, but let's say we have the static context. In the static context, this switching V1 circuit has VIP silent, and there's an easy mapping to make then between the, I've colored them as well. Correspondingly, there's an easy correspondence to make between the switching circuit and the static circuit. We just assign the positive and negative W static weights to the connections between neurons. That's an easy correspondence. In the moving circuit, we fix these weights, but now we have VIP. VIP is turned on. And it like the mapping doesn't seem that easy to make between the switching and the moving. But what we want essentially, what we want essentially is for the firing rate during movement to be equal to for the firing rate for the moving circuit to be equal to the firing rate for the switching circuit, at least for the pyramidal neuron. So this leads us to minimizing the loss function, right? We want to minimize FR moving minus FR switching. And the unknowns in this optimization problem will be connections to and from VIP. These are our unknowns. Also, a thing to note, this will be a constrained optimization problem because we're imposing Dale's law for biological realism, right? So now the question is, can we build a switching circuit, i.e. is our optimization problem being solved? And also, how many units do we need? So when we have no VIP units, you see that this loss is quite large. I'm adding VIP units. The loss is going down, but not as much as we'd like, maybe. I'm adding VIP, but I'm also adding feedback from the pyramidal connection, such that now the VIP neurons have information about the input that they didn't have previously. So you see here that for this circuit, our loss function decreases, but it doesn't increase much more than for 5 VIP neurons per unit space. So what conclusion we make that we're able to sort of switch. And we also in the paper, we put this like switching circuit to use, like to do some reconstructions and so on. But the basic idea is we can switch, and we can switch with few units, as opposed to we have 34 neurons per unit space in the model for pyramidal and SST, as opposed to 5 for VIP. So finally, like we can think of these neural architectures where in general we have switching circuits that I just described, and perhaps you have few switching units, as opposed to having, like, for example, a more a simple but sort of arduous and not very useful model is to think that every context has a certain circuit allocated to it, which means there are many connections to learn. And so this circuit could be this kind of switching circuit architecture could be advantageous both in biological circuits and perhaps even in artificial nets, which I'm trying right now. So review of the results, we have studied the biological circuit architecture that can adapt the static and moving context and switch between them. This circuit needs a recurrent connections from the pyramidal to VIP. and few switching VIP units are needed for the switch. And thank you all for your attention and for everybody who has helped me put this project together.
Thanks, Doris, for the great talk. It's very interesting. I've been thinking about questions, not just on purpose, it's just like, yeah, curious about these technical details a lot while I was listening to the talk. Yeah, maybe let me kick off with a question about the talk, some more technical. One thing is, I'm not really fully understanding about the difference between moving and switching. So when you talk about, so there's a slide where you impose a L2 norm between the weights during switching and moving. So my understanding, moving is like when animal is moving, right? Switching
right,
is you want to switch between these two states, like the static states and the moving state. Is that right? Or
right? Yeah, I might have actually made a mistake with like the names. So the moving circuit is our model ideal circuit for moving, which I remember I had these complex math equations that I just described, right? That's an idealized, that's what the animal would be ideally doing and applying Bayes' rule and like being awesome about getting the environment right. That's this circuit, the moving circuit. The switching circuit is like the V1 circuit when the animal is moving. So I should have said here switching circuit when animal is moving, right? And the switching circuit might not do the optimal thing exactly, but I'm trying to basically make this circuit, the switching circuit when the animal is moving, be the same as this idealized moving circuit, basically. That's why like this would be moving circuit versus switching circuit during movement, right? Because, so I should have like, perhaps made that more clear, but thanks that's, I hope this clarifies it.
Okay, yeah, I think I see. I understand that better now. Yeah, sorry, I just noticed the time is a little bit beyond 15 minutes for the first talk, so we probably need to move on to the second talk. But still, thanks, Doris, for the great talk. And people want to learn more about that, people can contact her offline to further discussion. Okay, so let me introduce Shraddha briefly. I just introduced her. She's a grad student working with Sarah Briggs at University of Rochester, and she's going to talk about attention differentially modulates multi-unit activity in the LGN and V1 of macaque monkeys. Okay, I'll just give the stage to Shraddha. Oh, you mute yourself, Shraddha.
Okay. All right. Okay, thanks for the introduction. So I'll be talking about a study on attentional modulation of multi-unit activity in the LGN and V1 of macaque monkeys. So since my project involves studying attention, I just want to briefly describe or define what we mean by attention in the context of this project and how we operationalize this definition to study attention. So most of you might be familiar with this famous video about viewers being asked to count the number of times the basketball was passed between these players wearing white. And most people were able to guess this correctly, but most of the same people also failed to notice that a person in a gorilla suit walked across the screen and spent almost 9 seconds on the video. So one way to think about attention is that it enhances the selection of behaviorally relevant targets at the cost of ignoring the distractor, which is like the gorilla over here. And often this has been helpful to increase accuracy in detection, discrimination, and search. And this has been shown across all sensory modalities. But a lot of the previous work has been focused on visual attention just because we have a very deep understanding of the structure and function of visual structures. So attention effects have been found all over the visual brain, including in the early steps, which include the LGN or lateral geniculate nucleus, which is the first synapse after the retina, as well as primary visual cortex V1. And usually these effects increase in size along the visual hierarchy. In addition to that, effect sizes also vary within the same structure. So effect sizes within the same structure vary with different neuronal classes, vary with task difficulty, where attention facilitation is higher with increasing task difficulty. Also varies depending on the task relevance of the neuronal responses. And surprisingly, also varies with measurements. So effects are different based on what measurement has been used to capture the neuronal activity. This is especially true in area V1. So on the left, you see a plot of attentional modulation index versus number of neurons. And as you can see, the peak of this plot is at 0. And this has been suggested to show that most neurons in V1 show no attentional modulation. And hence V1 is probably not involved in spatial attention. However, using fMRI, many other studies have shown much larger effect sizes, and the effect sizes are almost 20% larger than what we see in single V1 neurons. This is also seen in LGN, where effect sizes vary vastly across studies using different measurements. And so for this study, we decided to explore this further using an intermediate measure between single unit and fMRI, which is the multi-unit. And the question we pose is whether attentional effects measured with multi-unit activity are equivalent to single unit attentional modulation effects. So in order to study this, we had monkeys performing an attention task, which is the contrast change attention task. And to briefly explain this task, the monkey has to fixate at the central fixation dot, and the color of the fixation dot cues the monkey where he or she should attend. So in this case, the red color indicates that the monkey should attend at the lower drifting grating, which also overlaps with the receptor field of the attended location. And thanks to Doris, everyone now knows what a receptor field is. And in the attend away trials, the monkey has to attend away from the receptor field, which is the upper drifting grating. And at the end of the stimulus display period, the contrast will change of the cued location, and the monkey has to report that contrast change. But we want to ensure that the monkeys really understand this task and are paying attention to the cued location. And so we also include certain invalid trials where the contrast changes at the invalid location. And we would expect that the performance should drop. And so that's indeed what we find for all 3 monkeys, where solid lines is the individual session performance, and dotted lines is the mean accuracy. And we see that all 3 monkeys generally perform much better in the valid attention toward and attention away trials versus invalid. And once we have a proof of concept of a behavior, I just want to briefly draw your attention to this trial structure, and mainly this gray bar, which is the last 1 second before contrast change. And that's the time period I used for analyzing all the recording on neuronal data, which really ensures that the visual stimulation is the exact same on each trial, and we can now compare for different attentional effects. So to collect this data, we recorded from LGN and V1 while monkeys performed this task using single or multi-channel electrodes, and obtained the raw voltage signal and did some filtering processes and obtained this high frequency wideband signal. And because we really want to thoroughly test whether attention modulation of multi-unit is similar or different to single units, we chose to threshold using 3 different values and sort our multi-units in 3 different ways, shown here by 3 different colors. For example, for an example unit from monkey O, for each recorded unit, now we have 3 different multi-units as low, medium, and high. And as one would expect, the number of spikes reduces with the increasing threshold, but the visual dynamics and the attentional modulation are qualitatively consistent across these different multi-units. And we further subdivided or classified the data from V1 into different layers, namely the supragranular, granular, and infragranular. And so once we have all this data processed and classified, I'm now combining data across monkeys, sessions, and different recording contexts. And this is data from the medium threshold, where on average, the threshold was 2 standard deviations away from the mean of the wideband signal. And in red is the neuronal response from attend toward, and in blue is the neuronal response from attend away. So we can see that in LGN, both the curves are pretty much on top of each other, and we don't see any differences. And there are error bars in this plot, but it's pretty hard to see. And in SG, G, and IG in V1, we see that the red curve overrides the blue curve. And we see similar patterns of modulation in both the low and high threshold conditions. So to further quantify the differences between the attend toward and attend away condition, we use an attention index measure, which is mainly the difference over the sum of average firing rate and attend toward versus attend away trials. So in this example, PSTH, we compute the difference between this red curve and the blue curve across the entire time period. And this gives us an AI of 0.33. And because this is a bounded index, positive values means attentional facilitation and negative values means attentional suppression. So we compute this AI for all multi-units and combine across different structures and different thresholds. And over here, I'm showing histogram plots plotted with attention index on the x-axis and number of multi-units on the y-axis. So in general, we see that the distributions become broader with increasing threshold as the variance increases. But these dashed lines, which are the mean AIs for each condition, also shown in the table on the right, are pretty consistent across thresholds for each structure. So in LGN, we see that the mean AI is very small and close to 0 and not statistically different from 0. Whereas for SG, G, and IG, for low and medium threshold, we see that the line has shifted to the right and is significantly different from 0. Hence, these regions are on average facilitated by attention. And we don't see that for SG and G in high threshold, but the main takeaway here is that these effect sizes are much larger than what we would expect from single unit. These are almost tenfold more than reported effect sizes in single V1 neurons. So to further go into this data, we also looked at dynamics of the responses. And for this, I am plotting this data using confidence interval ranges. And these individual lines represent the range of the mean AI for each structure. And once again, because we know that attention is a dynamic process and shows fluctuations around 200 to 300 milliseconds, we decided to break the 1 second window into 3 smaller periods, which allows us to capture any dynamics if they are present in the multi-unit data. And we see that for low and medium threshold, the effects are pretty consistent across time for each structure. And this is very similar to what we saw on the previous slide, even though the data format was different. And for the high threshold, we see some interesting dynamics that emerge, especially in the beginning time period where we see that the G and IG are robustly facilitated by attention. And this was interesting to see because this reflects the local circuitry that we know from V1, where G and IG both receive input from LGN and are strongly interconnected. And it's consistent with what we know about attention effects within LGN and V1, where attention enhances the synaptic efficacy between LGN and V1 and also V1 local circuits. And also the fact that IG sends back feedback to LGN and attention enhances that feedback as well. So the main takeaway here is that effects are pretty consistent across thresholds and time, even though we start seeing more dynamics and changes in the magnitude in high threshold. And interestingly, the effects that we see for high threshold, both in terms of magnitude and dynamics, are very similar to what we've seen for single unit data from the exact same data set. So to conclude results, we mainly find differential attentional effects in LGN and V1. And these differences are pretty consistent across different multi-unit thresholds and across time periods. And mainly that we see attentional facilitation in V1, which is much larger than effect sizes using single V1 neurons. And it's actually interestingly more comparable to fMRI effect sizes. So this was really a simple exercise to point out that these effect sizes can really depend on what measurement we use and are not really illustrative of how a particular region is involved in attention. So the main takeaways are what I would call cautionary tale from this simple exercise of analyzing this data and making these comparisons, is that attentional effect sizes do depend on measurement. And often many studies combine multi-unit and single unit recordings to study attention, but that may not be appropriate depending on where we are recording from. And just the lack of effect size may not be indicative of the lack of a region's involvement in attention. So we must focus on more mechanistic accounts of attention and move beyond effects, just reporting mean effect sizes. And one candidate hypothesis that we propose for the differences that we see in LGN and V1 relies on the way multi-unit captures activity across neurons. So we know that multi-unit captures activity around 200 μm around the recording contact. This is just an illustration of what that would look like with 3 different neuron types. And so basically multi-units are pooling signals embedded in the functional architecture. And we also know that LGN and V1 are both locally heterogeneous, but also have very distinct functional architectures, where the main organizing principle in LGN is the layered structure, whereas in V1, it's the columnar structure. So that could explain one reason why we see these differences in effects and which are hugely inconsistent across what we know from previous work. So basically when we are talking about attentional modulation of population activity, we should think about that in the context of the underlying functional architecture. And with that, I would like to thank my lab and especially the previous members who collected this data. And thank you for attention. And I will take any comments and questions.
Okay, thanks, Shraddha, for the great talk about attention. Yeah, we're pretty tight on time. So, there's a question from the audience. Do you have LFP data from these areas, and how might that compare to multi-unit activity?
Yeah, so previous students have published on the LFP data for this same data set, and they find interesting dynamics based on what time period they're looking at and which are consistent with feed forward and feedback mechanisms, but on average, the amplitude of the effects is not as high. And I think that's the main point, where just looking at the amplitude or magnitude of the effect really varies with the measurement and doesn't really tell us much about what a region is doing in attention.
Okay.
But you can find the paper. It has been published 2 years ago or a year ago.
Okay, thanks. Hope that answered the question. We have another question. Meanwhile, maybe Adil, you can try to set up the sharing. Just one quick question. How would you reconcile the MUA results with single neuron and the fMRI results?
Right, and I think basically we have shown through other work that we can find these mechanistic details in the diversity of responses in V1 with single neuron. But I think the main takeaway is that instead of thinking of it as reconciling, maybe we need to think that the measurement is a reflection of several different factors, including the functional architecture. So I think really exactly how to measure attention or how to think of attention must be done across multiple different parameters and not just attention index. That could be one way to think about it. Like I think one way to further explore this project would be to test out few different analyses with the multi-unit activity and see like how it affects the variability or Fano factor or like synaptic efficacy between the two regions. And that could provide a better insight into how consistent these two measures are when we see more holistically with the many different ways we think about attention.
Okay, thanks, Shraddha. Given the time limit, okay, we have one more question from the audience from Habiba. So, but we have time limit, so maybe we can save it for till the last of the end of the session to see if we have more time to have Shraddha to finish that question. Yeah, thank you. Thanks, Shraddha, again. Next, we have Adil, who's a PI at King's College London, and we'll talk more about learning and attention. So I'll just give the stage to Adil.
Thank you very much, Angie. Yes, as Angie said, my name is Adil Khan. I'm a PI at King's College London. Right. So in order to survive, animals often need to enhance their ability to detect and then respond to particular behaviorally relevant stimuli. For example, on the time scale of hours to days, animals can gradually learn the behavioral relevance of novel stimuli. And in this example, for instance, this monkey has clearly learned the behavioral relevance of this bottle-shaped object and the reward it contains. In parallel, at a shorter time scale of seconds, as very nicely introduced in the previous talk, animals can also attend to stimuli that are behaviorally relevant at a given time. So for example, this boy who's clearly very able to attend to one object while filtering out a large number of other stimuli from his environment. Now, these two phenomena, learning and attention, therefore serve a very similar purpose, which is to enhance the animal's ability to detect and respond to behaviorally relevant stimuli. And both of these phenomena are believed to be dependent on cortical plasticity. And so the broad question that I'm interested in addressing is, how do both learning and attention change the information processing in the same patch of sensory cortex, right? So, and to address this question, what I will do is I'll first describe how neurons change their responses as mice learn to discriminate visual stimuli, and then how the same neurons subsequently change their responses as these animals switch between attending to and ignoring the same visual stimuli. Right, so to do this, so this is the behavioral paradigm which we developed, which is, so head-fixed mice run through this virtual environment. On the walls of this environment, we can present different stimuli. the mice are trained to perform a very simple go, no-go discrimination task. Whenever they see vertical stripes, they have to lick a reward spout to get a reward. Whenever they see angled stripes, they have to not lick. So this behavior is nice because it's very rapidly learned. Mice become experts in 7 to 8 days. And it's V1 dependent, which you confirmed by silencing V1 while the animals were doing the task. So that's great, but to really tell if V1 is really involved in learning the behavioral relevance of these stimuli, what we need to do is to record from V1 as the animal performs the task. And to do this, what we used is in vivo 2-photon calcium imaging. And so the experiment would look something like this. As the animal is performing the task, we record from this patch of cortex using 2-photon imaging, and at the same time recording behavioral variables such as the pupil diameters, the running speed, etc. So since we know that interneurons, and again, very nicely introduced by Doris earlier, we know that interneurons exert a very powerful influence on cortical processing, and that different classes of interneurons can play very different roles in cortical processing. So we wanted to identify these different classes of interneurons in our data set. And for this, we used a post-hoc immunostaining approach, in which at the end of the experiment, we would take extract the brain, slice it parallel to the imaging plane, and then immunostain the slice for PV, SOM, and VIP markers. And then using image registration, we can find the same neurons back in the originally recorded in vivo data during the behavior. So then at the end, we can go back to our data and say, well, this neuron was clearly a VIP neuron, this neuron was a PV neuron, this neuron was a SOM neuron, and so on. And so with this approach, we obtain this simultaneous activity from PV, SOM, VIP, and pyramidal neurons, and across this behavioral paradigm. So now with this data set, we asked, how do things change with learning? And the most relevant metric for us was the stimulus selectivity, which we defined as the difference in the neural response amplitude to stimulus 1 minus the response to stimulus 2 normalized by the standard deviation. So just a very simple metric. And we asked what happens with learning. And so here's an example neuron, an example pyramidal cell, which pre-learning is not very stimulus selective. It fires approximately the same degree to the 2 stimuli, but post-learning, it becomes very selective, firing mostly for one and not much for the other stimulus. So you can take this metric, average it across the population, and ask what happens across the population. And here what we see is over learning, SOM and VIP interneurons don't change significantly with learning, but we see a very robust increase in stimulus selectivity for pyramidal cells and for PV interneurons. So this was already a very interesting result for us. In particular, we were quite intrigued by the fact that PV interneurons increased their stimulus selectivity, which is kind of contrary to the dominant model of that PV cells are just broadly integrating their inputs from the surrounding pyramidal cells. We dug into this quite a lot. I won't go into this in detail. This is published. But what we think we find is that there's a reorganization of the recurrent connectivity such that there are pyramidal PV subnetworks which form, in which cells connect to each other more strongly if they prefer the same oriented stimuli. So I'll just skip over this, but ask, okay, having established these results, what happens with now attention, right? So to study attention, we use the following behavioral paradigm. We trained mice to go in blockwise between a visual block, which is exactly the same as the behavioral task I just described, that go, no-go visual discrimination paradigm. And after 100 or so trials of the visual block, we switched to an olfactory block. And the task is a very similar odor discrimination, where it's odor 1, lick, odor 2, don't lick. But on a fraction of trials, we throw in the very same visual stimuli, which now the animal, it's completely irrelevant to the task and the animal has to ignore. And the animal, we won't tell the animal when the blocks transition, they figure it out through the changing reward contingencies. And these mice are extremely good at switching back and forth between ignoring and attending the same visual stimuli. So here is the behavioral data. The y-axis is behavioral accuracy. 2 is approximately 85% accuracy. So you can see in the olfactory block, they're performing very, very accurate odor discriminations while completely ignoring the visual stimuli. And just a few seconds later, when you switch to the visual block, the animals are very accurately discriminating between these same 2 visual stimuli. And you can make them switch back and forth as many times as you like. So mice are performing this behavioral switching very, very robustly. And then we can ask what happens to the neural selectivity during the ignore condition and the attend condition of this attention switching paradigm. So here's an example neuron. It's you can see that in the ignore condition, it's not very selective, and in the attend condition, it becomes selective. So clearly there is an ability of attention to induce selectivity. And you can again take this value of selectivity, average it across all neurons, and compare ignore and attend conditions. And what we find is very similar to previously is this ignore that the somatostatin and VIP interneurons don't change their selectivity with attention. But again, we see a very robust increase in pyramidal and PV cells. And from just a few slides ago, you will notice this is very similar to what we saw with learning. An increase in pyramidal and PV, no change in SOM and VIP. So on average, the changes which we see during learning and attention are very similar across these 4 cell classes. And so the next question which we wanted to dig into is, of course, what is happening at the level of individual neurons? Do the same highly plastic cells drive both of these changes? Or in other words, if an individual neuron changes a lot with learning, will the same neuron change a lot with attention also? And since we had recorded from the very same neurons from pre-learning to post-learning to ignore to attend, we could directly address this question. And what we find very interestingly is there's absolutely no correlation between the amount cell changes its selectivity with learning and the amount that same cell changes its selectivity with attention. So selectivity changes are uncorrelated at a single cell level. And mind you, this does not mean that there are two separate populations, one doing learning and one doing attention. No, it just means that they're uncorrelated. So we confirmed that this is not due to any large-scale changes in response properties, which means because from the end of the post-learning of the learning paradigm, it takes 1 to 2 days for them to become experts at the attention switching. So in that time, we compared that the post-learning, the selectivity alone in the post-learning condition versus the selectivity in the attend block of the attention switching task, and that was extremely correlated. So it's showing that the cells are still preferring basically the same stimuli. It's the delta of that selectivity which is uncorrelated. So and then we looked at, of course, at the interneurons, and they showed basically the same effect. No correlation of the amount, not much correlation of the amount that they change with learning versus attention. So essentially, the changes in average selectivity can be very, very similar on average across the population, but at the single cell level, these are occurring through independent populations. So this really leads us to believe that there are distinct mechanisms underlying learning and attention-driven changes, and what might these distinct mechanisms look like? We've looking at a number of places, but I had time to just go through one, which is how the response magnitude changes. So here, what I've plotted is, if you take the response curve in the post-learning condition and subtract the response curve in the pre-learning condition, so post minus pre of the same neuron aligned to this one to stimulus 1. So this is what we call the difference PSTH. And this is each row is one neuron. And if you look at this first learning data, you can see that there's a large number of cells which are blue, which is negative. And negative values essentially means that the neuron reduces its responses to the same stimulus during learning. So there's a dominance of reduction in responses with learning. But with attention, you can see there's a bit more of an even spread. There's some enhancements and some reduction. And so you can plot the histograms of just averaging from 0 to 1, and you can see this clearly large bump of negative values for learning, and attention more nicely spread across 0, in fact, a little bit more enhancement than suppression. Now, again, since we recorded from the same neurons from learning and attention, we can ask, if an individual neuron tends to be suppressed with learning, is it also tending to be suppressed with attention? And the answer is no, there is absolutely no correlation. Once again, the same neurons can do completely different things with learning and attention. So very versatile behavior depending on the behavioral demands. And so finally, just to zoom in on this really interesting subset of neurons, which we call recruited neurons. These are the neurons which start off non-selective and become selective, either over learning or with attention, right? So if we just take those neurons, what do we see? So here are those neurons which over learning, they start off non-selective by definition. They become selective. So we're just plotting the average PSTH of all of these, about 200 or so neurons. They become selective, not by boosting the response to one of the two stimuli, but rather by selectively suppressing the response to one of the two stimuli. But in contrast with attention, non-selective cells become selective by a combination of enhancement of responses and a bit of suppression. Similarly for the neurons, and this is for neurons which end up preferring stimulus 1, neurons which prefer stimulus 2, same story. Learning is happening entirely through suppression. Attention is happening mostly through enhancement and not much change for suppression. So essentially, learning is driven by selective suppression. Attention is largely driven by selective enhancement. And so this really underlines the fact that there are very distinct implementations of these attention-related increase and learning-related increase in selectivity. So just to summarize, what I've shown you is that learning and attention both increase pyramidal and PV selectivity on average, but distinct mechanisms underlie the selectivity learning across the changes across learning and attention. What we think is happening is learning is driven by selective suppression, attention is driven by enhancement plus suppression. So thanks a lot to the people with whom I did this work, our collaborators and funders, and I have a postdoc position available in the lab, so please write to me if you're interested. And thank you very much for your attention.
Thanks, Adil, for the great talk on learning intention, a very thorough study about neural selectivity on for these two different tasks. We have time for one question. We have a question from the audience, Elizabeth. Have you ever overlapped the visual and olfactory cues?
Yes, actually we did do that. And the problem is it's a bit of a mess when you overlap them. The major problem, I think, is that the onsets of olfactory stimuli are never going to be as sharply defined as the onsets of visual stimuli. We did it nevertheless, and we never really looked into it too much in detail, but it's actually not so surprising that we don't really see too much of responses to odors when we're looking in V1. So it was not really that interesting ultimately. And so finally what we did is we just separated out the things completely. So we never really overlapped them much in this analysis. We just kept this visual stimulus comes on, goes off, and then the odor comes on, just to keep it clean.
Okay, thanks. Maybe one quick question. Have you looked at changes in correlated variability between cells across learning and attention state?
Yeah,
more generally,
absolutely.
how does it relate to the study by Ni et al. 2018 that show a shared components in learning and attention?
Yeah, that's a great question. And this is exactly, so one of the things I mentioned we're looking at is in this direction. Here, so since we recorded simultaneously from PV, pyramidal, SOM, and VIP, we can actually look at correlation between every possible combination of cell pairs that way. And what we find is that here's the result essentially, that you have 10 possible combinations, and that's summarized in the square where the thickness of the line indicates how much change in correlation there was. And with learning, there's this dramatic widespread reduction in correlations across everything we measure, almost everything we measure, most strikingly between SOM PV, SOM pyramidal, and SOM VIP. So really SOM cells get decorrelated from the rest of the network. And in contrast with attention, we don't see much changes. You can see most of these lines are very thin, except SOM and VIP within their own population change. So SOM gets more correlated and VIP gets less correlated. And we think, and so I mean, we hope to put, once we have this preprint out, you can have a look, but we've got this model in collaboration with Claudia Klopat's group, which has come up with so Kata there has come up with a model in which she's able to explain this difference, why we get this particular change on the basis of top-down modulations impinging on this very structured circuit. So yeah, I'll be very happy to share you to talk about this if I had more time.
Okay, thanks. That's great. We have just finished everything on time. Thanks again for our great speakers, Doris, Adil, and Shredder, for the great talk. And thanks for the audience questions and for your participants. Yeah, that's basically the end of the session. And people can tweet on Twitter with hashtag NMC3 for continuing discussion. Okay. Yeah, really enjoy the session and have a great time today.
Thank you very much, Angie.
Thank you. Thank you very much.
Okay.
And then
Thank
bye-bye.
you. Bye.
Thanks. See ya.
Bye-bye.
All right, let's wait for a little bit. Everyone looks much colder up there.
It's freezing. At least
That's
like
pretty cold.
For being like early in the season, it's already like only like 0 degrees outside right now.
Yay for Canada.
my timer ready.
We've got like 30 seconds.
Mm-hmm.
Okay, guys, I'm going behind the scenes. Good luck.
Thank you. All right. See you. Let's wait 30 seconds. Yeah, this yesterday we were just talking about that there are a lot of people attending on YouTube instead of Zoom. So
I've been doing.
Yeah.
Yeah.
We should start on time now. So welcome to this session, and you're in room 1 right now, and we have a very interesting line of talks today about how rest anxiety varies as they are in the house in different conditions. And I feel their experience is very relatable during quarantine. And I will introduce our first speaker as she set up her slides. So Emma Littler, she is a master's student in cognitive neuroscience at Queen's University. She studied fear and anxiety responses with a particular interest on what might cause increased susceptibility or increased resilience to experiencing fear and anxiety. So Emma, I'm looking forward to your talk.
Thank you so much. So I'll just share my screen. All right, can everyone see that?
Yeah.
Okay, so thank you so much for that great introduction. So yeah, like I said, I'll be talking about the influence of individually ventilated cage conditions on rats' anxiety-related behaviors. Before we jump into some of the background, I just want to give you guys a brief overview of sort of why this research is important. So all research in Canada is overseen by the Canadian Council on Animal Care, otherwise known as the CCAC. And they operate with the principles of replacement, refinement, and reduction. And today we're going to be focusing on the topic of refinement. So the CCAC defines refinement as the modification of husbandry or experimental procedures to minimize pain and distress in research animals. And as part of the goal of refinement, research equipment is constantly evolving to better protect research animals. And one major component of this is the refinement to rodent housing equipment. However, it is also very important to note that rodent behavior can be altered by the type of housing, and that's what we'll be talking about today. So one somewhat recent refinement to rodent housing equipment is the development of individually ventilated cages, or IVCs. And in many years, lots of research facilities are adopting the use of these IVCs over conventional open top cages. As you can see in the picture at the top with an open top cage, they're a shoebox style cage featuring a wire lid that permits the free transfer of air from inside the cage to outside the cage, whereas the IVC, shown by the picture on the bottom, is hermetically sealed and is thus under forced air ventilation. The use of IVCs does have very clear benefits. The forced air ventilation and the hermetic seal decreases the spread of pathogens between the animals. It allows for very efficient removal of CO2 buildup inside the cage and allows for very precise temperature and humidity control. However, there have also been some unanticipated drawbacks to the use of IVCs. So there is some evidence to suggest that rodents housed in IVCs behave differently than rodents that are housed in conventional open top cages. For example, lodging colleagues found that when they switched over from the use of conventional cages to IVCs, the behavioral phenotype of their mouse model of schizophrenia vanished. Similarly, when Shan et al. Switched to IVCs, the behavioral phenotype of their serotonin transporter knockout rats was also eliminated. And both researchers suggested that IVCs may be increasing rodent stress levels and thus disrupting typical behavior, but the exact cause for this increase remains unknown. So one cage condition that I mentioned that is unique to IVCs is this forced air ventilation system. And this has been posed as a possible explanation for the increased stress levels. The forced air system introduces variables such as draughts, noises, and vibrations that were never present in the conventional open-top cages. So the forced air ventilation used for IVCs can either be operated under two different pressure settings, negative pressure and positive pressure. Under positive pressure, air is pushed into the cage through the ventilation system and then passively diffuses out of the cage through a filtered lid at the top. And we call this positive pressure because the pressure inside the cage is higher than the pressure outside the cage. It has been shown to be effective in reducing the spread of diseases and infections between the animals, since they're receiving a constant stream of fresh filtered air and they're never actually exposed to the air of other animals around them. However, in positive pressure, there's a high number of air changes per hour inside the cage and increased drafts, which can lead to dried out bedding, which has been linked to increased aggressive behavior. And additionally, there is some research to show that rodents actually prefer cages with a fewer amount of air changes per hour. So in contrast to positive pressure, under negative pressure, air is being sucked out of the cage through the ventilation system, but passively diffuses in through the filtered lid at the top. And again, we call this negative pressure because the pressure inside the cage is negative of that of the pressure outside the cage. This has been shown to be reducing the exposure to allergens for those of us who work with the animals, since all the air from inside the cage is being sucked into the ventilation system. So as the environments of these two cages is quite different, one of the first goals of this study was to examine if the anxiety-related behavior of rats differs when they're housed in IVCs under positive or negative pressure. And like I said, since some research has suggested that rodents prefer environments with a fewer amount of air changes per hour and fewer drafts and vibrations, we predicted that animals housed under positive pressure might exhibit higher anxiety-like behavior. So as researchers who are ultimately interested in studying stress and anxiety, we're also interested in looking at any other housing conditions that could possibly impact anxiety-like behavior. And one variable of particular interest to our lab is the social housing of rodents. Social housing shows a very robust stress protective effect on many social species, including rodents, and is a phenomenon termed social buffering. So rodents who are pair housed usually show decreased anxiety-like behavior compared to rodents that are single housed. And this effect is very well documented in traditional open top cages. However, at present, it's not known if this social buffering effect carries over to rodents that are housed in IVCs. And so an additional goal of this study was to examine any social buffering effects in rodents housed in IVCs. So while many biological pathways likely contribute to the phenomenon of social buffering, a large amount of evidence suggests that the oxytocin system plays a major role. And oxytocin is a neuropeptide that is primarily produced in the paraventricular nucleus and the supraoptic nucleus of the hypothalamus. It's released in response to stressful stimuli and acts as an anxiolytic that's reducing anxiety. It's also implicated in prosocial behaviors and is released in response to tactile stimulation. And this is important because when rats are pair housed, they engage in physical play behaviors and thus tactile stimulation that releases oxytocin. And additionally, when more oxytocin is being released, more is actually being produced. And so we would expect that pair-housed animals would have a higher number of oxytocin producing neurons due to upregulation of the oxytocin system. And so since we're ultimately concerned with understanding what IVC housing conditions might be contributing to stress, and if the social housing of rodents can reduce stress levels in IVCs, the final goal of this study was to examine if the oxytocin system is implicated in any of these variables. So to summarize, our first goal was to examine the influence of air pressure on behavior. As I mentioned, positive pressure environments are possibly more stressful. And so we predicted that rats housed under positive pressure would exhibit higher anxiety-like behavior. A second goal was to examine any effects of social buffering on anxiety-like behavior. And since social buffering is very well documented in open top cages, we also predicted that rats pair housed in IVCs would also exhibit lower anxiety-like behavior. And finally, our last goal was to explore the influence of the oxytocin system on behavior. And we predicted that pair-housed rats would have a higher number of oxytocin producing neurons in the PVN due to upregulation of the oxytocin system. So 40 male Long Evans rats were used for this study, and upon arrival to our animal facility, they were randomly assigned to their housing conditions in IVCs. Half were assigned to housing under negative pressure and half were assigned to housing under positive pressure. And then within that, half were assigned to pair housing and half were assigned to single housing, and that resulted in the following groups and ends. So after remaining in their housing conditions for roughly 5 weeks, all animals underwent behavioral testing in the elevated plus maze, which I'll walk you through on the next slide. And then after behavioral testing, their brains were harvested and underwent immunohistochemical staining for oxytocin-producing neurons in the paraventricular nucleus. So the elevated plus maze is a wooden maze that contains 2 closed arms and 2 open arms in the shape of a cross, like you can see in the picture. And during testing, rats are placed in the center of the maze and they're left to explore for about 5 minutes. And this test relies on rodents' innate fear of open spaces and their tendency to spend more time in dark and enclosed spaces, where they're theoretically more protected from potential predation. When placed in the maze, rats tend to spend the majority of the 5 minutes in the closed arms. And so any level of open arm activity can then be used as a measure of anxiety-like behavior, with more open arm activity indicating less anxiety-like behavior. And the most common measure that we use to quantify this is the percentage of time that they spend in the open arms. So the results from the EPM didn't actually reveal any main effects of housing or air pressure on their own, but did reveal a significant interaction. And the interaction was followed up with post hoc tests that revealed that only under positive pressure was there a main effect of housing, such that pair housed rats spent more time on the open arms than single housed rats under positive pressure. And these results are actually quite interesting because we did originally predict that there would be an overall effect of housing with all pair housed animals spending more time on the open arms, but we only see this effect under positive pressure. And this leads us to believe, like we originally predicted, that positive pressure is a more stressful environment. And as you can see in the graph, under positive pressure, the single housed rat spent the least amount of time on the open arm. And that maybe due to this high stressful environment, the effects of the social buffering are more pronounced. So I'll go over the results from the oxytocin. So contrary to what we originally thought, there were actually no differences in the overall number of oxytocin producing neurons between groups. We originally did think that pair housed rats would have a higher number due to upregulation of the oxytocin system. However, we did see a significant positive correlation between the number of oxytocin producing neurons and open arm activity in the EPM, with more neurons being correlated with more open arm activity. And this further suggests that oxytocin has stress reducing properties. So to summarize, it does seem like positive pressure is a more stressful environment for rodents. As I said earlier, drafts inside the cages and the higher number of air changes per hour may be responsible for this. And this is in line with previous evidence that actually shows that mice living in IVCs with a higher number of air changes per hour displayed increased anxiety-like behavior on the EPM. We only saw effects of social buffering under positive pressure. So if positive pressure is truly a more stressful environment, it may be more sensitive to the stress-reducing effects of social buffering. However, it is still odd that we didn't see an overall effect of pair housing because it is a very well-documented phenomenon. And again, this does lead us to believe that IVCs might be influencing typical rodent behavior in general. And finally, although we didn't see any differences in the number of oxytocin-producing neurons between groups, there was a positive correlation between the number of neurons and open arm activity. And this is in line with previous evidence that also shows similar findings. So before I finish, I just want to leave you with some thought-provoking questions and some future directions. So first, what are the implications of these findings for the well-being of laboratory rodents? The ultimate goal is the refinement of housing conditions to better serve laboratory animals and reduce any suffering. So future research should explore how housing conditions can impact rodent stress levels. And our lab actually has future plans to compare the use of IVCs to open-top cages. And second, how does this impact our ability to conduct behavioral research? Because it does seem like IVCs are impacting typical behavior. And as behavioral researchers, anything that's impacting typical rodent behavior can confound our results. And so more future research should look into how any environmental factors, including housing conditions, can impact rodent behavior. So thank you for listening. And if you have any questions, let me know.
Thank you very much for the interesting talk. Let's see if we have questions. Please post questions to the Q&A session, and while we're waiting for the questions, I will ask you something. This is very interesting to me, and I think like rodents are relatively more olfactory oriented, But would that, like, when they're housed individually, can they see other rats? Would that help?
So that's actually a really interesting question and something that I didn't really touch on in the presentation because I didn't have time. But the thing with IVCs is that because they're hermetically sealed, they can't smell any other rodents around them. And because rodents are such social animals and do rely on a lot of olfactory cues, that is another variable that we're actually looking at, is the transmission of olfactory cues between the animals. When rodents are housed in cages next to each other, they don't have great vision, so they actually can't see any of the animals around them, but they do get olfactory cues if they're housed in those conventional cages where the air can transfer between both of them.
Hmm. Yeah, I forgot that they can't really see that well.
Yeah.
Yeah, that's really interesting. I think I have other questions if there's nobody else is asking. And do you think the positive pressure that's maybe induced, like, matters more is they also probably need to smell themselves? I don't know. Would that be the case?
Smell themselves? Like how so?
If it is positively ventilated, it would seem that whatever their own odor they're producing, like it will feel like a home cage. It probably actually smells like themselves. This is just
yeah. No, that's an interesting question because under both pressure systems, like the air is being removed from the cage like either way. Under positive pressure, it's a little bit more intense because the air is being like pushed in versus like sucked out. I don't really know much about like how rodents sort of like nest in their house or nest in their cages and then like respond to like what they've deemed as like their like safe space. I'm not really sure, but that's actually a really interesting thing to think about because in open top cages, they would not be getting that like change in air. And so I guess their cage would smell similar to them all the time.
Mm-hmm. Very cool.
Yeah.
All right, now it's time for us to move on to our next speaker. Our next speaker is Alex Prosserman. He is a first-year graduate student at Queen's University. He completed this project for his fourth-year undergraduate honor thesis with Dr. Janet Menars. He hoped to shed some light on the importance of understanding how individual ventilated cage system can impact lab rodents and their behavior. So I'm really looking forward to your talk, Alex. Take it away.
Thank you. So can you hear me? Perfect. Okay. So hi, everyone. My name is Alex Prosserman, and I'm a first-year grad student studying neuroscience at Queen's University in Kingston, Canada. So today I'll be talking about the influence of social housing and air pressure on rats' anxiety-related behavior, similar to Emma. So to start, most federal committees concerned with the regulation of animal care are mandating the principle of refinement, which is changing some aspect of an experiment that results in a reduction or replacement of animals, or a reduction of any pain, stress, or distress that animals may experience. One of the most recent refinements is the replacement of classical open-top cages, shown here on the left, with new individually ventilated cages, which I'll now refer to as IVCs, shown here on the right. IVCs are sealed cages where air must pass through a filtering system before entering the cage. And the cages have two air ventilators to control the air pressure, one pushing air into the cage and another pulling air out of the cage. The cages can be ventilated under either positive or negative air pressure, and this can be a kind of a confusing topic, so I'm going to walk you through it. Under positive air pressure, there's more air being ventilated into the cage than is being ventilated out. So as you can see, there's a higher concentration of air inside the cage than outside the cage. Because of this, the air inside the cage passively diffuses out in an attempt to balance this concentration gradient. As all of the air entering the cage is filtered, rodents can be protected from airborne diseases. However, a drawback of positive pressure is that the air may dry out the cage environment, which has caused aggression in rats. Conversely, under negative air pressure, more air is being pulled out of the cage than is being pushed in. So therefore, in an attempt to balance the concentration gradient, some air passively diffuses back into the cage. And as all of the air leaving the cage is filtered, this can protect lab staff from airborne allergens within the cage. IVCs are also beneficial because frequent air changes improve its microclimate by eliminating the buildup of carbon dioxide and ammonia. However, there are some drawbacks to using IVCs. Firstly, some evidence suggests that rodents living in IVCs can consume food in an atypical manner. Secondly, IVCs can alter behavioral phenotypes, such as a well-characterized genetic model of schizophrenia that was completely altered when switching from OTCs to IVCs. And finally, and most importantly for my study, some evidence suggests that IVCs can increase rats' anxiety levels. So because IVCs might increase rats' anxiety, we thought maybe we can alleviate that by pair housing them, because it's common for rats housed in pairs to be less anxious than rats housed alone. So therefore, our primary question was whether single and socially housed rats living in IVCs would differ in their responses to an anxiety test. And also a supplemental goal of our study was to determine if rodent behavior could be influenced by maintaining the IVCs under either positive or negative air pressure. Social housing may also have implications on the body's oxytocin system. Oxytocin is a hormone produced by cells in the paraventricular nucleus and the supraoptic nucleus of the hypothalamus. Oxytocin has anxiety-reducing effects on rats. For example, oxytocin administration decreased rats' corticosterone response to noise stress. So this brings me to my third question. Could social housing in IVCs also influence the density of oxytocin-producing cells in the hypothalamus? So to answer these questions, I used 40 male Long Evans rats from Charles River, Quebec. They lived on a 12-hour light and dark cycle with free access to food and water. And when they arrived, 10 rats were randomly assigned to each of the following 4 conditions. The first was single housing in positive air pressure. The second was single housing negative air pressure. The third was paired housing in positive air pressure. And then the last was paired housing in negative air pressure. So after the rats lived in their respective housing conditions for 6 weeks, I conducted the novelty-induced suppression of feeding test, also called the NSFT. This test compares a rat's latency to consume a tasty snack in a familiar and novel environment. So what I did is for 4 consecutive habituation days in a row, the rats were placed in a clear open top cage with a bowl containing a graham cracker, shown here by the picture on the left. Then I measured the time it took for each rat to initiate the consumption of the graham cracker. And if they didn't begin eating after 10 minutes, I ended the trial and placed each rat and the graham cracker back in their home cage. Then on the 5th day, I conducted the same test in a novel room with a novel cage and bowl, shown here by the picture on the right. And prolonged times to begin feeding were an indication of anxiety. So before I conducted the test, I predicted that single housing would increase the rats' latencies to initiate snack consumption in the NSFT, suggesting increased anxiety-like behavior. I also predicted that positive pressure would increase their latencies, because as I mentioned, positive pressure can dry out the cage environment, which has caused aggression before. So what did I find? Well, my first result illustrates that there was no effect of social housing. So here on the y-axis is the rat's latency to initiate snack consumption measured in seconds. And on the x-axis was the testing day with habituation days 1, 2, 3, and 4, and then the novel cage test on the 5th day. And as you can see, there is no difference in the rat's latencies to initiate snack consumption if they were pair housed, shown here by the solid line, or if they were single housed, shown here by the dotted line. And honestly, this result completely surprised me, as the anxiety-reducing effects of social housing are quite robust in the literature. So my first thought was, could these results be due to an error in the testing procedure? But as you can see here, I obtained a characteristic U-shaped latency profile of the NSFT test, whereby the highest latencies occurred on days when novelty was a component. That is on the first habituation day and during the novel cage test day. And this was irrespective of housing and air pressure conditions. So this suggested that my failure to observe evidence of social buffering does not appear due to problems with the testing procedure, and that IVCs may be inherently stressful for rats. So next, and counter to my original hypothesis, I observed a marked main effect of air pressure. And that is rats housed under negative air pressure, as indicated by the solid line, took significantly longer than those housed under positive air pressure, as indicated by the dotted line, to initiate snack consumption. And this implies that the rats living under negative air pressure were more anxious than the rats living under positive air pressure. So importantly, there were no group differences in body weight, which suggests that appetitive factors did not contribute to these group differences. One possibility is that the rats housed under negative air pressure perceived a relatively greater threat from the possibility of facing conspecifics in the colony, that is, other rats in the colony. So under negative air pressure, the rats were exposed to cues from the colony room, such as the sounds and scents of other rodents. it follows that the rats living under negative air pressure might have been more aware of the presence of other rats in the colony room. And since rodents naturally compete for food sources, the rats living under negative air pressure may have suspected that they were entering the territory of other rodents during testing, and thus exercised greater caution to initiate snack consumption. Conversely, the rats living under positive air pressure were not exposed to olfactory cues in the colony room, which might explain why they were less cautious during the test. But ultimately, it's not really clear why the rats living under negative air pressure took longer to initiate consumption in the NSFT. So onto the immunohistochemistry component. So to count the number of oxytocin producing cells in the hypothalamus, I used antibodies that specifically binded to and labeled those cells. Then I used a microscope to count the number of immunoreactive cells. And I predicted that single-housed rats would have fewer oxytocin labeled cells in the hypothalamus. And my immunohistochemical analysis revealed a marginally significant housing and air pressure interaction, such that single-housed rats housed under negative air pressure had significantly fewer oxytocin labeled cells in the posterior parvocellular region of the paraventricular nucleus relative to single-housed rats under positive air pressure. And given the purported role of oxytocin in anxiety reduction, it seems likely that single-housed rats under negative air pressure were more anxious than single-housed rats under positive air pressure. And this outcome maps onto my main effective air pressure in the NSFT, but it's not clear why this was only apparent in the single-housed group. And if anyone has any ideas or comments, I'd love to hear from you about that. So to summarize, firstly, there is no evidence that social housing influenced the rats' latencies to initiate snack consumption across the habituation or test trials in the NSFT. Secondly, there is a marked and unexpected main effective air pressure, such that the rats housed under negative air pressure took longer to bite the palatable snack than those housed under positive air pressure. Finally, single-housed rats under positive air pressure displayed more oxytocin-labeled cells in the PAPO of the hypothalamus. And these results suggest that IVCs can alter the well-characterized anxiety-reducing effect of social housing in laboratory rodents, and actually might in fact influence other well-characterized displays of rodent behavior. So it's really important that we further comprehend how individually ventilated cage systems can impact laboratory rodents and their behaviors. And this would both to ensure that research efforts are robust and to advance the well-being of animals used in research. And at the very least, my findings suggest that researchers need to cautiously consider the form of housing condition used for experimental rodents. So to finish up, I'd like to sincerely thank my supervisor, Dr. Janet Menard, as well as NSERC for funding our research, and Samantha Irwin and Emma Littler for their support and help with this research. So thank you.
Thank you so much. And let's see if there are questions. While we're waiting for questions, I will ask something. So I didn't quite get it that how the negative one, what was your explanation for when the negative pressure will make them feel like more competitive for food?
Because under negative air pressure, they're exposed to sounds and scents from the colony room. So my explanation for that is they might have suspected during testing that they were entering the territory of other rodents and therefore exercised greater caution to initiate snack consumption, whereas the positive pressure rats weren't exposed to these cues. So during testing, they would possibly be less cautious.
So we have a question. Wonderful talk. One question, is this phenotype related to FVC specific to rats or can be seen in mice as well?
In the study, we just used rats. I'm actually not too sure if it can be seen in mice as well, but to the best of my knowledge, it is only rats.
Very interesting. I think I even have more questions. Yeah, do you think it is the nature of the task that it is sort of, because social housing gives them the sense that there is a possible competitor for food, do you think that might be weakening the benefits of social housing because the behavioral task is very food related and it introduces a sense of competition?
Yeah, definitely. That's a really good point. I didn't think about that one before, but yeah, definitely that could have an effect.
That's really good. I want a tasty treat, but
Yeah.
and great. Now I think we should move on to our next speaker. Our next speaker is Ben Stretch. He's a master's student at Queen's University, and his research focuses on how lab conditions in animal colonies can affect indices of anxiety. And Ben, I'm looking forward to your talk.
Okay, can everyone hear me? Yeah, perfect. Okay, so hey everyone, my name is Ben Stretch, and today I'm going to be talking to you about my recent work, evidence of social buffering in rats housed in individually ventilated cages, also known as IVCs. So a little bit of background before we really get going. So historically, rodents have been kept in these open top cages or OTCs, and those use one ventilation system for a group of cages. So you can see an IVC in kind of the top right-hand image there. So it's really just a plastic tub with a wire lid. Recently, however, there's been a big push to switch over to these individually ventilated cages or IVCs. And you can see one of those in the image at the bottom. And so that's actually a hermetically sealed plexiglass chamber. And just like the name suggests, it actually has its own individual ventilation system. And so at the time of the study, it was really unclear to what extent the use of IVCs actually affected behavioral outcomes in rodents. So something else that's good to understand when we're talking about IVCs is that they can be held under two different pressure conditions. So under positive pressure, the internal cage pressure is higher than that of the surrounding room. Air is forced in through the ventilation system and can passively flow out. And negative air pressure is just the opposite. So air can passively flow into the cage, and it actually is forced out through the ventilation system. So part of the reason that there was kind of this big push to start using IVC systems is that they did have a lot of benefits, including a decreased cost, space, and number of cage cleanings that are required. They reduce the possibility of airborne disease outbreak in rats and also protect staff from aeroallergen exposure. And finally, they also prevent the buildup of volatile organic compounds like ammonia and carbon dioxide. That being said, a lot of the early research that was done that used IVCs did note some behavioral abnormalities, including an increased sensitivity to drugs, altered consumption of food and water, altered behavioral phenotypes, and higher overall anxiety levels. And so this last point, the higher overall anxiety, this was something that we really wanted to kind of research further, because since laboratory animals spend really a great deal of time in their own home cage, if that environment is anxiety inducing, then it would be a source of chronic stress, which would have a significant impact on research outcomes. So one way to kind of counteract anxiety is through the use of social housing. So a lot of research has shown that social isolation causes both the dysfunction of neurotransmitters and neural hormone dysregulation. And previous studies have also indicated that social housing typically reduces stress and anxiety, a term that's often coined as social buffering. But again, it was really unclear if the protective effects of social housing would still occur if animals were pair housed in an IVC system. And finally, we also decided to look at the oxytocin system for this study. So oxytocin is released from the supraoptic nuclei and the paraventricular nuclei of the hypothalamus in response to stressful environmental stimuli. And so it's also important to note that oxytocin is actually a self-regulating system. So the more oxytocin that's released means that more is actually being produced as well. And so social contact has been shown to release oxytocin. So what that means is that pair housing animals that will generally increase their social contact, and that will typically result in an upregulation of the oxytocin system. But once again, all of these studies have been done in OTCs, and so it was unclear if this would still be the same in an IVC system. So the study design, we took 40 male Long Evans rats and split them into 4 housing conditions. So 10 were housed in pairs under positive pressure, 10 were housed in pairs under negative pressure, 10 were housed alone under positive pressure, and the last 10 were housed alone under negative pressure. And then after spending 8 weeks acclimatizing to their respective housing conditions, all of the rats underwent the shock probe burying test. So the shock probe burying chamber is really just a plexiglass cube with an open top. And then a small electrified probe is actually inserted into the chamber. And then when the rat interacts with the probe, they receive a mild electrical shock. And so kind of the natural defensive behavior that this elicits is that the rat actually tries to bury the probe. And so defensive burying is the term that's used to describe when the rat takes its front paws and actually begins to push the wood chip bedding towards the probe. And so a lot of previous studies have indicated that higher levels of defensive burying typically correlate with higher levels of anxiety. We also measured the number of stretch attend postures. And so a stretch attend is when the rat stretches its whole body towards the perceived threat. In this case, it would be the probe. And then they quickly withdraw and retreat while keeping their eyes kind of locked on that probe. We also measured the number of rears, or the number of times that the rat stood up on its hind paws. And so this is important to measure because typically when a rat's faced with a threat, it's going to have increased levels of defensive behaviors like burying and like stretch attends. But rearing actually doesn't have a defensive purpose. So when faced with a threat, non-defensive behaviors are typically suppressed. And finally, the last two measures there, the number of contact-induced shocks and the score of each behavioral reaction to each shock. The last two measures are really just used to kind of control for confounds. So it's pretty logical to assume that if a rat gets shocked a lot more, that would also cause higher levels of anxiety, which would also cause higher levels of burying. So the goal of kind of taking the measurements of these defensive behaviors is that we want it to be not due to the effects of simply interacting with the probe itself. So I had two main predictions for this study. First, that pair-housed rats would be less anxious as indexed by the shock probe burying test compared to the single-housed rats. And secondly, that pair-housed rats would also have a higher density of hypothalamic oxytocin-producing cells compared to single-housed rats. I also did compare positive and negative air pressure, but because there hasn't really been an existing body of literature that's compared anxiety in terms of positive and negative air pressures in IVCs, I couldn't really make a directional prediction. So here are the results from the shock probe burying test. So you can see from the graphs that for the three main measures, burying, stretch attends, and rearing, there weren't actually differences between positive and negative air pressure. However, the pair-housed animals did bury significantly more. They did have significantly higher levels of stretch attend postures, and they had significantly lower levels of rearing. So this does suggest that the pair-housed animals actually had higher levels of these kind of classic defensive behaviors. And so this is really surprising because in the prevailing literature, pair-housing animals typically reduces their anxiety levels, and that's typically reflected in a decrease in defensive burying. Furthermore, to make it just that little bit extra weird, the single-housed animals buried not only less than pair-housed animals, but actually when we compared the levels of the single-housed animals to those levels of defensive burying in all of the previous studies, they actually had significantly lower burying levels on average. So something to keep in mind with these kind of strange results is that all of the previous literature was done using an OTC system, while we used an IVC system. And so keep these kind of surprising results in mind. I will be coming back to them. So after the behavioral results, we conducted an immunohistochemical analysis. So the rat brain tissue was processed to label oxytocin-producing cells in the hypothalamus. And then at that point, we're actually able to quantify the number of oxytocin-producing cells using a light microscope. So you can see here right away that the pair-housed animals and the single-housed animals under positive pressure, those three groups didn't actually significantly differ from each other. However, the single-housed negative pressure animals did see a significant decrease in oxytocin-labeled cells. And so something that's interesting to note from the literature is that chronic stress can actually cause the suppression of oxytocin production. So then it could be that the single-housed negative pressure animals were under chronic stress. And if that is the case, then that kind of begs the question, does this mean that the pair-housed negative pressure animals, them being paired with a conspecific, did that have a protective effect that prevented a similar decrease in oxytocin-producing cells? And kind of to sum up all of the results that we got, firstly, just to reiterate, our findings weren't consistent with our initial hypotheses. And so as I kind of got into on the previous slide, chronic stress is one explanation that could explain why the single-housed negative pressure animals saw a decrease in oxytocin-producing cells. And what's also important to note is that the literature suggests that not only can chronic stress cause this decrease, but it specifically does that in the parvocellular regions of the PVN, which was the exact region that I was investigating. So then to kind of wrap it all up, we kind of took a look and said, okay, so we've come up with this hypothesis of chronic stress. Can we kind of use chronic stress to explain the results of the shock probe burying test? And as it turns out, yes, we can. And it relates to the idea of active and passive coping strategies. So an active coping strategy is one where an animal kind of directly deals or handles the threat in front of it. And a passive coping strategy is one where it actually just actively avoids the problem. And so while previous studies have indicated that acute levels of stress cause an increase in defensive burying, chronic stress actually causes a decrease in defensive burying. And it does that through the suppression of